Alright. I think I finally got everything working. Whew. OBS crashed. Twice just now. So if you were there for the first two attempts at getting this to work, good on you. Thank you for arriving to the stream early. Oh man. I hope that this stream doesn't explode on itself when I try to play Death Stranding, because that was the plan, but I know that my hardware might not be able to handle it, so I don't know. We'll see. If Death Stranding doesn't work, I'll have to switch to a different game. I have a couple of games that we can switch to, but then I have to rebrand the entire stream. Ugh. I guess we can do some Photoshop editing if we want. But for now, you're probably wondering, if you are even able to watch the stream, what we're doing here today, while my roommates go play Nintendo Land in the living room behind me. Yeah, this is going to be a 24-hour stream, or at least that's the plan, unless I totally get destroyed by my mental fog throughout this endeavor. I don't know why I'm so obsessed with wanting to do a 24-hour stream of MCAT prep. I've been wanting to do the stream for months now. Only ever had time to do it now, which is also partially due to the fact that I'm totally screwed for this test. And it is rapidly approaching a lot sooner than you might expect. But, aside from all of that, I don't know. I guess I really wanted bragging rights for, pulling an all-nighter on, the MCAT. Which is really interesting, because what am I even... what is the point of that? Why do I care so much? No one cares about what specific test you do an all-nighter for. This isn't your V card, bro. This is your ability to handle time management skills and tests and exams. But I don't know. I just never got to do that during high school or in college. And by get to do it, I mean I have never pushed myself to such an extent that I would need to do that or something. I don't know. There were many a time I accidentally slept through the time that I was expecting to study, though, so that might be a part of the reason why. I don't know. In any case, always wanted to do this. I am frankly not prepared at all, because I am very tired right now. Which is a great thing to say when I already know that this is going to be twice the amount of length of a normal day for me. Basically only do like 12 hours of productive stuff, which doesn't even include or isn't entirely encapsulated by my educational productivity, academic productivity. No, I am including errands and stuff like that. Ugh. So, this is going to be extremely interesting. Hopefully we can figure out some strategies to keep me awake and keep me attentive throughout this journey. Of course, if Death Stranding crashes OBS again, we're gonna be a little bit more worried about some other stuff. But for now, I think that's enough talking. Oh, wait. I need to explain the plan for Death Stranding and breaks in general. Every 2 hours, or rather, every hour and a half of studying, I plan on dropping in a half-hour break. The problem being that we're already halfway, or we're already a half hour in to the stream, and yeah, this is cutting into my study time, but it is what it is. All that setup was a problem. Actually, I'm hoping that the stream is running properly. I haven't checked in a second. Yep, okay, we're good. So, regardless, I'm gonna be hopping into Death Stranding at 9:30, and then every, yeah, as I've mentioned, hour and a half. We'll do a half hour Death Stranding, or we might switch to something like Signalis, which I definitely want to play, but I was hoping to play Death Stranding because I feel like that's the better game for when you're half asleep. Although I guess if I did Signalis, I could probably finish the game. It's not a 6-hour game either, though. It's like 8 to 10 hours. Whatever. Death Stranding for 6 hours. We'd be chilling. It's mostly going to be cutscenes, if I remember correctly. Whatever. I think that's all the explanation I need to do for now. Unfortunately, people are probably going to hop into the stream, not knowing what's going on. And then I'll explain again eventually. I don't know. Watch out if you are worried about Death Stranding spoilers if you come back later. Oh, and speaking of which, I don't think this stream's probably gonna stay up as a VOD anyway. Lot of copyrighted music going on here. I'm listening to the Xenoblade 3 OST just because I've been inspired by Flute Guy's performance over in the Game Awards, which was absolutely phenomenal. Okay. Is that good? Is that it? Good? Is that it? Are we good? My mind combining stuff already. This is fantastic. All right, let's go.
What is a non-coding RNA?
What is it?
A non-coding RNA, or an mcRNA, as it is
abbreviated,
is a functional RNA molecule that actually skips this last step and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. And there are many examples of non-coding RNAs, including microRNAs, ribosomal RNA, transfer RNA, the list goes on and on. And as we...
Okay, so ncRNAs are going to be along the pathway from DNA to transcription to... Yep, there it is. We already have it right here. ncRNAs are right there. Beautiful.
We go through each of these different types and examples of non-coding RNAs, you'll start to see that there's sort of an emerging theme here, and that is that most of these non-coding RNAs participate in either transcription or translation in one capacity or another. So let's start off with microRNAs. MicroRNAs, or miRNAs, function in transcriptional and post-transcriptional regulation of gene expression, and they do this by base pairing with complementary sequences within mRNA or messenger RNA molecules, and this usually results in gene silencing through translational repression or target degradation. In essence, the mRNA to which these microRNAs bind are prevented from being translated, or they are sent on a pathway for degradation. Now, the next set of non-coding RNAs that we'll be talking about are all involved in translation. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery used to translate mRNA into proteins, and it is made up of one type of RNA molecule, ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids, and so this is another type of non-coding RNA that you'll see in translation. The third type is called SNO RNA, which stands for small nucleolar RNA, and it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear RNAs, primarily through methylation.
Well, I'm already confused. So, what are microRNAs in this case?
So let's start off with microRNAs. MicroRNAs, or miRNAs, function in transcriptional and post-transcriptional regulation of gene expression.
Yes.
And they do this by base pairing with complementary sequences within mRNA.
Okay, but are microRNAs...
And this usually results in gene silencing
through
translational repression or target degradation. In essence, the mRNA to which these microRNAs bind are prevented from being translated, or they are sent on a pathway for degradation.
Right. So I understand that they'll attach to mRNAs. What I'm not clear on is... Oh, well, I guess non-coding RNAs are just a huge umbrella for all sorts of RNA that just don't code. So in that case, miRNA, or microRNA, going to be going right here, which will be translation. So let's go ahead and put gene silencing as its job. And then we already have RNA and rRNA and tRNA. So we'll be good for the split second that they're going to talk about that. And then SNO, SNO RNA. What is that?
The third type is called SNO RNA, which stands for small nucleolar RNA, and it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear RNAs, primarily through methylation, which is the addition of methyl groups, or pseudo-uridylation, which is the addition of an isomer of the nucleoside.
Pseudo-uridylation. Okay. Ah, yes, I've definitely heard of that before. Okay. Protease, it's a small. This is a part of translation. Wait, how is that part of translation?
Which stands for small nucleolar RNA, and it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear RNAs, primarily through methylation, which is the addition of methyl groups, or pseudouridylation, which is the addition of an isomer of the nucleoside uridine. Now, another class of non-coding RNAs are the small nuclear RNAs, or SNRNAs, not to be confused with the SNO RNAs, the small nucleolar RNAs that we just. The third type is called SNO RNA, which stands for small nucleolar RNA, and it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear.
That guide. Sorry, what? They guide what? They guide covalent modification. Oh, okay. So they help out in this system here. Oh yeah, you can see my mouse. Nice. Yeah, okay, so they guide this system. Alright, in that case, I think we can go ahead and put this here. Small nucleolar RNA. I've been stuck on DNA for a while. Maybe we can go ahead and switch to different concepts later on, so that I don't have to just keep on doing the same topic over and over again. Though I do dread switching to math. Oof, that's not going to be fun. Or will it? We'll see. For now, though, here we are. It helps out with translation. That's good. That's very nice.
Ribosomal RNA,
very helpful.
RNA, and small nuclear RNAs, primarily through methylation, which is the addition of methyl groups, or pseudouridylation, which is the addition of an isomer of the nucleoside uridine. Now, another class of non-coding RNAs are the small nuclear RNAs, or SNRNAs, not to be confused with the SNO RNAs, the small nucleolar RNAs that we just talked about. Now, small nuclear RNAs get their name from the fact that the average length of these RNA molecules is approximately 150 nucleotides, and their primary function is in the processing of pre-mRNA in the nucleus. They also aid in the regulation of transcription factors, or a particular RNA polymerase 2, as well as maintaining telomeres, which are the regions of repetitive nucleotide sequences at the end of a chromatid, which protects the end of the chromosome from deterioration during chromosomal replication. Now, snRNA can be associated with a set of specific proteins and form complexes that are called small nuclear ribonucleic proteins, or snRNPs, or sometimes people just call them SNRPs. And there is a special SNRP complex called the spliceosome, made up of 5 small nuclear RNAs and over 150 proteins, that is responsible for splicing or removing the introns contained in messenger RNA, which is a major step in the post-transcriptional modification that takes.
Okay, this is connected to the pre-mRNA, and the spliceosome. So it's over here. Okay, we can do this. We can just move this up a little bit. Snip out the introns. And leave them be. Or I can move them here. How do I want to play this? Yeah, we can go there. Move up the description of spliceosome. It's kind of on its own. There we go. Alright, now we can go ahead and attack the spliceosome with its 2 components, snRNA and snRNP. It is defined as small nuclear RNA. Ooh, speaking of attacks, I can't wait for OChem because I have forgotten everything with OChem and I really want to remember everything because OChem is pretty cool. Spliceosome, and then let's go ahead and place another shape here, which is going to be the snRNP, which I don't even know what that stands for.
Takes place in the nucleus of eukaryotes. And the way that the spliceosome does this is that it binds to specific sequences in the pre-messenger RNA strand and performs 2 sequential trans-esterification reactions that splice out the intron and then ligate the 2 exons to form a mature mRNA. So now you know a little bit more of some examples of non-coding RNAs and some of the functions that they perform within the cell.
Just gonna put SNRP here. And then we'll deal with it later once it becomes more relevant. All right, nice. Let's move on then, shall we? Oh no, don't replay the video. How do I move on to the next one? Why do I not see any info on that? Oops, that is not it. Those are the courses available at Khan Academy. Again, not sponsored. Although I would totally be down to be sponsored. Hit me up, Khan Academy. Oncogenes. There we go. It's already lagging. I don't know if we're going to be able to play Death Stranding like this. Unless I close everything. Oh boy.
In essence, some sort of tumor-inducing agent, or it can also be totally spontaneous. Now, proto-oncogenes code for proteins that help to regulate cell growth and differentiation, which makes sense since the essence of a tumor, whether it is a cancer
I'm curious. Hold up, I'm gonna pause. Let me check my task manager and see how much memory and CPU usage I'm processing here. Well, OBS is always going to use up a shitload of my CPU, but that's okay. Okay. 52% of my memory is being used by Opera GX, which I'm currently using, as opposed to my usual daily driver of Firefox. OBS is using 50% of my GPU here, which is probably more concerning, considering that Stranding is going to be using a lot. Is there any way to reduce my GPU load on the OBS side of things? I could lower my bitrate or something. Assuming that the thing will allow me to. Although I don't think I can do that midstream. Shoot. I can't adjust it midstream. Alright. What is an acceptable bitrate? That affected nothing. Am I misunderstanding how this works? To be fair, I do need to stream more. Oh my god. I don't think this is gonna work, boys. This is looking kind of concerning here. The problem is my GPU is not that phenomenal. It's one of those built into your laptop little chips that are at least 2 generations beyond. Oh, 3. Just remember they came out with the 40 series. It's a 1050 Ti. Oof. I don't know if it can do it.
In fact, the words oncogene and oncology, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are 3 main mechanisms: deletion or point mutation, gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start the first one. Deletion or point mutation encoding sequence of the DNA in the gene itself or within a regulatory region such as a promoter region can lead to either a protein that is produced in the same normal amounts but is hyperactive for some reason, or maybe there is some sort of loss of regulation and the normal protein is just overexpressed. Gene amplification or an increase in mRNA stability that prolongs the existence of the mRNA and thus its activity in the cell can lead to a normal protein that is overexpressed. And then finally, chromosomal rearrangement involves translocation of a gene to a nearby regulatory sequence that then causes this normal protein, the gene product, to be overexpressed. Or you could possibly have fusion to an actively transcribed gene which...
Where is it? Do I not have oncogenes? Okay. Okay. Need to make a new branch here, but I'm not sure where. Sorry. Redefine it real quick.
Genes that code for proteins that normally drive... Oncogenes are genes that code for proteins that normally drive cell growth. They start out as proto-oncogenes, and then something happens to
cell
convert that protein
growth
into a full-blown oncogene. In essence, some sort of tumor-inducing agent, or it could also be...
Cell growth. DNA transcription, translation. Proteins. Enzymatic protein function. Confirmation. Man, this is what happens when I don't look at this map for like months. Oh man, I do not know where to put this. I mean, where's my section on genes? Gene silencing epigenetics. Wait, no, why would it wouldn't be included in epigenetics, I think. Okay, I guess it would just come off of DNA. Everything comes off of DNA, huh? Man, I just had to make it an absolute mess. Okay, let's see. Could do... Jeez, that's so annoying. There we go. I'll just push DNA damage and repair a little bit to the left. Maybe I could just move this. Maybe flip it around. I really wish that this program could just detect when I'm trying to flip my my group of, you know, shapes, and try to realize that I just want to move them and rotate them and choose the shortest path to between two objects, but I guess they just can't do it. All right, there we go. DNA, which is can be endogenous or exogenous. I feel like that can just be easily implied by the fact that endogenous is here, but that's whatever. I'm gonna keep it. Maintain continuity. All right. Please don't lag. There we go. Okay. Now we have a bit of room. Not much, though. Okay. I can... Oh my god. Okay, oncogenes. Proto oncogene. This is way too big. What the heck? Whoops. Okay. Leading into oncogene.
Totally spontaneous. Now, proto-oncogenes code for proteins that help to regulate cell growth and differentiation, which makes sense since the essence of a tumor, whether it is a cancerous or benign one, is unregulated cell growth. In fact, the words oncogene and oncology, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are three main mechanisms: deletion or point mutation, gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start with the first one. Deletion or point mutation encoding sequence of the DNA in the gene itself or within a regulatory region such as a promoter region can lead to either a protein that is produced in the same normal amounts but is hyperactive for some reason, or maybe there is some sort of loss of regulation and the normal protein is just overexpressed. Gene amplification, or an increase in mRNA stability that prolongs the existence of the mRNA and thus its activity in the cell, can lead to a normal protein that is overexpressed. And then finally, chromosomal rearrangement involves translocation of a gene to a nearby regulatory sequence that then causes this normal protein, the gene product, to be overexpressed. Or you could possibly have fusion to an actively transcribed gene which overexpresses the fusion protein or leads to a hyperactive fusion protein. And so really you can start to see a theme here emerging where the key idea is that you have either a normal protein that is just overexpressed, basically too much of the normal protein, or you can have normal expression but the protein itself is just a hyperactive one. Now, there are tons of examples of oncogenes, including the Stark oncogene, RAS, MYC, receptor tyrosine kinase, and also cytoplasmic tyrosine kinases. So let's talk about each one of those here. Stark was the first confirmed.
Have we not covered mutations yet? I feel like we have. No, okay. Well, in that case. Well, really? Not even deletion? That's a little weird, don't you think? Excuse you. I had to ask Miro. No, wow, okay. I see, I do not have that much information, even though my map already looks like this. Alright, in that case, we can just cover all the bases here. Deletion / point mutation. And basically, this is all to... These are all things that cause cancer, right? Overexpressed protein or fusion protein. Right? I'm not misunderstanding that part.
Gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start with the first one. Deletion or point mutation encoding sequence of the DNA in the gene itself or within a regulatory region, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are three main mechanisms: deletion or point mutation, gene
Oh, I'm dumb. Okay. So it's just a switch from proto to onco. Okay, alright. So then we're just gonna put these over here then. Deletion, point mutation. Hello. Opera. That's not what I was asking you to do. There we go. I want to search up on Google. Make sure that I can see a definition of the proto-oncogene so that I actually understand what's going on here. So what, they don't code for growth hormones or something. Not yet. Oh man, my computer's already lagging. This is bad. Gene involved in normal cell growth, mutations, changes in proto-oncogene. Okay, so proto-oncogenes are the normal cells. They're the chill ones. They're the ones that are supposed to be there. The ones that we need to survive. Oncogenes are the bad ones, the ones that we don't like, which are the cancer-causing ones. Okay. See, that makes more sense, because I remember understanding oncogenes being the cancer genes, but, proto. It's always confusing when science just decides to name things based on their potential to be something else, which is just fantastic. Alright. Gene amplification. Yep, makes sense. Yep. How do I represent increase? Oh yeah, ink. Increase stability. Point mutation, chromosomal rearrangement. Okay. Chromosomal rearrangement. I might need to just like close all my Opera windows in order to get Death Stranding to actually work. Shouldn't be too big of an ordeal, but it's gonna be annoying for sure. Luckily, this setup is pretty quick to actually get going once everything's in place. When things aren't in place, it's it is a problem. It's really annoying. Well, that matters when I have everything good. Everything good here? Oh god. Okay, this is there's a lot going on in this section right now. Okay. Here, here, here. If I can just move up the go. Oh my god. It auto routed to the other one. Don't do that. To point out that you're working together or separately or independently to create oncogenes. So go there and then you. Oh my god. There we go. Down here. Nice. Fantastic. Can afford to move down a little bit further. What a mess. Okay. All right.
It's the first confirmed oncogene, which was discovered in 1970 and was termed SARC for sarcoma,
Sarc.
which is a tumor
Sarcoma,
of
yay.
connective
As
tissue cells.
in,
It was actually an oncogene
not
discovered
yay.
in the chicken retrovirus. And so SARC codes for a non-receptor protein tyrosine kinase, so it phosphorylates specific tyrosine residues in other proteins. Another example of an oncogene is the RAS oncogene, which codes for a small GTPase, which hydrolyzes GTP into GDP and phosphate. This protein is activated by growth factor signaling and functions like a binary switch, sort of an on-off switch in growth signaling pathways. Examples of downstream effectors of RAS includes the protein MAPK, which is a type of kinase that in turn regulates genes that mediate cell proliferation. You can see examples of RAS oncogene mutations in thyroid tumors, certain leukemias, and cancers of the pancreas and colon. The MYC oncogene codes for a transcription factor that induces cell proliferation.
Whoa, okay. SRC, RAS, and then MYC. Okay. Mick. Not to be confused with Nick. Okay. Do you think I need to remember which oncogene causes which cancers? I don't know. Also, I don't know why I said yay for sarcoma. I meant to say that sarcastically, but that sounded way too genuine. What the hell? Nick?
And there's a very common translocation involving MYC between chromosomes 8, where MYC oncogene is found, and 14, which leads to a certain type of lymphoma called Burkitt's lymphoma. The RTK oncogene stands for receptor tyrosine kinase, which is responsible for adding phosphate groups to other proteins to turn them on or off. These are similar to the SRC gene, which is a non-receptor tyrosine kinase, the difference being the location of the protein, either at the cell surface as receptor or not as a receptor. So say you have a cell surface receptor that receives a signal from outside of the cell. Well, then that signal gets propagated or transmitted into the cell via these receptor tyrosine kinases that add phosphate groups to the target protein, specifically on tyrosine residues. And they can cause cancer by turning a receptor constitutively or permanently on in the absence of signals from outside the cell. Some well-known examples of receptor tyrosine kinases are vascular endothelial growth factor, or VEGF, epidermal growth factor, or EGFR, and platelet-derived growth factor,
Wait a minute. Getting a little confused because RTKs are just that's a receptor, that is not a gene. What? Okay. Is it just referring to the genes that affect RTKs? Are those not the genes below? What? No, these are growth factors. Transcript, please. Cell surface receptor. Okay, so it's an oncogene specific to affecting RTKs. That's the whole point. Yeah, okay. That is pretty straightforward. Okay. And then all of these other ones that they're mentioning are just growth factors, of which I'm not sure I need to remember. Kind of would like to include them, but I'm not really sure if that's going to be helpful. Vascular engines. These are just examples. Yeah, they're just examples. I'll just leave it alone for now. The important part is that RTK is here, yay.
Via these receptor tyrosine kinases that add phosphate groups to the target protein.
Me and the boys.
And they can cause cancer by turning
causing cancer
constitutively or permanently on in the absence of signals from outside the cell.
By destroying our individual receptors on these cells. Bruh.
And finally, we have
Could
the
you do
cytoplasmic
this?
tyrosine kinases, which mediate responses to the activation of receptors of cell proliferation, migration, differentiation, and survival. A well-known example is the BCR-ABL gene in chronic myelogenous leukemia, or CML, also known as the Philadelphia chromosome. It is a fusion of
So then CTK would be over here.
parts of DNA from chromosome 22 and chromosome 9. So basically, a part of chromosome 22, which contains the BCR gene, fuses with a fragment from chromosome 9, which contains the ABL gene. When these two chromosome fragments fuse, the genes also fuse, creating this new gene, the BCR-ABL gene. The fused gene codes for a protein that displays high protein tyrosine kinase activity, which is actually due to the ABL half of the protein. And the unregulated expression of this protein activates other proteins that are involved in cell cycle and cell division, which causes a cell to grow and divide uncontrollably, basically becoming cancerous. So as a result, the Philadelphia chromosome is associated with chronic myelogenous leukemia, a certain type of leukemia, as I mentioned before, as well as other forms of leukemias, which are cancers of white blood cells.
Okay. On to tumor suppressors.
Tumor suppressor genes are those genes whose protein products either have a halting effect on the regulation of the cell cycle, or they can also promote apoptosis, or sometimes both. So in other words, these proteins are like big stop signs that act as safety checks to help stop the mistakes in cell division that could lead to uncontrolled cell growth and cancer. Now, there are several sort of categories of tumor suppressor proteins. Those that recognize DNA damage and either repair it or initiate programmed cell death, apoptosis, if it can't be repaired, so DNA repair proteins. Then there are those proteins that act as repressors of genes that are essential for the continuation of the cell cycle. So if these genes are actively repressed and thus not expressed...
Okay, maybe I need a cancer-specific branch, although I feel like it's gonna be a big branch that I need to edit later. Makes it awkward that I branched off of DNA. Let's see here. I mean, the whole point of cancer is that it's affecting cell's ability to realize that it's been fucked up and needs to change, or it needs to die, basically. And the cell's just like, nah, bruh, I'm not supposed to die yet. I just keep going, but not do anything. And then that kind of messes with the whole system that we got in place. So, I guess tuner suppressors can be... I'd have to move DNA repair down. Just gonna move everything down. Yeah, I might need to move down everything. Okay, let's do that. Whoa, not you. Not Central Dogma. Come on, man. Central Dogma needs to be solid. It needs to be on its own. Okay. There we are. Sufficiently moved. Okay. And now we can learn about cancer all we want. Fantastic. I hope one day this YouTube channel will get big enough to where we can do all sorts of charity streams or different kinds of, oh, basically on everything that is important to better life on Earth. But specifically, cancer treatment, funding. Really need to figure out how to beat cancer in all of its many forms. And man, like, not quite the, it's kind of the final boss of medical research, I think. But man, it sucks. Cancer's terrible. Always cool to see events like GDQ promote funding for cancer research. All right. That aside, everyone needs to learn a little bit about how cancer works. So let's continue and see what tumor suppressors are going to be doing.
So you have cell cycle repressors. With tumor suppressors, there is this concept called the two-hit hypothesis, in which both alleles, and remember that alleles are basically the copies for a certain gene, and you have two copies for any given gene, one on the chromosome that you got from your mom and one on the chromosome that you got from your dad. Now, in the two-hit hypothesis, both alleles must be mutated before the effect is manifested, because if only one of the alleles for the gene is damaged, then you have this sort of backup second copy that can still produce the protective protein. So you need two hits, one hit for each of the alleles that you have. Another way you can think of this is that in mutated oncogenes, these alleles are typically dominant. So a mutation in only one of the alleles yields the cancerous phenotype. But with a mutated tumor suppressor allele, these mutations are recessive because both alleles must be mutated in order to lead to the cancerous phenotype. The two-hit
Makes
hypothesis was
sense.
proposed with cases of retinoblastoma, a rapidly developing cancer that originates from the immature cells of the retina, the light-detecting tissue of your eye. And I'll write this as PRB for retinoblastoma protein. Now, the retinoblastoma protein prevents the cell from replicating when its DNA is damaged, and it does this by preventing progression of the cell cycle from G1 into the S phase, or synthesis phase. So the retinoblastoma protein binds and inhibits transcription factors, which normally push the cell into the S phase. And this complex acts as a growth suppressor, and so the cell remains in the G1 phase. This complex also attracts a histone deacetylase protein to the chromatin, which reduces transcription of S phase promoting factors. And you can remember this by
I know this is just an example, but I may as well include it because it seems to be a illustrative example of the concept. So let's go ahead and type that in. Retinoblastoma.
recalling that histone deacetylase leads to chromatin condensation, or transcriptionally inactive chromatin. So this also further suppresses DNA synthesis. Another very well-known tumor suppressor protein is the p53 protein. Homozygous loss of this protein is found in up to 65% of colon cancers, 50% of lung cancers, and also in breast cancer. So this is clearly a very critical tumor suppressor protein. And so p53 activates DNA repair proteins when DNA
Yep,
has
I've definitely seen
and
that
also
around.
arrest growth by holding the cell cycle hostage, if you will, at the G1 to S regulation point. And this gives DNA repair proteins some time to fix the damage and allow for continuation of the cell cycle. So specifically, p53 binds DNA and activates several genes, including ones that code for a protein called p21, which binds the cyclin CDK or cyclin-dependent kinase complex, which is actually the complex responsible for pushing the cell from the G1 to S phase in the cell cycle. p53 also functions in the initiation of apoptosis if the damage to DNA is irreparable. One significant exception to the two-hit rule for tumor suppressor genes is with certain mutations of the p53 gene product, which can then result in what is called a dominant negative, meaning that a mutated p53 protein can prevent the protein product of the normal allele from functioning. So don't forget to sort of keep that in the back of your mind when you're thinking about tumor suppressor genes.
Sure. Make sure to keep that in the back of your mind. There's a lot of information here. Oncogenes? Oh my god, we finished lesson 5 on gene control. On to lesson 6. Oh, there we are. Genetic mutations. I was wondering where they were. Oh man, it's gonna be a long boy. Ooh. I like having practice questions at the start, but I'd rather just get to the content. Today we're just working on the map. Worrying too much about practice problems
Today I
at
give
the
you a quick
moment.
introduction to genetic mutations.
I'm going to leave that
First, let's go over
to
the central dogma of
you
molecular
world
biology.
to figure
Which is
that
the idea
out.
that genetic information in a cell is stored in the form of DNA, and this DNA is used to generate complementary RNA through a process called transcription. That RNA is then used to synthesize a corresponding protein through the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U, or T, and C pairs with G. Next, our RNA will be used to generate protein through translation. And remember that during this process, RNA nucleotides are read in groups of 3, called codons, in order to generate corresponding amino acids. Now, just very generally, we say that mutations have the effect of making this synthesized protein not turn out quite right. So I'm going to give a quick shout out to sickle cell disease, which is an example of a disease that's caused by a
Shout
So you
out!
may remember that there is a protein in red blood cells
Shout out to
which is also
sickle
called
cell
hemoglobin. Hemoglobin
disease.
is a protein that coordinates to iron ions in order to hold
Oh,
oxygen.
fantastic. Yep, very famous disease. Oh shoot, where are we putting these things? It's just a general section on genetic mutations. I mean, it might have been included in proto-oncogenes, but like, I don't think that's really necessary to be combined. So let's move here. Oh my god, everything is connected to DNA. It all stems from DNA, which it does. I mean, duh. It's DNA. It's kind of too OP, you know? Does everything, truly. Okay. Kind of the basis of all life, you know? So, makes sense. Probably should have tried to save space somehow, but no. So this is fine. Gonna work out, I swear. Probably. Oh no, it's not. Oh, this is bad. This is really bad. Okay. Methylation. Inscription. The lac operon. I like how we're only doing mutations now. I feel like that's we could have just. Oh, this would have been easier if we just did everything related to DNA in one section and then continue. That's a bit much to ask for, I guess. Because it's kind of important to see everything from a larger, more impactful angle with these bigger concepts that we have down at the protein aisle. And then going a little bit more microscopic. Whatever.
Molecules and transport them throughout the body. Now, the mutation that causes sickle cell disease results in a mutated form of hemoglobin called HbS being formed, where the S is for the word sickle. And the difference between normal hemoglobin and HbS is that one glutamate amino acid residue is being replaced with a valine amino acid residue. And this small change results in all of these mutated HbS proteins aggregating together in a red blood cell, which makes it very difficult for that red blood cell to transport oxygen effectively. Now, just a side point, remember that red blood cells are initially generated from hematopoietic stem cells through a process called hematopoiesis. So where are mutations found, and how do they come up in the first place? Well, let's look at a couple different possible mistakes that could lead to an incorrectly produced protein. So first, we'll see what happens if a cell makes a mistake during translation, and we'll stick with our example of sickle cell disease from before. So let's say that we have a sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is
We're going to translation.
RNA
All right.
GAG. Now that GAG would normally correspond to a glutamate residue during translation, but a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with sickle cell disease. But notice that if a mutation happens during translation, the cell will only produce one mutated hemoglobin, or HbS, for each overall mistake. And since cells are making tons and tons of hemoglobin, just one mutated protein might not have that big of an effect on the cell. So we can say that mistakes during translation probably don't cause mutations like the one associated with sickle cell disease. So next, we'll look at mistakes during transcription. Again, we have our CTC piece of DNA, which would normally make GAG on RNA, but maybe a mistake occurs which leads to the transcription of a GUG instead, which would then code for the valine associated with mutated hemoglobin. Now, if this mistake occurred, the cell would only make a few mutated hemoglobins for each mistake, since the strand of messenger RNA will only be translated a couple of times before being degraded.
So it has to be a DNA level mutation for like significant cell-wide problems.
Then our corresponding RNA from transcription will be changed, and ultimately a valine will be produced instead of a glutamic acid.
Yep.
Now, since the cell's DNA stores all of its genetic information, that mistake would lead to all future hemoglobins produced from that gene being mutated. So overall, we can say that mutations will usually result from mistakes in a cell's DNA, and not from the RNA or the protein. So where do these types of mutations come from? Well, there are 2 ways a person can get a genetic mutation. The first is that they inherit it from their parents. Remember that DNA is passed down from parents to offspring, so if we have a mutated father here, then there's a good chance.
We're gonna make a mutation branch. It's gonna go right here, and it's gonna be awesome. Or well, mutations are awesome. Most of the time they, well, a lot of the time they end up just screwing with you and really messing you up. But the whole point is that eventually some mutations occur so that the species can have more genetic diversity, and genetic diversity allows for evolution because the best fit members of the species will be more likely to, you know, reproduce and send along those mutated genes, which will eventually become the norm in wherever that region of the world is. And species in that region. Anyway. Mutations can be inherited. True. That's important.
So one of his kids will inherit that mutated gene the same way that the child might inherit any amount of that parent's DNA. The other possibility is that the mutation will come on spontaneously, which is where a person suddenly gets a mutation in their DNA without their parents having had the same mutation. And spontaneous mutations can come from many different sources, with just a few examples being from DNA replication errors, environmental factors like certain poisons, or it's also possible that
the sun is a deadly laser.
Well, first we learn that mutations originate at the DNA level, and not at the RNA or protein level. But the effects of a mutation, like the example we gave of sickle cell disease, are found with problems with the proteins that are ultimately expressed by the mutated DNA.
Just hit that thing with UV light, guys. Come on.
Are usually found at the protein level. And finally, we learned that mutations are either inherited from parents or entirely spontaneously.
Okay, cool. How come it's not saving my history on these videos? Come on, Khan Academy. What's going on here? I'm a little disappointed.
So today we're going to talk about the different types of genetic mutations that you find in a cell. But first, I want to review the central dogma of molecular
Luckily,
biology.
streaming this is going to allow me to be able to check back and see where I left off, so that's all good.
Now, if you mutate one of the nucleotides on DNA, like let's say turning this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what are the different types of mutations? Well, the first type of mutations we're going to talk about are called point mutations. Now here I've just written out a random sequence of DNA, which is just a repeating pattern of CTC, which would code for a repeating sequence of GAG in the RNA strand, and finally a protein sequence of 3 glutamate amino acids. So a point mutation is when one of our DNA bases is replaced with another. So in this example, a thymine base is being replaced with an adenine base, which leads to a change in one RNA nucleotide and ultimately a change in one amino acid. Another type of mutation is called frame shift, which works a little differently. So first, I'll write out the same DNA, RNA, and protein sequences from before, but now instead of changing one base to another, I'm going to add one to the sequence. And here I've thrown in this extra cytosine base that I've written in blue. Now naturally, this change would lead to an additional guanine base being in the result
Or
of
a deletion.
It
doesn't have to be a
the reading frame
addition.
of the RNA. Remember that RNA is read in groups of 3, or codons, when being translated to form protein. But now, since we've added an extra G here, all of the codons coming after that extra G will look a little differently. Now, instead of having 3 GAG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed, and in this example, they'll be changed from glutamate to glycine. So you can see that frame shift mutations usually have more significant effects on the final protein than point mutations do. Now, it's important to recognize that both of these mutations are classified and named for how they affect the cell's DNA structure, and aren't really named for how they affect the resulting protein. Now, our next type of mutations are nonsense mutations and missense mutations. Let's say we have a DNA sequence that normally generates RNA and codes for a cysteine amino acid. A nonsense mutation is
How do I want to play this? Do I just list out each mutation? It's weird that frame shift is on its own and they don't have deletion under it. But okay. I don't even have to include the word mutation on each of these. That's nice and convenient. Okay. Maybe I should have spontaneous inherited on the same side of mutations. Yeah, okay, we'll do that. Then I'll have some space below mutations to include more stuff. Oh my god, guys. Come on. Work with me here, Miro. Work with me. Point mutations. And then frame shift. Leads. Oh my god. Nonsense and then missense. Which both sound like nonsense. Thank you, biology. Fantastic naming scheme here.
Now, missense mutations are a little different, and they're any genetic mutation that changes an amino acid from one to another. So in this example, our mutation is changing the resulting amino acid from a cysteine to a tryptophan. Now you can see that nonsense mutations probably affect the resulting protein a lot more than missense mutations do, since that new stop codon that we're creating could chop off a huge section of the protein instead of just changing one amino acid to another. So now we can divide the missense mutations even further into a bunch of smaller categories. Silent mutations are when the mutation doesn't actually affect the protein at all. Since many different RNA codons can code the same amino acid, it's possible that the mutation might not affect the protein at all. So in this example, CCA, CCG, CCT, and CCC in a section of DNA will all end up coding for glycine. So if you change the third base,
why are there multiple codes for
new amino acid is of the same type as the original. So here I have a
for amino acids? What's up with that? Like, is it to keep silent mutations possible in the case of a mutation event that has less likelihood of wreaking havoc on the cell, or what? What's going on here? Conservative. Or is there some other function? Or maybe there's no function at all.
And aspartate, which are both acidic amino acids. So a mutation that swapped out an aspartate for a glutamate would be a conservative mutation. Finally, a non-conservative mutation is one where the new amino acid is of a different type from the original. So here we have a serine amino acid, which is a small polar amino acid, being replaced with phenylalanine, which is a large non-polar aromatic amino acid. And this would be an example of a non-conservative mutation, since serine and phenylalanine are different types of amino acids. Now I'll point out again that all of these mutations are classified and named for how they affect the resulting proteins, and aren't really named for how they affect the cell's DNA. So let's look at a quick example. Sickle cell disease is a disorder where hemoglobin, or HB, which is a protein found in human blood, is mutated into a less active form, which we're going to call HBS. And it results from a single glutamate residue being converted into a valine residue. Now we can classify this mutation as a point mutation, since only one DNA base is affected, but we can also say that it's a non-conservative missense mutation, since glutamate is being swapped out for valine, and the two are different types of amino acids, since glutamate is an acidic amino acid, and valine is a non-polar one. So what did we learn? Well, first we learned that mutations originate at the DNA level, but show their effects on the protein level. And second, we learned that we can classify different types of mutations by either their effects on DNA or their effects on protein. In reference to DNA, we have point and frame shift mutations, and in reference to protein, we have missense and nonsense mutations.
I'm realizing it must be hard to see on... Actually, I'm streaming at 1080p, and then OBS is telling me that this is in like 420p or something. 480, I mean. Just because it's scrunched up into that one little window that I can't really expand that easily because of my setup, which is a little annoying, but alright. So I'm assuming that you guys can see everything that I'm working on, even though it might not look fantastic. Also, I've noticed that our music stopped for a little bit. Yes, continue watching. Watching.
So
Where
today
is
we're
it?
going
I
to talk
don't
about
even
causes
hear
of
it.
genetic mutations. But first, let's just do a quick
What
review
track
of
is
the idea that
this?
mutations are mistakes in a cell's DNA. And there are two main types of
Tribal
mutations that we see in nucleic acid
tableland.
And the first is called point mutations, and that's when one DNA base is switched out for another, which usually results in a change to one codon in the RNA sequence. Frameshift mutations are when the reading frame
Yeah, I
of the
think I
RNA is
do
altered,
hear it.
and while the actual nucleotides in the RNA sequence haven't changed that much,
There we
the
go.
reading frame of the RNA strand has shifted,
Yep,
meaning
it's
that many
definitely
different
there.
RNA codons will change as a result. And we're going to take a look into what causes these point and frameshift mutations. So point mutations are caused by base substitution, which is when one DNA base is substituted for another. And there are a couple of different types of base substitution. A transition is when you have a substitution of adenine for guanine or vice versa, which is a swap between purines, or a
Okay.
substitution of cytosine for thymine, or also vice versa, which is a swap between two
I'm gonna
purines.
expand on each of these mutation types. Okay.
And in this type of base substitution, you have either a purine being replaced with a pyrimidine, or a pyrimidine being replaced with a purine. Now, the last kind of mutation that can lead to a point mutation is a mispairing, which some people call mismatching, and that's when a DNA strand has a non-Watson-Crick base pairing. Normally, A pairs with T and G pairs with C, but when you have a mispairing, that's when A and C pair up, or when G and T pair up. And it's much more common for mispairings to occur between a purine and a pyrimidine, as opposed to between two purines like A and G pairing up, or two pyrimidines like C and T pairing up. Next, we're going to talk about frame shift mutations.
Whoa, hold up. That's where they're gonna mention deletion and thinking addition, but it's not addition, is it? Additive? No, not point. Point is what we're talking about right now. What in the world is it? The one where you add a nucleotide. Well, whatever. They're about to get to it, so I'm not going to worry about my ability to remember information from a year ago. Work with the information I got now. Transition, transversion, and then mispairing. I keep looking at the clock. I shouldn't be doing that. I already have an alarm to tell me when I get to play Death Stranding, but I don't know, I'm kind of excited. Well, I'm also mostly nervous on whether or not it'll let me play the game. Didn't really get to test that thoroughly, which I could have, considering that I already know what happens at the start of the game. Don't know what happens after, though. But whatever, we'll work with what we got. Just hope that I get to play Death Stranding and not have to switch to Signalis, which I do also really want to play, but really wanted to spend this MCAT prep time with Norman Reedus, the man, the myth, the legend.
This would then be transcribed into an RNA strand with repeating GAG units and an extra G on the end, and our three codons would be the three GAG units, which would then each translate to a glutamate amino acid. Now, one way you can cause a frame shift mutation is through an insertion, and that's when an extra DNA base finds its way into our sequence. So here we have this extra
insertion, of course.
And this additional C base would lead to an G being thrown into
insertion and deletion, yeah, that makes more sense.
RNA strands during translation. So now instead of three
They just don't, they're not like named opposite of one another, but they are opposites.
The other way
Very
to cause
cool.
a frame shift mutation is through a base deletion. So in a deletion, we drop off one of our bases from our original sequence. So here I've dropped that first thymine base, and this would also result in a shift of the RNA reading frame. Now, instead of having three GAG codons, we have a GGG codon and two AGG codons, which would lead to a protein with a glycine and two arginine amino acids. So overall, insertions and deletions can both lead to frame shift mutation. Now, we can also talk about large-scale mutations, which instead of being at the level of individual nucleotides, are usually seen at the chromosomal level and can affect many genes instead of just a few base pairs. So first, we'll talk about translocation, which is when a gene from one chromosome is swapped for another gene on a different chromosome. Now, it's important to see that translocation refers to gene swapping between non-homologous chromosomes, which means that if this blue chromosome were chromosome 10, then the green one could be any chromosome assigned to chromosome 10.
Okay, large-scale mutations. What, like crossing over? Crossover mutations? I didn't even know that those were classified as mutations. I just assumed that it was just a standard thing that happens. Crossing over. Okay, large scale mutations then. to be there.
The process of crossing over that occurs during meiosis between homologous chromosomes. The next large-scale mutation we'll talk about is chromosomal inversion, and that's when two genes on the same chromosome switch places. So here, our green and blue genes are being swapped and end up on different parts of the chromosome after the mutation. Now, since both of these mutations don't always affect the individual nucleotides coding for a gene, it's important to see that many of these types of mutations affect how a gene's expression is regulated, in addition to changing what the genes actually code for. Remember that the position of a gene on a chromosome partly determines how it's regulated, and this could be due to histone configuration, promoter regions, or any other regulatory process. So what
Yeah, that
do?
makes sense.
Well,
Okay, whoa. Why did it suddenly go full screen on me? Hello.
Mutations, which can be caused by transitions, transversions, and mispairings. And we also have frame shift mutations, which can be caused by insertions or deletions.
Translocation and inversion. Okay. And inversion. Interesting. That makes sense, though. You suddenly move the gene to like a completely different spot. It's just like, where regulation? Where are my regulatory segments? Where are they? Right, mutagens and carcinogens. Back to talking about cancer. Ooh, wait, maybe we can connect mutations over to cancer. Ooh.
But first, I want to review the central dogma of molecular biology and how the genetic information of a cell is stored in the form of DNA.
Oh my god.
Which is then transcribed to RNA and then translated to
He's reviewing again.
Nucleotides from DNA are transcribed to their complementary forms.
He's at it again. Oh lord. The central dogma is back.
Then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what is a mutagen? Well, a mutagen is any chemical substance or physical event that can cause genetic mutations. Chemical substances like certain poisons could be mutagens, or physical events like UV light or different kinds of radiation could also be mutagenic. And we classify mutagens into two different categories. So let's say we have a person over here. A mutagen could be classified as endogenous if it comes from inside this person's body, and it's some mutagen that's already found in the organism. But an exogenous mutation is one that comes from outside the affected organism, something that's from the external environment. So what are some examples of...
This looks more like I'm gonna add a special branch called mutagens, and then carcinogens are gonna be right off that mutagen branch, leading right to cancer. Alright, sure, why not? It's just not as satisfying, though. It's not the connection I want. This is like a completely outside of the system thing. Okay. Mutagens leading to carcinogens. Which lead to cancer. All right.
Some endogenous mutagens. Well, the most significant endogenous mutagens are what we call reactive oxygen species, or ROS. And ROS are naturally occurring metabolites in the human body that are produced by mitochondria during oxidative phosphorylation. So if we have this guy here who's about to chow down on a big meal, you can expect that during the metabolism of the meal, his mitochondria will produce a bunch of ROS, like O2 dot minus, which we call superoxide, which is an oxygen molecule with one extra electron, as well as some hydrogen peroxide, which is another ROS that your body can produce. Now, reactive oxygen species, as you may be able to tell by their name, contain oxygen, like both of these examples do, but they're also highly reactive with different cell components, including DNA. And by reacting with DNA, they can actually cause significant damage to a cell's genetic code. One example of this type of damage is the double-strand break, and ROS can actually break a DNA's double helix into two smaller pieces. And you can see why this type of reaction could cause a mutation, since it quite significantly changes the structure of the cell's DNA. The next type of DNA damage that ROS can cause is base modification, and that's when the nucleic acid bases are changed or swapped around, and that can pretty readily cause point mutations, or maybe even other kinds. Now, you may be wondering, why would a cell ever make something that could damage itself? Well, it turns out that ROS actually have a couple of beneficial effects on a cell, and cells actually have a couple of ways to make sure that they don't cause damage. But sometimes ROS levels get really high, and cells can't deal with them anymore. And we call this oxidative stress. And antioxidants are something that your doctor might have told you they're good for you, and it turns out that part of what antioxidants do is help make sure that ROS don't damage your DNA. Now let's look at a couple examples of exogenous mutagens.
There you go.
There are many different types of...
Time to buy all of the antioxidants off the shelves because we don't want to cause all these crazy mutations. That makes sense. Although I'm assuming that we don't need as many antioxidants as companies would like us to believe, but hey, it is what it is. I'm sure that in certain situations you need plenty. Okay. We are running out of space. We are trying our best to maintain some semblance of order. Oh my lord. Look at this. Am I really about to try and move all of this at once? Okay. I guess this can be like here and then. Ooh, this is rough. This is kind of too close to call. Oh my goodness, it really just did that, didn't it? Oh my god. Oh wait, I can just move the two-hit hypothesis. There we go. Alright, we're good. I have a bit more space to work with my mutagens. Okay. Apparently not as much space to click on the right things, though. Strand species. Double strand breaks and base modification. And this is going to be the last set of notes before I switch over to the other thing. Double strand break. Okay. Alright, it's 9:30. I'm going to have to put this in really quickly. Then I get 30 minutes to attempt to understand Kojima's video game. And then I get to go back to studying. Modification. I'm assuming that there's going to be a save spot within 30 minutes of the game starting. That might not be true. Antioxidants.
Now let's look at a couple examples of exogenous mutagens, and there are many different types of exogenous mutagens, but we're really only going to talk about two. Now, intercalators are one example, and one of them is called ethidium bromide, which you may be familiar with if you've ever done a PCR experiment before. And what ethidium bromide will do is
I have done a
jump into
PCR
DNA molecule and
experiment.
stick itself between the two strands. And when
Wow.
these intercalators intercalate into the DNA, they can deform the structure of the DNA and cause some serious problems. Base analogs like 5-bromouracil, which we also call 5-BU, pretends to be a certain base, but then acts differently than that base normally would. So in the case of 5-BU, it's an analog of uracil and looks a lot like it, but once it's incorporated into DNA, it can shift between two different forms. In its keto form, it will pair best with adenine, while it's in enol form, it will pair best with guanine. Now, if you're familiar with organic chemistry, you might know that 5-BU can convert between its keto and enol form through something called a tautomerization reaction. And overall, you can see how this base analog might be able to induce mutations in a DNA strand. Now, the last thing we're going to talk about is what a carcinogen is. Now, carcinogens can be mutagens, but not all of them are. But in general, you can say that a carcinogen is something that can lead to cancer, which, if you remember, is when cells in an organism divide uncontrollably and can form big masses of cells called tumors. Now, some carcinogens will work by making mutations in DNA that lead to cancer, but sometimes they might carry out their effects simply by increasing the rate at which a bunch of cells divide without actually affecting their DNA. And some examples of carcinogens are tobacco, which come from cigarettes, asbestos, which used to be used as home insulation, and even UV radiation. So, what did we learn? Well, first we learned that mutagens are chemical or physical substances.
Yeah, no kidding. The train station over at my place spent the entire summer getting rid of asbestos. And I had to use the train that entire summer because of the job that I had over the summer. Oh, fantastic. Love being near carcinogens. It's great. Exogenous. Intercalators and base analogs. That makes sense. logs. Oh my goodness, this map is a mess. Okay. Okay. We're good. All right, time to switch. Oh man, this setup. I have to go ahead and delete this entire thing. But first I'm gonna use the bathroom. I'll be right back. I'm not even 2 hours in, I'm already yawning. Alright, let's get rid of all of this stuff and pull up Hide Your Kids Kojima game, video game. Oh boy, this is gonna be a pain to switch between, but I get 30 minutes regardless. And hopefully OBS won't crash. If it does, then we're gonna have some problems. I guess I'll include loading times, which is not ideal considering that it is on my hard drive as opposed to my SSD. Okay, now it's blotting out the sky. Cool. I love it when it does that. Did I not adjust the size of the screen? Why? Why is it so huge? What is going on? What is happening? Where am I? Who am I? Why is the dog gay? Kojima Productions. Okay, I can afford to have this underneath me. There we go. It's a little awkward that you can just see everything. You can see where my camera ends. So this setup's not fantastic. But it's good enough. Where is it? Maybe I'll just have it on top of me. Oof. Kojima's about to top me. Ooh. Okay. Here we go. Here is a video game that hopefully won't crash my computer. Right. I've heard that the game is not that difficult, but it can be changed at any time, so we're just gonna go normal. Yeah, sure. Let's go. Maxor, why are you showing me a normal-sized crap on my screen? Oh no, I included loading times. Oh no, look at this! This is terrible! Oh no, I'm only gonna be able to play it for like 15 minutes at a time! Oh, that's kind of annoying. Oh wow, this is bad. This is like really bad. I know it takes a while for my computer to load things, but this is absurd. It's not even at 1%. Maybe I should have switched to Signalis regardless. Oh wait, you can't even see me. I just realized. Oh, I have to alt tab. There we go. Hello. Hi. Video game loading. Look at this thing. It's not moving. What? Oh no. Am I about to spend 30 minutes waiting for it to load? Is this how my time's gonna be spent? Oh my god. Alright, if we spend 30 minutes loading, then we're switching to Signalis next time during the break. It is what it is. I just thought that this would be more thematically appropriate, you know? Walking through America trying to make connections, you know, learning about medicine, helping people, stuff like that. Well, this Stranding is really cool, though. It seems like Kojima read a psych textbook before making it, which is always fantastic. I mean, Evangelion is the thing, so love it when creative people do that. Or neurobio. Neuroscience textbook, maybe. The game is concerned about oxytocin levels and human connection and relation. I categorization of these concepts is messy. I would associate it with psychology, but I mean it's the same, basically. Okay, wait. I might be able to play the video game. It's only been 5 minutes. We're getting there. It's gonna get there eventually, I'm sure. So, how's your weekend been going? I hope it's been going quite well. And if you're watching this in the future, then I'm surprised. That's amazing. Also because I have a feeling that this is going to get taken down, but it hasn't been taken down yet, so maybe YouTube's smiling down upon me. As opposed to when I was streaming Eminence and Shadow and it got immediately blocked in all countries. Which is hilarious, by the way. Even their sponsored streams got blocked. But I did not get sponsored by them, so oh well. It was a little embarrassing anyway, because my mom called me in the middle of playing the best gotcha game created. Embarrassing. Called me twice on 2 different phones. Luckily, I didn't end up doxing my family through there because changed their names and such as you do with your contacts. But man, whack. I gotta make sure to communicate about when I'm streaming and when I'm not. Specifically when I'm streaming on my phone. Oh my god. Oh, ooh, it's moving. The line is moving now. The line is moving. And we don't even need a fast pass system for it. I feel like Defunct Land is like the best type of advertisement that Disneyland could ever ask for. Aside from Disney movies. Alright, fine. It's just like it's about the parks though, specifically. Which makes it more hype to go there. Because now you're like, ooh, I'm invested in the park itself as opposed to the IPs. So if you're not enjoying the park because you're respecting the IPs, then you might leave. But in my case, I want to be there because of the park. And to see what kind of messed up shit happens in it. Oh. Bob Iger back as Prez, I think. If I remember correctly. That's wild. That's so wild. Man was such a meme. And he left. And then he came back. Oh no. Disney. Oh, Disney, Disney. Do not mess around like this. Wow, we're going on 10 minutes. 40% of the way there. Will I only be able to play 10 minutes of the game? Okay, I might have to change the schedule maybe. So I might do... It's kind of annoying. Oh, I don't want the ratio to be higher. I want the 1/4 ratio or less. So I guess that means 6 hours of studying with 2 hours of gameplay. Or I could do 3 hours of studying, then 1 hour of gameplay, which is probably more doable. It's probably more accessible. I mean, 3 hours. 3 hours of studying, though. Ugh, a little annoying, but we'll make it work. I could do 2 hours. No, no. I think 3 hours and the 1 hour might be the way to go. I'm gonna go ahead and stretch though. You can go ahead and look at 2B's leg. Covered up by the green screen. Which looks great, by the way. This is my new ring light, which makes my room not look like the inside of a dungeon. A Dark Souls dungeon. And instead I have like the power of the sun right there. Oh, in the form of a wheel. The wheel of power. Oh man, this is not gonna work. Oh, wait. 8%. We got 8% in the past 2 minutes that we've been loading this game. Wow. Amazing. I don't know. I mean, I kind of like the setup though. It's kind of slick though. I don't know. I want to switch to a different game. Oh my god. Okay. 10 minutes load time. That's what we're working with, baby. So I get to play for 20 minutes. Oh, it's better than nothing. Still loading. Okay. Image on the left. Wait. On the left cannot. I have to look at OBS to make sure that it's doing this properly. Okay. I'll work with this. Okay. Statistical analysis indicated dooms for the date of birth is hard to establish your own ability levels. Please enter your birthday. Great. I don't know why I've been trying to hide my birthday for such a long time by like giving different places the wrong birthday. It's just like if a verification thing entirely relies on birthday information, that's just dumb because that's public information, basically. I don't want to hide my birthday because it's a banger of a birthday. Higher levels Doom's ability is the constellations Cancer. I'm when am I? I forgot that what I am. Oh well. Oh well. Guess I don't have dooms. I can see that the cutscenes are gonna look great. Hmm. Rope and the stick. Yeah, cool. Don't really get it just yet.
Once there was an explosion.
Oh baby, he's talking about the explosions. Explosion. It's all because of Megumin's spin-off coming out.
Which set a planet spinning in that space.
Mm-hmm.
Once there was an explosion. A bang which gave rise to life as we know it. And then came the next explosion.
Oh shoot. Need to make sure that I mute this. Game audio gonna be muted a bit. Really nice music though. I have to watch out if I even want this stream to stay up though. Just a little bit. Deo Kojingles. Hell yeah. I'm so worried about the cutscenes. Luckily, I've seen all the cutscenes at the start of this show. Or show. This game. It's gonna suck for you guys if it doesn't look. Ugh. Whatever. The rest of my videography is basically unsynced. Oh man, this is a loud. This is a loud game. Game is loud as heck. Whoa. Controller vibrating. Whoa. Oh yeah, the audio is already misaligned. This is pretty bad. Look at this, guys. Oh no. This is so sad. Why is it all... why does this always happen? Audio gets desynced so terribly. Nah, we gotta switch to Signalis. This isn't gonna work. I'll just have to do this on my own. Wait. Did it just resync itself? I'm so confused, but I guess it worked out. Okay, so all I have to do is just press the options button and I'll resync, I guess. Nice. Alright. We're in this. We can play the game. Hello. I've seen this cutscene so many times, and it still totally looks like you hit her. But I get... Alright. Before I spoil more of the game, like... Oh my god. The attention to detail in this game is insane, and I appreciate it. Oh. So sad. Same. You know what? Maybe I can, I'll just change my setup here. Make it, like, make me smaller and in the corner. And then the game gets to be front and center. Even though the game's going to be running at 720p, I mean, it is what it is, right? But, okay. Oh yeah, it's inverted. Okay. Not inverted. It's actually normal. The problem being that I play with inverted y-axes, so I need some help here. Where is it? Camera controls need to be inverted. Yeah. Okay. Transfer cargo. I can do that. Have to scan. Ah, gotcha. Okay. Is there motion blur right now? Or is that just my computer lagging? Because if it's motion blur, then I'm gonna be sick. Oh god. Actually, if it's more concerning if it isn't motion blur, because... Uh-oh, okay, that is going to be a thing that's gonna bother me. That's fine. It's my computer trying to load this the environment. Oh, that's so sad. Right, where are we headed? We're gonna go there. So what, do I need to climb up there, or where am I headed? It's a cliff right there, Sam. That's a cliff, dude. Oh, wait. Okay, we're good. Like a little crevice here. Just slip through. climb continuously. Okay. But I failed to see a reason why I would do that right now. Sounds good to me. Norman Reedus. There he is. There's our boy. In all of his glory. Time acceleration is such a cool thing. Such a great concept. Trying to avoid the rain. They'll just make you old. Oh boy. We have all of the vibrator settings on. Ah, my controller. It's going places. Bruh. Is there a BT outside my room? Because it's cold as hell. Man, I remember the first few reveals of this game. Such amazing trailers. I'm so excited. Because Death Stranding 2 got announced a couple days ago. Oh, shoot. It sees right through my hair. Hey, why does it do that? I don't know. This game is totally gonna make me do the thing where you hold your breath along with the characters. Oh no. What's up, ghosts? It's your boy. And then a third one shows up. Oh my god. My hair. My hair is chroma keyed. Dialed in. Fuzzy hair. Should just like wear a hat. I don't have it. I mean, I have my Megumin hat, but that's gonna be way more distracting than hair being like chroma keyed. I'm 3 minutes away from where I'm supposed to stop.
What the hell?
This isn't gonna work,
Stab
boys.
you so hard.
This is not gonna work.
Tears. A chiral allergy.
I'm gonna need another 30
So
minutes.
you have dooms like me.
I've got the
Just the entire cutscene's just been 30 minutes.
What's your level? You can see them, right?
No, but I can sense them.
Level 2 then.
What are you doing here?
Trying to stay dry. Same as you. Time falls let up. My name is Fragile.
Yeah, I've heard of you.
Honestly, even at the lowest settings, at 720p, this game looks amazing. Like, look, the anti-aliasing is non-existent, but like, it's a damn cool looking game. Regardless, I just hope that I'm able to play it properly. Maybe I can turn it up the settings just like one notch.
You want it?
Surely that's not gonna wreck my computer, right?
A cryptobite a day
Computer.
makes the time fall away.
Cryptobite. Nom nom nom. I should have like a snack to eat whenever she does this, but I don't have that many snacks, so
Wanna come
I'd
work
rather
with me?
not.
Must be tough out here on your own.
Well, she's gonna leave us for a second there.
Yeah.
You wanna show up for a bit?
Express had plenty of people.
Plenty of traitors. Not
Raiders.
much left of us now, save for a few honest folks. And on top of that, not much left of me either. Got soaked from neck to toe.
I can't help you with that. I make deliveries. That's all.
So the timefall, like, can you, like, is the time acceleration property just from falling onto your body, like actively moving water, or is it, or can it be like still water? Because earlier we saw the photo like float
Those things never stay
on some
calm.
water. Seemed totally fine. A little confused, but you know, it is what it is. Damn, Norman. Ah, looking hot. Goddamn.
The time fall fast
Foreshadowing
forwards
moment.
whatever it touches. But it can't wash everything away. The past just won't let go. I'll see you around. Sam Porter Bridges.
Okay, so clearly this is not going to work. I'm going to have to add another 30 minutes, and then we're going to have an extended length studying session. Oh god, 4 hours and a half. Is it worth it, Sam? Is it worth it? It better be. Blue bars, stamina gouge. Alright, cool. I mean, I think part of it is like, can I play this at 1080? Like, if I can, then it'll probably look amazing. I'm gonna try it. I'm going for it, folks. I'm going for it. It looks marginally better. No, it does look actually a lot better, but like, I don't need to play it at 1080. Oh shoot. Oh. Gotta watch out with music. Guess I probably want to climb down this way. Can I sprint? I actually don't know the controls. Compass mode, scan terrain, hold breath. Change posture. Okay, so I have to press. I don't think I want to sprint this much. Yeah, L2, R2 is the triggers. Small drugs. I wonder what drugs, though. Aw. Rip. Dang, his stamina drops faster than I expected. Woo. This is incredibly fast. Okay. Wait, I thought holding things in your hands was a hold action, not a toggle. Right now it seems like a toggle. So I'm headed over to Central Knot. Right? Wait. That doesn't look like the city, though. Wait, I'm a little confused. Maybe this is this signifying a thing that needs to go to Central Knot? Yeah, that's okay, that's how this works. I'm gonna eat one of these bugs. Okay, that's how you heal. Cool. Oh wait, I can just press triangle. That works too. I feel bad that I can't let you guys listen to the music. I mean, it's also loud as hell on my end. Oh god, it's loud. Alright, there's a lot of input lag on my end. Oh wait, can I work with the audio in this settings? Because that might help a lot with what I'm going through right now. Because it's loud as heck. There we go. That's probably what I should be playing at. Oh, it's still kind of loud. It's fine. Sam? Oh, we have a jump button. Okay. Can I just... Do I take fall damage? I definitely take fall damage. Kojima definitely implemented every single point of realism. Oh god. Okay. I lost a bit of health, but it's not as much as I was expecting. I mean, it was the tiniest ledge ever, but still. You know? Can I just climb this rock? How do I climb? Oh god. I just... feel like I'm pressing the wrong button for jumping and climbing. Like I'm jumping when I'm trying to climb and climbing when I'm trying to jump. Whatever. We made it. I really want to try, like, carrying the kind of level of stuff that he carries everywhere. Especially when it gets super ridiculous later on in the game. Do I need to run? At this point, I feel like running would just make things a lot more difficult for no reason. Yes, it is live, Peanut. Seems like a rerun. No, this is very much live, although the chat might be a little bit behind. This up. Yes, it's live. Hello. Trust me, if it was pre-recorded, I'll have a lot more interesting plans for that, and you wouldn't be able to tell. I just wasn't expecting to actually join the stream. Yeah, it's a bit delayed. Oh, wow. Yeah, well, I'm not really working with the best... Oh, shit. With the best setup right now, so it's kind of a mess. But hey, we got another 22 hours to figure it out. Oh, shit. Oh, my God. I don't even know what kind of stuff I even have on my back right now. Yeah, I don't know what latency I've set the stream to. Usually I prefer low, just because I hate delays on streams, but... I don't know if I made that super consistent. Yeah, first time playing for me. I've seen, like, some parts of the first, like, 8 hours of this game. So I know what happens. It's kind of a shame that I have to play at the lowest settings, because my computer can't really handle it. But I am very much excited for the second game, even though I haven't even played this first one. I just know that I'll like it. Why would I need to zoom in? Awesome. Alright, we got 20 more minutes before I switch back to studying. Oh boy. It's gonna be real awkward because I know that there's gonna be a long-ass cutscene next. Ah, well. Oh wait, I get graded because of my deliveries? Ooh, I love getting graded. Likes might be a currency in this game, but what about dislikes? It's like YouTube. I deleted the dislikes. Yeah. I didn't get to play PT myself. I'm sure that it's pretty easy to emulate. If I really, really wanted to. But, man, the story behind that entire... Like, all the shit that happened at Konami is messed up. And I'm glad that he's able to make all sorts of weird stuff in his own studio. So it's fantastic. All right. The remakes, yeah, there have been a lot of people trying to remake PT. Because man, when that game or when that demo came out, I love how people call it a game when it's a demo. The whole point is that it's a demo. It's so incredibly short. Well, short if you know everything that you're supposed to do. Extremely long if you don't know what the hell is going on. Quarantine. Oh yeah, this is gonna go great. Yeah, fuck it, I'm Norman Reedus. Oh, right. Yeah, sorry about that. I have to turn on and off the game audio just in case the music kicks in.
Just saying.
Because
And I
I
can't
don't want my
keep
stream to be taken down that early.
That's why
I mean, we just started.
a bridge baby, huh?
With its help and you, we'll be able to stay one step ahead of them. Makes me feel like shit every time.
Well, you are plugging into the other side. Freaks me out too.
Baby. The baby.
Roll out.
Oh, is there a streamer specific mode? Okay. Yeah, I know that some games have that, but I wasn't sure if this one did. Where in the settings is it? Alright, I'll check it as soon as we get this cutscene over with. Oh boy. Ah, my my laptop is working real hard for this one.
The world was different when I was a kid. America is a country. Anybody could go anywhere they damn well please. No need for couriers like yourself. We had highways, airplanes. Hell, we could even visit other countries.
What are these fences for? Trying to protect against BTs?
Hard to imagine it now. As you can see, the Death Stranding poked us full of holes. Fucked us beyond all recognition. And if you were lucky enough to survive, the timefall came and washed you away. Then those freaks from the beach showed up. worlds of the living and the dead all mixed together. And that's when folks started pulling up in the cities. Couriers like yourself got put up on a pedestal.
I find it interesting that this game is all about human connection, but we still have cities, so makes you wonder if people just don't go outside their rooms. It's clearly supposed to be an allegory for what's going on now, or what happened during the pandemic even.
We're gonna have to cut to the BTs.
Oh shit. Oh my god. Rainbow. I don't think that's a rainbow. A lot's going on.
Engine stalled.
Those assholes got us right where they want us. Get us out of here.
Sam, did you see anything?
They couldn't have at least brought a lighter with them. My specs, shoot. 8th gen i7 from Intel and an NVIDIA GTX 1050 Ti. Which would normally be fine for this game. But the problem is I'm streaming. Which everything just chugs whenever I'm streaming. Which is to be expected. But man, I'm just too overambitious. Wait a minute. A lot is happening, and none of it is moving on my screen. Okay, wait. Oh, okay. There we go. Now we're back. It's one of those things where the audio just kind of loads up first. 4 gigs of what? 4 gigs of RAM? Well, you can't run it while streaming, I'm pretty sure. But I feel like if you have the same card, it should be okay to run at least at these the lowest settings. It's just OBS takes up so much of my GPU that I can't really do much with it. It's too bad. At least with this, the cutscenes aren't like completely misaligned with the audio. Because I can reset it whenever I want, which I can't say the same for my other games that I stream on this channel. If you've seen like my Resident Evil playthrough, oh, it's a mess. Jill like says something and then her voice comes in like 10 minutes later. No, not a minute. It's like 10 seconds of delay or something. See, the audio actually like matches what's going on, so I can appreciate that. Well, an allegory of like of what was already happening, and then a prediction of what. Yeah. A prediction. You're right. I always love the shot of him like covering his face with his glove, which is like covered in time ball water. It's just like, well, I guess your face is gonna look like an old man now. Feels bad. Although I am still confused about whether still water produces the same effects as running water. Yeah, that makes sense. 2019. Oh man, it was like right before. Like only a few months before. Damn, Kojima. Studying on a Sunday. I mean, assuming that I would have a test on Monday, that would be normal. But no, this I'm not even taking this test for like weeks. The problem being that I have a lot of like a lot of commitments on the weekdays, and then I don't want to like postpone this studying until next weekend. So we're doing it now, baby. Doing it now. Also, my sleep schedule is totally fucked. So a 24-hour stream might be exactly what I need here. I'm just casually talking over this guy just being dragged away. He's fine. He'll be okay. Man, I remember when this game was still being revealed and stuff, and this is like one of the first trailers they showed. What is going on? What is going on right now? Just glance through it before entering class. Yeah, well, the MCAT is a rather big test. I don't think you can just glance through the entirety of medical information in the year 2022. But I wish I could. I am pretty good at cramming, but I think you're gonna see me study a lot over this coming weeks. Big. Oh damn, I never actually I've seen this cutscene like a couple of times, but I've never noticed that he gets like sucked into the like hands at the top of the head or something. That's intense. And by the way, Peanut, I recommend watching White Light's 7-hour like summary of this game, because he covers everything regarding thematics and like generally there's just like a lot of great information in that video without spoiling. So I've only spoiled up to like however long I was watching that video. Well, I mean, it's not an essay. The MCAT is an 8-hour multiple choice test. So it's kind of hard to BS that other than just to like guess A for everything. I mean, I'll at least get 1/4 of them right? I mean, yeah, Zane, you can talk about whatever for the most part. Obviously, don't dox me. But I mean, yeah, pre-medical. That's what that's my studying. That's what it's all about. In the meantime, Norman is having an absolute moment right now. Oh my god. Why did Kojima send me to this land? What is this? Oh, that's how he gets the handprints on his body. Okay. And then, of course, the C-section scar on his stomach. I'm really curious how much of this exposition is going to be like revisited later, or how much of it is just going to be pure symbolism that we just like pretend that doesn't have like a reason to be there, or a reason that we need to like figure out. Yeah, to be fair, I probably went into a bunch of different deep dives into all sorts of shows and games that I really like, and then I just promptly forget all of the lore. Like, Resident Evil is like one of those things where it's just like, I've seen so much gameplay from all across the entirety of the franchise, including a bunch of spin-offs. And it's just like, bro, I do not understand what's happening in that franchise. Oh shit. Saying, what the fuck? I can't be handing out my credit card information that easily. Come on, man. At least trick me into saying it. Like, if you want to be a pro gamer, you should grab your parents' credit card and tell me the numbers on the front and the 3 epic numbers on the back. Oh, okay. Move. Right. Oh, wait, what? Oh, this is weird. I just need to get closer. Hey, what's up? This is gonna grow great. Alright, as soon as this cutscene is over, I have to save. And then I'll switch back to studying. Wait, audio got desynced again. Oh, audio's still desynced. That's too bad. I think we're still desynced, boys. I feel like this is all this Norman grunting is happening like 3 seconds after. Yeah, okay, there it is. Okay, hang on. No, it didn't. Ah, the audio's so random.
Once there was an explosion. A bang which gave rise to life as we know it.
Norman, can we like resync your audio somehow? How do I do that? There's no options accessible in that pause screen. It's too bad.
An explosion. That will be our last.
Or wait, was it not decent? I'm so confused. Oh my god, I'm gonna have to wait for it to load before I can save. Another 10 minutes incoming. An entire 10 minutes of load times. This is what happens when I'm not playing on a PS5 with the lightning fast SSD. Norman Reedus and the Funky Fetus. You're goddamn right. It do be quite funky. Look at this dude. Trying to teach me about the game's mechanics, which is just walking. Advanced walking. Oh, baby. I'm trying to imagine a scenario where I could do something similar to Norman in this game, where I have to like carry a bunch of stuff on my back and then deliver it to somewhere else, like for a video or something. Can't think of a reason why I would do that. Unless it's to like bring emergency like supplies to a place that is in the middle of a natural disaster, in which case, I mean, would not want to monetize that through putting it on YouTube, so I would probably just do that without a camera. Maybe with it. No. And it's just what would be a lighthearted situation for this? I guess that there just isn't one that exists. The amount of stuff. Like in all other situations, I would just be expected to drive a car and bring it over. Populus trichocarpa. Ah, yes, I know the entire genomic information of such a species, indeed. Isn't that about plants and stuff? I mean, specifically, I specialize. My emphasis is on neurobiology, so want to ask me about grid cells? I got you. They're like cells in a grid to allow your brain to recognize where you are in certain locations. Our brain has like a bunch of different systems that interact with one another to memorize or well, to internalize location information. Yeah. The genome code. Is that the amount of base pairings in that genome? Oh shoot, how many are there in humans? I forget. Wait, are we in the millions? No, that number's too low. What number is that? Can you expand, please? Can you elaborate further? Same number in PT. Oh shit, what did I do? I just pressed something. Uh-oh. Come on, game. I just need to save and then I'll switch back and then Peanut, well, I'll be back playing the video game in like 4 hours, which means oh man, 3 a.m. at my time. I'll be able to play Death Stranding again. Should have been playing Signalis. Would be faster to switch between them. But I've already made the thumbnail, and the thumbnail looks awesome. Okay, it's not that awesome, but it's a good thumbnail. It's a nice thumbnail. I like it. The tree. Okay, so it's a species of tree that is related to PT. Well, I mean, does it even matter at this point? Like, all the information about PT, because Konami's moved on to make a new Silent Hill game, which looks interesting, to be honest. I don't know if I'll play it, though. I mean, I actually haven't played any Silent Hill games, which is really sad because I know that I'll like it. I need to play Silent Hill 2, which they're remaking, but I don't know if I should play the remake, or rather, if I should wait for the remake because I'm sure that they're gonna change some stuff and it's not gonna be the same experience and stuff like that. Whereas with like Resident Evil 2 remake, I think I made the right choice in choosing the remake over the other one because I'm just not interested in tank controls, but I don't know if the gameplay of 2 is going to be the same situation. Are we 200k base pairs? I don't remember. I mean, you guys can easily search it up on your end. Right now, I'm just trying to wait for this game to load. Yeah, Kojima. So, I was really hoping that he would announce his independent title at the Game Awards as opposed to Death Stranding 2. I'm very excited for Death Stranding 2, but I'm way more interested in what he's gonna do for that independent title, which is probably going to be a horror focused title. So that's where like the PT inspirations come into play, I guess. Inspirations, he made the game, but you know what I mean. Where PT comes into play again. It's my boy, Guillermo del Toro. Yes.
I'm a doctor. Well, a coroner.
Originally. Yo, I'm dead. Again? Oh no.
Call me dead man. I'm well acquainted with the dead. Not like you, of course. Contrary to the name, I've never actually died.
Man, I love this man.
I would advise against that.
Or rather, the actor.
I'm no expert, but I can assure you it's for your own protection. See?
Probably one of my favorite directors.
So I'm a prisoner.
I find it hilarious that he's such good friends with Kojima.
They're cutting-edge devices that keep us all connected.
I mean, generally, Kojima just is such a hilarious human being to me. In the best way possible, with the most amount of respect.
Yup. Bridges. Humanity's best hope.
He wears all of his inspirations on his sleeve.
Avoiding extinction, if you prefer. Right.
Definitely inspiration for me.
Watch me. Try pressing it against your skin, like this. Relax. That just means your body's got a good connection to the cufflink. Look.
Hu Tao moment.
The cuffs will watch over you 24 hours a day.
Gee, I wonder why I like Hu Tao so much. I may or may not be interested in stories about death.
2 days. During
You could argue that Death Stranding is PT.
liberty of collecting fluids.
Yeah, I think I've heard some lore regarding PT. I don't remember exactly, so it might be related somehow, but
Makes you very
I don't know exactly. Oh, well, I guess a fetus shows up in PT. No, I don't know.
Central Knot was obliterated in the annihilation. Game over, man. Place is great.
Game over.
The only ones
With
to
the
get in
Metal
middle
Gear styling, that's hilarious.
for obvious reasons, and your broken bridge, baby. Is it all right? It's been marked for disposal.
We injected it with 7,000 liters of Monster Energy.
We lost everyone.
Still not back to full working condition.
Disposal. My team, HQ, all of Redefort, every soul in Central Knot City.
Well, rip.
We're at our base in capital Knot City now, or should I say, our new headquarters. Sudbury seemed the logical choice, but our implementation team has been decimated, and there's only so much they can do in 2 days. Things here are a mess. Fortunately, the director and his support team were out of town at the time, so the chain of command remains intact.
It's a character
So soon.
in a game called Genshin Impact that I really want because it's a gacha game.
This stencil is from when you repatriated?
And she hasn't shown up in the video games for like
What's the other mark from?
A clingy woman in a cave.
she's at like 400 days now. 400 days since we've seen Hutao in Genshin Impact.
Phobia. No wonder you were out there alone, where no one could touch you. I'll try to be more careful, Sam. So, the job is an urgent delivery. I need you to bring the president some morphine.
Why does he have a scar over his head?
Is gone. You talking about the mayor of Central Knot?
What kind of brain-related surgery did he have to go through?
Here, America lives on, Sam.
America.
The president is in the final stages of cancer.
Hey, we were just learning about cancer
There's still time.
like an hour ago.
Why me?
Look, Sam. Do as I ask, and I promise it will all make sense.
Why don't you do it?
Because I'm not really here.
And then walks through him. Bruh.
Apologies. This is just a chirogram. And here, come the tears. Allergic reaction to the chiral transmission. I'm actually over in the isolation ward in the big triangle building. Here is the morphine. Bridges hereby enters into a contract with Sam Porter.
Come on. You got morphine there. What's this about? Tell me the truth.
The truth, Sam.
Oh yes, I have seen that.
Last president.
I think I've seen every single trailer for this game. I was very excited for it to release.
To say no?
But then I never had the chance to play it because I was for various reasons. Only now I'm starting to play it because it's on Game Pass. And my my friend went ahead and gave me the chance to have like 2 months of Game Pass because he's not really interested in it, I guess. And he was like, "Oh, I have codes because Discord gave it to me," and I was like, "Okay, sure." Yeah, who crashes your game and the upside down color inverted of the image of the child and in Death Stranding, you've seen the trailer has the letter J written on it. Okay. Well, yeah. Maybe I should rewatch some playthroughs of PT or something like that. I'm guessing that is probably important. No, read us in the funky. Okay. Alright, guys. Very happy that we're very invested in this game. Oh. Norman jumping. Look at this Spider-Man pose. Doing that full squat in midair. Eee.
Delivery order for Sam Porter Bridges. Request transport of morphine to isolation ward for immediate administration to the president.
Alright. Sounds like a plan. Guillermo del Toro. I'm just gonna totally forget their in-game names. I want this, okay, I want to save here. Is that it? Did I save? Oh god. I have to... Okay. This game's gonna take 10 minutes to load up the next time I play it, but it won't be for another 4... Yeah, I'm not gonna play this game for another 4 hours. Alright, boys. I'm headed into the abyss. Alright. I'll be right back. Give me a moment, and then I'll start studying the MCAT again. Thanks, Omni. Games Radar. Or release of Dead Stranding. Oh, okay. Yeah, maybe I'll look at that. Now though, do what we're actually here for, which is study. Study we will for a very long time. This is not how I expected this to go. Though, to be honest, I was probably just ignoring the fact that my setup is weak. Bruh. Oh, whoops. Hold up. Gotta pull up Opera for that lighter usage of my RAM. Although, considering that I have to delete or I have to close all my windows every time I try to play the game, doesn't even matter that I'm using a different browser than my usual. Oh well. Let's see here. Where's my history? Can I just search? Can I just Okay, perfect. Okay, and then this. Okay, while I change some stuff in the settings, I'm gonna hide myself. Okay, let's pull up Khan Academy again. Mutagens and carcinogens. Ooh, it even remembers the shape of the window. Wow, that's really helpful actually. Ooh, that's really nice. I like that. Oh yeah, that is really nice. So now I can just click this. My Xenoblade music's back. What is the last thing? What am I? The time. The time's important. Where is my time thing? Shoot, now it's too. Oh, that's not what I want. At least I know what I'm looking for. What? How come that? Why are you like this, Opera? What did I do to you? What did I do to deserve this? Day at... Man, that's disappointing. Oh well, okay. I'll just have to reconfigure my settings again anyway. It's fine. Okay. Well, won't get to play as Norman Reedus in The Funky Fetus for another 4 hours. See you then. While I play the best game ever, which is MCAT studying. And I need to make sure I have my timer down the corner without doxing myself. That would be very much appreciated. And OBS is hard to like work with sometimes. Probably because I haven't loaded up my OBS Studio with all of the plugins. Right, I need to update this. Okay. And then Khan Academy, where is it? There we are. All right. Needs to be much bigger. Norman returns at 3 a.m. Oh my god, this text is huge. Actually ridiculous. How do I change text in OBS? Wait. Is there no way to adjust it? What's going on here? Cannot adjust text size. OBS, please. What? It doesn't even wrap around itself, so I have to like... Oh, that looks jank. That looks really jank. What? That's silly. I don't want to do that. Norman returns. Oh, whoops. I like how I can use my left arrow key to like move assets in OBS, but I can't use... Or I can use my left and right, but I can't use up and down. Because OBS interprets that as a scene change, which is interesting. I don't really understand why it thinks that way, but okay. Why not? Returns at 3. Hey, my time zone. PT. Yo, this is very appropriate for considering what game we're playing. Very nice. Planned, completely planned by on my part. Yeah, baby. Okay. Hello, I'm back. Took me like a good 10 minutes to get my setup back, which is hilarious because that's how long the game takes to load. So maybe I shouldn't yell too much at Kojima Productions for optimizing poorly for my computer. But it's really just my limitations, my hardware limitations at play. The effects of mutations.
So today we're going to talk about the overall effects of a genetic mutation, and how mutations impact the affected organism as a whole. But first, I want to review the central dogma of molecular biology, and how genetic information in a cell is stored in the form of DNA, which is then transcribed to form RNA, which is then translated to form protein. Now, nucleotides from DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3, to code for specific amino acids in a larger protein. Now, if you mutate one of the nucleotides on DNA, like turn a stymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are generally mistakes in a cell's DNA that lead to abnormal protein production. So are mutations good or are they bad? And what kind of effect do they have on the affected organism? Well, there isn't really a good answer to this question at all, and there are many, many different types of mutations out there that can result in big structural changes, like the little pictures I've drawn out here, or may result in little cell changes that may go completely unnoticed. It's very difficult to call a mutation good or bad, though, since it really depends on a huge number of things, including the environment that the organism lives in. So let's look at an example of a good mutation. So the bacteria Streptococcus pneumoniae is the bacteria that you typically see associated with pneumonia, and one of the more popular treatments for pneumonia is giving the infected person an antibiotic like penicillin, which would help kill all of the bacteria and get rid of the disease. But sometimes you can find some mutated Streptococcus bacteria that will have a special trait that makes them resistant to penicillin, and now penicillin won't kill them as easily as it'll kill the bacteria without the mutation. Now, we call this a good mutation because the bacteria are living in a human host where they're likely to encounter this deadly penicillin, and being resistant to antibiotics like penicillin would then be beneficial to the bacteria. And just to clarify, I'm calling this a good mutation for the bacteria, not really for the human infected, since it'll be harder for them to get rid of the bacteria that are resistant
That's
to
a
certain
good
antibiotics.
clarification
So
there.
now let's look at an example of a bad mutation.
No,
So
it's good for the bacteria though. Not so much for you.
CFTR gene. Now, I'm not really going to go into detail about how this mutation actually hurts you, but I'll leave you with the idea that what it does is it makes the mucus that you'd find in a person's lungs really, really thick, which makes it really hard for people affected with the disease to breathe.
Thick.
So in general, we can say that the mutation causing system
Dummy thick mucus.
But mutations aren't strictly good or strictly bad. In fact, there are some
Clap
mutations
of my mucus lining.
Sickle cell disease results from a mutation in a protein called hemoglobin.
Alerts the guards.
And this mutation turns hemoglobin into a much less functional form, which we'll call HbS. And it's much less efficient at moving oxygen around the human body. But another effect of sickle cell disease is that it makes the diseased person less susceptible to malaria. Now, malaria is a parasite that grows and multiplies in red blood cells and can have a lot of nasty effects on the host organism. And the malaria parasite can't really grow as well in red blood cells that are affected with sickle cell disease. So, in this case, the mutation associated with this disease has one bad effect, which is that the HbS isn't as good as carrying oxygen around the body, but also a good effect in that it makes it less likely that the diseased person will be affected by malaria.
And then if you have
blood cells. So what did we learn?
the dominant and recessive alleles,
There are some exceptions to this rule.
you get the dual wielding of the benefits of sickle cell without having the messed up stuff in sickle cell. Well, the cells are still messed up, but not all blood cells are gonna look like sickles, and so that's pretty helpful compared to if you had a fully homozygous. Homozygous sickle cell gene situation. Very cool. I don't think I need to take any notes there. That's just a nice overview of why mutations are important. Oh no. You can't see on no, you can't really see it on your end, but this is Mendelian genetics, which
An introduction to Mendelian genetics. Now,
this is
before we
just
start, let's
math.
review the idea that human cells contain 46 chromosomes, which contain the DNA that makes each cell
Complicated
unique.
now.
And 23 of these chromosomes
Math,
are
but
inherited from a person's
simple.
father, and 23 were
Simple
inherited from a
math.
mother. So we can say that each person is made up of a combination of genetic
Oh
code from
boy.
both of their parents. Now, sometimes
how
we
am
like
I
to say we have
gonna-
23 pairs of chromosomes, instead of saying that we have 46 total, because that way we remind ourselves that for each chromosome we have a maternal and paternal copy. Now, the first thing I want to introduce is the term allele. Now, if we have a chromosome here, then an allele is one small section on that chromosome that codes for a specific gene that makes you, you. Now, since humans have at least 2 copies of each chromosome, we can say that humans usually have at least 2 alleles for every specific gene, one allele from their mother and one from their father. So let's look at an example, and we'll start by talking about blood type. Now, I'm sure that you've heard that blood types are usually named with letters like A, B, and O, but what does that actually mean? Well, there's a specific allele that codes for blood type. And let's say that we have this guy here, and his alleles both code for blood type A, and I'll use the letter A for that. And let's say we have this girl here who has one allele coding for A and another allele coding for blood type O. Now, for the guy, if he has both alleles coding for blood type A, then it's pretty clear that when you check his actual blood type, it will be A. But for the girl, we're not so sure, since she has one of each. So now I'm going to introduce a couple new terms to you, and the first is that since the guy has 2 alleles that code for the same thing, both code for blood type A, then we say that this guy is homozygous. And homo means the 2 alleles are the same, homo the same. And zygous refers to the mixture of DNA that he got from his parents. So someone who is homozygous got the same allele from both parents.
Oh my
Now,
god, how do we not even cover,
Well, it
like,
turns out that she's going to
alleles yet? And where would I even put that? Oh my god. Where does one... Wait. Oh no. Oh, I should have put like a genetics branch, but like the whole this entire thing is genetics. This entire section is about genetics. Okay, is it appropriate to branch alleles off of chromosomes? Or do I have to make another branch off of DNA? And considering how many branches I have off of DNA, it's probably not that big of a deal. Oh wait, I need to make this purple. Because it's part of the pathway along along which the central dogma resides. Mutations are important. DNA damage. Wait a minute. Isn't DNA... DNA damage isn't the same as mutations. Never mind. It could be in the situation... No. It could be in situations where mutagens just completely mutilate the double strand. Like... Oh god. If you were to split your DNA, that's... I can imagine that that's not good. Okay, I'll have to move this section up. Create more space. And then I'll just like make a DNA or Mendelian genetics branch off to the side here. That's the game plan. That's the game plan. Here we go. Wait, where's my music again? Why does Xenoblade not continue? Why you pause? Throw chromatin. Okay. What do I call it? Genetics? Call it alleles? No. No. Let's go with Medallion. I'll go with alleles. Assuming that we haven't seen that term earlier, in which case we would run into an awkward situation where things are supposed to connect over to other things. Parallel. That's funny. Okay, cool. We're good. So, homozygous.
Have blood type A, and that's because the A allele is the dominant allele, while the O allele is the recessive allele. When an allele is dominant, that means that if someone has two different alleles, it'll be the dominant one that wins. So in this case, since A is dominant over O, which is recessive, A will win, and she'll have blood type A. Now, since this girl has two different alleles, we call her heterozygous, since hetero means different, and zygous refers to the same thing, the mixture of DNA that she got from her parents. Now, I want to introduce two more terms. We can describe a person's genes in two different ways. We can look at the person's individual alleles, and we call this the genotype. So for this guy, his genotype is AA, referring to his two alleles, which both code for blood type A. But we can also look at a person's physical traits, which we call the phenotype. So for this guy and girl, the phenotype would be blood type A. So you can see that genotype and phenotype are different, but it is possible for two different genotypes.
Okay, wait a minute. Wait, is homozygous referring to the gene or the allele? I always get confused on the terminology here. It's like, it's a homozygous gene, right? Homozygous. Having two identical alleles of a particular gene or genes. So you can have a homozygous genotype. So it's an adjective to be placed or to be modify genotype. I should have another window on the screen for you guys to see what I'm Googling. But it's very simple. Like, I literally just Google the terms off to the side. Okay, sure. So, in that case, I'll delete this and then put genotype here. And then phenotype will be right above it, and then genotype will branch into homozygous and heterozygous. While phenotype doesn't really branch to anything, it's just going to be right above genotype. Phenotype. Having 3 hours, yeah. Having 3 hours study sessions probably is more accurate to the test experience after all, so maybe this is a blessing in disguise. Zygous and heterozygous? Of a genotype? Oh, come on, Miro. You're so close to greatness. Oh my god, my voice. Already getting wrecked after 3 hours. That's why I need to make sure I hydrate. I also have plenty of coffee and tea to keep me going. Next time I need to snack on something, you know. I can't. I gotta make sure to not load up caffeine, or rather, not load up on sugar too much, and balance it out with just pure aqua. Pure. Oh man, it doesn't reach around. Pure aqua. Can't really see her that well. I have a Konosuba poster right next to me. You guys barely ever get to see any videos because it's at too awkward of an angle. You'd have to like look at me from the side profile in order to see them. And then Megumin's like all the way at the top. So then I have to tilt it up so then you'd only see this part. And then at that point I'm just like creating my own GigaChad meme.
Phenotypes to make the same phenotype, since some alleles are dominant over others. Now let's talk about gene inheritance for a bit. So let's say that our guy and girl from before have offspring together. We can use something called a Punnett square to determine what different genotypes their kids could have.
Ooh, congratulations.
Alleles are on separate chromosomes, so each parent can their 2 alleles to a child, and the Punnett square allows you to
I
determine
just
all
love
those
it
combinations.
whenever examples introduce 2 characters and they're just like, oh, they just have kids now. And it's just like, whoa, okay. That relationship moved instantly. That was crazy. God damn.
Now let's see what happens if we change our father's genotype to match our mother's genotype. Now, only 1/4 of the children will have the AA genotype. Half will have the AO genotype, since the order of the alleles doesn't matter. O, A, and AO are the same. And 1/4 will have the OO genotype. So this means that
Not
75%
like
of the children will have
this.
blood type A
Punnett
phenotype, since
squares.
the AA and AO make blood type A. But 25% of the children will have the blood type O phenotype.
It's not so bad when it's the
Well, first
2 alleles,
and the difference between homozygous
but
and heterozygous,
when
as
we're
well
going
as
to
between
4,
dominant
it's just like,
traits in relation to
bro,
alleles.
I
Second,
don't
we
want
learned
to
about
do
the difference
this.
between phenotype and phenotype, and how the
I don't
genotype
want to
versus
do
phenotype
this
different
anymore.
traits of the DNA code.
Mendel,
And finally, we learned about how we can use a Punnett
why
square
did
to
you
determine
do
how many alleles
this?
will be inherited from 2 parents.
Square. Punnett square. Miro's telling me that I spelled it wrong, but I thought I spelled it right. What? Why?
An introduction to Mendelian genetics. Now before genotype and phenotype, and how the genotype refers to determine how different alleles will be inherited
What?
from 2
2
R's
N's
that
and
have
2
the A in them, and 2
T's.
will have
Yeah. What? I got that right. The hell, guys? Co-dominance and incomplete dominance. Jeez. Maybe I can branch Mendelian genetics off of alleles. And that's how we do this. Because alleles is the general concept, and then Mendelian genetics is the umbrella, like, field, essentially. So I can just do this. And then Mendel's just gonna have his own branch. Play around and have fun with all the fruit flies. Just making an incessant amount of generations of themselves. And peas, of course. Peas and fruit flies. Although, wait. Mendel worked on peas, and then he worked on fruit flies as well, right? Or am I thinking of a different guy that worked off of Mendel's ideas, and then did a fruit fly related experiment? I don't know.
So today we're going to talk about co-dominance and incomplete dominance. But
Oh
first,
boy,
let's
here
review the
we
example
go.
of blood type, and how someone with the same two alleles coding for the same trait would be called homozygous.
We're really gonna
and someone
stick with
with different
blood
alleles would be
type
called
as the
heterozygous.
example, huh?
Also
Alright.
remember the concept of dominant and recessive alleles, and how the A allele is
The
dominant
relevant
over the
one.
O allele in this example. This means that the same phenotype, blood type A, can result from these two different genotypes. Now, the example that I just gave you was an example of complete dominance. So if a person had genotype AO, since our phenotype is just blood type A, it means that the A allele is completely dominant over the O allele, and only the A allele from the genotype is expressed in a phenotype. But there are actually three different patterns of dominance that I want you to be familiar with. And to explain this, I'm going to use a different example. So let's say we have this flower, and the red petal phenotype is coded for by the red R allele, and the blue flower phenotype is coded for by the blue R allele. So I'm going to introduce three different patterns of dominance, and they are complete dominance, which you've already heard of, co-dominance, and also incomplete dominance. And I'm going to explain what these two new patterns are through this flower example. So let's start by looking at three different genotypes and the phenotypes that you would see for each of them under each different dominance pattern. So we'll start with the genotype 2 red Rs, which we can expect that in all cases the flower petals will be red, since we only have red Rs in the genotype. Similarly, if our genotype had 2 blue Rs, then we can expect that in all cases the flower petals will be blue, since we only have blue Rs in the genotype. Now these three different dominance patterns change when we look at the heterozygous example, and that's what makes these three patterns different. Now we're already familiar with the example of complete dominance, so if we said that the red R is dominant over the blue R, then this would make the heterozygous phenotype a red flower for complete dominance. Now what co-dominance is when the heterozygous phenotype shows a flower with some red petals and some blue petals. So it's when the two alleles are dominant together, they are co-dominant, and traits of both alleles show up in the phenotype. Now what incomplete...
This Xenoblade track is reminding me of another track from Zelda. I don't know what it is off the top of my head that it's reminding me of. What are you? Da 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 da. I think it sounded like Zelda's song or Zelda's theme to me or something. It sounds nothing like it though. Reminds me of it. I'm math. Ugh. It just had to combine the two regions into High Macta Wildwoods. Macna. And then I forgot the name of the other one. Margatha? Margaret? Margit? Elden Ring? Bill Clinton? No.
complete dominance is when a heterozygous phenotype shows a mixture of the two alleles. So in this case, the red and blue flower petals may combine to form a purple flower. Neither allele is completely dominant over the other, and instead
Yay.
the two, being incompletely dominant, mix together. So what did we learn? Well, if we assume the heterozygous genotype, red R, blue R, then there are three different dominance patterns that we might see for a specific trait. In complete dominance, only one allele in the genotype, the dominant allele, is seen in the phenotype. And this was the example with the red flower. In co-dominance, both alleles in the genotype are seen in the phenotype. And this was the example with the flower with both red and blue petals. And finally, in incomplete dominance, a mixture of the alleles in the genotype is seen in the phenotype. And this was the example with the purple flower.
That makes sense. That makes sense. What can I say? But that makes sense. Oh my god, we're almost at Hardy Weinberg. Oh lord, he coming. Or they coming.
In the last video, I drew this grid in order to understand better the different combinations of alleles
Oh
that
my
came from my mom or
god,
my dad.
it's
And this grid that I drew is
25
called a Punnett
minutes
square.
long. What the
Punnett
fuck?
square. And I looked up what Punnett means, and it turns out, and this might be the biggest takeaway from this video, that when you go to the farmer's market or you go to the produce and you see those little baskets, you see those little baskets that often you'll see maybe strawberries or blueberries sitting in, they have this little grid.
What is he even talking about? What?
A bunch of strawberries in them, like that. That green basket is a punnet. That's a punnet. Apparently, in some
That's
countries
a
they call it
punnet?
punnet.
Wow, I'm learning so much.
UK viewers can correct me if I'm wrong, and so I guess
Okay,
that's
okay, guys.
the B
I'm just
situation
gonna
that
do the
where
pro
one trait
gamer
is necessarily
move of
dominant,
skipping
you
through
have the situation.
his
So incomplete
explanation.
dominance. So incomplete, two plants were to reproduce, and the traits for red and white petals, I guess we could say, are incomplete dominant or incompletely dominant, or they blend. And if I were to say,
You know what could be interesting? I'll just like color it like this. And then color this like red. Then the lettering is gonna be like blue. Okay, that's too hard to see. Oh man, that's too hard to see. Okay, and then I'll color this like orange. And then that represent. It looks terrible. Why? Orange doesn't even look like red. It's just yellow. What the heck? Can I get it more orange, please? There we go. That's not a very good representation of incomplete dominance, but whatever.
hey, what's the probability of having a pink plant? And now when I'm talking about pink, this of course is a phenotype. So the probability of pink, well, let's look at the different combinations. How many of these are pink? This one is pink, and this is pink. So two are pink of a... Oh, then this combination would have been a 2.
I'm surprised that they have such a long video on Punnett squares, because Punnett squares are very straightforward until you get to the big boys. The big Punnett squares is where it gets crazy.
Let's assume that we're talking about traits that assort independently, and we cross two hybrids. So this is called a dihybrid cross, very fancy word. But it's just
Oh,
an idea
here
of
we
the
go.
power of the
A
principle.
dihybrid
So let's say
cross,
both
perfect.
parents are, so they're both hybrids, which means
Yeah,
that they
see,
both have,
this
they
is
both
where
have
we get
dominant brown eye alleles.
ridiculous.
I think hopefully you're not getting too tired here.
This is
So
very
we have a
silly.
Capital B, big teeth.
What a gamer
So if I want big teeth,
spreadsheet.
and brown eyes.
Ugh. What is this, an RPG?
So big teeth and brown eyes.
The
So we have those
spreadsheets.
both, the dominant. So if you have either a capital B or a capital
Oh
A, you have
god.
big teeth and brown eyes. So this is big teeth and brown eyes. This is big teeth right here, brown eyes there, big teeth. Or maybe I should say brown eyes and big teeth, because that's the order that I wrote right here. Brown eyes and big teeth, brown eyes and big teeth. Even though I have a recessive trait here, the brown eyes dominate, and I have a small teeth here, but the big teeth dominate. This is 3 combinations of brown eyes and little teeth. 3. And if I were to say
Oh my god, it's just counting. Why are we just counting, guys?
explore incomplete dominance when there's blending, where
Where's
red and white are
the-
genes, or you could even use it when there's co-dominance and when you have multiple alleles, where it's not just 2 different versions of the gene, there's actually 3 different versions. So
Could show me the math at least. That'd be interesting information. I don't remember the way that you can just calculate the answers for everything.
now that we're familiar with the idea of allele
Okay,
frequency,
Hardy
let's build on
Weinberg,
that
here we go.
To develop the Hardy, I'm just in a new color, and actually let me do it right over here. The Hardy
Oh my god.
Weinberg.
Oh my god, we're
Weinberg
gaming.
principle, which is a really useful principle for thinking
my
through
God.
what allele frequencies might be, or what probability you have if you found someone of what percentage of the population might be homozygous recessive or homozygous dominant, or might be a heterozygote. And it really builds on the work we've already seen with allele frequency. Now, before we go into that, we're going to make some assumptions, and these are all just assumptions that get us a stable allele frequency in the population from generation to generation. We're going to assume that there's no selection, no natural selection is going, or even unnatural selection is going on that would change the allele frequency. So it's not like people with one of the alleles or another are going to be more or less likely to reproduce and have viable offspring. We're also going to assume no mutation. So we're going to assume that one of these alleles isn't from generation to generation turning to another one, or turning into maybe a different, a new type of trait, whether it's green eyes or whatever else it might be. And we're also going to assume large populations. So that would definitely throw out the example that we looked at in the last video, which I did just to understand the notion of allele frequencies. Where we say, hey, look, 1 out of the 4 of the genes in this population, or 1/4 of the alleles in this population, are the dominant brown, while 3/4 or 75% were the recessive blue. We're going to assume large populations, so many, and so that's so that
Controllers
very
in the
small
way.
populations, you can imagine that depending on how these reproduce, it's very easy to get to changes in frequencies. But in larger populations, that helps us make the assumption that we have stable allele frequencies. So once again, this is also that we have stable allele frequencies. Now, based on that, we've already seen if we take the frequency of the dominant trait, which we can denote with P, and to that we add the frequency of the recessive trait, allele, let me be very careful here, the frequency of the dominant allele, and to that we add the frequency of the recessive allele, what's that going to be? Well, you see in this case, it adds up to 100% or 1, and it's always going to add up to 100% or 1, because we're assuming that there's only 1 of 2 alleles in the population. So you have a 100% chance of getting one of these two. That whatever percentage is going to be, whatever the frequency here is, 100% minus that is going to be whatever Q is. So these two things are going to be equal to 100% or equal to 1. And now we can start to do a little bit of interesting mathematics. It'll allow us to start thinking about things like homozygotes and heterozygotes. And so to do that, let's square both sides. So let's square both sides of this, a little bit of algebra in biology class. And so when you square the left-hand side, this is just squaring a binomial. You might want to review it if this looks like Latin to you. There's many algebra videos on Khan Academy that go into this.
I like how
This is
they're
going to
just
be
like,
B
oh
squared
my god,
plus
maybe you don't know how to
2
square. Oh
plus
shit. shit.
2 times PQ plus 2 times PQ
Oh
plus
no.
1 squared. And of course, 1 squared is
Math.
now, what are each of these?
My
What are each of these?
weakness.
Let's just think about something. P squared is the same thing as P times P. Well, P is the frequency of your dominant allele. So this is the frequency of your dominant allele, the percentage of the allele population, I guess you could say, the allele frequency that is dominant, and you're multiplying that times again. Well, another way to think about P is this is the probability if you were to randomly pick one of these genes. So if you were randomly to pick one of these 4 genes, and here I'm just using my oversimplified population, of course, for the truths that we're about to surface to be true, you're going to have to assume a large population. But in this one right over here, one way to view P is what's the probability if I were to pick a gene at random? What's the probability that it is the variant, or it is the dominant allele? What is the probability that it represents the brown variant? So that's one way to view P. So the probability of getting let's just write it that way, a capital B, a dominant brown allele.
Oh man,
So
I set up
dominant alleles. Or another way,
alarms for every single time I get to play Death Stranding, but now that we've changed the schedule, I have to like rework everything. Okay, let's see, 3 o'clock, and then 4. Reach 4 o'clock. 4 a.m. 3 hours after 4 a.m. is 7 a.m. Then I can play the game again or play until 8 a.m. Then 3 hours after 8 a.m. 11, which I'll play until 12. And then 12, 3, then 4. And then I'll have a special break at 5. Break at 5 p.m. Oh, man, how am I gonna work that into the schedule? I guess I'll just, yeah, I'll just treat it as a special break. And then ignore it for the schedule to continue. And then I get to have my Death Stranding gameplay at 7, and then we'll be done after that. It'll be the end of the 24 hours when I play Death Stranding at 7. Okay, cool. Stable allele frequency.
way of thinking about it, this is the probability for someone in the population to be homozygous dominant. So it's the probability of someone being capital B and capital B. And so what is, by the same logic, what is Q squared? Well, Q squared, that's just Q times Q. Q is the probability of getting 1 recessive allele. So this is the probability of getting 2 recessive alleles, one from your mother and one from your father. So this is the probability of if you were kind of randomly born into this population, of getting 2 recessive alleles. Now what is this middle term right over here? Well, P times Q. So PQ, so one way to think about it, if you said, what's the probability that from your mother you get randomly, you know nothing about them, or if you pick a random mother and a random father, what's the probability, I have to be careful here, so if you're just randomly getting alleles, what's the probability that from on one side you're going to get the dominant, and from the second side you're going to get the recessive? So that would be PQ, that would be P times Q. So that's getting it from, say, from one parent, and then that's from the second parent. But what about the other way around? From the first parent, you have a Q probability of getting the recessive one, and from the second parent, you have a P probability of getting the dominant one. So there's 2 ways of becoming a heterozygote. And so if you add these 2 probabilities, what do you get? These are both PQ. I'm just changing the order of multiplication. You sum these two, you get 2 PQ. So this is the probability of being a heterozygote. So this is a pretty neat result. Just by making a few assumptions and reasoning through this notion of allele frequency, we're able to come up with this expression that actually is fairly powerful in thinking about allele frequency in a population, and actually the different genotype frequencies in a population. You see, it all makes sense. These all add up to 1. The probability of someone being homozygous dominant plus the probability of someone being a heterozygote plus the probability of someone being a homozygous recessive, they're going to add up to 100% because someone's going to have to be one of these 3 things. Now I'm going to leave you there in this video. In the next video, we're actually going to use this Hardy-Weinberg equation to actually come up with some very interesting results about a population.
Right. Okay. Applying the Hardy. Oh, that's it? That's the entirety of Mendelian genetics? Oh, this must be a long video. Oh boy, here we go.
So let's stick
Really?
The simplification.
Okay.
There's a gene for eye color, and it only comes with 2 variants.
I mean, I guess Hardy-Weinberg is pretty simple.
Eye color, and it has
But
the
okay.
recessive variant, which codes for blue eye color. So if you have if either one of your alleles is capital B, you're gonna have brown eyes. The only way to have blue eyes is to have lowercase.
I like how someone could probably watch the stream
Now let's say that in a population,
and be prepared for something like AP Bio or General Biology. This is some like, this is just like pure review of that
Can we figure out
specific class.
which is the frequency of the dominant allele? Can we figure this out? And can we figure out Q, which is the frequency of
I mean, I took an advanced genetics class after that,
we
but
saw the Hardy-Weinberg equation. Can we figure these things out given this information? Well, let's revisit
that was a
the
little
Hardy-Weinberg
different.
equation, which we worked out in the previous video, but I'll rewrite it right now. It says the allele frequency for the dominant allele frequency squared plus 2 times the dominant allele frequency times the recessive allele frequency plus the recessive allele frequency squared is equal to 1. And we saw that this just comes from the idea that P plus Q is going to be equal to 1. There's a 100% chance if you were to randomly pick a gene that it's one of these two variants. Now, when you say 9% has blue eyes, what does that mean? Well, the only way to have blue eyes is if your genotype is homozygous recessive, because if you have a capital B in here, then you're going to have brown eyes. So we can say that 9% also has this genotype. Or you could say that the frequency in the population of this genotype is 9%. But we've already seen that's exactly what this term right over here is. That's this Q squared term. This is the probability, one way to think about it, this is the Q, of course, is the frequency of the recessive allele. Now this is you could view this as the probability of getting 2 of the recessive alleles. It's going to be, if you're in your population, it's going to be 9%. So we could say Q squared is equal to 9%. Or another way to think about it, this term.
Yeah, I always get confused by Hardy-Weinberg just because you have to take the square, like it's the one that, like the phrase this squared is equal to that percentage. You have to remember that, or else it's just like, wait a minute. Wait a minute. This isn't the pure frequency of the allele. This is the frequency of the phenotype, of which there has to be 2 alleles that are recessive in order to result in that phenotype. It makes sense when you like work it out in your head, but like when you're just doing the problem, it's just like I see numbers, I put number into the equation. That's probably why I'm really bad at math.
30% express our code for the recessive allele, are the recessive variant.
My math is just about remembering what equations do what and why they do it. Speaking of which, oh man, our chemistry thing down here, I don't remember. I do not remember how this works. I do not remember how this works. I need to review that.
Based on this, and once again, a simple equation, but these really neat ideas are starting to pop out of it. Based on just this information, we're now able to say that 49% of the population is going to have a genotype of capital B, capital B.
It's probably like the least important part of my MCAT review
that's going to be
that I'm doing.
times 0.7
I'm spending so much time on.
times 0.3 times 0.3. So let's see, that's going to be 2 times 0.21. So this is going to be, this right over here is going to be 0.42. Or another way to think about it is 42% of this population is going to have the genotype uppercase B and lowercase b. And you see they all add up. 49% plus 42%.
That makes sense.
Plus 9%.
All very logical, as opposed to whatever the hell is going on in the chemistry section. I can't keep in mind all these concentrations, bro. These concentrations be flipping around, dude. Damn. Alright, cool. Well, are we almost done with Unit 5? Please tell me we are almost done with... Oh my god. There's still more... Oh, we get to talk about gel electrophoresis. Alright, that's fun. PCR. Okay. Alright. These are actually important
Say
sections.
that you have some vials here, and you know that in the
Very
solution you have
nice.
fragments of DNA in each of these. And what you're curious about, what about the DNA
Whoops.
fragments in this first vial, in vial number 1? How long are those fragments? How many base pairs? How long are they? Well, you might say, well, why don't I just take them out and count them? Except for the fact that they're incredibly small and incredibly hard to handle. Even a fairly large fragment of DNA, let's say we're talking about something that's on the order of 5,000 base pairs, well, that's going to be approximately 1 to 2 micrometers long. If you were to completely stretch it out, and we can't even start to think about how thin the actual diameter is. If we just, but lengthwise, the long way.
DNA technology. Let's make a section for DNA technology, and then we're almost done here. I think, probably. I imagine this is the last section regarding DNA. Hope. Hope and hope. I guess it can just be next to alleles. You get to have your time in the spotlight next to alleles. That's fine. Yeah, I like how enzymes is down here. But enzymes should be up. Oh god. Maybe I'll have to move it up here with the proteins. The enzymes are just like a special type of protein, so it's... Are there enzymes in this section, though? Like, do I actually mention enzymes here? Huh. Well, non-enzymatic protein, but I mean, yeah, that's non-proteins, or non-enzymatic proteins. Huh. Oh, well. Okay, cool. Don't have to worry too much about that then. I'll just keep working. Let's bring around DNA technology here. Like super biotech. I mean, it is biotech, but like DNA biotech. Yee boy. Yee boy. Yeehaw. Alright, DNA technology includes gel electrophoresis. Gel electrophoresis pretty simple. I just forget specific. Like, I forget which direction it goes sometimes. I'm like, what is the base charge of DNA, which is apparently negative now that I'm looking at the thing. Because we all know that it has to go from the little wells to the opposite end, so... Electrophoresis. Yeah, gel electrophoresis is always one of those things that is just so fun in the classroom. Like, yo, I extracted this DNA and it's moving. Oh my gosh. Oh my god, it's morbing. The DNA, it's morbing. Oh shit. Oh my god. Okay. What am I doing? Yes, we're good.
It's only going to be 1 to 2 micrometers, which is super duper small. This is 1 to 2 thousandths of a millimeter. So that's not going to help us to somehow try to manipulate it physically with our hands or with kind of rough tools. So how do we do that? And we can have other vials there. How do we see how long it on?
Oh, come on.
Well, let me make it a bit easier to read.
I was trying to click the thing.
And it would come
Let's restart.
Let's say that you have some vials here, and you know that in the solution you have fragments of DNA in each of these. And what you're curious about, well, what about the DNA fragments in this first vial, in vial number 1? How long are those fragments? How many base pairs? How long are they? Well, you might say, well, why don't I just take them out and count them? Except for the fact that they're incredibly small and incredibly hard to handle. Even a fairly large fragment of DNA, let's say we're talking about something that's on the order of 5,000 5,000 base pairs, well, that's going to be approximately 1 to 2 micrometers long. If you were to completely stretch it out, and we can't even start to think about how thin the actual diameter is. We just, but lengthwise, the long way, it's only going to be 1 to 2 micrometers, which is super duper small. This is 1 to 2 thousandths of a millimeter. So that's not going to help us.
DNA small.
With our hands or with kind of rough tools.
Like
so how do we do that?
VTuber Shark.
How long the
small.
sitting in those vials actually are. And the technique we're going to use, gel electrophoresis, actually could be used for DNA strands, could be used for RNA.
Now I'm just imagining Gura, like her face edited onto a double helix.
And it's called gel electrophoresis because it involves a gel.
What am I doing?
It involves electric charge, and phoresis is just referring to the fact that we're going to cause the DNA fragments to migrate through a gel because of the charge. So phoresis is referring to the migration or the movement of the actual DNA. So how do we do this? Well, here's our setup right over here.
How am I like cold wearing this many layers?
Embedded in a buffer solution. So this gel, the most typical one is agarose gel, polysaccharide that we get from seaweed, and it's literally a gel. It's a gelatinous material.
Other jacket
and
on
what
top.
we're going to do is we're going to take samples. So we might take a little sample from this one.
This is crazy.
This well. Right here, and you can do these wells as divots in gel. You could take a little sample from here and put it into
going further beyond.
Put a sample from here, and you could put it in that
next
well.
stage
This buffer,
in my
the
Super
buffer,
Saiyan form.
Water with some salt in it, and the buffer is going to keep the pH from going too far out of bounds as we place a charge across this entire thing. Because if the pH gets too far in the basic or acidic side, it might actually affect the DNA or affect the charge on the DNA. And what we're going to do is
I had put on the
whole.
whole. I could have just avoided the whole like green screen debacle had I just worn this earlier.
Is going to be our positive electrode.
Man.
We're going to use the fact that DNA has a negative charge at the typical pHs, or the pHs that we are going to be dealing with. And we can go back into previous videos, and we can see it right over here. You see these negative charges on our phosphate backbone. And so what is going to happen? What is going to happen once we connect both of these to a power source, and this side is negative and this side?
So we can put the negative electrode here, the buffer, and then headphones are getting caught in my stuff. I mean, I've been contemplating changing out these headphones for something else. I mean, I have to buy these things like every year or so. Like it's not that big of a purchase, especially if I'm taking care of it like well. As long as I don't yank it, it'll last for pretty long. The problem is like my setup is so annoying to deal with. The wire in the way. That I could upgrade to like a really good set of headphones that are like Bluetooth enabled, you know. That'd be pretty cool. But I don't know. These hold sentimental value, or at least the specific type of headphone. And if you know, you know who I'm talking about that has worn these before. But at the same time, they're just like really good headphones. I keep calling them headphones. It's just annoying calling them like clip-ons or whatever they're called. I don't know what the technical term for this thing is, but it whatever. I'll just keep wearing them, I guess. And then I have Raycon, but they're not sponsoring me, so I'm not going to mention them too often until they sponsor me. Come on, guys. I have an entire 40 subscribers. It's worth it, I swear. You'll make bank. Bank. I like how the Xenoblade music is just like softly playing some epic track right now, but like it's so low on my end that I can barely hear it. I could afford to turn it up, I guess. Because I don't think volume matters particularly much in when it comes to DMCA requests. So we can afford to make it a little bit louder. Yeah, okay. Just unfortunate that I guess I can turn up Khan Academy, but it's not really gonna help much. No, I'm pretty tired already. This is just whack. We're going all in, baby. Oh yeah, this is much nicer. I can actually hear what I'm listening to. Featuring Norman Reedus. What a glorious thumbnail. Okay. Enough patting myself on the back. I think I'm good here. There we go.
Well, the DNA is going to want to migrate. Now, let's think about what will happen. Will
I'm watching myself.
Or will longer things migrate further? Well, you might say, well, longer things are going to have more negative charge, so maybe they go farther away. But then you also have to remember that they're also moving more mass. So their charge per mass is going to be the same regardless of length. And so what determines how far something gets, how much it migrates over a certain amount of time, is how small it is. Remember, we have this agarose gel, and people are still studying the exact mechanism of how this DNA or these macromolecules actually.
Well, now the music sounds too loud, and it's distracting. See, dang. That's crazy. I keep saying crazy, by the way, but like... I mean... Obviously, I'm not talking about mental health whenever I say that word, okay? Just want to make that super clear. I understand that, like, when it comes to colloquial usage of the word crazy, I think most people understand that it's not referring to any mental health related situation or condition or anything like that of that nature. It's unfortunate if they do refer to a person as with that word, then it becomes problematic. But for the most part, for like usage in terms referring to situations and such, I really don't think I need to police myself when it comes to that. But who knows, maybe that'll change over time. But for now, just gonna be chilling.
Things are going to be able to go through the gaps easier than the larger things. And so if you let some time pass, some of the DNA, let's say this DNA gets around there, let's say, and I'm just color you actually when you see these colors, let's say this DNA gets around that far, so it doesn't get as far. Let's say that this DNA doesn't migrate. Let's say it has some that migrates that far, let's say it has some that migrates that far. And so if you just saw this, you wait some amount of time, and you were to come back, and you were to see this migration, you were to see this
only 2 times speed.
And the longer you wait, the further these things.
It's just too slow.
All the way over the other edge.
Too slow.
If you just saw this, you say, okay, well, this strand right over here, these must be smaller DNA molecules. They must be shorter. These must be a little bit longer, and these must be even longer than that. And this grouping right over here is going to be the longest of all. So this was a mixture of some longer strands and still longer ones, but not quite as long. And for example, maybe there are some really short strands. Maybe there were some really short strands. what I'm drawing as that orange group right over here. So what I just did right over here, this could tell you the relative length of these strands, but how would you actually measure them? Well, that's where you can go find standardized solutions, which we call a DNA ladder. So let's say you go get the DNA ladder. I'm going to draw it in pink. So you literally could buy this. You can buy it online. And the standard solution, let's say it separates like this. So it separates like that goes there. Let's say some of it goes like there, and some of it goes like there. Well, you would be able to know from the labeling, or whichever one you choose to buy, that this grouping here, this is all of the DNA that is 5,000 base pairs. Let's say this right over here is 1,500 base pairs, and let's say this over here is 500 base pairs long. And so now you can use this DNA ladder, these standardized ones, to gauge how many base pairs these are. So you say, okay, this blue one here, this is a bunch of DNA that's a little bit longer than 500 base pairs, but shorter than 1,500 base pairs. You can see this green one here. Well, it's a little bit longer than 1,500 base pairs. It didn't migrate quite as far as this big bundle of 1,500 base pairs did. And so then you can get a better approximation, and you can choose your ladder based on what you think you are going to find there, what you're actually going to look for. Now, the other thing to appreciate is when you see the DNA having migrated this far, you might say, okay, is this one DNA strand? Is that one DNA strand that I'm looking at? And just going back to the measurements, no, that is many DNAs that you're looking at. They're not all stretched out like that. Remember, even something that is 5,000 base pairs long is only going to be 1 to 2 micrometers if you stretch it out. So you wouldn't even be able to see it. It's a thousandth of a millimeter. You wouldn't even be able to see it. So this is many molecules of DNA is migrating that far. And they would have to be that small to be able to migrate through that polysaccharide gel. Now, the last thing you're probably saying is, okay, wait, but how am I even seeing it? Okay,
all right, sorry, I'll be right back. Need to take an idle meeting. Oh, hot mic, hot mic. Make sure that I mute because I don't know what my roommates are saying. I'm just gonna err on the side of safety. I'll be right back though. Oh, I just realized I was muted. Evil Tostitos have arrived. Because these are green, they're like lime flavored. They're very bad for you. Doesn't even matter. I shouldn't even say anything stuff like it's bad for your health because obviously, duh, chips in it, or no, crisp in it. I'm lacking my British. I haven't played Xenoblade in like 3 months. Has it been 3 months? Maybe 4. It's been a while. Damn, it's December. I forgot about that. Yeah, I haven't played in a while because my my Joy-Con have incredible drift. Also salsa. That's not green screen. There we go. My chroma key doesn't care about salsa. Okay. Hold on. Not gonna be eating a full meal until 4 a.m. It's my breakfast. Did want to eat some fun food. In the meantime. And don't worry, I'll start exercising for once in my life. Throughout this week. Not only offset what I'm doing here, but Just like as a general, keep myself in the zone kind of habit to prepare myself for the MCAT. I do like beans, actually. The problem being, when you eat too many beans, you get super gassy, and I don't like that. But I love Mexican cuisine. It's like probably my second favorite. Regional cuisine, like cuisine defined by its region. Second to like Thai food. I love Thai food with a burning passion and way too much bias. It's ridiculous.
This DNA, especially if they're these super, super small molecules. And the answer is you put some type of marker on the DNA that will make them visible, some type of dye or something that might become fluorescent. And one of the typical things that people often use is ethidium bromide. And ethidium bromide is called an intercalating agent. And it's a molecule, you can see the ethidium right over here. These are two backbones of DNA. You can see the base pairs bonding here. And then this right over here, that is ethidium that has fitted.
Ethidium bromide. I'm pretty sure typing with one hand is like equally as fast as my usual typing with two. Just because typing with my left is just like me using two fingers. Whereas I'm using like my entire hand with my right. I have really bad typing habits. Developed over years.
Itself. That's why we call it intercalating. It has fit itself in between the rungs of the ladder. And when it does so inside of DNA, it actually becomes fluorescent when you apply UV light to it. So if you put this ethidium bromide into all of your DNA right over here, and then as it migrates, and then if you were trying to turn on a UV light, it would become fluorescent, and you would actually see these things. And so if you wanted to see what it actually would look like, well, this is what it would look like when you were to, if you were
Zane, do you just want me to play Among Us live on stream or something? I'll do it.
And it would come up with standardized measurements. Maybe that's our 5,000 base pairs. This right over here is our 1,500 base pairs, and this right over here is our 500 base pairs. And then
I'll just be like,
wait,
explain to me how gel electrophoresis works. And if you can't explain it, then you're sus. Everyone will, and everyone clapped. Everyone agreed.
So now you know what's actually going on here. This isn't a strand of DNA. This is a bunch of DNA that has been tagged with some type of a dye or ethidium bromide or something like that. And it's a bunch of those molecules, and they've migrated based on the charge. They're trying to get away from the negative charge to the positive charge.
Or I can play Fall Guys.
Right over here are able to get further because they're able to get.
Although I don't have that game.
Gel.
Well, actually, I only have Among Us Mobile, which is even worse. You really need voice chat in that game. Else it's just not as fun.
I'm here with Emily, our biology content fellow, to talk about PCR, or polymerase chain reaction, which you've actually done a lot of. Why have you done PCR?
PCR was kind of the mainstay of my graduate project, where I built all sorts of different recombinant DNA molecules and used them to learn things about plants.
And so what does PCR in particular do?
PCR basically makes you a lot of copies of a particular fragment of DNA that you're interested in.
And so how does that, like, why would you need to make a lot of copies of a particular fragment of DNA?
So you might want to be making lots of copies so that you can clone it into a plasmid and then do some other experiments with it. That's a big use.
So when we talked about cloning, and we're talking about sticking.
Here we go, boys. PCR. This is the famous one. This is the one that everyone does in their little laboratories. Trying to be like, yo, we found something out. We did some crazy shit. We discovered CRISPR. Hey. Heyo. So yeah, here we go. PCR. Beautiful reaction. Very important to progression of science. I almost sound sarcastic. I'm not. I'm just don't really have I feel like it's just it speaks for itself. Don't really need to hype it up.
Taking a fragment of DNA inside of a plasmid, it's not like you're just taking one fragment into one plasmid. You're doing that with many, so you need a lot of fragments of DNA.
Exactly. That is exactly it.
And you might start with a very small sample of DNA, and so you just really need to, where else would you have to do PCR?
PCR is used a lot in forensics. It's also used a lot in medical diagnostics. So this could actually be your DNA that was being checked to see if you have a gene that would predispose you to a particular condition. All sorts of really practical applications.
Because it's hard to identify just one fragment of that gene. So you want to make copies, or as they say, amplify it, so that you can write it in gels and stuff and see how all of those molecules, how big they are, or something like that.
Exactly. If you were just looking in your DNA pulled out,
Sorry,
that would be really
my
interesting.
sinuses.
You can really zoom in and look at just the thing you need to see.
Okay, so you've drawn some diagrams here, and I actually have never done PCR, but you have. So I'm going to tell you how I understand it happening, and you tell me if this makes sense. So what you drew over here, this is double-stranded DNA, just and this could have been from a sample of someone's hair or whatever else. And let's say we want to replicate or make many copies of a fragment of this. And so let's say the fragment that we really care about is the fragment roughly from there to there. This part is what we want to make. We want to make multiple copies of. And so this first step, denaturation.
It's a weird word.
It's a weird word. You have 96 degrees Celsius, so this is almost at the boiling point, so it's quite hot, and that separates the two strands.
Precisely.
And so once they're separated, then you can cool things down, although this still isn't that cool. 55 degrees Celsius would be very uncomfortable. But you would cool it down to this, and then these primers show up. And so, one thing to remind ourselves is this process is happening inside a test tube or in a big solution. So you heat it up, the DNA, the two strands separate, and you just have this primer lying around.
So the primer is something that you've ordered from a company, and you've ordered a lot of it. So you put in a ton of primer in your reaction, so that there's a really good chance that when you get to this step here called annealing, that a primer is going to bind to many of your pieces of DNA.
So this is our solution. Is this all happening in water?
Water with some salts and stuff floating around, yeah.
Okay, so we have our solution right over here. You put whatever your initial DNA sample is in there, and once again, it's a very small amount. You put a lot of that primer, so you want to put that in a lot of surplus when you do this magenta color. You obviously wouldn't see it in real life, you just all dissolve.
It would just look like a drop of liquid.
It would look like, but for visualization, you put a lot of primer, and so you heat it up, the DNA strands separate, and then when you cool it back down, this primer is going to be specific to the ends of the region that you want to copy.
Exactly.
And so when you order online or wherever that you want a certain primer, you're going to pick the sequence of that primer to be specific to the region you want to copy.
Exactly, that's super important.
Okay, and so when you cool it back down, the primer attaches, and then you heat it back up, not quite to the 96 degrees Celsius, but to the 72 degrees Celsius, where you extend those. And I'm assuming, since it's called polymerase chain reaction, that this is where the polymerase is involved.
That is exactly where the polymerase comes in.
So the polymerase is what is actually extending this. And is it?
There's some seriously epic music going on right now while I'm just letting these two explain how PCR works.
As you were mentioning, even the cool step of this process is not something that your body would want to be hanging out in. So the polymerase is actually from a really heat-tolerant microorganism.
And what is that? It's called a Taq polymerase.
Thermophilus aquaticus, I think, makes quite a mouthful.
And they found it at heated vents. This organism that is able to stand these high temperatures. But that I guess leads to another question: why do you have to heat it up to begin with? I guess just to separate the two strands.
That's really the key reason. You just have to get them apart. You don't have an enzyme to do it the way you might in a cell, so heat does the trick.
Okay, so I get it. So this is one step. I'm getting at least the polymerase part of the PCR, where you heat it up, the strands separate, then you have all of this extra primer there. The primer, because there's so much primer, the primer is much more likely to bind to at least at this part of the sequence than for these two strands to get back together at this point. And then you have the polymerase, the Taq polymerase in particular, and you would have added that at the beginning. The Taq polymerase, I'll do it in a yellow color. So you would also put all that Taq polymerase in there. And once again, these things aren't robots. They don't know exactly what they do. They just bump into things in the right way and react in the right way. And then you also have to add a bunch of nucleotides.
Yes, absolutely. Your reaction is not going to work if you forget the nucleotides.
So the Taq polymerase, when you heat it back up again after the primers have been attached, is when you start adding all of these nucleotides. And what, do you just wait a certain amount of time? Will it just keep going on forever?
It'll keep going on for a while. Usually you do pick the length of that step to match how much time you expect the polymerase to need to complete your fragment. But it kind of will stop. Either it'll fall off. Or it'll stop when you go on to the next step.
Okay, so I get this is so far. So far we have, after one cycle, you've written here, after one cycle we would have doubled at least that part of the sequence that we care about. Although we might have copied even beyond that sequence. So where's the chain reaction come into this?
So I guess you can interpret chain reaction in two ways, and one is that's sort of what the polymerase does, is it adds things to make a chain. But there's actually even more of a chain reaction dimension here, and that's that we're actually getting this kind of exponential process going on.
So you do it one cycle, you get to this situation right here. You heat it up, the strands separate, you cool it down, the primers attach, then you heat it up again, the Taq polymerase does its job. And like all polymerase, it goes from the 5' to the 3' direction. We talked about that in the application. So now you have two strands, but now since all that stuff is in that solution, you can heat it up again. Now these two strands can turn into, or these two double strands can now turn into 4 single strands. Then you can cool it down again. Now they get primers attached to them, and then it's still the same primer because we still care about the same sequence. And then that can keep, and so now you go from 1 to 2 to 4, and so you keep repeating this. So how many times would it be typical for you to repeat the cycle?
So like 35 might be a pretty typical number of cycles to do. Depends a little what you're doing. You're going to do it a lot of times.
And so if you do this 35 times, I mean each time you're multiplying by 2, so it's going to be 2 to the 35th power, which is well over 1 billion times. So and how long would that take? You've done that.
Depends on the length of your fragment, but usually like 2 to 3 hours.
So in 2 to 3 hours, you can start with one fragment and get into the billions.
If it's perfectly efficient, which I wish it always were, but you usually get quite a few pieces made.
And one thing that I've always wondered when I first learned about this, and I'd like to go to the lab and do this with you, is is okay. I get that you have your primer and then the polymerase is just going to extend it like that. But I was like, well, it's going to be, how does it know where to stop? And you explain, well, on that first pass, it might not know where to stop. But then when you start going in the other direction, it's going to, so over here, and when it goes in the other direction, it's not going to have anything else to copy.
Exactly.
And so then so most of the billions of molecules that you produce are going to be both ends, kind of a nice clean cut.
The vast majority. Exactly.
Fascinating.
Makes sense. Pretty big brain, not gonna lie. Pretty big brain, not gonna lie. I can just say that about every single thing that we're gonna learn today. Pretty big brain of them to figure that out. Not gonna lie, man. That's pretty gamer of them. That's a pro gamer move right there. Why is Kirby here?
Alright, so let's say that you've got this little guy over here, and he's got his shoes, and just happy smile. So this guy right here is our protein. So let's look at how this protein was created. So in order to make protein, we have to start out with our base, and in this case, our base is DNA.
It's Kirby.
DNA,
It's not something else.
RNA, and then that messenger RNA eventually leads to
I know what you're thinking. You weebs.
happy guy over here.
You
This is pretty straightforward,
weebs.
but what if we wanted to go in reverse? What if we started out with the protein, and we wanted to figure out what its DNA sequence was, so if we wanted to go in this direction. So let's look at how this is done. Now, scientists thought that it would be nice to basically be able to type in the name of any protein that they're interested in, and automatically...
It's not like my channel really
Now, that is known
gives
as
the impression that I'm a huge weeb though. Because I really don't play that many
So let's look at how this
anime
So we'll
games.
start out with our protein.
Except for
And
Cell Glow.
This chain of amino acids. So amino acids basically are formed from messenger RNA. So if we know the amino acid sequence of our protein, we know what the messenger RNA sequence is based on the codon table that we all are too familiar with. So if we have the messenger RNA sequence, what we do is we add an enzyme known as reverse transcriptase. Reverse transcriptase. And when we add reverse transcriptase, basically takes this messenger RNA and makes a complementary DNA sequence to the messenger RNA, and that's known as cDNA. The C stands for complementary. So complementary DNA, one thing to keep in mind, is single-stranded DNA. So normally DNA in our cells is double-stranded DNA, but complementary DNA is single-stranded. So in order to generate double-stranded DNA, we need to add another enzyme known as DNA polymerase. DNA polymerase. And the DNA polymerase basically generates double-stranded DNA. So this is basically step one of the process of creating a DNA. Step one. And now let's look at step two. So now that we have our double-stranded DNA, what we need to do is sequence it. So in order to sequence it, we'll start out with our double-stranded DNA, and we'll basically inject it into some sort of cloning vector, such as a plasmid or a virus cloning vector. And that cloning vector can then be, and then you can take that cloning vector and add it to some bacteria, and it'll basically infect the bacteria, and the bacteria will basically produce lots and lots of this DNA, this double-stranded DNA. So that's a process known as amplification. And once we have lots of double-stranded DNA, we'll go and sequence that double-stranded DNA. And basically, once we have that sequence, we'll put the sequence into a large database that's readily accessible online, and that database will basically populate the DNA library. So now anybody that is interested in the
Okay. DNA library. Oops, wrong way. How do I summarize this? Without typing the entire thing. Right. cDNA, which with a DNA polymerizer, or DNA polymerase, I mean, polymerizer. Sounds like Yu-Gi-Oh all over again. Inject into cloning vector. Application. And then you get your sequence. Done. All right. Very nice.
The DNA sequence of a particular protein, you can just go into this library and pull up the genetic sequence of the protein of interest.
I will take this chip and eat it. Meanwhile, DNA cloning. All right.
Let's talk a little bit about DNA cloning, which is all about making identical copies of a piece of DNA. And usually it's a piece of DNA that codes for something we care about. It is a gene that will express itself as a protein that we think is useful in some way. Now, you might have also heard the term cloning in terms of the Clone Wars and Star Wars, or Dolly the sheep. And that is a related idea. If you're cloning an animal or an organism, like a sheep, well, then you are creating an animal that has the exact genetic material as the original animal. But when we
Dolly
talk about
hype.
DNA cloning, we're talking about something a little bit simpler. Although, as we'll see, it's still quite fascinating. It's identical copies of a piece of DNA. So how do we do that? Well, let's say that this is a strand of DNA right over here, and I'm just drawing it as a line, but this is a double-stranded, and I'll just write it down. This is double-stranded. I don't want to have to take the trouble of keep drawing the multiple strands. Actually, let me just try to draw the two strands, just so we remind ourselves. So there we go. This is the double-stranded DNA, and let's say that this part of this DNA has a gene that we want to clone. We want to make copies of this right over here. So gene to clone, gene to clone. Well, the first thing we want to do is we want to cut this gene out somehow. And the way we do that is using restriction enzymes. And there's a bunch of different restriction enzymes, and I personally find it fascinating that we as a civilization have gotten to the point that we can find and identify these enzymes, and we know at what points of DNA that they can cut, they recognize specific sequences, and then we can figure out, well, which restriction enzyme should we use to cut out different pieces of DNA, but we have gotten to that point as a civilization. So we use restriction enzymes. We might use one restriction enzyme, different color here, that latches on right over here and identifies the genetic sequence right over here and cuts right in the right place. So that might be a restriction enzyme right over there, and then you might use another restriction enzyme that identifies with the sequence at the other side that we want to cut. So let me label these. Those things right over there, those are restriction enzymes. Restriction enzymes. And so now you would have, after you apply the restriction enzymes, you will have just that gene. You might have a little bit left over on either side, but essentially you have cut out the gene. You've used restriction enzymes to cut out your gene, and then what you want to do is you want to paste it into what we'll call a plasmid. And a plasmid is a piece of genetic material that sits outside of chromosomes, but that can reproduce along, or that could replicate along with the machinery of the genetic machinery of the organism, or it can even express itself just like the genes of the organism that are in the chromosomes express themselves. So this is where we cut. Let me write this. We cut out the gene, and then we want to paste it. Then we want to paste it into a plasmid. And plasmids tend to be circular DNA, so we will paste it into a plasmid. And in order for them to fit, there's oftentimes these overhangs over here. So you might have an overhang over there, you might have an overhang over there. And so the plasmid that we're placing in might have complementary base pairs over the overhangs, which will allow it will become easier for them to react with each other if they have these overhangs. So we're pasting it into the plasmid. And this is amazing because obviously DNA, this isn't something we can manipulate with our hands the way that we would copy and paste things with tape. You're making these solutions and you're applying restriction enzymes. The restriction enzymes are just in mass cutting these things. They're bumping in just the right way to cause this reaction to happen. Then you're taking those genes and then you're putting them with the plasmids that happen to have the right sequences at their ends so that they match up. And then you also put in a bunch of DNA ligase to connect the backbones right over here. And we also saw DNA ligase when we studied replication. So that is DNA ligase, which you can think of it as helping to do the pasting. And so now we have this plasmid, and we want to insert it into an organism that can make the copies for us. And an organism that's typically used is, or a type of organism is bacteria, and E. coli in particular. And so what we could do is, let's say that we have let's say you have a vial right over here. You have a vial, and it has a solution in it with a bunch of E. coli. You actually wouldn't be able to see it visually, but there's E. coli in that solution, and then you would put your plasmid, which would be even harder to see, in that solution, and somehow we want the E. coli, we want the bacteria to take up the plasmid. And the technique that's typically done is giving some type of a shock to the system that makes the bacteria take up the plasmids. And the typical shock is a heat shock, and this isn't fully understood how the heat shock works, but it does, and so people have been using this for some time. So if you have a bacteria, right over here. It has its existing DNA.
I bet some random scientist was just like, what if we suddenly increase the temperature?
You put it in the presence of our plasmid.
What would happen?
You put it in the presence of our plasmid, and you apply the heat shock.
Whoa.
Some of that bacteria is going to take in the plasmid. It's going to take in the plasmid. And so just like that, it's going to take it in. And so what you then do is you place the solution that has your bacteria, some of which will have taken up the plasmid, and you put it, and then you try to grow the bacteria on a plate. So let me draw that. So here we have a plate to grow our bacteria on. And it has nutrients right over here that bacteria can grow on. It has nutrients. it has nutrients. And so you could say, okay, we'll put this here, and then a bunch of bacteria will just grow. So you would see things like this, which would be many, 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 many cells of bacteria. There would be colonies of bacteria. You could just let them grow. But there's a problem here, because I mentioned some of the bacteria will take up the plasmids and some won't. And so you won't know, hey, when this bacteria, when it keeps replicating, it might form one of these. It might form one of these colonies. So this is a colony that you like. So this one is a good colony. Put a check mark.
You guys ready for the midnight frappuccino? I'm not. I'm concerned about what this stream will do to my body. Luckily, we're learning biology, so I'll learn exactly what happens. It's another 2.5 hours until Norman Reedus storms my stream and takes my laptop hostage. There we go. We need more space. Space. I see the problem. I need more spaces here. So that it doesn't like clip everything thinking that it's one word. What in the world? Yo. This, okay. About to say this bottle knows that I'm not supposed to be drinking it at this time. Not sponsored by Starbucks. I wish I was, though. Okay. Is that gonna be my new YouTube catchphrase? Oh boy. Sigh. I mean, it's hard to keep on chatting when I'm just doing work, boys. I'm just working hard. And putting in all of the inputs into my map. My big, glorious map. Which I'll probably, like, look at in a week's time and be like, I can't understand how what this is. I can't understand how this works. This is ridiculous.
There, but maybe this colony is formed by a initial bacteria or set of bacteria that did not take up the plasmid, so it won't contain the actual gene in question, so you don't want that one. So how do you select for the bacteria that actually took up the plasmid? Well, what you do is, besides the gene that you care about, that you want to make copies of, you also place a gene for antibiotic resistance in your plasmid. So now you have a gene for antibiotic resistance here. And so only the bacteria, and I think it's amazing that we as humanity are able to do these types of things, but now only the bacteria that have taken up the plasmid will have that antibiotic resistance. And so what you do is, in your nutrients, you do nutrients plus antibiotics, plus an antibiotic. And so this one will survive because it has that resistance, it has that gene that allows it to not be susceptible to the antibiotics, but these are not going to survive. They're not even going to happen. They're not even going to grow because there's antibiotics mixed in with those nutrients. And so this is a pretty cool thing. You started with the gene that you cared about, you cut and pasted it into our plasmid. Let me write the labels down. Into our plasmid. That also contained a gene that gave antibiotic resistance to any bacteria that takes up the plasmid. You put these plasmids in the presence of the bacteria. You provide some type of a shock, maybe a heat shock, so that some of the bacteria takes it up. And then the bacteria starts reproducing. And as it reproduces, it also is reproducing the plasmids. And because it has this antibiotic resistance, it is going to grow on this nutrient antibiotic mixture, and the other bacteria that did not take up the plasmid are not going to grow. And so just like that, you can take this colony right over here and put it into another solution, or continue to grow it, and you will have multiple copies of that gene that are inside of that bacteria. Now the next question, and I'm oversimplifying things fairly dramatically, is, well, how do you now have a bunch of bacteria that have a bunch of copies of that gene? How do you make use of it? Well, the bacteria themselves, let's say that gene is for something you want to manufacture, say insulin for diabetics. Well, you can actually use that bacteria's machinery, use its reproductive machinery to keep replicating the genetic information. But you can also use its productive machinery, I guess you could say. It's going to express its existing DNA, but it can also express the genes that are on the plasmid. In fact, that's what gives that's what would give the bacteria its antibiotic resistance. Because if this gene was, say, for insulin, well, then the bacteria will produce a bunch of insulin molecules, which you might be able to use in some way. And I'm not going to go into all the details of how you will get the insulin out and how you can make use of it. But needless to say, it's pretty cool that we could even get to this point.
Oh my god, why are there so many DNA technologies we're going through? This is way too dank. Way too much, boys. Way too much. Hold to take this moment to point out that I am not interested in making these streams these streams are not designed for a particular demographic. I may say boys, but like I don't care who you are. You can just watch my streams. Doesn't really matter where you come from, what you're doing, all that stuff that you may be experiencing in life because you have different DNA from other people. Shouldn't matter here. We're all here to just chill. We're all humans at the end of the day. As we can see through this extremely long stream about mostly human biology. Well, I mean, this is general biology, DNA, you know, but I just have a lot of default phrases that I need to work out.
Alright, so in this video we're going to be talking about something known as DNA hybridization. DNA hybridization. Now, what is DNA hybridization? Well, basically what it... So let's work through an example to try and explain what DNA hybridization is. So let's imagine that we have two cells. So over here we have cell A, and over here we have cell B. Now let's imagine that cell A is a cancer cell. So this is a cancer cell, and cell B over here is a normal cell. This is normal. Now, cancer cells basically have the ability to proliferate and grow and metastasize and move throughout the body. So basically they have this unregulated cell growth. And the reason that the cell growth is unregulated is because there are various mutations that cause changes in the proteins that are expressed and changes in the regulation of the cell cycle. And there are hundreds and hundreds of different mutations, hundreds of different proteins that can be affected, and all of them can lead to cancer. Now, it's producing different proteins in different amounts. Now, what are kind of the two options that we have for certain genes? So let's imagine that we have gene A over here. So if this is gene A, what are the two options? Either gene A can be upregulated or it can be downregulated. So if it's upregulated, then what we have is we have the gene products, which is mRNA and eventually protein. We have a lot more of the mRNA and the protein that gene A encodes for. And what that basically means is that let's imagine that gene A encodes for a protein that will induce cellular proliferation and will allow that cell to go and metastasize throughout the body. Well, if we have a lot more of gene A being expressed, either because the promoter is upregulated or for whatever reason, now we have lots and lots of this protein that basically allows cellular proliferation to occur. We have lots of this protein floating around the cell, and we have this cancer cell proliferating uncontrollably. So another option is if we have gene B. So if we have gene B could be downregulated. And that basically means that that gene B isn't producing its gene product. And what if that gene product were something that basically stops the cell from proliferating? Well, if we have less inhibition, then we basically have more proliferation. And the third option for any specific gene in a cancer cell, so let's say gene C, is that there's no change. So there's just no change. So what we want to do is use DNA hybridization technology in order to assay the gene transcription profiles of a cancer cell compared to a normal cell. And in order to do that, we need to do something known as a microarray. So a microarray. Now, what is a microarray? Well, array basically means that we're assaying a whole bunch of different things. In this case, we're assaying the transcription profiles of a bunch of different genes. And micro just means that it's small. So
Assaying.
So let's say we have a microarray.
Okay. DNA hybridization, utilizing microarrays. Pretty big brain, not gonna lie. It's kind of cool technology you got there. Shame if someone were to try to study it in a 24-hour stream.
Chip. So let's say that we've got this chip, and it's basically just this square, and this chip has a lot of different holes in it. So let's imagine that we've got just lots and lots of holes. So we have just hundreds of these holes. I'll just drop you for simplicity sake. So we have a bunch of these holes on the micro.
I remember I was trying to interview for a biotech company. Like, I was like, hey yo, what's a microarray? I was like, what the fuck are you guys talking about? Oh boy. I like biology, but yeah.
In this cancer cell, gene A is upregulated for whatever reason. The gene A is upregulated, it's being overtranscribed, and that means that there's lots and lots of the gene A mRNA floating around in the cell. So there's just a bunch of the gene A mRNA. And this is in comparison to the normal amount of gene A products, which might just be a few gene A mRNA. Now, what we can do is we can
I like it
and we can break it apart, and we can label the mRNA with a certain color.
in the frame, in the context of behavior. So like, that's why neuroscience is my interest. I mean, neuroscience, psychology. But I fail to see how I mean, I'm not gonna rag on stuff that like, clearly this is important and this will influence behavior, but like in the sense that it's people reacting to it existing, so you know.
decide, okay, is it a brighter yellow or is it a brighter blue? If it's a brighter yellow color, if you see mainly yellow, that means that you have a lot more of that specific gene's products being expressed in the cancer cell compared to the normal cell. Now let's imagine that we look at a downregulated gene. So if we look at a downregulated gene, let's just draw another well. So let's draw a well over here. Now if we look at a downregulated gene, we've got lots of the gene B mRNA, the complementary strands inside as well, and we're going to have very few of the gene B mRNA in the cancer cell and then a lot more in the normal cell. And once again, we'll label the gene B mRNA with a yellow fluorescent label, and over here, over there we're going to label it blue. So we'll label it this blue fluorescent label. And once again, we're going to lyse the cells and we're going to expose the intracellular contents to this well. And what we're going to have is we're going to have very few of the gene B products binding, and we're going to have a lot of the normal cell, of the gene B byproducts in the normal cell binding. So when you look at this well, it's going to pop up as mainly blue. And once again, the computer's going to read this and it's going to notice, oh, well, this well has mainly a blue fluorescent label, which means that the normal cell is expressing a normal amount and there's a lot less of that gene being expressed in the cancer cell. So this is kind of the idea of a microarray chip in assaying the gene expression profile in a cancer cell versus a normal cell. It's able to tell you whether a specific gene is upregulated or downregulated, and you're also able to see if a specific gene has no change. And if there's no change, then instead of seeing either a yellow or a blue dot, you would see something kind of in the middle. So maybe you'd see a green dot. And that's basically a quick way in order to look at a whole bunch of different genes on a single chip and try and quickly determine which gene is upregulated or downregulated in the cancer cell compared to a normal cell. And this can help you tailor your therapy. So let's say that you know that this well right here is for a specific protein and you have a drug that's able to target that protein. Well, now you're able to tailor your therapy for that individual patient using this microarray technology.
Okay, makes sense. Oh my god, how many? Alright, we're almost done here. We're almost done with genes.
Normally when we think about cloning, we think
My god.
in this sense. So let's say that we got a baby and it's just so cute that we want two of them. So we can go and clone a baby. Well, this isn't exactly what we're talking about when we talk about cloning in scientific sense.
Oh my god.
What that means.
He's so quiet.
Let's say that and there's a certain gene, I'll call it gene A for now, and I want to clone that gene. So how do I go about cloning the gene A? Well, the first step is to isolate the messenger RNA from the gene of interest. So if we have the messenger RNA, what we need to do is we need to convert it into DNA. So if we add reverse transcriptase, which is an enzyme that will go and actually create DNA from RNA, we'll end up with something known as complementary DNA. Now, complementary DNA, the C stands for, basically the complementary DNA sequence to the mRNA sequence. Now, keep in mind that the complementary DNA only contains exons, so the introns have already been sliced out. And we talked about introns and exons in the earlier video. So next, what we have to do is we take this cDNA, this complementary DNA, and now what we want to do is we want to amplify it. We want to create lots and lots of the complementary DNA so that we can have plenty of cDNA to work with. So in order to do that, we have to take this complementary cDNA and transform it into a plasmid. Now, what does transform mean? Well, what it basically means is that we're taking the complementary DNA and we're basically...
I really wish my setup had better ergonomics. My chair is absolutely destroying me right now. I don't know how I'm going to be able to sit in this for much longer.
So now that we have this plasmid, we want to actually infect bacteria with the plasmid, so we're going to put it into bacteria. And once it's in the bacteria, what we want to do is we want to actually add antibiotics to the culture of bacteria. So we're going to add antibiotics, and since the plasmid contains antibiotic-resistant genes, the bacteria that were successfully transfected will survive, and all the other ones that didn't have the plasmid inserted will die. So now we've got a pure culture of bacteria that all contain the plasmid, which contains the gene of interest, the DNA of interest. And now the bacteria just does the same, so it's just going to start to replicate. The bacteria is going to replicate, and as it's replicating, it's going to produce lots and lots of the gene of interest. It's going to produce lots of the RNA of interest, and basically what we just did through this process is we just cloned the gene of interest. And that's what we're talking about when we're talking about cloning, at least from a scientific point of view.
Didn't we already cover this? Was that not like DNA libraries combined with DNA cloning? I'm confused. I mean, I guess the end result is mRNA, but that was the same thing. Basically the same thing. I don't... Okay, whatever. Cool. Cool. Cool concept, bro. Very epic. Appreciated.
So in this video, I'm going to be talking about something known as a southern blot.
Whoops.
So a southern blot basically allows you to visualize a specific piece of DNA that you're interested in. So let's imagine that we have a cup and it's filled with DNA. So it's got just a whole bunch of DNA inside. And there's just lots and lots of the DNA. And let's imagine that I'm specifically interested in one piece. So let's imagine that I'm interested in gene A. And I want to see if gene A is inside of this cup, if it's inside of this long piece of DNA. Now, in order to figure out whether or not gene A is inside this cup, basically we have to do this process known as a southern blot. And we'll break it up into a couple of different steps. So step one, what we're going to do is we're going to take this DNA and we're going to cleave it. So take the DNA and cleave it. So let me draw that out. So we're going to take this big old strand, we're going to remove it outside of the cup over here. So we've got the big strand, and we're going to cut it up, we're going to expose it to enzymes that will basically cleave the DNA in a whole bunch of different parts. And that will result in lots of these smaller pieces of DNA. And so that's basically the first step. So we've got a bunch of small little pieces of DNA. Now, step two, what do we do? Well, what we're going to do is we're going to take all these tiny little DNA fragments, and we're going to run them on a gel. So specifically, we're going to do a gel electrophoresis.
Oh shoot. Okay.
And I made a video on gel electrophoresis. If you want to refresh, you can watch that video. But basically, the gel electrophoresis will help us separate these DNA fragments based on size and based on charge. So let's just diagram that out. So we're going to take these DNA fragments and we're going to run them on a gel. So let's imagine that this is the gel, and we add the DNA fragments in different wells. So the fragments are going to move down the gel, and they're going to basically be separated based on size and based on charge. So we're going to have these fragments separated like so. So now we've got this gel, and we've got the DNA fragments separated by size on this gel. So the next step, step number 3, is basically we're going to take this gel and we're going to transfer it to a filter. So transfer the gel onto a filter. And what the filter will basically allow us to do is it allows us to visualize, because this gel is very flimsy, so we want to transfer it onto a filter. So what we'll do is we'll take a filter that's basically the same size as the gel, and we're going to basically just put it right on top of the gel for a little bit, and the fragments will basically transfer onto the filter. So now we're going to have a filter with these fragments, and the filter is a lot sturdier than the gel. So this is the filter, and we'll just write that down over here, and it's sturdier than the gel. Okay, so the next step, step number 4, is we're going to take the filter and we're going to expose it to a radio-labeled D piece of DNA. So expose to radio-labeled DNA. Now, this radio-labeled DNA is going to be the complement to our gene of interest. So we're interested in finding out if gene A is present in this massive DNA over here. So what we do is we're going to take the complementary sequence to gene A and radio-label it and expose it to this filter. So let's imagine that the radio-labeled piece of DNA is this pink
Filter?
And let's imagine that we do have gene A.
Whoa, hold on. This dude is talking about a filter, but I've never even heard of a filter in gel electrophoresis. Filter. Okay, so wait, what? Filter. We're gonna put radio-labeled DNA into the filter through gel electrophoresis. So maybe I'll just put the DNA here. Oh, so now it moves over the arrow to the closest point of connection. Okay. I see how it is. I don't like how it is, but this is fine. This is perfectly fine.
Expose the radio-labeled DNA to this filter paper, it's going to anneal to our gene of interest. So we're going to have this radio-labeled piece of DNA stuck to this DNA fragment, which is its complement. So in order to visualize this radio-labeled piece of DNA,
I don't know what's going on, dude.
to an X-ray film in order to
This is crazy.
To X-ray. And the X-ray basically will shoot a bunch of X-rays, and since this piece of DNA is radio-labeled, it will pop up on the X-ray film. So we're going to have a film.
Wait, what?
That film over here, so we'll have this film, and basically the...
It's not rather, the radiation the probe itself exposes the film, resulting in a signal where the probe was bound. So the probe is... Okay, so there's a radioactive probe that is attached to a specific DNA fragment, and then that's what radio-labeled DNA is, I guess. And then so we are looking for the base pair length of that, I guess.
Basically, the only thing that will pop up is this fragment over here, and that fragment will have a control, and we'll be able to say, okay, well, since we have this fragment, it's basically the radio-labeled piece of DNA, and since we see the radio-labeled DNA, it means that it was bound to this gene A, which means that gene A was in this cup of DNA. Have you ever wondered how we sequence DNA? Well, let's just take a quick look at DNA sequencing. So we're going to break down DNA sequencing into 3 different steps. So the first step is you take the sample of DNA that you're interested in sequencing, and you basically use PCR to amplify the sample. So by using PCR in order to amplify the sample, you're able to generate lots and lots of DNA fragments. So the next thing that you do is normally in PCR you have to add the growing strand, the substrate from which it can grow. So
remove this. I'm gonna put DNA sequencing up here.
You got an OH group here, you got an H group here, you have a base, and you got carbon group, oxygen, hydrogen. So this is what a normal nucleotide looks like. Interspersed in the PCR, what you also want to add is you want to add in something known as a dideoxynucleotide. So a dideoxynucleotide looks something like this. It's basically exactly the same thing, but it only has a hydrogen here. So this oxygen is removed. And what that basically does is if this dideoxynucleotide, we can abbreviate DENTP, if this incorporates into the growing strand. Since there's no oxygen group here, the strand can no longer elongate. So you basically have termination of strand elongation as soon as this DDNTP incorporates. So what you can do is you can actually fluorescently label the different dideoxynucleotides. So for example, we have 4 different options. So we can label all the G's blue, we can label all the A's red, all the T's green, and all the C's orange. And so basically what you have is you have these dideoxynucleotides with different fluorescent labels getting incorporated into the growing strand. And since PCR is able to amplify and create millions and millions of DNA fragments, you can basically, what you can do is you'll have strands of different lengths. So let's just kind of look at an example. So let's imagine that we've got
Yeah, and then you line them up later. Yep.
another one, and then just randomly, all of a sudden, we have a dideoxynucleotide being incorporated here. And this would stop elongation of the strand. So you have a DNA strand, that's just 4 nucleotides long. And after another round of PCR, what we might have is 1, 2, 3, 4, 5, 6. It's just growing, and all of a sudden, whoa, what happened? You got a dideoxynucleotide being incorporated. And so basically you just do this, and after you've got millions of samples, you'll eventually be able to have something that looks like this. You'll have maybe just 1 regular nucleotide, and you've got a dideoxynucleotide incorporated, or you might have maybe, let's say, 2 of them, so you'll have 2, and then you've got so what you can basically do is you can see that you have strands and they're elongating, and different strands are terminated at different points by a dideoxynucleotide. And so basically the next step is you use gel electrophoresis in order to separate the strands by size. So when you run all the different fragments on a gel, it'll separate them by size. And then you can just have a computer go in and analyze all the fluorescent labels. So if it sees here that you've got this blue fluorescent light, then it knows that the second nucleotide in the sequence is a G. So it'll say G. And then it'll look here, and it'll say, okay, well, this is a C. It'll look here, it'll say we have another G, and so on and so forth. And basically the computer is able to, by reading these fluorescent labels, these fluorescent tags, it's able to give you a DNA sequence. And so this is basically an overview of how DNA sequencing works.
They might cover it later, but there's another technique that I'm aware of that seems a little bit more efficient. Or maybe, oh, they won't cover it in depth. I'll have to look at my notes from that class, because that's more advanced genetics.
Basically the process where a gene is used to synthesize some sort of product. So you go from a gene to a product. And
There's like a shotgun technique where you just like, you can just sequence a bunch of little, no, you can sequence a bunch of different DNA fragments and then line them up. And then it's just like, oh shit. Guess that works.
Now, how do we determine what the function of a gene is? How do we determine if a specific gene is exactly what?
Because what they were talking about was very sequential. Unless I'm thinking that technique plus something else, in which case, okay, maybe that's just a progression of ideas rather than a different idea completely. I don't know, I was playing Bloodborne when I was studying for that class, so right now my mind is just imagining that graveyard section in Bloodborne. Oh dear. This is why I can't, or I shouldn't study along with playing a game. Just, I mean, obviously you're not supposed to do that, but I am distinctly aware of why. Oh my god. Because now whenever I think of genetics, I just think Bloodborne, which is kind of appropriate considering its themes, I guess. Kind of. Not really. Kind of. Bloodborne, good game. Very good.
So if you know that there's this homologous sequence somewhere else in the genome and it goes for a specific protein, then you know that the function of that protein, then you know that the gene of interest might create a protein that has a similar function.
What?
So
what?
Well, it's basically the process where
what is this video?
to synthesize some sort of product.
Gene expression, yes, okay, and then it produce what?
So basically you go from a gene to a product.
Yes. Why is there milk? What is happening? What is going on?
There's a gene here, and we have digestion of milk. So this process is known as a knockout. We know that this gene didn't really have much to do with digesting the milk. But if we knock it out and the cell's no longer able to digest the milk, then we know that this gene A had something to do with the digestion of the milk. So this process is known as a knockout. So basically you're
knockout genes.
What the function is of the gene. So if you knock out a gene,
Okay.
so you basically create a knockout loop and study its effects.
Knockout genes important. They're not really telling me how they like make knockout genes or how they knock out genes. They're just talking about them. Okay.
And if you know what that homologous sequence does, then you have a pretty good idea of what that gene might do. So if you know that there's this homologous sequence somewhere else in the genome and it goes for a specific protein, then you know that the function of that protein a similar function. So what is gene expression? You know that and you know the function of that protein, then you know that the gene of interest might create a protein that has a similar function. So what is, there's this
Oh my god. Okay, sequence homologous. Homologous sequences. Okay, maybe I just need to put reverse genetics or something. So reverse genetics use knockout genes to find homologous sequences? Is that where we're going with this? I mean, sure, I guess. Why not?
So what are some applications of DNA technology? Applications, DNA technology. All right, well, let's first look at medicine. So what are some applications of DNA technology in medicine? Well, one of the two big things where recombinant DNA technology was first used was to create insulin and human growth hormone. Human growth hormone. So before the advent of recombinant DNA technology, insulin and growth hormone were really, really hard to manufacture. You basically had to isolate it from another human, purify it, and then give it to patients. But with recombinant DNA technology, you can basically just grow these proteins in E. coli. You can grow them and culture them in E. coli bacteria. So this really has changed the way that medicine is practiced, and it's really helped a whole bunch of people. So vaccines is another application of DNA technology. A while ago, vaccines were made by first denaturing the disease, and then after the disease had been weakened, they would inject it into a human, and they would hope that their immune system would be able to put up a fight against the weakened virus, and that way in the future, if they were infected with that virus, they would at least have some kind of immune response towards the virus. The problem with this was that the patient would sometimes get the disease because you're injecting a weak virus, but sometimes it wasn't weak enough. So with DNA technology, they can actually recreate the outer shell of the virus and inject that. So it's a lot more cost effective, and it doesn't have the risk of actually causing the disease in the host. So this is much safer, and it is cheaper, and it's very it produces a better immune response. And so some vaccines that we actually use recombinant DNA technology to create include the Hep B virus and Hep B virus and the herpes virus and malaria. So these are some applications of DNA technology in medicine. Another cool application
Yeah,
is in
vaccines.
solving crimes, so in forensics. So there are parts of the genome known as non-coding regions of the genome.
Dude, anti-vax movements are whack.
Forensic scientists identify specific individuals.
So whack.
So they can look at things like short tandem repeats, STRs, and these are basically short sequences of DNA, like 2 to 6 base pairs long, and they're normally found in really high amounts. They're just these short repeats that are found in really high amounts and to varying degrees between different individuals. So if they actually sequence these short tandem repeats, they can identify specific individuals given a DNA sample. They can also look at mitochondrial DNA. So mitochondrial DNA is inherited from your mother, and it's found in really high amounts within an individual cell. So even if there's very little sample available, forensic scientists can analyze mitochondrial DNA in order to identify a potential suspect. Another technology that is used in forensic science.
There's so much misinformation out there, man.
Looking at short tandem repeats found on the Y chromosome.
You know, the simplest shit.
DNA technology has helped scientists pick out individuals.
Sometimes it's the simplest shit.
On DNA samples that people
when you can hacks. I mean, true, I guess. Unfortunately, I'm not dialed into the Matrix. Unlike you, I presume, so I would not be able to hacks like that. Yeah, got him. Got him.
To create more crops to feed a growing population of individuals. And it also helps with the economy because then you've got farmers that are growing all their crops, and if there was some sort of bacteria or virus that destroyed their entire crop, then that farmer would not get paid for that season. So by transgenically modifying the crop so that it's resistant to specific things, then they're able to grow their crops, sell it, and feed individuals.
Yay. DNA tech. Very cool. Very awesome. Very useful. But what about ethics, though? Ooh, time to take an ethics class?
So DNA technologies are really cool, and they've provided us with a lot of really good things. However, there are some safety and ethical issues that surround DNA technology. So back in 1975, there was a conference on...
Well, it's not even stupidity necessarily, it's just,
can be particularly risky, and so they implemented study guidelines to try and minimize that risk. The NIH later issued formal guidelines for recombinant DNA work.
it's just really unfortunate, circumstances that kind of lead to, you know, a vulnerability to specific types of misinformation.
So that's one example of a safety concern using DNA technology. Another safety concern is how do you protect researchers that are working with recombinant DNA from being...
I mean, sure, you might be more biologically like vulnerable to misinformation too, but I highly doubt that that's the main issue we're facing as a globalized society. It's just more like there's so much information provided in different ways across different platforms, across different groups, or within different groups, within pockets of populations. So
What if an individual is perfectly normal genetically? Is it okay if we put in genes that help that make them smarter or faster? What are the ethics surrounding that? So those are some ethical issues. So that would be genetic modification. So another ethical concern that has been brought up is we're able to genetically...
for some, yeah,
So forensic scientists
yeah.
are able to synthesize DNA. They're able to figure out who an individual... They're able to figure out what individual left a DNA sample at a crime scene, for example. So we're getting better genetic fingerprinting. But what are the ethics around that? What if the government was able to track every single person that opened a certain door based on...
Or there's like a monetary or some other type of incentive to spread misinformation as well. There's that as well. But you know, you can tell when that's the case because usually they're like making entire movies about things that are like, I think everyone knows that that's not true, but like, why? They're banking off of interesting stories that people would typically think are not true, but then might kind of push certain ideas to the forefront that imply other things. It's a complicated mess. Indeed.
One criterion that we use to define biological life is that the organism must have the ability to reproduce. And the parent organism must have the ability to pass on to its offspring.
Wait, why are we still talking about DNA? No! We're still on DNA! Oh my god! It's still here! Excuse me, can I check how much left is there on Academy? Where are we on this now? How are we not done with genetics? What is going on? I didn't realize it was this long. Holy shit. Okay, population dynamics is different. Okay, there we go. Yeah. And then we'll get into metabolism soon. Okay, cool. I don't know what... Okay. Well, I mean, evolution is still part of genetics. But soon we'll get to Gibbs free energy. Oh my god. We're almost there, boys. And gals. And everyone.
Some sort of material that contains the instructions for living. And we call this material genetic material. What is genetic material? Well, today we know that it's DNA, or deoxyribonucleic acid, but this wasn't always so obvious. We're going to go through a couple of earlier experiments that help prove to us that DNA is genetic material. The first experiment we're going to discuss was one that was conducted by a scientist by the name of Friedrich Nietzsche. And Nietzsche lived from 1904
I thought they said Nietzsche, and I was like,
Nietzsche worked with cells that he took out of pus.
Hey, yo, God dead?
And lymphocytes happen to have
God dead?
across the cell nucleus might look something like that.
God dead, and we have killed him? Hey, yo.
And because the nuclei are so large, Nietzsche was able to isolate the material inside the nucleus.
Okay.
And he analyzed this material and came to the conclusion that it was made up of two components. The first was protein.
Oh my god, I can't add more branches, guys. This is ridiculous. I don't... Where do I even put this?
Nietzsche was the first one to isolate and identify
What?
nucleic acid.
What is even the plan? What is this? Oh my god.
From 1850 to 1924. And Roux worked with cells that were dividing, and he analyzed these cells. So I drew a cell here, and I drew a couple of organelles also. Let's just label them. We have the endoplasm.
Pretty sure I don't need to know biological history, though.
And singular mitochondria.
I need to know the researchers that
and I also drew some vesicles. And of course, in
found all these things. I don't know.
And we're going to say that this cell is undergoing mitosis right now, so I'm going to...
Alright, fine. I'll just include a section anyway. I think it's important to know these figures anyway. I'm just too lazy to write down their names sometimes. Just pretty sure I won't be tested on, like... You know... There won't be information like memory recall questions on these people. Just, I don't think that's gonna happen. But who knows? Shout out to Mishler. Shout out. Arigato, Mishra-san. Good shit, man.
(1844 to 1895). And Misha worked with cells that he took out of pus, and today we know that these are lymphocytes. And lymphocytes happen to have very large nuclei, so if that's the cell, the nucleus might look something like that. And of course, there's this material inside the nucleus. And because the nuclei are so large, Misha was able to isolate the material inside the nucleus. And he analyzed this material and came to the conclusion that it was made up of two components. The first was protein, and the second was this substance that acted like an acid, and so we called it nucleic acid. Nucleic because it came from the nucleus. And so Misha was the first one to isolate and identify nucleic acid. The next experiment we're going to talk about was one conducted by a scientist who lived at about the same time. His name was Wilhelm Roux, and he lived from 1850 to 1924. And Roux worked with cells that were dividing, and he analyzed these cells. So I drew a cell here, and I drew a couple of organelles also. Let's just label them. We have the endoplasmic reticulum here. We have a couple of mitochondria. In singular, it's mitochondrion. And then we have a couple of ribosomes. I'm just going to abbreviate it like that. And I also drew some vesicles. And of course, in the center, we have the nucleus. And we're going to say that this cell is undergoing mitosis right now, so I'm going to draw the chromosomes like that in the center. I'm going to draw 4 chromosomes. And I'm also going to draw the mitotic spindle, which will help to separate the chromosomes into the two cells.
Oh, did they take a picture of the...
And what Roux noticed was
Yo!
that
The X's are
organelles were not just
splitting!
in a very organized and even fashion. You can see the cell
Oh
on the left
my
got
god.
mitochondria, the cell on the right only got 2, the cell on the left got 3 ribosomes, the one on the right only got 2. But he noticed that the material in the nucleus divided in an extremely organized fashion, so each nucleus got exactly 4 chromosomes. And so Roux concluded that because the material in the nucleus was divided in a very orderly and even fashion, that must be the genetic material. And this makes sense because the genetic material or instructions for living is the most important thing that the parent cell has to pass on to the daughter cell. If one of the daughter cells didn't get enough mitochondria, well, that's not so terrible. As long as it has the correct instructions for living, it can make more mitochondria. If one of the cells accidentally didn't get a ribosome, well, if it has the right instructions for living, it can make ribosomes. So the conclusion here was that genetic material is made up of protein and or nucleic acid. This was the conclusion that Friedrich Miescher's experiment and Roux's experiment helped prove.
Huh. Oh yeah, the city does sound like this. Okay. For a second there, I thought that was Nier Automata. I was like, did this switch to my others playlist? Ah, okay, yeah. Yeah, the city does deserve to, like, have that kind of... rustic? This kind of music. Very nice. Feels lived in.
We spoke about the experiments of Friedrich Miescher and Wilhelm Roux, and how their experiments helped show that genetic material is made up of either protein or nucleic acid, or perhaps both. So the next logical question to ask is, well, which one is it? Is it protein, is it nucleic acid, or perhaps it's both? So this question was answered with the experiment of 2 scientists, Alfred Hershey and Martha Chase. And in their famous Hershey-Chase experiment that was published in 1952, they showed that it's nucleic acid that's genetic material and not protein. So Hershey and Chase worked with bacteriophages.
Are we sure we're not talking about
bacteriophage is
chocolate?
a virus that specifically infects bacterial cells, and it can also be referred to as
Chocolate Hershey's?
So
Oh
what is
shit.
bacteriophage? Well, it has nucleic acid, which can be either DNA or RNA. In this video, I'm just going to refer to all nucleic acid as DNA, but keep in mind that when I say that, it can also mean RNA, because in some viruses, nucleic acid is RNA. And that nucleic acid, or DNA, is surrounded by a protein coat. And how do bacteriophages infect bacterial cells? Well, they get kind of close to the bacterial cell and sort of sit on top of it and inject their DNA into the bacterial cell. Then their DNA gets integrated into the bacterial cell's DNA, which I'm going to draw right here. So here's the bacterial cell's DNA. So the virus's DNA gets somehow integrated, and now this bacterial cell is going to produce a whole bunch of viruses. So this is some background information. Now let's talk about Hershey and Chase's actual experiment. Hershey and Chase took some phages, and they put these phages in a medium, which means a broth that has a lot of nutrients, so that these phages can now multiply and reproduce and make a lot more of themselves. But they wanted to label the protein coat of the new generation of viruses, and how are they going to do that? So they made sure that all of the amino acids that were in this broth had in them radioactive sulfur. So they were labeled with sulfur 35, which is one of the radioactive isotopes of
Got
sulfur.
'em.
And in this way, they can kind of keep track of where the protein is going and what's happening with the protein. And the reason that they chose sulfur for this part of the experiment is because they wanted to label the protein in particular. And
Sulfur.
sulfur is found in amino acids, which
Whoa.
means proteins with sulfur is not found in DNA. So this is a good way to make sure that they label the protein but not the DNA. So now when this phage, or the phages that are put into the medium, reproduce, they're going to take nutrients from this broth and among those nutrients are amino acids, and they're going to incorporate those labeled amino acids into their protein coats. So they produce a generation of viruses that have radioactively labeled protein coats. And now they allow this generation of viruses to infect a bacterial cell. So you can see they climb on top of the bacterial cell and inject their DNA into the cell. So remember, their DNA gets incorporated into the DNA of the bacterial cell, and the bacterial cell will produce a whole bunch of viruses. Now take note that the original protein coats, of course, remain outside of the cell. And Hershey and Chase now wanted to separate the protein coats from the bacterial cells. So they centrifuge this mixture to kind of get rid of the protein coats. So let's get rid of them. And notice how the phages inside this bacterial cell, they just have a regular protein coat that's not radioactively labeled with S35. That's why they're drawn in green. And so now what they did was they took the bacterial cells, lysed them, which means they made them burst, and they analyzed the viruses. And they saw that the viruses were not at all radioactively labeled. So they concluded that the protein coat must have remained outside the cell, outside the bacterial
Oh
cell.
my god.
And if the protein coat remained outside of the bacterial cell, then it must be that that is not genetic material, because in order for this bacterial cell to produce viruses, it had to have contained the genetic material of the virus. And if the protein coat remained outside, it must be
What?
Let's talk about the
That's
second part. Of
crazy,
So
dude.
again, they took a phage or a couple of phages and put it in a medium with a lot of mutations so that it can reproduce and make lots of viruses. But this time, they wanted to label not the protein coat, but the nucleic acid inside. And again, I'm just going to refer to nucleic acid as DNA, but keep in mind that it can also be RNA. So they want to label the DNA, and how are they going to do that? So they made sure that all of the nucleotides that were in the broth, and the viruses are going to need nucleotides to make their DNA, so all of them were radioactively labeled with phosphorus 32. So P32 is radioactive isotope of phosphorus, and this is how they're going to label the DNA. And the reason they chose phosphorus 32 is because phosphorus is found in DNA nucleotides, but it is not found in amino acids, so it will not get integrated into the protein coat. So it's a great way to differentiate between the two. So they put them in the broth and allow them to reproduce, and they produce a generation of viruses that have this radioactively labeled DNA, which I drew in that magenta, and of course the protein coat isn't green because it's not labeled in any way. So they allow these viruses, these phages, to infect the bacterial cell. The protein coat remains outside, and it injects the DNA into
Oh
the
wait,
bacterial cell.
shoot. Is this Hershey and Chase as well? I wasn't paying attention.
And the protein coats remain outside. And now they're going to centrifuge this mixture of protein coats and bacterial cells because they want to get rid of the protein coats. So they spin it in a centrifuge, and the protein coats, which are less dense, will be spinning, and the bacterial cells, which are heavier, end up in the pellet. So we're going to get rid
What the
of
heck?
that.
I'm
So
supposed to
we're gonna
reset my sleep schedule by
Now,
the end of
Chase
this,
sliced
but
the
I'm already
viruses
resetting it
and they
now.
analyzed
This is
the viruses inside, and the viruses have a lot of nucleotides,
Ugh.
P32, and so there's a lot of radioactively labeled DNA. Maybe not all the DNA.
You know what?
radioactively labeled
Maybe I
phosphorus,
should
because
just
some of the nutrients came from the bacterial cell, but a fair number of the viruses have
make sure to
radioactively
study
labeled
right
DNA
before I
inside.
sleep so
And
that I
they
can
concluded
like make sure that I
since the
sleep
DNA
faster.
entered the cell, it must be DNA that's genetic material,
Ugh.
or it should really be more
It's not
specific,
even
it's nucleic
okay.
acid.
It's pretty
It's genetic
boring.
material.
It's
In
only
order for the
boring
bacterial
because
cells
it's
to produce viruses,
presented
it has to have
boringly.
genetic material. And since there's this radioactively
The
labeled
concepts are
DNA
cool.
in the viruses, they included that nucleic acid is genetic material. We mentioned that Hershey and Chase published the findings of their experiment in 1952.
Okay,
and
Hershey and
another
Chase did
writer
both of
in
those
1953
because they were like,
James
"Yo,
Watson
holy
and
shit,
published their thesis paper, which
bacteriophages!
actually
What?"
identified the structure of DNA or nucleic
Watson
acid.
and Crick,
So they were a very
but
tremendous amount of research that was happening during their time and before their time.
there's also
and they identified the
Rosalind
structure of DNA,
Franklin,
and they
so
told us
please
that
don't
DNA is
forget
a
her.
double-stranded helix with
Incredibly
a
based.
sugar phosphate backbone. The sugar in this case being the
Rosalind Franklin.
And then what you see on the
That's
inside
right.
kind of what looks like the rungs of a ladder. Those
Am
are
I
the
allowed to say
nucleotides.
that?
And there are 4 of them.
Am I allowed to say that about
thymine,
famous scientists?
guanine, and cytosine.
Oh, I don't know if I
And
am.
adenine and thymine pair up with each other, guanine and cytosine pair up with each other. So let's just recap the 4 experiments that we discussed. We spoke about Friedrich Miescher, and Miescher was the first one to isolate and identify nucleic acids. Then we spoke about Wilhelm Roux, and Roux's experiments helped show that it was the material in the nucleus that was genetic material, but still at that point, people weren't quite sure if it's protein or DNA, and many people thought it was protein because proteins are more complex than nucleic acids. Then we spoke about Hershey and Chase, and how they helped prove that it was nucleic acid that's genetic material and not protein. And then Watson and Crick actually identified the structure of nucleic acids.
Yeah, after stealing Rosalind Franklin's picture of the double helix, and they were like, "Oh my god, we were right the whole time," even though they had a triple helix in their original model. Oh, those silly dudes. Not that I dislike Watson and Crick. I'm just saying, it's so silly how they figured that out. They were like, "Yo, what? What is this? Look at this, Crick. Whoa. It's a circle with 2 things coming off of it, or going towards, it's like an X, I think. Anyway, they were like, yo, okay, that's a double helix. That makes sense. But not to discredit their work, because that was still, like, it still took some work to actually make the model, based on the little, you know, a picture is helpful, but it was limited, so, you know, they still had to do some legwork to figure out or to interpret it properly. So that's cool. Double helix. Watson and Crick. And Franklin. We didn't end up getting a Nobel Prize. Sad. Sad boy ours. Actually, I'm curious. Did she ever get a Nobel Prize? I think she might have gotten she might have gotten it posthumously because she died like pretty soon after Watson and Crick got theirs. Sad. Probably because she was working with so much radioactive stuff, though. I mean, it is what it is. Oh, it didn't. They didn't make. So Nobel Prizes can't be handed out posthumously, so that's too bad. Rest in peace.
By this point in the biology playlist, you're probably wondering a very natural question. How is gender determined in an organism? And it's not an obvious answer because throughout the animal kingdom, it's actually determined in different ways. In some creatures, especially some
this
types
is
of
supposed to be sex.
reptiles, it's environmental. Not all reptiles, but certain cases of it could be maybe the temperature in which the embryo develops will dictate whether it's male or female, or other environmental
We
factors.
are
and other types of animals,
epic
especially mammals, one example. It's genetic
gamers, and we
So your next question
can
is, hey, Sal,
understand
so
what
write this down. In
they're
mammals,
talking about without having to think too deeply on
It's
it.
genetic. So like, hey, maybe there are different alleles, a male or a female allele. Then like, hey, there's so many different characteristics that make that differentiate a man from a woman. Maybe it would have to be a whole set of genes that have to work together. And to some degree, your second answer would be more correct. So let me just draw it's even more than just a set of genes. It's actually whole chromosomes determine it. So let me draw a nucleus. That's going to be my nucleus, and this is going to be the nucleus for a man. So 22 of the pairs of chromosomes are just regular non-sex determining chromosomes. So I could just do, that's one of the homologous 2, 4, 6, 8, 10, 12, 14. I can just keep going, and you eventually have 22 pairs. So these 22 pairs right there, they're called autosomal. And those are just our standard pairs of chromosomes that code for different things. Each of these right here is a homologous pair. Homologous.
Okay, finally, we're talking about chromosomes, so I get to edit this.
They code for the same genes. If eye color is on this gene, it's also on that gene. And the other gene, the homologous pair,
How am I gonna do this?
on either one. That determines what you But these are just kind of the standard genes that have nothing to do with our gender. And then you have these two other special chromosomes. I'll do this one. It'll be a long brown one, and I'll do a short blue one. And the first thing you'll notice is that they don't look homologous. How could they code for the same thing when the blue one's short and the brown one's long? And that's true. They aren't homologous. And these we'll call our sex-determining chromosomes. Sex-determining chromosomes. And the long one right here, it's been the convention is to call that the X chromosome. Let me scroll down a little bit. And the blue one right there, for that as the Y chromosome. And to figure out whether something is a male or a female, it's a pretty simple system. If you got a Y chromosome, you are a male. So let me write that down. So this nucleus that I drew just here, obviously you have a whole broader cell all around here. This is the nucleus for a man. So if you have an X chromosome, and we'll talk about in a second why you can only get that from your mom.
Man assigned
a Y chromosome from your dad. Male. If you get an X
male
chromosome
assigned at
from your mom
birth.
and an X chromosome from your dad, you're going to be a female.
This gendered man or
but it kind of shows what all the different possibilities are. So let's say this is your mom's genotype for her sex-determining chromosome. She's got 2 X's. That's what makes her your mom and not your dad. And then your dad has an X and a Y. And a Y chromosome. I should do capital. And has a Y chromosome. We could do a Punnett square. What are all the different combinations of offspring? Well, your mom could give this X chromosome, and in conjunction with this X chromosome from your dad, this would produce a female. Your mom could give this other X chromosome with that X chromosome. That would be a female as well. Or, well, your mom's always going to be donating an X chromosome, and then your dad is going to donate either the X or the Y. So in this case, it'll be the Y chromosome. So these would be female, and those would be male, and it works out nicely that half are female and half are male. But a very interesting and somewhat ironic.
Sal's making all the mistakes today.
Whether the offspring are male or female. Is it the mom or the dad? Well, the mom always donates an X chromosome. So in no way does the mom...
I'm almost expecting like a message at the bottom just to say, Sal actually got this in.
By whether, let me just draw a bunch of dads got a lot of sperm.
Oh my god. Sal, please! Come on, man. I'm pretty sure this video is just outdated in terms of how we understand in terms of how we understand gender identity and sex assigned to birth. So
you have these. I mean, it's not just the case with kings. It's probably true.
We're epic
of
gamers.
our
We
civilizations
understand
as
what they're
well.
talking
And
about.
you have these men who are obsessed with producing a male heir to kind of take over the family name, and in the case of Henry VIII, take over a country. And they become very disappointed, and they tend to blame their wives when the wives keep producing females. But it's all their fault. Henry VIII.
Yeah, it's all their fault because they're the ones giving out the wives.
The general notion is that look, he
Bruh,
became upset with her that she
what
was
are you
producing a male heir,
doing?
and he found a reason to get her essentially decapitated, even though it was all his fault. His fault. He was maybe producing a lot more sperm that looked like that than was looking like this. He eventually does produce a male heir, so he was, and if we assume that it was his child, then obviously he was producing some of these, but for the most part it was all Henry VIII's fault. So that's why I say there's a little bit of irony here, that the people doing the blame are the people to blame for the lack of a male heir. Now, one question that might immediately pop up in your head is, Sal, is everything on these chromosomes related to just our, maybe sex-determining traits, or are there other stuff on them? So let me draw some chromosomes. So let's say that's an X chromosome, and this is a Y chromosome. Now the X chromosome, it does code for a lot more things, although it is kind of famously gene poor. It codes for on the order of 1,500 genes. And the Y chromosome is pretty, I mean, it's the most gene poor of all the chromosomes. It only codes for on the order of 78 genes. I just looked this up, and who knows exactly 78. What it tells you is it does very little other than determining what the gender is. And the way it determines that, it does have one gene on it called the SRY gene. You don't have to know that. SRY. That plays a role in the development of testes, or the male sexual organ. So if you have this around, this
Pretty cool.
gene right here can start coding for things that will eventually lead to the development of the testicles. And if you don't have that around, that won't happen. So you'll end up with a female. And I'm making gross oversimplifications here. But everything I've dealt with so far is this is okay.
Man, thank God it codes for the testes where P is stored. Oh, fantastic. Oh, man. I love how the meme of P is stored in the balls is single-handedly like perpetuated by med students that are just bored and making memes. It's just the simplest meme to make. Ah yes, where is P stored? The balls. Genius. And what if you don't have any? Wait a minute. Wait a minute. Hold up. That's impossible. Yay, okay, cool.
Clearly plays a role in determining sex, but you do have other traits on these genes. For example, and the famous cases all deal with specific disorders. So, for example, colorblindness.
Really interesting that like
I should say.
X is so much longer though,
cause
especially considering that
blindness, which I did.
If you have two X's, sometimes you get
colorblindness and also hemophilia. Hemophilia. This is an inability of your blood to clot. Actually, there's several types.
Sometimes you get some crazy,
both of these are mutations
like,
on the X chromosome. Mutations on the X chromosome.
co-dominant
and they're recessive mutations.
expressions.
So
Co-dominant and incomplete dominant expression, gene expressions.
Have to have the hemophilia mutation in order for you to show the phenotype of having hemophilia. So, for example, if there's a woman, and let's say this is her genotype, she has one regular X chromosome, and then she has one X chromosome that has the, I'll put a little superscript there for hemophilia. She has the hemophilia mutation. She's just going to be a carrier. Her phenotype right here, her phenotype is going to be no hemophilia. No hemophilia. She'll have no problem clotting her blood. The only way that a woman could be a hemophiliac is if she gets two versions of this, because this is a recessive mutation.
Yeah, that's why it's so
this individual will be
way less likely for it to happen.
Now,
yep.
They only have one X chromosome. So for a man to exhibit hemophilia, to have this phenotype, he just needs it only on the one X chromosome he has. So, and then the other one's a Y chromosome. So this man will have hemophilia. So a natural question should be arising is, hey, this guy, let's just say that this is a relatively infrequent mutation that arises on X chromosome. The question is, who's more likely to have hemophilia, a male or a female? All else equal, who's more likely to have it? Well, this is a relatively infrequent allele.
A person with XY.
Two versions of it. So if the frequency of it, let's say that the frequency of it, and I looked it up before this video, roughly they say between 1 in 5 to 10,000 men exhibit hemophilia. So
yes.
say that the allele frequency of this is 1 in 7,000 is the frequency of XH, right? The hemophilia version of the X chromosome. And that's why 1 in 7,000 men display it, because it's completely determined whether, there's a 1 in 7,000 chance that this X chromosome they get is the hemophilia version. Who cares what the Y chromosome they get is, because that essentially doesn't code at all for the blood clotting factors and all the things that drive hemophilia. Now, for a woman to get hemophilia, what has to happen? She has to have two X chromosomes with a mutation. Well, the probability of each of them having the mutation is 1 in 7,000. So the probability of her having hemophilia is 1 in 7,000 times 1 in 7,000, or that's 1 in, 49 million. So, as you can imagine, the incidence of hemophilia in women is much lower than the incidence of hemophilia in men. And in general, for any sex-linked trait, if it's recessive, if it's a recessive sex-linked trait, which means men will, if they have it, they're going to show it because they don't have another X chromosome to dominate it. Or for women to show it, she's have to have both versions of it. The incidence in men is going to be I guess we could say so. Let's say that P is, or let me write M is the incidence in men. Incidence. I'm spelling badly. Incidence in men. Then the incidence in women will be what? You could view this as the allele frequency of that mutation on the X chromosome. So women have to get two versions of it. So the women's frequency
What in the world is he?
You might say, hey, that looks like a bigger number. I'm squaring it. You have to remember that these numbers, the frequency is less than 1. So
Saul, I'm listening to you at 2 times speed. How are you making so many mistakes?
Anyway, hopefully you found that interesting.
What is this,
how we all become men and women, and even better, you know whom to blame when
what is going on?
I guess male-focused parents are having trouble getting their son.
Okay, cool. Sex-linked traits are very interesting, yeah. Oh, great. Now this track sounds like Genshin. Okay. I'm getting reminded of every single game except for Xenoblade by listening to Xenoblade's soundtrack. God damn it.
Let's take a look at the nucleus of a cell that's just starting to undergo meiosis. So we have these 46 chromosomes, and beforehand each chromosome probably looks something like that. That's the centromere middle. And then as meiosis is beginning, they will duplicate into, each chromosome will duplicate into something like that. That's again that centromere middle. So we
Yeah,
have
they
these
duplicate
23
during
purple chromosomes.
synthesis.
These are the maternal chromosomes.
Oh my
And then we're going to see that
goodness.
the blue ones are the paternal chromosomes. They come in homologous pairs, and the nucleus that we're looking at must belong to the cell of the male, because right over here you can see that is the Y chromosome. It's a bit smaller than most of the other chromosomes. I'm going to digress just for a moment to clarify a very common point of confusion, and that is that if you look over here at these two chromosomes, well, this is considered one chromosome, but so is this called one chromosome. And this can be confusing because this is only one chromatid.
It's
Two chromosomes, so
super
why are we calling them
confusing.
chromosome? And the answer to that question is because we count chromosomes by the number of centromeres. So this single chromatid has one centromere, but these two chromatids are attached in the middle by one centromere. So we also call that one
God damn
chromosome.
it.
Just keep that in mind. It's just a technical point to know that even though they're different, they're each
I
still
feel
considered
like
one chromosome.
genetics
And
would be
this
so much
chromosome, these
easier
chromotids are
to
different.
understand
A
if we
copy.
had
Anyway,
two separate
so we
terms
have these 46
for
chromosomes, 23 homologous
chromosome
pairs,
with
and
just
they're not
one
not necessarily arranged in the way that I drew them. I just drew it that way for the sake of organization.
one
But we
copy
have these pairs, and we're going to focus on
of the
one
fucking
pair of homologous chromosomes. But I want you to keep in mind throughout this video that whatever we're describing that's happening to this pair of homologous chromosomes is also happening to the other 22 pairs of homologous chromosomes. Just that it would be too hard to depict in a video how that's happening, but keep in mind it's not just happening to the pair that we're talking about, but what we're just going to be describing is happening to all of the pairs of chromosomes in the nucleus.
Okay.
So here we have our pair of homologous chromosomes, and during prophase 1 of meiosis, the homologous chromosomes pair up with each other and form a unit called a tetrad.
Oh wait a minute.
And
Do I
it's
have
called tetrad because, well,
meiosis
tetra means
here?
four,
Oh no,
and
do
so
I
it
have
has four
to
elements, right?
do I
One,
have to
two,
write
3, and 4. And the process during which the homologous chromosomes pair up with each other is called synapsis. So during synapsis, the homologous chromosomes will get a little bit closer to each other, something like that. And at a certain spot, they might actually cross over or overlap. So I'm going to circle that spot, and that's called the chiasma. And in some cases, another thing happens. This protein complex that resembles something like a railroad track forms. We'll see in a
I
moment.
know what I have to
And
do,
this is
but I
called
don't know if I have
the
the strength to
synaptomer
do it.
complex.
I have to
You can
fit
actually see
meiosis in here somewhere.
So we form the synaptomer complex, and with the synaptomer complex, these two chromatids, the ones that are crossing over, will actually swap material downward of that protein.
Oh
So
my
we're gonna get something that looks
god.
like that. Look at how the purple chromosome now has some blue over there, and look at how the blue chromosome now has some purple over there. And the way that
Okay. I mean, I could just mention genetic recombination. I mean, they're not going over meiosis specifically here. But if I were to put meiosis somewhere, I'd have to put it there anyway. Oh my god. What have I done? There's so many different branches off of DNA. Why are there so many branches off of DNA? This is crazy. Redis returns at 3 a.m. PT. I feel like I can have this below my face. This shouldn't need to do this here. Well, I'm gonna take this moment to stretch a bit. Be right back. Okay. Ah, I'm gonna be real. I do not have the best attention span. And this is a very long study segment. So I'm gonna quickly do my Genshin dailies. And I'm too lazy to put it up on screen. Or rather, I'm kind of also worried about putting it up on screen just because, like, I don't want to put any more load on the stream than than I need to. There's already a lot going on. So instead, we're going to do this. Okay, there we go. Alright, boys. Genshin dailies. This is the way. Oh, oh. Oh, what? I didn't spend any money in this game. I didn't do that. That's not a thing I would do. What in the world is this Misty Dungeon thing? I don't know. Just gonna leave that alone. Okay, let's see here. Where are all of the dailies? Where are they? There's one over there. Is it just gonna be a running one? It's gonna be one where I have to run to a goal, isn't it? I'm gonna be like, yo, run towards this thing and kill the slimes on the way, dude. Do it. I'll just be like, no, I don't want to do that. I wonder how, like, big this screen looks when your guys is in. Does it actually look like you can see what's going on in the game? Because I may as well just do this. Like, this is a nice setup. Real easy. Oh, wait. Why do we have alarm during this time? A little confused, but okay. Yep, knew it. Time challenge. There's the slimes, just as I thought. Sorry, where am I? Okay, I'm flying over there. Big brain. Huge. Just run or just fly over. Dummy. Hell yeah. This is the way. This is great. This is a great setup. But Nick, we can't hear the audio. Well, I mean, we could do this. You hear the audio now? Wait, do you actually hear the audio? Hmm. That actually works. Nice. What in the world? Okay. I'm so confused by this scenario. This fisherman was just like, yo, there's no monsters here, and then monsters just appeared. Or was he saying that there were here? But then that they don't follow human concepts of... I don't understand, but okay, this is kind of interesting. Oh, whoops. My bad. Oh my god, what? Why am I fighting like a boss for this daily, dude? What is going on? I'm literally doing no damage. But this fisherman just killed it. Okay. Of course, of course. As you do. My man's just fishing. He's just fishing. I don't get it, but very cool. Okay, we're just here to do dailies. Just the dailies. 'Cause I'm remembering that I need to do some other stuff in this game, but like, man, I do not want to spend any more time on this than we need to. Honestly, I'm not even sure if I consider this a break. Alright, whatever. Just do this. Oh. There's another guy just randomly over there. Okay. Oh, shit. Okay. Okay, last one. Kicked over my stuff. Bro, this just feels like more chores, though. Wait, what? Where is it? It's over there. Okay. Oops. Oh my god. I mean, please. Video game. Does a video game be like, look at this anime girls fighting, yay? Okay, well, this is cool. Wonderful. But where's Norman Reedus? Sure, this may have waifus, but it doesn't have the waifu. Reedus. And it's not directed by Hideo Kojingles. So what's even the point, really? Wait, what? Hold up. Wrong person. There we are. And dailies are done. Good work. Good work. Honestly, yeah, agreed. Where is Saul Goodman? I mean, I can get Yanfei right now, if I roll right now, but no. I want Saul. We better call Saul right now. Alright, that's enough stalling. Let's get back to it. There we go. Alright. Okay, so
What happened was that the DNA in the chromosome, actually some bonds in that DNA broke and the DNAs just kind of swapped places. So what we just described, this process by which the two chromosomes swap information, is called crossing over. Or another way to say this is genetic recombination. And let's see why this is called genetic recombination. So we're going to fast forward to the end of meiosis 2, where the chromosomes get...
I feel like we just did something related to genetic recombination just now. Where is it? Where did we talk about this? Oh my god. Oh no. This has mutated into something that is way beyond my comprehension at the moment. Crossing over. Oh, translocation? It might be translocation down here. That's probably it. Yeah. It's not exactly a good spot to put it, though. I'm just gonna put it here. Crossing over. Genetic recombination. It's gonna be here. I have put my flag in the ground, my stake, my, you know, I'm placing everything on this one move. Boom. Genetic recombination is going to be here. I have determined. Yes. Which is not confusing at all because the arrows are totally going in all of the right places. All of the understandable places. Makes sense. No, that looks terrible. Oh no. Oh no, I'm gonna be so confused. Oh no, this isn't it. Are you serious? What is happening? Move this. Okay. Alright, this is fine. This is probably fine.
It's split into two, and all the chromatids get separated into different gametes. And I want to pause and remind you that everything we're describing that's happening to this pair of chromosomes is also happening to all the other 22 pairs of homologous chromosomes. But anyway, so now let's put each one of the chromatids in a different gamete, and look at how we get 4 different gametes. And we can call these two gametes recombinant. And we're calling them recombinant because they have a combination of alleles that's new. We haven't had this combination of alleles in the And just to clarify things, let's see what the gametes would look like if crossing over did not happen. So let's go back to our original chromosomes, and let's split them. And let's put them into 4 different gametes. And we are going to get that. And you can see that in this case, we only have 2.
No glasses,
gametes.
gang.
So we can see how
No
genetic
glasses.
recombination increases genetic variability, which is usually a good thing. One of the very interesting things about genetic recombination is that you can actually use genetic recombination to figure out the distance between genes on a chromosome. And if you were to do this to
Yeah,
all
you
genes on a
can
chromosome,
do
you can actually
that.
map out
Oh,
chromosomes to
yeah,
figure out
you
exactly where the
can
genes
do
are.
that,
And we're
but
going to explore
then
that concept.
oh
So we're looking at a pair of
no.
homologous chromosomes. Now let's just say that the orange chromosome
Okay,
is the maternal chromosome, so it comes from the father.
let's.
And
Oh
let's say that the yellow one is the
wow,
maternal
that
chromosome.
is off to a completely different
And just to remind
planet.
yourself, so these are
On
sister
gene
chromatids.
mapping. Gene mapping, baby.
That means
Getting
that
into
they're
the
identical
cool
chromatids,
shit.
they have identical genes
Hell
on them.
yes.
So those colored circles
Finally.
that I do represent just some genes that I randomly picked.
Only
And
took
you can
us
see that I have
5
two sister chromatids that
five
are
and
the
a
same
half
color,
hours.
they're the same. And for that matter, these are also sister chromatids. These 2 yellow chromatids are also sister chromatids. And I'm just going to review a bit to give us some context. So normally in the cell, the paternal chromosome would just look something like that with a centromere mill, and the maternal chromosome would also look something like that. But during meiosis or mitosis, when they replicate, this chromosome will turn into that, so it's going to duplicate itself, and each one of those strands is a sister chromatid, and so will the maternal chromosome turn into something that looks like that. But this is still considered one chromosome, and this is considered one chromosome. And that can be a little bit confusing because how could this be one chromosome and this also be one chromosome if this X has double the material as the original strand? And the answer to that question is because we count chromosomes by the amount of centromeres that are present. So this has 1 centromere, it's considered one chromosome. This X has 1 centromere, so it's considered one chromosome. However, they're not quite the same. This on top it's just 1 chromatid, and the bottom chromosome is made up of 2 chromatids, so try not to get confused. Even though we're calling this one chromosome, it's really made up of 2 identical sister chromatids. But anyway, that was a bit of a digression. Let's focus again on our homologous chromosomes. So I picked a couple of genes that I just put on, and we're going to focus on 3 genes in particular. And to make this a bit more real or relevant, let's just say that the green genes represent, I don't know, maybe complexion, maybe like a dark complexion versus a lighter complexion. And on the maternal chromosome, I also drew it in green to remind us that these are homologous chromosomes or homologous alleles. In other words, these are both alleles that code for complexion, but different shades of green because they're probably different versions. So these green genes also code for complexion. And let's say that this darker purple or magenta gene codes for, let's say, hair color. So that means that the lighter purple on the maternal chromosome also code for hair color. And then let's say that the blue genes code for eye color. So we have dark blue on the paternal chromosome and light blue on the maternal chromosome. And let's then focus on the sister chromatids where genetic recombination occurs. So these 2 strands are going to swap genetic information between them. Maybe like a chunk on the bottom will swap, maybe something in the middle will swap, maybe something on top will swap. Actually, genetic recombination also occurs between sister chromatids. However, sister chromatids are identical, so it would be of no consequence. Anyway, so let's look at the 2 chromatids where genetic recombination is happening, the 2 that I circled. Let's take a closer look at those. So here are the 2 chromatids that are going to exchange genetic information and undergo recombination. And I want to ask you a question. So I'm going to give you 2 choices, and I want you to try to figure out which one is more likely. So for the first choice, let's look at the purple and green genes. And the question is, what's more likely?
Hi
Is it more likely
boys,
that the
I'm
purple and green
across the
genes undergo
streams.
recombination? And when I say that, I
Gonna
mean that
cross
the
the
genes that are coded
streams
for
with
one chromosome
this.
separate.
Oh
you can see that
no.
this purple gene will get separated from that gene, and this lighter purple
Okay,
gene will
we
get
did
separated
it.
from that gene. So is it more likely
Chromatid
that the purple
references genetic
green
recombination.
genes recombine?
No.
Or the way I like to view it, get separated?
This is
Or
this
is
is
it more
not
likely
it.
that the blue and
This
purple
is
genes
too
recombine and get
much.
separated?
What have I done? Too much of a
And
programmer
when you think about
move.
this is,
I'm
well,
not ready
what's
for
the
that
distance
kind of
between them?
thing.
So the distance between
Just
the purple
yet.
and green genes is something like that. So in order for recombination to occur with respect to the purple and green genes, it would have to happen somewhere over here. Or on the other side, it doesn't matter, it doesn't make a difference. So you have this whole distance. Whereas if you wanted recombination to occur with respect to the purple and blue genes, you have a very small area to work with. It would have to happen somewhere here. So somewhere along, this line of the chromosome. So that means that it's much more likely for recombination to happen with respect to the purple and green genes. That is more likely because you have this entire distance to work with. If these two chromatids swap genetic information anywhere along this stretch of the chromosome, so the green and purple genes will separate or recombine. And the blue and purple genes are less likely to recombine because recombination would have to occur only in this little sliver of chromosome, and that's just smaller than the other part of the chromosome that we were looking at. So we just learned a very important concept, and that is that the further apart two genes are, the more likely it is that they will recombine. I'm actually not sure if that's the proper way to use the word, but I just use it that way. So I'm going to put it in quotes. And again, when I say recombine, I mean that two genes that were originally on the same chromosome get separated. And then the next thing we learned is that the closer two genes are to each other, I'll abbreviate each other just EO, the less likely it is that they will recombine.
Big brain. Big brain, not gonna lie.
The centimorgan is the unit of measurement that we use to measure distance on a chromosome.
No.
And another way to say centimorgan is a genetic map unit.
Centimorgan.
Or M.U, which stands for map unit.
Oh, map units.
Because I think it's important. It kind of ties in distance to what
map
happens with
unit.
recombination.
Oh, wow. I didn't know they actually had a
And
different
so a
name.
centimorgan is the distance between genes, just going to abbreviate between like that, for which one product of meiosis in 100 is recombinant. And to put that in simpler terms, it means that if two genes are 1 centimorgan apart, it means that 1 out of 100 times, or 1% of the time that meiosis happens, those two genes will be recombinant or separate or recombine. And we'll actually do an example of this to illustrate what this means. So here we have our two chromatids again, and let's just say that the distance between the purple and green genes is 25 map units. Remember, a map unit is the same thing as a centimorgan, and let's say that the distance between the blue and purple genes is 6 map units. So this is clearly not drawn to scale very well, but it's just an estimate. So let's first focus on the purple and green genes. So if they're 25 map units apart, so remember if two genes are 1 map unit apart, that means that 1% of the time they'll recombine. So these are 25 map units apart. So that means that 25% of the time that meiosis happens, recombination will occur with respect to the purple and green genes. So let's see what that looks like. So we're going to see that in this spot right over here, the chromatids just swap information. So let's draw what that would look like. So let's draw our paternal chromosome, or at least part of it, and then we'll draw our, actually I'm going to draw that a little bit higher because I need more room. So here's our paternal chromosome, and then our maternal chromosome, or chromatid, and then since they kind of swapped in this spot, so I'm going to draw yellow right over here. That's the part that came from the maternal chromosome, and then I'm going to draw orange over here. That's the part from the paternal chromosome. And now let's fill in our genes. So the blue genes will just stay where they were, so we just leave them put. And the same goes for the purple genes. But then we have that piece of maternal chromosome, so we get that hunter green gene over here, and then we get the lime green gene right over here. So this is what I mean by recombination being a separation of genes. So this purple gene and that lime green gene were on the same chromosome before, but they got separated. They're not on the same chromosome anymore. And the same applies to these two genes. Now let's look at the blue and purple genes. So they're 6 map units apart. So that means that 6% of the time that meiosis happens, the purple and blue genes will get separated. So we're going to say that upwards of this spot, the chromatids swap information. So let's draw that. So here we'll draw a part of our paternal. That's the paternal chromosome and the maternal one. And they swapped somewhere like over here. So let's fill that in. So that's the part of the maternal chromosome that lands up, that's the part of the maternal chromosome that ends up on the paternal one, and the orange over here. And now let's fill in our genes. So let's first fill in the ones that just stay put. So we have that lime green gene, then we have that hunter green gene, and our purples also stay put. But the blue genes swap chromosomes. So we have our dark blue gene over here, and our light blue gene over here. And again, take note of how they swap places, or how they separate. So these two genes were together on the same chromosome before, but not anymore, and the same for these two genes. So let's just tie this back into the bigger concept going on. If we were to do a statistical analysis of how often certain recombinations happen, that can help us map out the genes on a chromosome. Normally when we think about DNA, we think about the nucleus of a cell, and that's because a cell's DNA is contained in its nucleus. But there are actually a few exceptions to this general rule. So there are certain organelles that actually have their own DNA, and two very famous examples of this are the mitochondria and chloroplasts.
Passed down from mother to mother, same exact
mitochondria.
mitochondria. It's freaking wild.
Own DNA, but they can actually replicate their DNA and replicate themselves independently of the nucleus.
All the way back to our ancestors in Africa. That's how we were able to trace it.
And they're sometimes referred to as
That's
the
wild,
powerhouse of the
man.
cell because they break
That's
down glucose
awesome.
to make this high energy molecule called ATP, and then the cell takes this ATP and uses it for all sorts of cellular processes. And the mitochondrial DNA, written like that, mtDNA, has about 37 genes in it. And these genes
Powers the cell, more like
in the mitochondria. Let's talk a bit about chloroplasts. So chloroplasts are
most
the
important
found in
connection
cells.
to our ancestry.
They're also found in algae cells. And chloroplasts are the site of photosynthesis. If we want to be more specific, so you have these stacks called granum. Well, in singular it's granum, plural it's grana, and those granum are made up of these, that's an M over there. And those granum are made up of these little circles called thylakoids. And photosynthesis happens within these thylakoids. So during photosynthesis, sunlight is harnessed, of course, with a bunch of
Why are we talking about
glucose. So
Mitochondria
and
and chloroplast
food comes from. It's actually
chloroplast
making
at the
glucose, making
moment.
its own food, and then that glucose goes to the mitochondria of that cell and gets broken down, make ATP, and then the cell uses that for like, whatever it needs to do. The DNA in chloroplast, sometimes written cpDNA, has about 100 genes, and these genes also, most of them have to do with proteins or things that are involved in photosynthesis. And the reason that this is interesting is, well, let's take a look at how sexual reproduction normally takes place. We have an egg cell, and the nucleus of this egg cell has only half the amount of DNA that a normal cell in that organism would have, so we call that.
I'm starting to realize a problem. If we want to make a connection to something else from any point on this map that I've made, painstakingly made this entire time, I probably want to connect over to mitochondrial DNA through DNA. No, that's just another branch of DNA. Where do I even go? How would I even do that? Is that even possible? Maybe I can put it here and then... Oh my god, I'm about to do a pro gamer move. Gonna connect DNA all the way up here. And I'm gonna connect it to extranuclear inheritance. Oh man, I feel like something's stuck at the back of my throat, which is never good because that's usually like similar to one of my cold symptoms, but I don't think it's that. I think I'm just tired. And just like stuff is just like coalescing in my body. Back there somewhere. Goddamn. I feel like my body temperature has probably dropped a lot too. Doesn't help that I'm using fans to like cool down my PC. Fans on top of fans. Which has diminishing returns, to be honest, but not like I can really test that out. I really need like some kind of application to calculate PC temperatures for me because I'm not really clear on how much I'm
Remember, the
using.
sperm cell is really much smaller than an egg cell, so this is in no way drawn to scale.
Or
And the sperm cell also
how
has
far I'm
half the
pushing
amount
it.
of DNA that cells in this organism normally have. So that's also N. But then they fuse to make a zygote. And this zygote is 2N. It has the normal amount of DNA that a cell in this organism would have. Half of it comes from the egg cell, and half of it comes from the sperm cell. And then this zygote is going to divide into 2 cells, and then those 2 cells, of course, divide further. And this goes on and on until, excuse me, until there are enough cells to put together an organism. But this egg cell, well, it's a fully developed cell, and it not only has genetic information, but it has organelles in the cytoplasm. So it has these mitochondria in the cytoplasm, and those mitochondria have DNA in it, which I'm just going to scroll some aside. And the zygote also has those mitochondria. Because remember, the zygote is, well, practically an egg cell, but with the only difference being that its nucleus has the additional DNA of the sperm cell. And remember, the sperm cell does not donate anything to the egg cell except for half of the DNA in the nucleus. It does not give the egg cell and the zygote anything else. So you have a zygote with those mitochondria, and of course they have their DNA in it. And then when this zygote replicates itself, so it replicates the nucleus, but it also replicates the mitochondria in the cytoplasm. And these cells will, I'm going to skip out the nucleus, we're just showing the mitochondria. So they have these mitochondria, but these mitochondria came only from the egg cell. None of those mitochondria came from the sperm cell. And so this brings us to the concept of maternal inheritance. And maternal inheritance, well, it's basically like exactly the way it sounds. It's inheritance that happens only from the maternal line or only from the egg cell. So right here, we're showing that the mitochondria that this organism will eventually have originates from the mitochondria that came only from the egg cell and not from the sperm cell, and therefore exhibits maternal inheritance. So both mitochondria and chloroplasts exhibit maternal inheritance because they are in the egg cell that eventually becomes the organism.
Oh wait.
And maternal inheritance,
Whoops.
it's interesting
Yeah,
to note,
both
is
both
contrary
chloroplasts would have. Yeah.
to Mendelian genetics. So maternal inheritance is contrary to Mendelian genetics because Mendelian genetics assumes that half of the DNA comes from the egg cell, half from the sperm cell. It does not take into account any sort of genetic information that comes from only one of the gametes, for example, just from the egg cell. And in fact, everything we just described here can be referred to as extranuclear inheritance. So extranuclear inheritance would refer to any genes that are passed on from structures that are not in the nucleus. So extranuclear meaning outside of the nucleus. Mitochondria and chloroplasts are outside of the nucleus, so they when they are inherited, we refer to it as extranuclear inheritance. So now that we've introduced extranuclear inheritance, let's actually take a look at one of the early experiments that helped to discover extranuclear inheritance.
Ooh, talking about history again. I love that. Totally down to learn all about extranuclear inheritance and the history behind how we discovered it. Definitely. How long did it take for
There was a
AMC
scientist by the name
to
of
approve all this?
Carl Correns. And he was somewhat
Have
of
they
a
approved of it
He
recently?
lived from
Because
1964
oh
to
boy.
1933. And Gregor Mendel
Carl Correns.
lived from 1822 to 1884. So they were contemporaries, but Correns was younger than Mendel, and we will soon see that Carl Correns helped discover some things that do not fall into the category of Mendelian genetics. So Carl Correns did a lot of experiments with this plant called the 4 o'clock plant. That's how we know it, which is what you're looking at. I know it looks a little bit like a tree, but it's supposed to be a plant. And actually, the scientific name for the 4 o'clock plant is Mirabilis jalapenos.
Hey yo, trees ain't plants.
But anyway,
Wait a
so we did
minute.
a lot of experiments with the 4 o'clock plant. And one very
What are you
interesting thing about
talking
this
about?
plant is that you can have within the same plant leaves that are a lot of different colors. So let's say that this is the main branch of our plant. And
Ooh,
we're going to
Kev's
say
battle.
that the leaves
Ooh,
that
let's go.
come off anywhere on this branch are going to be white. So there are our white leaves, and we're going to say that any leaves that come off of this branch are going to be green. So those are our green leaves. And then the leaves that come off of this
Okay.
branch are variegated.
Come on.
And that means that they have a pattern of green and white mixed.
W-
So they are
what?
variegated leaves. And why is it that?
What is that word?
So let's take a look at some of the cells.
Variegated?
Cell
Va-
that's from a
what?
leaf that's green. So this is in a leaf that's green, so that's our cell. Here's our
Just
nucleus.
as I'm
This is what
getting
our cell
hyped up
is. Square, it's
to
a
learn
plant cells have a
more,
somewhat more vivid shape.
I am struck with a sudden realization that I have no idea what that word is.
And remember,
Variegated?
chloroplasts have their own DNA, sometimes referred to as cpDNA. And chloroplast DNA has in it basically the stuff that the chloroplast needs to carry out photosynthesis. And one of the genes in the chloroplast DNA is a gene that makes chlorophyll, which is a pigment that's involved in photosynthesis.
Different
And chlorophyll is
colors.
what makes the leaf green. It's a pigment
Right.
that turns the leaf green. If you
I
want more
probably
specific,
would have realized that
all
had
the
I
colors
paid
in sunlight,
attention.
except for green, so green is really reflective. But anyway, the point is that chlorophyll makes the leaf green.
Okay, okay,
So now
okay.
let's take a look
Very cool.
At a cell that comes from a white leaf. So this cell comes from a white leaf. So again, we'll draw our nucleus, and then it has chloroplasts. But the chloroplasts in this cell, the DNA in those chloroplasts have a mutation. So the cpDNA has a mutation that does not allow it to produce chlorophyll, or allows it to produce only a very, very tiny amount of chlorophyll because it needs chlorophyll to survive, but it's not enough that the leaf will be green. So I'm just going to write very, very tiny amount of chlorophyll, and that's why it appears white. And then the variegated leaves, well, they have some cells that have regular chloroplasts that make chlorophyll, and then they have some cells with the mutated chloroplasts, or chloroplasts with mutated DNA, that do not make chlorophyll, or rather make a very small amount. And then they actually have a third type of cell, which, maybe you're guessing it already, have both types of chloroplasts. They have chloroplasts that do make chlorophyll, and then they have chloroplasts that have mutated DNA and do not make much chlorophyll. And what Correns noticed, when he crossed a whole bunch of these plants together, he noticed that the progeny had nothing to do with the sperm cell or the pollen cell. It had only to do with the egg cell. So wherever he would take the seed from, the seed is where the egg cell is located. If he took it from a branch that only had white leaves, all the progeny had only white leaves. It didn't matter what the pollen cell was. And the same if he took a seed from a branch that had only green leaves, so all the progeny had all green leaves, no matter where the pollen cell came from. And this is because the trait that we're looking at, the color of the leaf, well, that's determined by the DNA in the chloroplast, and the chloroplast exhibits maternal inheritance. It is going to be inherited only through the egg cell, or through the maternal line, or another way to say this, it exhibits extranuclear inheritance, because the chloroplast has DNA that's outside of the nucleus. Let's take a closer look at what Carl Correns did. So we have this chart to help us out. In the first column, we have the egg cell of the female, that's the seed, and then we're going to cross it with what we have in the second column, which is the pollen cell, that's the male gamete, and they all look the same because it doesn't make any difference in our case. And then we have the third column, our zygote, or the result. So let's look at our first row. So we have this XL that came from a branch that had leaves that were only white. So it came from a flower plant with only white leaves. And when we cross it with a pollen cell, no matter what that pollen cell is, no matter where it came from, we always get the same result. We will always get a plant with only white leaves. Let's look at our second row. So we have this XL that came from a branch that had only green leaves. And again, no matter what we cross it with, no matter where the pollen cell came from, a white leaf, a variegated leaf, a green leaf, or a flower green leaf, so we always get the same result. We get a plant that has only green leaves. It gets a little bit more interesting when we look at the XLs from the variegated parts of the plant, because there are 3 different types of XLs we could have. We could have one that resembles the cell you see if it came from a flower with only white leaves. Then we have another XL that looks like that. It looks like the XL you'd find in a plant that had only green leaves. And then we have this third type of interesting cell that has a combination of the quote-unquote normal chloroplasts that are green, and then it has some chloroplasts that have that mutation that don't allow it to make chlorophyll or make a very small amount of chlorophyll. So that's kind of mixed. Anyway, let's look at egg type 1. We cross it with a pollen cell. We always get the same result. We get a plant with only white leaves. And then we look at XL type 2. Whatever we cross it with, we get only green leaves. And then if the XL type 3, so the zygote of course will have both types of chloroplasts, but remember this zygote is going to divide further, and it's
Dude, there's only so much that I can commentate over.
Some of the cells will look like this with the chloroplast.
Look at this shit.
Some of them are going to look like that with the regular
It's
chloroplast,
so
and some are going to
straightforward.
do that
It's
in the
literally a lie.
pattern. You might have some leaves that are white, you might have some leaves that are green.
Gee, I
So,
wonder what
one
happened.
key cool message is, as I explained before,
I
because the
understand
trait we're looking
this is
at-
the process of science,
Because the gene for that trait is in the chloroplast,
but damn.
it exhibits maternal inheritance. Maternal inheritance.
Oh, yes. Okay, that's cool. Alright.
Because this inheritance has to do with DNA that's outside of
It's
DNA.
just
So
like
exhibits maternal inheritance,
watching,
it has nothing to do- this particular trait
you know,
is
like
not passed down to the
strategic
only passed down to the
RPG
female.
gameplay
As we explained before, because the chloroplasts are coming only from the XL.
without the Fog of
Chloroplasts
War
is zygote. It only
as an
contains
audience
the
member.
So therefore,
But the
the
players
chloroplast
are still
is
playing with Fog of
maternal
War
inheritance.
because it's back then
And
and
the
they
same
don't
process
have
applies to the
ways to
mitochondria.
turn off the fog.
So we explained mitochondria also has its own DNA, and so if a person were to have a disease that had to do with the DNA inside of the mitochondria, we would know that that person got it from his or her mother.
Shout out to
Because
Corins,
the mitochondria also exhibits maternal inheritance. There's one more thing about extranuclear inheritance that
my
and
favorite
that is,
protagonist
why
of
is it
Fire
that
Emblem.
mitochondria and chloroplasts have their own DNA? Is there something that can explain that? And there is the endosymbiotic theory. Seeks to explain why mitochondria and chloroplasts have their own DNA. And this theory tells us that mitochondria and chloroplasts were once independent prokaryotes. So they lived independently. So of course if they're independent,
Oh,
they
shit,
have their own
okay.
DNA. But eventually, they joined what I'm going to call a ancestral eukaryotic cell.
Yep,
In case you're
and
wondering,
then
like, what exactly
Nom,
to say about
the
what happened
eukaryote, just was like, yo,
So what happened is
let's
that the
join
mitochondria,
forces.
the chloroplasts joined an ancestral eukaryotic cell. And I'm going to call it ancestral because it's not exactly a eukaryotic cell that we see today, but it's the cell that would eventually become a eukaryotic cell. We could also call it a host cell because it's going to host mitochondria and chloroplasts. So let's put them inside. So now they're living in this host cell. And why do you think they would want to do this? So they're going to live together in symbiosis. And symbiosis is when organisms live together and each one kind of gives the other something and everybody gains something. So an example of this that you might have heard of is in our intestine, in our gut, we have bacteria E. coli, and at first glance that might seem like it's not such a good thing, but it's actually a really good thing because we give the E. coli a warm and cozy place to live, they get some nutrients from us, and in exchange, they make for us vitamin K, which is something very useful for us. So that's an example of symbiosis. Each one of the organisms kind
Based
of gets
E.
something
coli.
that's happy. So what's going on in the host cell? So the host cell gives the
Yo,
mitochondria
vitamin
a nice place
K.
to live and gives them nutrients, and in exchange, the
Holy
chloroplast makes the host
shit.
cell through photosynthesis,
Thank you,
and then the
E.
mitochondria
coli.
goes and produces ATP.
Awesome.
And then that ATP is used as energy in the mitochondria, uses some
So
of the ATP,
wholesome.
chloroplasts uses some of the
Giga Chat E.
ATP, and of course
coli.
the host cell uses some of the ATP. So everybody's happy, everybody's getting something. And then eventually this cell, evolved in many different ways, and of course it became the eukaryotic cell that we know this today, but not exactly the way it was because chloroplasts are not found in all eukaryotic cells. It's basically found in plants and algae, but that's the basic idea.
Taking some
So let's go
vitamins
back to
right now,
the
too.
endosymbiotic theory. So endo just means inside because the mitochondria and chloroplasts began to live inside a host cell, and symbiotic simply refers to symbiosis. Just going to draw an arrow down here, refers to symbiosis because each part, the mitochondria, chloroplast, and host cell, everybody's getting something. And so
I
this
take
would
my
explain why
vitamin D
mitochondria and
because
chloroplasts
I
are
haven't
because
seen the
once upon a
sun
time ago,
in,
they were independent
in
prokaryotes and
weeks.
lived on their own.
I have, but damn. Damn, bruh. I do not touch grass. Too many chloroplasts, man. Too many chloroplasts. Whoa, what? Oh, okay. Population dynamics. Let's go. Finally, we're talking natural selection.
So today
Oh
we're going to talk about
no,
a topic that's very similar
not.
to the idea of evolution. and that's
Oh,
natural
there's a
selection.
lot.
But before we
Okay.
get into that, I want to talk about what evolution isn't. So
Alright.
evolution isn't when some organism, like
Let's
the
do
monkey,
it.
magically transforms into a human.
I
And it's
got
also not when
this.
an organism changes in some way when it's in trouble, like a person growing wings after jumping a building. And I want to clarify that this is not what we're referring to.
Get
as
wrecked,
evolution.
Pokemon.
Evolution is a process that occurs to populations of an organism, not
Not how
individual
evolution
organisms. It occurs
works.
over huge amounts of time. And we're talking millions and millions of years for even small changes. So natural selection is one of the forces that ultimately drives evolution. But what is natural selection exactly? Well, why don't we jump right in and look at an example? Let's say it's 10,000 years ago and people survived by hunting and gathering, but they also have to worry about, being chased around by wild animals. So in order to survive, these people need to be able to find food, but they also need to be able to escape from predators. Well, let's say that one of the people
Oh my
has
god.
a special genetic trait and has slightly longer legs.
Yo, my
These longer
cytokines
legs put him in an advantage
are
because he's longer and he can run, let's say, 2 times
proliferating
faster
at
than us.
a
And this is more likely to
immensely
survive when a predator
fast
chases
rate.
him down. So what this also means that the guy
Okay,
with long
I'm
legs is more
distracting
likely to
y'all
originate if he's old
from
enough to find a mate, reproduce, and have children.
some.
Who would also have a special trait
That's
of longer legs
beyond
as genetic.
the scope
And
of the
because he's more likely to
MCAT.
have kids than anyone else over long periods of time, soon more and more of the population will have this special trait. Now, let's look at this idea again, but a little more deeply. And let's say there's 6 people in the world, and 2 of them have longer legs than everyone else. And let's say the ones with the longer legs have a 50% chance of surviving and reproducing, while the
Long
short-legged people have
legs.
only a 25% chance of
More
surviving
like
and
every
reproducing.
single
So that
anime
means one of the
character
2 long-legged
ever.
people and one of our 4 short-legged people here will reach an age
Takes
of
so
50.
long.
So now these people who survive will each have children,
Damn.
and naturally these children will resemble their parents.
In
And
the
children with long
world.
legs will also have long legs, and the children of short-legged people will have short legs. So now in our next generation we have 4 people with long legs and 4 with short legs.
Literally just like if you took
long legs than when we started. But let's
like
take
the
another
average leg
so half of our long-legged people will reproduce,
and
only
just
1/4 of
stretched
our
it.
short-legged people will reproduce. And this means
Anime
that our
legs,
generation will have
man.
8 long-legged kids and only 4
They be
short-legged
going
kids.
places.
Now, if we number our generations, generations 1, 2, and 3, we can see that in generation 1, 33% of the population was long-legged. In generation 2, 50% of the population was long-legged. And by generation 3, 67% of our population was long-legged. And this is all because that special trait of having longer legs made those people more likely to survive and reproduce than those with short legs. And this is the crux of how natural selection works. So why is it called natural selection in the first place? Well, let's take our example of the
My
short
God,
legged
are we
people.
going
Now,
through
here's the word
all of
selection,
the
because one trait is
like
advantageous
sound
to someone, and it's
effects in
selected to be
Xenoblade?
passed on to future generations more than other
Okay, there
traits.
we go.
On the other hand, selection can also apply to a disadvantageous trait. If
We
have people
will
with short
know
legs
our
and move slowly, then those
names.
people will be selected against
The
and will not
finale,
pass on traits
even
across
though
them, so
it doesn't really
Now, these are
reference
natural because
you
this
will
is
know our
really
names
specifically selecting which traits are good
within
and
the
which ones are bad.
finale.
It all has to
That's
do
too
with who has
bad.
the greatest probability of surviving. There's no one actually doing the selecting except nature itself. Now, finally, I just want to point out that natural selection does not apply to acquired characteristics. You know, if a father teaches his son how to hunt and this makes the child more likely to survive, that isn't a trait.
You know, I'm not really sure where to put evolution. Where do I put that in here? Like, is it? We just put it. Branching off of DNA. That's the easy answer. I can't just branch off everything from DNA. It's not helpful. At this point, I may as well just make this the center of my map. Okay? This is ridiculous. It's stupid. I don't like it. So instead, I mean, it's still gonna be the center if I chose something branching off of DNA, though. Oh, but I just don't want to make another one. We're 1/4 of the way there. Let's go. 1/4 of the way. Oh no. Okay, I'm gonna think about this and I'll be right back. Where to put Hell yeah, I returned to Weight of Life? Hell yes. Perfect. Yeah, sorry for the extended break. I need to lay down, because, like, it's not like I'm tired, it's just, it feels really like, I don't know, stifling, having to sit and do the same thing for an extended period of time. If I were to switch it up more often, then maybe it would be easier, but this stream's not about that. This stream's about sitting here and grinding. Like, not in Genshin. Not in a video game. We're grinding out freaking MCAT, which is supposed to stimulate my brain to get things together. Oh my god, I probably have broken so many DMCA notices. Uh-oh. Kind of just let Xenoblade play in the background. Oops. It is what it is. Oh, I want to play Xenoblade right now. I want to play Xenoblade 3. I have 1. I have 2. I actually have X with me too. Actually, no, I don't have 2. I was borrowing someone's copy of 2, so I don't actually have 2s myself. I have Torna, though. So, that's interesting. But yeah, I have X with me right now. Where is it, Nintendo? Where is it? Where is it? Give us X. Oh, that almost sounds like almost sounds like I was saying something else there, but no. Yeah. Honestly, X is probably not my cup of tea, or cross, or whatever you want to call it. Way too much MMO mechanics there. Oh, not even mechanics. MMO systems, just in general. Kind of frustrating to deal with sometimes, but you know, it is what it is. Also, the game, you can, like, the whole point is that you're supposed to traverse like this huge area, but it doesn't clearly show you where you're allowed to jump and like traverse. So I keep on trying to jump over cliffs, or like up cliffs that obviously aren't supposed to be jumped over, or not obviously meant to be jumped over. I don't know. It's a mess. It's a right mess. Oh man. But I probably will play Xenoblade X2 if they have that. That might be fun. Not really sure what that would look like though. I feel like they covered most of what they wanted to cover with X, and then they had the, you know, the ending is allowing for a sequel, but it's like, I don't, we'll see. We'll see what they do with it, if they ever do anything at all with it.
Since it's not genetic, and it's not absolutely passed on to children. So that's why we say that natural selection only applies to heritable traits, with heritable traits being any genetic trait. So what did we learn? Well, first we learned about the concept of natural selection, and how traits that help an organism survive are more likely to get passed on to offspring. Next, we learned that evolution, which is driven by natural selection, occurs to populations, not individuals, and occurs over a huge period of time.
Yeah, I think evolution just needs to be a separate thing. Yeah, evolution might need to be in a separate category altogether. Chemical, physical foundations. Foundations of behavior. No, that's not behavior. I mean, it would branch off of DNA, but it's like, it has to be heritable, like they just said. So I guess that would be. I'm just gonna put it over here just to be safe, and then we might be able to add it to something else, just like how I plan on moving the enzyme group over somewhere else. So evolution includes natural selection. Which only affects heritable traits, not acquired traits, because that's a separate thing altogether. Of course, memes are unironically the DNA of the soul. So, you know, there's like a separate heritable system in place for culture. Culture is heritable as well, in a more complicated interaction. But you know, that's beyond the scope of what we're talking about here, so that's all good. That's more psychological foundations of humans, humans specifically, because you don't see that in any other species. Because the whole reason why humans have been able to be so successful isn't necessarily because we're smarter than other animals on a baseline, from a baseline perspective, right? Like, our sheer intelligence doesn't necessarily like lead to us dominating our environment, but it is our accumulated experience that gives us that strength to overcome obstacles that no other species have even touched upon. Like, yeah, sure, some monkeys may use tools, but will they remember to use tools and build upon those tools and like build huge machines with those tools as a basis for like
So
physics
we're going to talk about
and
the concept of fitness and how it relates to natural
man,
selection.
it's wild.
But first, let's go over what natural selection is. So if you have a member of a population that has a special genetic trait like being really strong, then that person is more likely to live to an age where he or she can reproduce and pass on that special trait to offspring. Now, this example only really deals with surviving until the age where reproduction is possible, and doesn't really deal with the person's ability to actually reproduce once it gets to that age. Now, you should also remember that populations as a whole will evolve by natural selection, and not individual members of a population. So what is fitness, and what does this term mean? Well, fitness refers to an organism's total ability to pass on traits to offspring, and we can look at fitness as a combination of an organism's ability to survive to an age where it can reproduce, as well as the ability
I
to
keep
actually
thinking of the
reproduce.
Maxword
So, strong
joke
organisms probably have a higher survival rate than the
from
average
his
person.
Metal
Actually,
Gear Rising
it's
video.
nuts. And how well someone can reproduce is
That word is
fecundity, which is what we're talking about today. So what's
that word has
fecundity?
changed
Well, fecundity refers to how easily and how often
in my
organisms reproduce
mind.
offspring. Now, when you look at asexual reproduction,
It's
instead of sexual
ridiculous.
reproduction, fecundity of bacteria like E. coli is determined
Whoa,
by how
whoa,
quickly
whoa,
E. coli
whoa!
cells can
This
divide and increase
is
their population size.
whoa! What is
Pass on
happening?
traits to offspring. And we can look at fitness as a combination of an organism's ability to survive to an age where it can reproduce, as well as its ability to actually reproduce once it gets there. So
Yeah.
our strong guy probably has a higher survival rate.
But then
than
what's
the person. Actually
fecundity?
reproduces the same as everyone else. And how well someone can reproduce is determined by that person's fecundity, which is what we're going to talk about today. So what's fecundity exactly? Well, fecundity refers to how easily and how often an organism can produce offspring. Now, when you look at asexual reproduction instead of sexual reproduction, fecundity of bacteria like E. coli is determined by how quickly the E. coli cells can divide and increase their population size. When looking at sexual reproduction, fecundity refers to how well an organism can mate with another, and in the case of mammals like humans, carry and ultimately birth offspring. Now, when looking at the fecundity of humans, people often think that it only applies to female fertility, since the females are the ones that are actually carrying the children. But fecundity can also be a measure of a male's ability to produce offspring with a female mate. Now, fecundity is directly related to natural selection, because like any other special trait, high fecundity is selected for. So let's look at an example. Let's say we have a population of 6 people, 2 of which are red and 4 of which are blue. Now, the red and the blue people have the same chance of surviving to an age where they can reproduce, which is 50%. And what this means is that 1 of our red people will survive to an age where they can reproduce, and 2 of our blue people will survive. But the difference between the two is the red people who survive will each be able to produce 4 offspring, whereas the blue people who survive will only be able to produce 3 each. So by our second generation, we have 4 red people and 6 blue people. Now, 2 of our red people, 50%, will survive, and 3 of the blue people, also 50%, will survive as well. But once again, the surviving red people will each have 4 offspring, while the surviving blue people will only have 3 offspring each. So this means our third generation with 8 red people and 9 blue people. If we now number our generations 1, 2, and 3, we see that in generation 1, 40% of the population was red, while 67% of the population
Interesting.
was blue. In generation 2, 40% were red, and 60% were blue. And by generations 3, 47% were red, and 53% were blue. And this increase is all because the red people have a special trait of higher fecundity, which made them more able to have offspring than the blue people. And this means that fecundity is selected for by natural selection the same way a trait that benefits survival like strength would be. Now, another interesting way that fecundity is selected for has to do with mate selection. Now, when looking for a female mate, many males associate an attractive woman with words like curvy. And a curvy female would be one that has a healthy and robust body that was fit for bearing children. And this ability to easily bear children is a direct indicator of high fecundity. So, what did we learn? Well, first we learned that natural selection will select for individuals with traits that give them high fitness. And we can divide fitness into traits that will help with survival and traits that will help with reproduction, which are those that increase fecundity. Second, we learned that fecundity is selected for by natural selection.
Not going to
just
say
like any
anything.
other trait.
The way that our minds have evolved to understand what is and isn't attractive is so much more complicated than you might than you might understand from first blush. Like, yeah, childbearing assets are important, but it's like not it's not
So natural selection isn't the only force that drives evolution, and there are a few
not
alternative
the
forms of
only
selection
thing
as
that's
we treat.
important.
But before we
So
discuss, let's
it's
go over what
like,
natural selection
I
is in the
don't
first place. And it's the
I
idea
don't
that
know
if
what's
you have a member of a
going
population
on.
that has a special advantageous genetic trait, then that individual is more likely to live to an age where it can reproduce and pass on that special trait to their offspring. Also remember that natural selection selects for individuals with high fitness,
Societal
and fitness is a measure of an organism's total ability to pass on their genes to their
expectations
offspring.
are
And
way
it's a
too
combination of an
messed
organism's
up,
ability to
man.
survive to an age where it can reproduce, but also how well an organism can reproduce once
In
it
any
reaches
age, really.
also remember that
Any
populations are
era.
developed by natural selection, and not
It's never
individual members of
been
those populations.
never been great.
So what are natural selection's alternatives? Well, we're going to talk about two today, group selection and also artificial selection. So let's start with group selection. And this is the idea that genetic traits that benefit the population or group as a whole will still be selected for even if they don't directly increase the fitness of the individual with the trait. You know, words like altruism and and traits that relate to these ideas are what we're talking about when we think of group selection. And these traits can still be and it's a combination of an organism's ability to survive to an age where it
So
can
natural
reproduce
selection
those populations.
is a
So, still
type
be
of
selected for even
selection,
if they don't
but
directly increase the
then
fitness. So let's start with group selection. And this
there's
is the idea that genetic
other
traits that benefit
types
the
of
population
selection?
once it gets
What?
to that age.
I've heard of it about this. What? No, I think I know where they're going with this. Like, it's not always about fitness, because you can't always consider fitness in every single scenario. Sometimes random shit happens. And you just can't account for random shit.
Also remember that populations will evolve by natural selection, and not individual members of those populations.
Mhm.
So what are natural selection's alternatives? Well, we're going to talk about two today, group selection and also artificial selection. So let's start with group selection. And this is the idea that genetic traits that benefit the population or group as a whole will still be selected for even if they don't directly increase the fitness of the individual with the trait. You know, words like altruism and martyrdom come to mind, and traits that relate to these ideas are what we're talking about when we think of group selection. And these traits can still be selected for because entire populations evolve, not just individual members. So let's look at an example. Let's say a female human has children, and her children have children of their own. Why is it that this female grandmother is able to survive after she becomes unable to have children of her own? Let's say when she's already gone through menopause, which is when the female reproductive system shuts down. You know, any traits that would allow a human to live past this age couldn't be selected for by natural selection, since by the time those traits manifested, the person would have already lost their ability to reproduce. Well, it turns out that grandparents play a distinct role in taking care of their grandchildren, and since their care increases the
Big
survival
brain.
rate and thus the fitness of their grandchildren, this helps the group as a whole, and those traits that benefit survival until old age can then still be selected for by group selection.
Or big
So
Gino.
natural selection
I don't
help with survival until the age where
Just
reproduction
like,
is possible, but group selection
yeah.
accounts for all of those other traits that might help with survival after reproduction is no longer possible. So we talked about the first alternative to natural selection, and this was group selection. But what about artificial selection? Well, in order
Interestingly, from my understanding, there is evidence that homosexuality is important to multiple different types, multiple different species, specifically for a similar effect as like that grandmother taking care of their grandchildren situation. Like, a homosexual member of a family unit may help other siblings become more fit to pass on their genetics. So it's, you know, that's a really cool thing. Okay, so the stream is one half because YouTube, they only allow you to save live streams that are 12 hours long. So this is one half of a 24-hour stream. This is only going to be 12 hours long. Like this specific live stream, this YouTube link, is going to be 12 hours long, and then we'll switch to another one as soon as 8 a.m. rolls around. So we're almost there, and I'm almost going to be able to play Death Stranding. Let's go.
to find out what artificial selection is, let's take a step back and look at natural selection one more time. Remember that if we have a strong individual who's more likely to survive because of his or her strength, then that trait is said to be passed on to offspring more frequently than another trait. And this selection is said to be natural because it all has to do with the idea that the stronger person has a greater probability of surviving than someone else. Let's say a 75% chance instead of a 50% chance of surviving. There's no outside individual who's deciding and selecting for which traits are better than others. It all happens naturally. But that's exactly the difference between natural and artificial selection. In fact, some people call artificial selection unnatural selection. And let's explain this by jumping right into an example. If you have a farmer growing tomatoes, and some tomatoes grow bigger than others, then that farmer can literally select and choose which tomato seeds he uses to plant tomatoes next year. He's artificially selecting tomatoes for those that have a trait which makes them grow more fruit. And that's one of the main traits of tomatoes. Another great example of artificial selection is when scientists in the lab look at tomatoes'
It's
DNA
literally
and
just
select
like
for specific
artificial
traits
selection
in order to
is
give the farmer
selection made by
now, both of these
humans.
examples, since an outside being is selecting which traits are desired, we
So
say that the
silly,
tomatoes are
man.
undergoing artificial selection. instead of
Humans
natural
too
selection,
OP.
because it doesn't occur naturally. It's not just a matter of probability. So what did we learn? Well, first we learned that natural selection is not the only force driving evolution. We have group selection, which is the idea that traits benefiting the group over the individual with the trait can still be selected for. And we also talked about artificial selection, which is where an outside individual can literally choose which traits
When
within
one
the population will
species
pass on, instead of that
evolves
selection occurring naturally.
to force others to evolve. Oh, that's interesting. Humanity is so cool, man. Humanity based. Genetic drift. Let's go.
We've already made several videos over evolution, and just to remind ourselves what evolution is talking about is the change in heritable traits of a population over generations. And a lot of times you'll hear people say evolution and natural selection really in the same breath. But what we want to make a little bit clear in this video is that natural selection is one mechanism of evolution. It's the one most talked about because it is viewed as the primary mechanism.
This is what I was thinking about in terms of random shit.
So there's natural
Genetic
selection,
drift.
There is genetic.
Oh my god! Okay, wait.
This idea that you have variation in a population, you have different heritable.
I'm gonna pause this. We gotta change it up. I can't listen to a Xenoblade music while we're listening to genetic drift. Like, come on, man. Ain't no way we're gonna do that. Ooh, I can hear the rain from outside. Awesome. This is also the other reason why I wanted to play Death Stranding as opposed to Signalis. That, and because Signalis could be a game that I play with someone else, kind of like with Resident Evil, but I don't know. Might not do that. I might just do Signalis as a break through my MCAT streams, so that works too. What am I? Yeah. This is what I need to listen to while we talk about genetic drift.
Heritable traits. I'm going to depict those with different colors here. We have a population of living circles here, and they could come in blue or maybe magenta. Maybe they come in another variation too. Maybe there's yellow circles. And natural selection is all about which of these traits are most fit for the environment so that they can reproduce. So there might be something about being, say, blue that allows those circles to reproduce faster or to be less likely to
blue,
double even though they're slightly more likely to reproduce over time, over many generations, their numbers will increase and dominate, and the other numbers are less likely to, or the other trait is less likely to survive.
Oh my god! He's
for that blue trait. So this is all about
they're genetic
drifting. Now, genetic drift is also change in heritable traits.
Holy shit!
For an environment are the ones that necessarily survive. Genetic drift is really about random changes. And a good example of that I have right over here that we got from
lol random.
This is from OpenStack's College Biology. And this shows
lol,
so right over here,
the brown bunnies died, but the white ones did not.
And we have the gene.
Hey yo, wait a minute.
Color.
Wait a minute. Wait.
Versions of that gene, or we call them two alleles. You have the capital B version and you have the lowercase b. And capital B is.
Wait a minute.
Example that we're showing here. And so if you have two
interesting.
two of the white alleles, you're going to be white. If you have two of the brown alleles, the capital B, you're going to be brown. And if you're a heterozygote, you're still going to be brown. So as you can see here, there are several heterozygotes in this fairly small population. But if you just count the capital B's versus the lowercase b's, you see that we have an equal amount of each. And so the frequency, if you were to pick a random allele from this population, you're just as likely to pick a capital B than a lowercase b. And even though the phenotype you see a lot more brown, but these six brown here have both the uppercase B and the lowercase b. Now let's say they're in a population where whether you're brown or whether you are white, it confers no advantage. There's no more likelihood of surviving and reproducing if you're brown than white. But just by chance, by pure random chance, the 5 bunnies on the top are the ones that are able to reproduce, and the 5 bunnies on the bottom are not the ones that are able to reproduce. And you might be saying, hey, why did I pick those top 5? Why I didn't pick them? I'm just giving an example. It could have been the bottom 5. It could have been only these two, or the only 2 white ones were the ones that were able to reproduce. It's by pure random chance, or it could be because of traits that are unrelated to the alleles that we are talking about. But from the point of view of these alleles, it looks like random chance. And so in the next generation, those 5 rabbits reproduce, and you could have a situation like this. And just by random chance, as you can see, the capital B allele frequency has increased from 50% of the alleles in the population to 70%. And then it could be another random chance. I'm not saying this is necessarily going to happen. It could happen the other way. It could happen even though that first randomness happened. Maybe now all of a sudden this white rabbit
purely random. Let's go.
Maybe these 2 brown rabbits that are homozygous for the dominant trait are able to reproduce. Once it has nothing to do with fitness. And
no systemically induced probability.
Gone from the environment. And the reason why this happened isn't because the white allele somehow makes the bunnies less fit. In fact, it might have even conferred a little bit of an advantage. It might have been from the environment that the bunnies are in point of view. It might have been a better trait, but because of random chance, it disappears from the population. And
Yeah.
the general idea with genetic drift. So once again, just to compare, natural selection, you're selecting
It's a
certain
random
environments,
group of
selecting traits that are more favorable
bunnies
for
in
reproduction. While
the
genetic drift
like
is random, changes
Antarctic
changes
or
in
something.
the population. Now, as you can imagine, I just gave an
Just
example of 10 bunnies.
a bunch
And
of
what I
brown
described is
bunnies.
much more likely to happen with small populations. So
Small
much more likely,
populations.
more
Small population shout out.
with small populations. If I and we have videos on
Because with larger
the
populations,
likelihood of this
it's
happening with 10
just
bunnies versus the likelihood of what we just described happening
like
with 10 million
it's
bunnies
far
is
less
very different.
likely
It's much more likely
to
happen with a
have
small population. So a lot of the context of genetic drift, or when people talk about small populations, in fact, many times biologists are worried about small populations
and
because of
shift
genetic drift.
like
For random
this.
reasons, you can have less diversity, less variation in your population, and even favorable traits could be selected for by random chance. Now, there's two types of genetic drift that are often called out that cause extreme reductions in population and significantly reduce the populations. One is called the bottleneck effect. Let me write this down.
Yep, bottleneck.
The bottleneck effect, and then the other is called the founder effect.
Wait,
The founder.
wouldn't this be included within genetic drift?
They are both ideas where you have significant reduction in population for slightly different reasons. Bottleneck effect is you have some major disaster or event that kills off a lot of the population, so only a little bit of the population is able to survive.
This should
The
be
reason why it's called bottleneck is imagine
branching off of
you had a bottle here. So inside of that bottle, you
off
have
of
marbles of different colors.
Drifto.
So you have some yellow marbles, you have some magenta marbles, you have some blue
Right?
marbles.
I think.
Some blue marbles. So you have a lot of variation in your original
It's like
population.
genetic
So
drift
you
is
start
just
pouring them
defined
out
by
of
the
a bottle.
random
Maybe
chance.
some
It's
of them
not
have major disaster, and only two
not
of these
like
survive.
any
Or
more
say only four of these
complicated
survive.
than that,
So
I
you do that
think.
is, well, what are the marbles that are getting poured out of the bottle? It's really just a metaphor.
And then these two also have
But out of that disaster,
random
only a handful survive, and they might not have any traits that are more
elements.
desirable or more fit for the environment than everything else, but it's by random chance because of this disaster. They are the ones that survive, and so all of a sudden you have a massive reduction not only in the population but also in the variation of that population, and many alleles might have even disappeared, and so you have an extreme form of genetic drift actually occurring. Another example is founder effect, which is the same idea of a population becoming very small, but the founder effect isn't because of a natural disaster. It's, let's say you had a population, once again, you have a lot of different alleles in that population, you have a lot of variation.
Yeah, and then they just kind of leave. They're like,
So
I'm a head out
you have a lot of variation in this population.
and found
Let's say
my
they're all hanging
own
out in their
species.
community. They are surrounded by
Okay.
mountains, and I'm just making this up as I go. But let's say a couple of
Okay,
these blue characters were out
Very
walking one
cool.
day, and they maybe get separated from the rest of their population. Maybe they discover a little undiscovered mountain pass, and they go settle a new population
Whoopsies.
someplace. So that's what's called the founder
New
effect. These are the
species.
founders of a new population, and once again, by random chance, they just have a lot less variation. They're a smaller population, and they happen to be disproportionately or all blue in this case. And so now this population is going to a few you might have already had. This is just the process of this was genetic
After
drift,
many
where you have
generations.
many alleles will have disappeared because you have such a small population of blues here, and also because you have a small population, you're likely to have even more genetic drift. So it's a really interesting thing to think about. Evolution and natural selection are often talked about hand in hand, but natural selection isn't the
Because
only
they might-
mechanism of evolution. You also have genetic drift, which is really about not
Yeah,
selecting
they might
favorable
still
traits.
be the
It is
same
about
species. But for like a few generations, and then later on they're gonna be like, oh my god, I can't I can't have kids with that other group across the mountains. What's up with that? What's with that? Guess we ain't species anymore. Bruh. And then there's ligers.
So
Then it
today we'll
gets
talk about
confusing.
inbreeding and how it's not really the best for a population. So before we do that, let's review the concept of natural selection. And natural
Damn.
selection is the idea that
We're gonna
population is a
we're gonna do an
small
entire
is more likely to live to an age where they can reproduce and
oh, it's a
pass on
4-minute
special
video,
traits.
but still, an entire video dedicated
Having a big gene pool.
Alabama.
The more genetic diversity of the population
Yo!
allows the group to adapt.
Alabama shout out.
So what is
This is
inbreeding?
great.
Well, inbreeding is when people in a population will selectively have offspring with a certain smaller group within that larger population. And this can be for a bunch of reasons, like religion or culture, or maybe just because of preference. And when inbreeding occurs with non-human populations, it's almost always due to geographical barriers, where the greater population simply isn't accessible. Now, when people usually think
Yeah.
of inbreeding, words like incest come to mind. But inbreeding really isn't limited to members of the same family having offspring together. Now, lots of small
It's true.
religious and cultural groups in the world have some people with common ancestors and are only distantly related. So you can see that the effects of inbreeding can exist without close relatives actually having children together. So why is inbreeding a problem in the first place?
And then
Well,
sometimes with
so
like
Tay-Sachs disease is an autosomal recessive disorder. And what that means is that people with no copies of the genes are unaffected by the disease. And I've drawn these people in blue.
whatever.
People with just one copy of the gene are not affected by the disease, but are carriers for the gene.
Inbreeding
And I've drawn
genetics
these in
is
red.
like
People with two copies of the gene are affected by the disease. And I've drawn these
weirdly
in red.
complicated.
Carrier for Tay-Sachs. So here's just one copy of the gene. If we're
Ay,
looking at
ay,
the
ay.
general population, we can see that the odds of the person choosing a mate that's also a carrier for the disease are pretty low. And if he eventually has some kids, none of them will be affected by the disease, and only a few will even be carriers. It's likely that the copies of the gene will be so spread out among the population that it would be quite rare for two carriers to actually end up mating together. Now, if we look at an inbred population where a bunch more people could be carriers for the disease, the chances of our guy choosing a mate that's also a carrier are
Oh my god, they're all
so
anime
it's actually
characters.
a disease, but there's also a chance that some of his offspring who
Oh,
have copies of the
shit.
disease could actually be affected by the disease. Now, we just talked about an example with an autosomal recessive disorder, but maybe you're wondering how inbreeding affects autosomal dominant disorders. Well, let's look at Huntington's disease, which is autosomal dominant. And since this disease is autosomal dominant, the person has no copies of the gene and they'll be unaffected by the gene, and I've drawn these people in blue once again. However, if a person has either one or two copies of the gene, then that person will be affected by the disease either way, and I've drawn both of these people in red. The key difference in this case is that no matter who the guy has children with, even if the guy just has one copy of the Huntington's gene, there's still a chance that there will be children affected by the disease. Now, of course, if our guy has children with someone who is also affected by the disease, then
Our
more of his
guy.
children will be affected, but there's still a chance either way.
This guy,
Now, one of the
this
other
random
aspects that
dude.
is concerned about inbreeding affecting autosomal dominant diseases is that carriers for dominant disorders are generally aware that they're affected, and are well aware of the risks of them having diseased children.
Okay.
For recessive disorders, carriers usually don't have any symptoms at all, and they may not even know that they're carriers until they've had a diseased child. And this makes it much more important for people in inbred populations to seek genetic counseling so that they are aware of the risks of them having diseased children.
Genetic
So, once
counseling,
we
yeah,
learned-
that's
well, first
interesting.
we learned that certain inbred populations can have many more individuals that may carry a diseased chromosome than the general population. But we
I,
also learned that
I
even
don't
most
know where to
individuals
put
have
this on my
autosomal
map.
diseases, since those generally go more unnoticed. And
I don't think. What? Where would? I fail to see the relevance of this. Yes, inbreeding bad. I guess it's related to it's related to genetic diversity. I don't know where to put that. I mean, genetic diversity is like the reason why we have so many different mechanics related to genetics and trying to like cross over. I mean, it's most related to genetic recombination. Like that's the DNA mechanic for, or chromosomal mechanic for creating more genetic diversity beyond behavior. Biological mechanic, whatever mechanism. Mechanism. I'm thinking too much in game terms. But I don't really know if that's appropriate to just put. I don't know why we would. Whatever. I'm just gonna not include that right now. If I find that it's relevant somewhere else, I'm gonna include it. But it's just like, what? Okay. It's not related to evolution, because you're not really like evolving into a new species because
But before we look at that, let's go over the difference between
of
asexual reproduction
inbreeding.
and sexual reproduction.
It's
In
something
asexual reproduction, one organism with a single
something
bacterium
different.
divides into two daughter cells that are both genetically identical to the original cell. In sexual reproduction, two members of the same species will reproduce together in order to form genetically unique offspring. Now, in general, we say that organisms that reproduce asexually usually have low genetic diversity, whereas sexually reproducing species have high genetic diversity. So, what's a species? Now, this can be a very difficult question to answer.
Shoot.
For sexually
Okay, we're
reproducing
gonna talk
organisms, we
about
say that two
species.
organisms,
Let's
like
go.
this cat and this human, are members of different species. They're unable to have offspring together. However, for asexually reproducing organisms, like bacteria, protists, and archaea, it's a little more confusing. These species don't mate with other organisms, so we have a difficult time classifying them into different categories.
They're just
We
like,
call this the species
hey yo,
but in this video, we're going to spend
what
time
is going
on
on?
sexually reproducing organisms. And these are separated into different species by different forms of what we call
Okay.
reproductive isolation. And this is the idea that there are many forces that stop two different organisms from having offspring together. And we can divide these forces into two separate categories: prezygotic forms and post-zygotic forms. Prezygotic isolation refers to all the different forces that prevent two organisms from having offspring together that occur prior to the formation of the zygote. And remember that a zygote is a single cell that is made up of the genetic material of both organisms that reproduce together. Post-zygotic forms
Trying to put a square peg in a round hole. I always remember that. Just like mechanically, you can't do it. It's not possible. Why not? It's just not. It's just not possible. You're just not believing hard enough. Oh god. Okay, well, that's interesting. Isolation. And temporal and habitat isolation is actually uniquely relevant in the video game that we're going to be playing in 10 minutes. Ooh, that would be awesome. The video game. The video game. I keep saying that, like, ironically, but I'm kind of starting to worry that I'm just gonna start saying video game as opposed to game, and it's just gonna be really awkward. Not even gonna be that. Well, it's gonna be cringe. It's gonna be cringe. There's no doubt about it. It's like calling PCs computers. It's like, what? What did you just say? What did you just call that PC? Except that's what I call PCs all the time. Computer. I like the term machine for computers, though. New A dating online. Wow. Amazing. Okay, you're just gonna spam the chat. Alright. Cool, cool, very nice. Thank you for joining the God damn it, YouTube. God damn it. Give me a second, guys. Oh wait, I can actually I can actually moderate my own chat this time. Fantastic. Anyway, I needed to turn off that music anyway. And then we can switch over to Xenoblade again. Get Xenobladed, idiot. Okay, YouTube's just gonna take a split second to get me my mod privileges. And then I can totally wreck this fool who has invaded my chat with spam. How dare they do that? How dare they? Bruh. Shout out. Shout out to this random chatter who is trying to scam people. Very cool. Bruh. I ain't got time for scams, bro. I ain't got time for this. I'm trying to learn. Trying to... Wait, how do I... unwanted commercial content. Wait, I'm a little confused. I've rarely had to moderate on YouTube before, which is kind of sad now that I say it out loud. But is there not a, like, pure ban? Can you not ban people off of your channel? What? Okay, whatever. If it comes back, well, I'll deal with it. Okay. Now that we're okay, Opera, or chill, dude. Chill. I'm trying to like close your window. What's going on here? Wait, what was that? Okay. We're good. I was worried that it was showing my other screen, which should not be seen. Not that it matters because it doesn't reveal anything important, but...
All organisms mate at the same time. Some mate at night while others mate during the day. Some mate in spring while others mate in winter. If two organisms do not find mates at the same time, then they are temporally isolated.
Bruh.
Place where the organisms mate. Some mate for mating
I'm sorry, girl, but like,
don't find mates in the same place, and they're also
it's
isolated.
winter, bro.
If aren't a problem, then the next barrier is behavioral isolation.
Just not sexy, dog.
All organisms go through attracting a mate. Now, not all organisms will attract a mate the same way.
Oh.
One animal, like a bird, will attract a mate by singing.
Amazing. I would sexually reproduce, but it's winter, bro. Like, it's too cold outside. I'm shriveling, dude. Holy crap! My map just sent this this thing into the stratosphere, or maybe into the Marinara Trench. Marinara. What? Marania? Wait, no. The big trench. the deepest trench known to man. Whatever that's called. I always forget how to pronounce it. Isolation. I'm a bio student, not a geography one. Okay, geez. God, imagine me playing GeoGuessr. Oh no.
So we have behavioral isolation, and now we have mechanical isolation, which deals with the physical inability of two organisms to mate, even if they wanted to. Now, a great example of this
there it
is
is.
an elephant being unable to mate with a tiny mouse. If two organisms do mate successfully, they may still encounter
Right.
gametic isolation, which is when fertilization between the two
But
gametes
the mouse
forms a zygote
just.
is impossible. Now, once the zygote
Oh,
has been formed,
no.
we move on and look at post-zygotic forms of reproductive
Oh
isolation.
no, how
And
could
the
you
first
do
form
it?
is zygote
You
mortality.
can't do it
And this
to
occurs when
them.
even if the two gametes from the two organisms will dance to attract a mate. So we have behavioral isolation.
Wait, what?
And now we have
Hold up.
mechanical isolation.
I missed what, the
which deals
last
with the
one.
physical inability of two organisms to mate. even if they wanted to.
Mechanical,
A great
yes.
example of this is a huge animal like an elephant being unable to mate with a tiny mouse. If two organisms do mate successfully, they may still encounter gametic isolation, which is when fertilization
Gametic
between
isolation, that's what it was. Yeah. Mechanical and then gametic. Meme-ic? Are we memeing? Are you memeing? Do you mean it? What is a meme? A pathetic pile of secrets. But enough talk. Have at you. Was
two gametes to form
zygotic.
a zygote is impossible. Now, once the zygote has been formed, we can move on and look at post-zygotic forms of reproductive isolation. And the first form is zygote mortality. And this occurs when, even if the two gametes from the two organisms can fuse successfully and form a zygote, that zygote would have a high mortality rate and be unable to develop into a mature offspring. Next, we have hybrid inviability, which occurs when a zygote is able to grow into a mature offspring, but that offspring will
Hey,
have a high
yo,
mortality
wait.
rate and won't be able to grow into a mature adult.
God damn it.
Finally, we have the last form of reproductive isolation, which is hybrid sterility.
They just don't
And this is when
write
offspring
down
can
the
grow into a
name
mature adult,
of the...
which is when fertilization between the two gametes to form a zygote is impossible. Now, once the zygote
Have
has been formed, we move
symbols,
on to
but not
post-zygotic
the
forms of reproductive
actual
isolation. And the
thing.
first form is zygote mortality. And this
Oh, wait a minute. Oh, that's how they're structuring this. Okay, I can just follow their structure. Like, the rising levels of possible isolation, basically, right? That's what's going on. Okay, I see now. I see. That's actually pretty big brain. Wow. You're doing my work for me. It's fantastic. There's so much lag. I swear I'm not making this map slowly. It's the ping, guys. It's the ping. God damn.
This occurs when, even if the two gametes from the two organisms can fuse successfully and form a zygote, that zygote would have a high mortality rate and be unable to develop into a mature offspring. Next, we have hybrid inviability, which occurs when a zygote is able to grow into a mature offspring, but that offspring will have a high mortality rate and won't be able to grow into a mature adult. Finally, we have the last form of reproductive isolation, which is hybrid sterility. And this is when the offspring can grow into a mature and won't be able to grow into a mature
Inviolability. Right. Oh my god. Come on. This feels like
Finally,
low
the last form of
yield.
reproductive isolation, which is
I don't
hybrid
know.
sterility. And this is when the offspring can grow into a mature adult, but that mature adult is not able to mate and have offspring of its own. So if two sexually reproducing organisms are not isolated by any of these barriers, then we can generally say that they are members of the same species. So what did we learn? Well, first we learned about the species problem. and how
Oh,
classifying
it's
different
3
organisms into
a.m.
different species can be
It's
quite
3
difficult.
a.m., boys.
You know, we have a pretty good definition for sexually reproducing organisms, but
I
we
get to
don't
play
really have a
Death
species
Stranding
for
again.
organisms. And
Get
next,
to
we learned about
see
reproductive isolation, and how we can
my
say that two
boy.
sexually reproducing organisms are reproductively isolated if they
Norman
are unable to
Reedus.
freely produce fertile offspring together.
Okay. Now I have to just close everything. I wonder if like reloading will let me save my progress. Well, is this going to work? It's going to do the thing that I hope that it actually does. That would be very cool. Very cool. Okay. Hopefully that worked. All right. Death Stranding time. Death Stranding time. Oh, wait. Missed opportunity. Returns. Return. Norman Return. Oh god. This is comedy now? Is this what comedy means after streaming for 7 hours? Oh man, I'm scared for what happens at 8. Like, it's gonna get crazy. Gonna get wild, boys. Wait, what? Why is it up there now? What? Okay, that's weird. Hold up. It's like stuck up there somehow. Why would it even do that? That doesn't make sense. Right, enough memes about Norman Reedus and returning. Norman meme this. Okay. Continue. Continue the game. Okay. And then when we're done here, it's gonna be 1/3 of the way. Let's go. This is actually kind of hype. I kind of like this. It's I might like long form streaming. I don't know. Or will I regret those words? Find out next time on 24-hour live stream with Norman Reedus. No, I'm just gonna chill. Wait, now it's loading really fast. What is this? Okay, never mind. It's stuck at 21. This is what happens. This is what. Be right back. All right, let's freaking do this.
Beginning scan. Scanning bridges ID. Verifying ID. Clear. All clear. Welcome, Sam Porter Bridges.
Welcome. Cue cyberpunk music.
Beginning scan. Have a pleasant journey.
I will. It's gonna be awesome. It's time to transport morphine to the Bridges isolation ward. Also, Xbox is telling me that I have free stuff. Very cool. I love free.
On your cufflinks if you're lost, Sam. The isolation word should be pretty obvious.
Hmm.
If you're not sure how to get there, you can set markers and draw routes too. Try it out and you'll see what I mean.
Usually I never like mark in-game maps, but ever since I've started grinding through Genshin, I mean, yeah. Gotta start marking stuff on the map or else I'm just gonna like forget where the hell everything is. Alright. Come on, Sam, let's go.
Sam, if you want to
Yes?
confirm you're headed the right way, check your compass. It'll never steer you wrong, provided you have a destination.
I have a feeling this is not the compass. Wait, what? Wait, what? Okay. And then Sam just stops running. Very cool.
Central wasn't just home to Bridges HQ. It was also the seat of the Reconstructionist movement. If the president hadn't required special treatment, she would have never left.
Why does audio gotta load first? Always sad. See? Oh wow. My hoodie might actually be worse than my hair at... It's staying within my green screen. Jeez. Is this even playable at this point? Like... Ah, if it's not, then I'm just so disappointed. It was working earlier. It was fine. This what? Yeah. Oddly enough, someone else asked that exact same question earlier. Yeah, it's my first time playing Death Stranding. Always been interested in the game. Never ended up touching it. I have Game Pass now. Or at least for a little bit. Okay. That was interesting. And now I'm able to play it in lieu of the Death Stranding 2 announcement. Or DS2. Who knows, it might be Dark Souls 2, for all we know. Damn. You good? Okay. Wasn't sure if he was hearing things or something. I don't know. Oh, so now the cutscenes are working. I think I just jinxed myself again. Whatever. Yeah, it's most likely I'm just pushing my hardware too hard because this game is probably not meant to be played and streamed on like what, how old is my laptop? Like 7 years old? On a 7-year-old laptop? I don't think it's that old. 8th gen Intel Core or Intel CPU, I mean. I don't know.
Like I said, her vital signs are all trending the same way.
No chance of a recovery then?
None that I can see. No.
We should start making preparations.
Sam, it's me, Dead Man. Sorry, my mistake. I'm afraid the president's condition has deteriorated. Thank you. This will help to ease the pain. And allow her to speak with you in these final moments.
Oh, I just realized that he's wearing a stethoscope. I was wondering what that gold thing is. Why is it shaped like that? I like how he's like Hey, remember, it's... Okay, wait, I'm gonna be right back, though.
You're not exactly dressed for a reunion. Take that off before you come inside. That's the president's right-hand man, the director of bridges.
Die Hardman?
Oh, you were with Bridges 1? So, I guess no introductions are necessary.
Wow, okay, nice, I was muted.
10 years. Look at us. A bunch of deathless freaks meeting like this.
I was just commenting on your appearance as soon as I started up the game. Makes me wonder if YouTube's like promoting it or promoting my stream as soon as I open up the application. I don't know.
She's a bit out of it, but I know she'll recognize you.
Kind of doubt it. Maybe I'm just getting lucky. Or rather, you're getting lucky because this is only 1/4 of my stream.
We've brought Sam. We'll leave you two alone.
Sam, I knew you'd come back.
Come on, cutscene, you can do it.
Are you doing all right?
Okay, I think audio's synced up. Yes. Alright, awesome.
Amelie. You remember Amelie went west. Took her 3 years to cross. She's trying to rebuild.
Hope it's not gonna take us 3 years to cross.
Still going on about that. Huh?
Damn.
You're the one I wanted to send, Sam. Time's running out. Help Amelie. She needs you. Together, you can help us reconnect. You can make America whole. Sam, if we don't all come together again, humanity will not survive.
We don't need a country. Not anymore.
We do. Alone, we have no future.
No, America's
Wait,
finished.
sodium it's just
You're the
president.
Just salt.
Sam, listen to me. Please.
So I have seen this. Okay, I've seen like a decent amount of cutscenes. I don't know how far into the game, but I've always been curious about this. Does she know about his phobia? Is she reaching out for him regardless? Or I guess she would do this regardless of whether or not he has his condition, maybe.
I love you, Sam.
She is in her death throes. Oh, my hair.
I'll be waiting for you on the beach.
Madam President! Oh, no.
Doctor, we need your help.
All hands on deck here. She's in V-fib. Give me 150 cc's. She's not responding.
Stay with me. Madam President.
She's dropping.
Mhm.
Hold on.
What was that noise?
We're losing her.
Probably the medical equipment.
I'm a death.
Rip. Be stranded with love.
Listen,
Interesting.
no one can know that the president is dead. If word gets out, Bridges is finished. Now what happened here does not leave this room. Do you understand?
Yes, Director. But there's still the matter of the body. Without corpse disposal.
We've got that covered. Sam, before she died, the president made a contract with you.
What are you talking about?
As a member of Bridges, you're gonna work with the rest of us to rebuild America.
You think you can recruit me? Like she tried to?
Well, she succeeded. Look at your wrist. If that's not symbolic, I don't know what is.
Director, the cancer spread throughout her entire body. Harvesting organs is out of the question, and there is no need for an autopsy. Her body needs to be cremated before she necrotizes.
And if we don't, this place will turn into another crater.
What do you want me to do?
We don't have any porters right now. And the CD team you went into the field with are dead.
Yeah.
But the president's body has to be burned.
The road from Capital Knot City to the incinerator was compromised in the void out. Now the only way there is on foot, through the mountains. But the chiral density there is off the chart. It's gotta be BT's.
This job has requirements. Tombs. Repatriate.
So me? Why?
Sam, you're already on the clock.
Now get it done, Sam Porter Bridges.
Right, time to transport the corpse of our mother. Still amazed at how hard this game goes, like, from the start.
Can I have a moment? President Strand believed in American Reconstructionism. She worked tirelessly to bring the nation together again. And without her, there would be no bridges. She deserves a funeral with full honors. But we can't give her that. If she dies, America dies.
Without her, bridges will cease to be.
Listen. Her cremation must be carried out with the utmost secrecy.
I like how he seems to be lecturing Sam, even though he's the last person that would ever say anything about it.
America isn't dead yet.
He just said that it dies if Bridget dies.
She may be lost to us, but we still have an America worthy of the presidency.
Sorry, what?
Let's not get into it now. What matters is that we're going to finish your mother's work and rebuild America as she intended. That's the reason Bridges exists. So take the first step, Sam, and deliver the president's body to the incinerator.
If we don't all come together again, humanity will not survive. You're the one I wanted to send, Sam. Time's running out. I love you, Sam. I'll be waiting for you on the beach.
The facilities to the southwest. Remember that you'll be passing through BT territory.
We built the incinerator way up in the mountains so that the chiral matter in the smoke wouldn't reach the city. It won't be easy carrying a body up there.
Could have made a road. Could have made a road up there. I'm just saying, like, it's their facility. If this was further out, then that point is moot. But just for the one that's right over the mountain, guys couldn't have built a road for that one. Ah, whatever. We'll get there. Come on, Sam. Let's go. What hunky-dory quest we're on. Oh shit. Okay. Oh yeah, his name is Die Hardman. Is that actually referencing the movie, or is that just something else? I wonder. Oh, it's hard to like respond to the weight shift appropriately.
I don't think I need to remind you, but every minute counts when transporting corpses for disposal. 48 hours following cardiac arrest, they go necro and become a fresh BT to make our lives hell.
All right. We got 35 minutes to transport our mother's body up a mountain. Let's do it. All right, cool. Corpses are uniquely balancing, which is interesting. Actually, I'm curious about the icons in the bottom left. What is that? Oh, shoot. Oh, God. No, Sam, I just wanted to look at you.
Keep it together.
Yeah, sorry, man. Just want to look at you. This is definitely not one of those games where you want to, like, change the camera angle. Oh.
twins, she never stopped believing they could be reunited. People held on to their hope that America would rise again because they believed in her.
Wait, where am I going? Oh, I think it's that thing on the top of that mountain. How do I So what does that do? There we go. Okay, I see. So there's two mechanics that will allow me to pinpoint where I'm supposed to go. Well, there's one mechanic to pinpoint where I need to go, and then there's a separate mechanic for pinpointing where I'm allowed to. Alright. So by holding the cargo with both hands, does that slow me down, or does that allow me to just like stay stable for the longest amount of time? Ooh. The Strand-Type gameplay is starting to come into play. Let's go. Survey the surrounding area from a high vantage point. It's the function available at various structural levels. Okay. Well, let's see how it works. Okay, we see a ladder up there. And then... Okay. Oh my god. For some reason, this camera's not inverted. What the heck? Okay, whatever. Oh, well, since I used your, watchtower, you get a like. It is a good day. It's a very long day, but it's a good one. Oh, I don't even need to set markers. As soon as I see it, I get to profit off of the information. Getting into a rhythm. Chilling. So what's up with these? What is this about? I can't pick it up yet. Oh, okay. That's unfortunate. I even put a marker down. Aw, okay. Well, so much for that. I'm finally using my middle fingers for something. Oh, shit. Okay, wait. Hold up, hold up, hold up. Remix, remix, remix. Gonna have to mute the game for a bit while that music plays. Choosing the shortest way there is kind of not great. Gonna go around. This music is a lot louder than I expected it to be. Did not hear who was saying Sam, Sam, Sam super clearly. Wait. Oh, wait. Was I supposed to cross that? Wait, where am I going? Okay, there it is. Man, this game is definitely meant to be binged though. Longer playthroughs. Shame it's just gonna be an hour. Hour segments during the stream. It is what it is. Oh, is that Sam saying that to himself? That's so motivational. Wow. Okay, I see. Interesting. There's like a staircase over there. Oh man, why is this not inverted? That's so annoying. Okay, climbing anchor. Oh yeah, he is. You bet he is. The man with a plan. All right, I can turn the music back on. Whoa, 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 whoa. How do I? Not music. The sound. Still am locked out of the bathroom. Fantastic. Okay, well, whatever. He's using all of that upper body strength. God damn. Thank you, Igor. You can release the rope now, Sam.
Just a bit further now.
Wait, yeah, how? Wait, where am I going? Oh, I want to go that way, right? Very interesting mechanic. Tell me where I'm allowed to step. It's pretty cool. Oh man, Igor's my man. Really pulling through for me.
This one's for you.
Oh, whoops. Have to grab onto it first. There we go. Igor pulling through. Wait a minute. Now what? Okay, I guess I can climb up this way. Seems like a little complicated though. I think I can climb this way. This seems like this might be like an inefficient route. Oh, well, it's the route I'm taking, so see if I can get up there. Oh my god, Sam, do this. Okay. There we go. Making my way downtown, walking fast. This seems like this might not be the way. Oh, maybe. Maybe. I can climb over this little ledge here. Nice. Alright, we made it.
So that's the place.
Oh. All right. Yeet. Get out of here. R.I.P. the president and our mom. Oh, he does care. Well, there she goes. Mission accomplished. Interesting choice to name the element Chiralium.
Chiralium density in the area is climbing rapidly. Heavy timefall is imminent. Get back here, now.
Especially considering in like
Burn the other cargo while you're there.
like OChem. It's like basically molecules that mirror but can't overlap.
What's this?
Oh wait, am I thinking of a different thing? I'm thinking of a different thing.
But why is it here?
Because it's flagged for disposal. Central Knot's gone, and our headquarters along with it. Because that thing didn't do its job. It's all in Igor's logs.
But it's still alive.
It can't be repaired, and it can't survive outside the pod. Pity it if you want, but the decision has been made. It has to go. The director approved the order.
Dang, they're making holes into the concrete? Holy shit. These things are relentless.
Sam, do you copy? Damn it, must have lost you in the blackout. Sam, what's your status? Chiralium density is still increasing.
My status is fucked. There's tons of them.
Must be from the bodies we couldn't get up to the incinerator in time. Comes with the territory, I'm afraid.
Can you sit out the rain?
No. They're closing in on my position. I have to move before they realize I'm in here.
Sam, if one of those things eats you, it'll trigger a void out. You'll come back, sure, but the surrounding area will still be a crater.
Why does he come back? I
got an idea.
always get a little confused. Right. Baby is here. Let's go.
Maybe you are defective.
Ooh. Let's go. There we go. This is hype. Baby activated. I think he just winked at me.
Keep low. Move cautiously.
Okay. How in the world am I gonna do this? Okay, wait, I'm a little confused. I just move away, right? Wait, I don't understand. Wait, I'm a little confused. Hold up. Wait, I don't get it. What am I supposed to do? Oh, here we go. Is there no way to, like, crouch or something, or... I guess that's not part of the stealth? I don't get it. Okay, I'm out of here. Screw this. I don't really understand what's happening, but we're going. We're leaving. We're sprinting. We're gaming. Oh my god. Sam, please. Let's get over the mountain, Sam. Come on, we can do it. I don't know what's going on, but the soundtrack is kicking in, which is bad. That's a bad, bad thing. Oh god. Oh no, 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 no. It's coming, it's coming, it's... Oh shit. Oh lord, it's coming. Come on, Sam. Jump. Jump, Sam. Jump. Go, go. Come on. Oh my god. Why'd the camera just suddenly... I nearly jumped off a cliff because of that. That was messed up. Okay, I'm not really clear on how I got away from that, and I'm not really sure how to avoid that. But, hey, it worked out. And we got fish for days. Sashimi time. Ooh, sick roll. Select canteen. What, cause am I out of energy? Looks like I regained it, right? Wait, where am I going? Where am I trying to go? I go that way. And then loop around. Oh, well, that was an experience, but, one that wasn't too crazy, eh? Too undoable. Just gotta run harder. I learned that the hard way in our Stellar Glow playthrough. Wait. Missed my chance for a cryo bite. Got him.
So bad.
Aw. Don't ruin my immersion, dude. I'm sure they're very tasty. Oh man. Will walking through the rivers clean off this goop? Or is it just gonna cause me to turn into an old man? Voice crack. Time to take fall damage. Fall damage time. Oh, hi. More like DMCA time. No, 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 stop, 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 stop. Ah. There we go. Yeah. Man, this was the right game. I'm glad I chose this game for this 24-hour stream. So relaxing. Or wait, am I supposed to choose a game that would make me stay awake? I don't know. Oh yeah, I get to test this out. Does this clean off the goop then grind? Seems like it does. Oh, that's cool. Except I can't soak Sam's like upper body, I guess. Unless I really go deep diving. Oh, this reminds me of Xenoblade 3 has like a dirt and grime system, which is so wild. Do not expect that for that game. Because like you'd think most resources and priorities would be set for other parts of the system, but no, they have a you collect grime on your characters and you have to clean them off at a campsite. Super cool. Oh, probably want to. Oh, okay. Wow. Oh, is that to avoid me getting stoked? So that the time fall doesn't affect me? Still not clear on if the time fall is affecting the water there. Well, I don't know. I have to close the game in 6 minutes. Gotta make sure I don't trigger a cutscene. Or maybe I can. The cutscenes are long. These are some long cutscenes, boys. Long ass cutscenes. That's alright. Yep. 42,000 inhabitants. Hmm. Don't think I need to. I can see it right there. But thanks, Die Harmon. Okay. Pause the game real quick. Right back. Boy, howdy, I love it when my roommates close the frickin' door, making it look like someone's in there. And the fact that the door can get stuck a little bit. It's fantastic. Fantastic.
It's a miracle either of you made it back. When someone with dooms hooks up to a BB, your feelings and memories cause feedback, like with the speaker. It amplifies fears and stress, and runs the risk of taking you to a place from which you will not return. I'm sorry, but this BB has been pushed beyond its limits.
Sam, get cleaned up and come to the president's office. We need to talk.
Don't worry. It's okay. I'll always be with you. See the sunset. The day is ending. Let that yawn out.
Audio got desynced again. I don't think it's gonna resync though.
There's no
What? The kid's bubble is a heart? What in the world? How do you even do that? Hmm. Cool, cool, cool. All right. I'm doing good. I'm doing good.
Sam, I thought I told you dooms and BBs were a dangerous combination. Huh, perhaps a shower is in order. You're still covered in chiralium. Don't want the president to see you like that.
The hell you talking about? She's dead.
You're upset. Is this about BB-28?
No, he's just confused. She literally just died.
Well, in any event, it seems it owes you its life.
No, we owe the kid our lives. You too.
As you say, I shall look after it. Give my regards to the president.
Again, she's dead. And if there's another person, then we did not elect her. Alright. We'll let the cutscenes ride, just because it takes forever to load anyway. And then I'll grab something to eat, and we'll study for a little bit longer.
It's almost time to go, Sam. Here. It's a dream catcher.
And
Wear it when you sleep, and I'll keep the nightmares away. I'll always be with you.
I can't tell if it's desynced or not. I feel like the video is just ending as soon as the audio ends, even if it's lagging, which is super weird. Not a fan of that, but whatever.
Wash all that chiralium off, then come to the president's office.
Oh my god, I have to take a shower? What? I don't know how to do that. Hey, what's up? Can I not pull up the menu from here? Can I not choose to save? Can't save. Okay. Yeah, screw it. We'll just take a shower. Oh man, those scratches on his feet look painful.
For fuck's sake. Is it anything private anymore?
Oh, no. Except for taking a shower and going to the toilet, that is.
Fantastic.
Good. Come to the president's office as soon as you're done. She's waiting for you.
Okay. It's time to reunite America. It seems like in-engine cutscenes are totally fine, but for some reason, my laptop, every time it's like a pre-rendered cutscene, it's just lagging. It's just not there. It's not ready. It's struggling. Every time.
You did well, Sam. Thank you. Bridget may no longer be with us, but her legacy has a chance to live on. Sam, listen to me. America, Reconstructionism, her dream isn't dead.
I don't want to hear it.
This is the face of our new hope, our new America.
Homily.
My mother may be gone, but I'm here. And you, Sam? You're here too.
Been 10 years since you saw each other, right? And in all that time, she hasn't aged a day.
He knows why. My body's still on the beach. I don't get to grow older. But you do, Sam. You look good.
He says that with a lot of aggression. You look good. Damn. Okay.
Someone has to succeed Bridget, Sam. More importantly, someone has to carry on.
Just realized her name's Bridget because Bridges.
Samantha America Strand. Our new
Her middle name is actually America.
For our nation.
Yes.
Leadership will re-establish the UCA, the United Cities of America. This is how we'll rebuild our country. But we'll need your help to do it, Sam.
No, I'm through with this. I said my goodbyes to all of you when I said them to Bridget.
We never forgot about you, Sam. You ran away. You cut us off.
Emily put together an expedition. The best of Bridges won. And went west.
This is cutting into my study time. Oh boy.
I led them past craters in BT territory. I kept them safe while they searched for survivors.
Oh well, I have to cut the playtime short next time. Next time we get to play Death Stranding, which is going to be in 4 hours or 3 hours.
We convinced community after community to join the UCA, and we left behind our own people to help them. It took us 3 years, but we managed to make it all the way to Edgenot City.
All the way to
California, baby.
Jesus.
Yeah, but then everything went to hell. The team was wiped out, and Emily was taken.
But why?
Taken.
I'm not being kept in a cell or anything like that. I'm allowed to use their facilities and to speak with you whenever I want. I just can't leave the city.
It's all to safeguard the continued independence of Edgenot City. Well, that's what our keepers are saying, at least. They want the UCA to leave him be. Emily's their insurance policy in case we don't.
Who are these assholes again?
Homo Demons. Militant separatist group. They run Edgenot City.
I've heard of them. Bunch of terrorists who go around towns killing people and leaving craters.
Right. Nothing's off limits for them. Not even engineering void outs.
You think the suicide?
It's wild.
Not was one of theirs?
Could be. There are extremists like the demons everywhere, plotting in
They're just triggering like nuclear explosions everywhere they go.
No one organization to rule them all. Just a common ideology.
I know not everyone shares our vision for the future. If we Americans don't come together again, humanity won't survive.
Really does make me wonder what the rest of the world looks like.
I carried my mother's message to people all across the country. But not everyone was willing to accept it.
More than a few would rather stay isolated. Keep to themselves. Go it alone. Like you, Sam. They think that America can only be rebuilt by force, by men who tell them what to do, who take away their freedom and put them in shackles.
America's all about freedom.
You know better than the demons. Just another kind of cult.
They're not shackles, Sam. They're a symbol of our bonds. That's what we need right now. Not to stand apart, but to come together, to form chiral knots and reconnect.
Sam, we want you to go west and finish what Emily started. The people she left behind have been hard at work, setting up chiral network terminals. But these terminals are still isolated. We need you to bring them online. And for that, you'll need a Cupid. This contains all the necessary security and operations protocols to integrate a terminal into the chiral web. Take the Cupid west, Sam, and reconnect the people of our great nation.
That floating data to though.
Find Emily.
Like, why is it floating? Is it because like data is somehow the equivalent to magic in this universe?
I'll take over the presidency and carry my mother's work to save the country.
This was Bridget's dying wish. This is how we'll move forward.
Please, Sam. We need you.
I'm Sam Porter Bridges now. I'm not a Strand. Hell, I'm not even part of this outfit. You all saw to that. I'm not getting involved with you, or anyone else, ever again.
Wait!
See? It's like I'm not even here. Same as it ever was.
Sam! Sam! Hold on. Stop! Listen to me. America needs you. Both of you.
Yeah, covering the world in cable didn't bring an end to war and suffering. Don't act surprised when it all comes apart if you try to do it again. Round and round it goes. Connect, reconnect, it's not that simple.
All right, Sam. Just take it easy. I get it. You don't have to commit to anything now. Why not get some rest?
Good idea. We all need our sleep. Leave it too long and you're liable to reconnect with the other side.
Okay, well,
Necessary maintenance has been
we're not gonna follow this advice tonight.
fine-tuning for your benefit.
We ain't sleeping tonight, boys.
You can use it again.
It's just not happening. Not until 8 p.m.
For once, it would seem that dooms and BBs are a good combination. Or perhaps the two of you have something of an affinity for one another.
Oh. Oh, weird. I've been looking at the wrong screen this entire time. I have two screens up in OBS showing me two different OBS overlays. Currently it's using the Norman Returns one when I switched to the other one a long time ago. It's whatever. Doesn't really matter. Nothing's really happening in the corner of our vision anyway. It's kind of silly. Oh, wait. Oh no, has the gameplay been like this the whole time? Oh no, it was stuck on the top of the screen too? That's sad, actually.
Sam.
Oh my god.
Listen.
The audio is completely off.
You are Sam Strand.
No, not anymore. My name is Sam Porter Bridges.
There's no way to realign it?
Strand.
That's so sad.
Is part of rope or bond, while stranding means being washed up on the shore. And being stranded is when you can't go home. I'm stranded now, Sam. Here on the shore of the Pacific. Thousands of miles away. But our bond still holds.
Well,
Free. But we're still connected. Don't tell me we're not.
it's so unfortunate the audio is just completely out. Somehow it works though. I don't know why. This audio being completely off is working out.
Come back, Sam. I'll be waiting for you.
Wait!
Good morning, Sam.
Our cutscenes are so messed up. Totally messed up. I'm pretty sure there's more to that cutscene that we weren't able to see because of the audio being disconnected. Oh, that's sad. Okay, can I finally save?
Sam, this is Die Hardman.
God.
You were out like a log. Must have been beat. Private rooms come with all the basic amenities.
Okay, yes, I understand how a private room works.
Inspect your gear, review info, or whatever else. If you have any questions, feel free to ask our staff. But before you do, why not check things out for yourself?
Can I save? Can I save? Come on, try. Let me save. Let me save. Come on. Boom.
With that terminal there, you can browse the bridge's database, which has info on a range of subjects. You don't have clearance for everything, but there's still plenty you might find informative.
Well, I'm not interested. I'm trying to save. Oh, I guess it auto-saves, but regardless, it doesn't really matter. Because I can switch back to studying, boys. That's what this is all about, after all. And Norman returns at 7 a.m. Now I have to open up Opera. Get all my ducks in order. Oh, I see. My time clock is a little bit off, but that's fine. Man, what? My music now is just completely different in terms of shape. That's so silly. It was working fine earlier. Oh man. Oh well. It's fine. We'll just put it over there. Okay. Doesn't feel like cooperating today, I guess. Aren't you being a silly goose? Okay, there we go. Basic, Khan Academy. Oh, wow, we're already done with this lesson. Perfect. Now we can move on to bioenergetics. Oh wait, you guys can't see that. Hold up. There we go. Just doing the videos today, so I'm gonna skip the questions. Let's go jump into the video.
Our bodies are constantly active, whether we're sleeping or whether we're awake. Our bodies carrying out many chemical reactions to sustain life. And the question I want to explore in this video is, what allows these chemical reactions to proceed in the first place? You see, we have this vague idea that, the breakdown of nutrients to sugars and fats into carbon dioxide, and water releases energy to fuel the production of ATP, which is the energy currency in our body. Many textbooks go one step.
Let me search up how the saving system works in Signalis. Because I'm thinking that for the second half of this 24-hour live stream, we'll switch to Signalis instead of Death Stranding. Oh man, my laptop's chugging. It's not meant to do this for this long. Oh, I'm sorry. I'm sorry, Gurren. Gurren. Am I the only one that names my laptop? Am I the only one that names it after mechs throughout the various anime series I like? Maybe, maybe. To save your game, you must find and interact with red terminals. Is there limited saves, though? That's what I'm wondering. Yeah, for second half, I think we'll switch to Signalis. And then... Yeah, I mean... Ideally, I could find a game that is designed for short bursts of play. Oh, and then next... Yeah, so... The second half, we could try the Pomodoro style that I was trying to go for with that 1.5 hour and then 3 hour, or and then half hour of break. It's not really Pomodoro. It's based off of Anime Doro, which I got inspired by from this YouTuber that also got into medical school, and he has way better production values than I do. I mean, it is what it is. I'm not that interested in making the cleanest content ever. Oh wait, here we go. But for what I trade in cleanness, I gain some more organic content. I believe, at least. So that's what we're going for, baby. That's what we're going for. Organic. It's like organic chemistry. Okay, well. So now is the time when I would normally fall asleep. Like yesterday. Man. But now, starting now, I'm gonna force myself to stay awake so that I can reset everything. Uchi, why do you want to reset the world? Because I need to sleep at a better time schedule. The annoying thing about all of this is that I actually feel really tired, even though it's literally only been 12 hours since I've been awake. Like, I woke up at like 3:30 p.m. Like, the problem with messed up time schedules. Like, if you're sleeping at a time that makes you feel like if you haven't slept during a time that you're normally supposed to be sleeping, then I don't know, maybe there's some kind of subconscious well, circadian rhythms are those considered subconscious? Subconscious is a weird term. Regardless, either subconscious influences or circadian rhythms or some kind of societal value put into me. And I would like to emphasize that last one because I definitely have a lot of like preconceptions about how the body works that are solely based off of like my mom just being completely paranoid about the randomest of stuff. I'd expand on that, but I'm just very hungry and tired. I mean, like having your phone nearby you while you sleep is just like not a thing that she allowed me to do for a very long time. It's not like I put it that close to me, like a few feet away. It's just like, no, you can't do that, because radiation. It's just like, well, it's not really the waves that you're thinking of are not the same waves emitting from these devices, you know? These things utilize very different technology for that. So it's just... And yet I still have this this kind of lingering concern about putting my like phone next to my, or like near my head when I'm sleeping, so I still keep it like on a desk next to me as opposed to like right on the bed. Okay, let's see what's going on
to say
here.
that this process and other energy releasing processes, that is to say chemical reactions that release energy, textbooks say that these types of reactions have something called a negative delta G value, or a negative Gibbs free energy. In this video, we're going to talk about what the change in Gibbs free energy, or delta G as it's most commonly known, is, and what the sign of this numerical value tells us about the reaction. Now, in order to understand delta G, we need to be talking about a specific chemical reaction, because delta G is a quantity that's defined for a given reaction or a sum of reactions. So for the purposes of simplicity, let's say that we have the reaction of some hypothetical reaction where A is turning into a product B. Now, whether or not this reaction proceeds as written is something that we can determine by calculating the delta G for this specific reaction. So just to phrase this again, the delta G, or change in Gibbs free energy of a reaction, tells us very simply whether or not a reaction will occur. Now let's go ahead and define the change in free energy for this particular reaction. Now, as is implied by this delta sign, we're measuring a change. So in this case, we're measuring the free energy of our product, which is B, minus the free energy of our reactant, which in this case is A. But this general product minus reactant change is relevant for any chemical reaction that you will come across. Now at this point, right at the outset, I want to make 3 main points about this value delta G. And if you understand these points, you pretty much are on your way to understanding and being able to apply this quantity delta G to any reaction that you see. Now the first point I want to make has to do with units. So delta G is usually reported in units of, and these brackets just indicate that I'm telling you what the units are for this value. The units are generally reported as joules per mole of reactant. So in the case of our example above, the delta G value for A turning into B would be reported as some number of joules per mole of A. And this intuitively makes sense because we're talking about an energy change, and joules is a unit that's usually used for energy, and we generally refer to quantities in chemistry of reactants or products in terms of molar quantities. Now, the second point I want to make is that the change in Gibbs free energy is only concerned with the products and the reactants of a reaction, not the pathway of the reaction itself. It's what chemists call a state function. And this is a really important property of delta G that we take advantage of, especially in biochemistry, because it allows us to add the delta G value from multiple reactions that are taking place in an overall metabolic pathway. So to return to our example above, we had A turning into a product B, but what if the product B turned into another product C? If we wanted to calculate the overall Gibbs free energy for A going to C, we could instead calculate the individual delta G for each step of the reaction, that is A going to the product B and B going to the product C. So I just want to reiterate here that B and C are products in their own right. They're not transition states. But what we're saying here is that, in some cases, we may not be able to measure the change in Gibbs free energy going from A to C directly. So instead, we can add together the individual change in Gibbs free energy for each step, because remember, Gibbs free energy is a state function. And if we do that, we ultimately get the change in Gibbs free energy for the overall reaction of A going to C. Now, one fun way that I kind of remember this state function, like quality of delta G, as well as some other variables in chemistry, is that my chemistry professor used to tell us that life is not a state function. And this, of course, helps me remember that the definition of state function does not dig into the path of the reaction, because, of course, in life, it's all about the journey and not the destination. But in chemistry, sometimes it's the opposite. Now, the third point that I want to make is that delta G, unlike temperature, for example, which can be readily measured in a lab for a particular situation, delta G is something that can be calculated but not measured. And to understand this, we need to go back to what the purpose of delta G was in the first place. So remember, delta G, the value of it, tells us whether or not a reaction will occur. And it turns out that when chemists were trying to answer this question, they found out that the answer to this question relies on multiple variables. There's not just one thing that determines whether or not a reaction will occur. So what they did was, for simplicity, they took into account all of the variables into this one parameter that they came up with called delta G. And the way they did this was by creating an equation. So they said the change in Gibbs free energy is equal to the change in enthalpy or heat content of a particular reaction minus the temperature of the reaction times the change in entropy, or broadly speaking, randomness between products and reactants in a particular reaction. Therefore, as I mentioned before, we can go ahead and calculate one single value that takes into account all of the variables that affect the extent and degree to which a reaction will occur. And it turns out that we can actually measure the change in enthalpy, the temperature, and the change in entropy for a reaction. So that works out quite well. Now at this point, you probably have a question of, okay, I see that I have an equation to calculate delta G for a reaction, but what does this value that kind of pops out of this equation tell me about a reaction? So let's go ahead and go back to our hypothetical reaction of A going to B. Let's draw a diagram that will help us understand this reaction better. So I'm going to go ahead and draw a y-axis and an x-axis. On the y-axis will be the quantity free energy in units of joules, let's say, and on the x-axis will be the quantity of a reaction coordinate. And this is kind of an abstract parameter that simply is a way for us to kind of monitor the progress of a reaction over time. So this will make more sense when I actually indicate what we're putting in this diagram. So let's say that our reactants A have a much higher free energy than the products of our reaction, which is B in this case. And so what we can say about this, which hopefully is more clear by this visual diagram, is that the change in free energy, which remember is equal to products minus reactants, is negative, or we say it's less than 0. On the other hand, let's say that we started off with reactant A that had a much lower free energy than the product B. Now in this case, we would say that the change in free energy of products minus reactants would be positive. Now the key takeaway here is that for any chemical reaction that has a negative delta G value, we say that the reaction proceeds spontaneously. That is, it proceeds without an input of energy. So I'm just going to write spontaneous there. On the other hand, when a delta G value is positive, that is when the conversion of reactants to products requires a gain of energy, we say that it is a non-spontaneous reaction and cannot proceed unless there is an input of energy. Now one kind of loose analogy that helps me kind of think of these things more intuitively is to think about yoga breathing. So imagine that you're taking a deep, deep breath in, and all of this breath that you have inside of your body makes you feel kind of unstable and wanting to burst, right? So I kind of think of that as starting off at a high free energy state. So let's say we're starting off with A, and then as I breathe out, I kind of feel myself becoming more relaxed. I'm releasing energy, and that brings me to B, which has a lower free energy. And that, of course, breathing out is a spontaneous process.
Cool. Salad eating ASMR. Hmm? Man, I'm not even sure what to do about this tiredness I feel. I feel like I should watch something, like, for fun. But it's just like, I don't know if I want to watch someone else's video and, like, react to that. Right now, at least. Kind of defeats the point of the stream. Although I guess I'm reacting to this, but the problem is that this requires me to do something. But I don't want to necessarily have my hands on the keyboard the whole time. Whatever. Less than 0 is spontaneous. Right?
Think about yoga breathing. So imagine that you're taking...
Okay, yes. I'm a freaking genius. Let's go. I will freaking complete this unit today.
So in this video, I want to go ahead and talk more about the mathematical expression for delta G, or the change in Gibbs free energy, which I went ahead and already written out here. So let's remind ourselves that this change in free energy is a quantity that is equal to the change in enthalpy or heat content for a reaction minus the temperature at which the reaction is run times the change in entropy, or broadly speaking, disorder, in going from reactants to products for a particular chemical reaction. Okay, so that was kind of a mouthful, right? So my ultimate goal in making this video is to ultimately present to you an analogy that I hope will give you a better intuitive feel for how the terms of enthalpy and entropy contribute to the overall sign of delta G. Because remember, it is the sign of delta G, that is whether it's negative or positive, that tells us whether or not a reaction is spontaneous or non-spontaneous. But before talking about analogies, let's go ahead and get a better handle on enthalpy and entropy from more of a chemistry and mathematical lens. So recall that enthalpy is really a good proxy for the change in bond energy that occurs during a reaction. That is to say, whether or not energy was released or absorbed during a bond rearrangement. And because all systems want to achieve their lowest possible energy, if we're talking about enthalpy in isolation from entropy, let's say, generally speaking, we might intuitively say that having a negative delta H, which describes a release of energy, remember, going from reactants to products, would be more favorable than a positive delta H value. On the other hand, the second law of thermodynamics says that all systems tend toward disorder. And since entropy, or the change in entropy, is a
I'm wearing freaking fluffy socks, but I'm so cold. This is BS. Can't believe this. At least I have Xenoblade music to listen to. Oh.
measure of whether something is getting more disordered or less disordered. In isolation, if we're just talking about entropy, we might intuitively say that having a positive delta S value, that is to say describing an increase in entropy from going to products from reactants, would be more favorable than a negative delta S value. Now mathematically, our equation supports our hypothesis, right? A negative number minus a positive number will always be negative. That is to say, regardless of what the temperature is, the delta G value will always be negative, the reaction will always be spontaneous. But since delta H is not always negative, and since delta S is not always positive for all reactions, what will the delta G be for other combinations of delta H and delta S? So to explore this further, I thought we could go ahead and create a table like such to write out essentially the different signs that delta H and delta S could take on and determine for each of these combinations whether the delta G will be positive or negative. So we've already gone through one. We've said that when delta H is negative and delta S, is positive, delta G will be negative. But of course, there are other combinations too. Delta H could be positive and delta S could be negative, or both delta H and delta S could be positive, or delta H and delta S could both be negative. So let's go ahead and take these signs and plug and chug into our equation above. So if we have a positive number and we're subtracting a negative number, we are always going to get a positive number. So for all situations in which delta H is positive and delta S is negative, we will have a positive delta G value. This makes sense, right? Because it's essentially the opposite of the previous situation. But what about the case in which you have a positive number minus another positive number? Well, you might say it really depends on the relative magnitude of these two numbers, right? Because if you have a big positive number minus a smaller positive number, you would get a positive number. But if you had a small positive number minus a bigger positive number, you would get a negative number. So ultimately, it really depends. Delta G can either be positive or negative. And this value of temperature plays a pretty big role in kind of tipping the scale towards one direction or another. Because if it's really high, for example, our second term might be more positive, and therefore this overall value might be more negative. But if it's not very high and delta H is really, really large, well, perhaps then delta G would be positive. So finally, let's look at the situation where we have a negative number in our last row minus another negative number. Well, you might say, again, this really depends, right? If you have a small negative number and you're adding a very large positive quantity to it, it could be positive. But if it's a smaller number that you're adding onto a negative number, it could still be negative. So again, this really depends. And again, temperature plays a big role in tipping the scale one direction or the other. Now for the analogy that I want to present, I really want to focus on these last two rows here, in which temperature plays a big role in determining whether or not a reaction is spontaneous or not. So how can we understand this trade-off between delta H and delta S at high or low temperatures? So I'm going to go ahead and scroll down, and I'm going to present an analogy that I hope isn't too corny, but hopefully it's useful, and you might even consider thinking of your own analogy to help you understand this trade-off between entropy and enthalpy. So imagine you are a chemistry student, and for spring break you decide to go with your friends somewhere really tropical where there are a bunch of palm trees and great beaches. And one day you and your friends go ahead out to one of the beaches, and you and a couple friends set out, a large beach towel and decide to sunbathe on the beach, while some of your other friends decide to go out into the ocean and swim. Now, of course, being a chemistry nerd, you describe this phenomenon in terms of a chemical reaction. That is to say, a reaction in going from lying down on a beach towel to swimming in the water, or the reverse, that is going from the
Bruh. I must be a shy extrovert or something. I feel energized just chatting with my roommate for a split second, but then I purposely left the conversation. Because, I don't know, man. I'm just here to study right now, so I'm in the zone. When I'm in the zone. Try to get me out of it. So, feels bad. Poor timing. Well, good timing for me, but not mutually. It is what it is.
the water to lying on a beach towel. Now at this point, you begin thinking back to your equation for delta G, which remember
Meanwhile, I'm interacting with no one
you begin to realize the
live.
forward reaction that is going from a beach towel to the ocean.
Okay, it's a little different.
That is to say, more of your friends are in the water when the sun
Which is
really at its
interesting
peak and
because I'm
everyone
really
wants to
curious
go to
about
the water to cool
parasocial
off. On the other
relationships.
hand, the reverse reaction
I
that
would
is
like
going
to
from the water
study
to the beach
that,
towel is more
but,
favorable when the sun is less intense,
not today.
when it's less scorching hot outside and it's easier to relax on the beach. In other words, you conclude the sun is figuratively, or perhaps even literally here, analogous to the temperature variable in our equation. Depending on how hot the sun is, ultimately determines whether or not it is more spontaneous to go into the water or come out of the water. Now, since you happen to also be an English major and love metaphors, you go ahead and think up a metaphor for both the enthalpy and entropy contributions in your hypothetical chemical reaction. Enthalpy, you decide, you can think about in terms of the amount of energy that you're expending whether you're on a beach towel or in the water. So we all know that lying on a beach towel is super relaxing and very peaceful and requires little to no energy, and you're in a very, very stable place. But on the other hand, when you're swimming in the water, it requires a lot of energy to be expended because you're, of course, swimming and throwing around a beach ball. On the other hand, I'm going to go ahead and scroll down here. Lying on a beach towel can kind of get boring after a while, right? But being in the water is lots of fun, and there's really never a boring moment. And this interplay between fun and boring is something that you can kind of think of as your entropy variable. Now, in an ideal world, you want to be both relaxed and having fun. And in chemistry language, that's really another way of saying that you want to have a negative delta H value and a positive delta S value. But of course, in this particular setup, these things are mutually exclusive. So, what wins? Ultimately, in the end, you conclude that when the sun is out, this fun term, or what we're kind of calling entropy, wins, and the forward reaction is spontaneous. But when the sun isn't as hot outside, this relaxation or enthalpy term wins, and the reverse reaction is spontaneous. And with this mighty conclusion that you have come up with on this relaxing day at the beach, you go home and reminisce about how chemistry and life are far more similar than you once thought.
That analogy was not good. Not a good analogy. Not very helpful at all, dude. I'm just saying. Damn. It just made me more confused, even though Gibbs free energy is a pretty simple concept, for the most part. Or at least the equation is simple.
You are holding in your hands a glass of water. We're going to say that a glass of water is at room temperature, so that's about 20 degrees Celsius, and you are at body temperature, which is about 37 degrees Celsius. Now, intuitively, you know that after a while, your hand starts to feel kind of cold, right? So what?
Probably want to define H, T, and S. H is heat. Which I always get confused with temperature. Because we use them colloquially interchangeably. And then S is entropy.
What's going on here? In the physical sciences, we say that what's going on here is called a heat transfer. And the idea is that because we have two different things at different temperatures, specifically because our water is at a lower temperature, energy in the form of heat is going to travel from our body to the water, which means that our body is losing energy, and the cup of water here is gaining that energy. So we are becoming cold, and the cup of water is warming up. Now, when I was first learning about heat transfer, I didn't quite understand the difference between heat and temperature. So I just want to go ahead and briefly discuss what the difference is. Heat is the amount of energy that's transferred due to a change in temperature. So I'm going to go ahead and write that out here. Remember that whenever you see a gradient, whether it's a gradient of pressure or concentration, or in this case, a gradient of temperature, it always means that there is some type of potential energy that's stored up. And since the system wants to achieve its lowest energy possible, if there's no opposing force, the gradient wants to disappear. So this is the basis behind heat transfer. Of course, what's at this gradient is temperature, and temperature is an absolute quantity. Specifically, it's defined as the average kinetic energy of molecules in whatever we are measuring the temperature of.
Someone edited that into the flow of the video.
The point I really want to underscore here is that while temperature is an absolute measure of energy,
Can't trick me.
there's really no such thing as an absolute heat content. Heat should always be described as heat transfer because it is measuring the amount of heat that's either lost or gained. It's also important to emphasize that heat transfer occurs between a system and its surroundings, which I'm going to abbreviate as SIR. So to understand this, let's go ahead and revisit our example above. Now, what's really important to determine here is the direction of heat transfer. Now, as I talked about before, we can't really attribute an absolute value of heat content to either the system or the surroundings. But what we can say is what direction does this heat transfer occur in? What's getting hotter, essentially, and what's getting colder? So the way that physical scientists kind of denote heat transfer is with the lowercase letter q. And so in the case of our system, which is our hand, which is becoming colder, the way that this would be denoted is with a minus q, because heat energy is being lost from the system. On the other hand, what you would find after a while, if you kept holding this glass of water and then set it down on the counter and measure the temperature, is that the temperature of the water would have increased. And essentially, what is going on here is that the energy lost from the system is being absorbed by the surrounding. And so in this case, because heat energy is being gained, this would be denoted as plus q. To state this phenomenon more generally, it's fair to say that the heat that is either lost or gained by the system is equal and opposite in magnitude to the heat that's either lost or absorbed by the surroundings.
I like how the only thing that I got from that was to put heat transfer here. Sometimes the least amount of information is all you need for everything to make sense. Wait. Oh, wait, no, enthalpy is H. Wait. It's an enthalpy heat? Okay, wait, they're gonna explain it.
So we've been kind of talking kind of at a very macro level about heat transfer by using an example of heat being transferred between our hand and a glass of water. So now let's go more of a micro level. Let's talk about heat transfer and chemical reactions. So when we're talking about heat transfer and chemical reactions, we're talking about the term called enthalpy. Now, it turns out that if we think about chemical reactions as our system, so go ahead and write that here, chemical reactions can either release or absorb energy that can change the temperature of its surroundings, which we generally think of when we're talking about chemical reactions as, the solution that the chemical reaction is taking place in. And of course, if the chemical reaction is taking place in our body, the surroundings really just our body, which you can think of really as just a big bag of water. So the question might be on your mind, why did chemists come up with this fancy term enthalpy to describe the heat transfer of chemical reactions? It turns out that enthalpy is a very useful quantity to calculate for many chemical reactions, because not only does it tell us something about heat transfer, but it also is a component of Gibbs free energy, which is an important parameter that chemists use to determine whether or not a reaction will take place or not. Generally speaking, I think it's okay to conceptualize with some simplification that enthalpy is essentially just a fancy term to describe heat transfer for chemical reactions. And because we can never define an absolute quantity of heat, because remember, heat is the amount of energy transferred, it's not an absolute term, enthalpy is always referred to as a change in enthalpy, and oftentimes it's written as delta H. And specifically, this change in enthalpy describes the change in heat energy, that is, whether heat was lost or gained from the perspective of the system. So even though there is this kind of important interplay, this conservation of energy between the system and the surroundings, this term enthalpy is really just telling us what's happening from the perspective of the system. And I think this will make more sense as I give you an example below. But before I do that, I just want to note what the units of enthalpy are. And the units of enthalpy are joules per mole of reactant in the chemical reaction. And this of course makes sense because joules is a unit of energy, and we're talking about heat, which is a form of energy. And it's notable to know that having it per mole allows us to take the amount of whatever is reacting into account. This is something that we can't really take into account if we just, say, measure the change in temperature that occurred over the course of a reaction. Alright, so now let's go ahead and take a look at an example. So I'm going to go ahead and scroll down. Let's go ahead and use an example that you will be fairly familiar with. So when our bodies get super hot, to cool ourselves down, our bodies essentially evaporate water from the surface of our body to help cool ourselves down. And this is really just a fancy way of saying that we start sweating, right? Now, the chemical reaction for sweating we can really think about as just essentially the evaporation of water. So that is to say, taking water from the liquid phase and turning it into water vapor, or water in the gaseous phase. The change in enthalpy for this particular reaction, and note that I'm saying for this particular reaction for our system, is defined as the enthalpy of our product, which is our water in its gaseous form, minus the enthalpy of our reactant, which is water in its liquid form. This, of course, is a way to conceptualize enthalpy, but remember, it's really actually not possible to measure an absolute quantity of enthalpy for anything. We can only ever measure this change in enthalpy, which involves monitoring the change in temperature of surroundings during the course of a reaction, as well as some other considerations that we're not going to go into in this video. But just to note that, it's really the change in enthalpy that we're measuring and not these absolute quantities, even though technically this is kind of how we're conceptualizing enthalpy. So the key idea here about this process of evaporation is that it requires energy to occur. Just think about boiling water. In order to get, water into its gaseous phase, you need to heat it up. So the fact that we're adding energy to our system should tell you something about what the sign of this change in enthalpy should be. The products are essentially gaining more energy in the form of heat than the reactants. So we say that in a process that absorbs heat, the change in enthalpy is greater than 0. In other words, the change in enthalpy is positive. And whenever a reaction has a positive change in enthalpy, which by definition means that heat is being absorbed, we call it an endothermic reaction. And the way that I kind of like to remember this is that endo is, I think, a Latin prefix for within. So heat is going into our system, which is our chemical reaction. So you're probably wondering at this point, well, if we're absorbing heat, how is this connected to us cooling off? Well, this is kind of the important point that I kind of alluded to before. This change in enthalpy only tells us what's going on with our system, which of course is our chemical reaction. But our body, in this case, is our surrounding. So we can use this relationship above that tells us that the heat lost or gained by the system is equal and opposite to the heat lost or gained by the surroundings. So in this case, we know that our system is absorbing heat, which means that our surroundings, our body, must be losing heat, and that's how we cool down. Now you can also imagine that we have chemical reactions in which the change in enthalpy is less than 0, and we call these types of reactions exothermic reactions. And the way I kind of think about these is that this X term is kind of a Latin prefix for out of, so heat essentially is going out of the system instead of being absorbed. One example of a very prevalent reaction in our bodies that is exothermic is the hydrolysis, or reaction with water, of ATP, which is of course the energy currency of our cells. So ATP reacts with water, and it loses a phosphate group to become ADP and a free phosphate group. And
ADP, let's go. ADP, shout out.
Or in other words, it releases heat. The fact that this reaction is exothermic is physiologically significant for many reasons, but one of those reasons is when we're talking about shivering. So we all know that we shiver when we're cold, and the reason we do that is because we want to warm ourselves up. And that heat energy is indirectly tied to this hydrolysis of ATP, which releases heat and allows our muscles to contract to warm us up. So just to wrap things up here, I think the key takeaway is that enthalpy describes heat transfers for chemical reactions, and notably it's from the perspective of the chemical reaction, not the surroundings. And so chemical reactions can either lose heat, in which case they're classified as exothermic reactions and having a negative enthalpy, or they require an input of heat, and they're classified as endothermic and have a positive change in enthalpy. In this video, I want to go ahead and talk about thermodynamics and kinetics. I remember that when I was first learning about these two things in the context of chemical reactions, I used to hear phrases like, oh, this chemical reaction, let's say hypothetically A going to B, is kinetically favorable, so it must proceed. But then the next day when I go to class, I hear another statement that said, oh, well, this reaction A going to B is thermodynamically favorable, so it must proceed. So in this video, I want to go ahead and clarify these two statements for you and really understand what it means to be thermodynamically favored and or kinetically favored. So to get us started, I've already drawn kind of two plots so that we can plot out the free energy change that occurs. For the forward reaction, we'll do that on the left side, and the reverse reaction, which we'll do on the right side. So before we get into that, let's go ahead and label our axes. So our y-axis in both cases will be measuring free energy, which is in units of joule. And our x-axis will be a abstract dimension called the reaction coordinate, which essentially allows us to monitor the progress of a reaction. So now I'm going to go ahead and say that the forward reaction will draw out in this teal color, and the reverse reaction will go ahead and draw out in this pink color. So let's go ahead and start with the forward reaction. So I'm just going to go ahead and say that the forward reaction has a negative delta G value. So remember, that means it's spontaneous, and visually that means that our reactants start off at a higher free energy than our products. Now, this of course means that the reverse reaction, which will have the same magnitude of delta G, that is, the free energy change will be at the same numerical value, but of course, since the reaction is going the opposite direction, the sign of delta G will be now positive. And visually, we're saying essentially that our reactant, which in this case is B, starts out at a lower free energy than our product, which is A. Now, thus far, in drawing this free energy diagram, we've just been talking about thermodynamics, but it turns out that there's also a kinetic energy barrier for the conversion of reactants to products, regardless of whether the reaction is spontaneous or non-spontaneous. This kinetic barrier of energy is referred to as the free energy of activation, or simply activation energy. So I'm going to go ahead and put in parentheses E sub A, which we'll say stands for activation energy. And remember that delta G, of course, is talking about thermodynamics. With that said, let's go ahead and add this kinetic energy barrier to our diagrams. And we can do this by understanding that the activation energy is defined as the amount of energy that is required to form a high energy intermediate during the course of the reaction. In other words, in our hypothetical reaction of A going to B, it proceeds through an intermediate that is a high energy chemical product that won't last very long, but is important in the conversion of A to B or vice versa. So in our diagrams here, we can go ahead and indicate that there is some intermediate, so in between our reactant and our product, that is at a much higher energy than everything else. And we can go ahead and then connect the dots. So go ahead and essentially draw a line from reactants to products that includes our high energy intermediate. And this ultimately allows us to see the presence of the activation energy as well as the change in free energy. So let's go ahead and actually label these things. So on the left side here, remember our change.
Is it time to do a pro gamer move? Oh man, it might be. It might be time to move enzymes finally. It's gonna be so painful to try and do right now. But it's probably worth it. It's gonna make this map very much right skewed. Or is it left skewed if we're talking statistics? It's gonna be very annoying to deal with, but let's do it. My music stopped too. Hate to see it. Okay, enzymes are over. No, this is protein. Protein's over here, so I can move. Oh no, I have to move this entire thing down. Okay. Can I add this to the group? Yes, I can. Perfect. Move it down like so. Don't have to move it too far down though. Very nice. And now I need to open up the left side of enzymes. Allow for connection to be made. Okay. So I can move this a little bit further away. And these. Oh no. Is there a way to deselect? Can I deselect that? These. Oh no. No, that's just it's just gonna duplicate. Alright. I'll just take these and then run with it. Switch this to the bottom of the active site, moves this here. There we are. Not the best, but it's an open road to enzymes. So it leaves an ease of access to the concept. Oh no, it's going to... No, okay. I can just include these without including the others. One day I'm gonna have to revisit this and clean it up so that it's actually usable. For the time being, this is like not it. Oh my god, look at this. Look at this. Everything branches off of the central dogma, which makes sense because it's the central dogma. God damn. Okay. They really weren't kidding about the central part. And so it begins. We need a loop around thermodynamics and kinetics. So this is thermodynamics. And I guess, what leads off of what? Yeah, I feel like those are two separate concepts, right? Thermodynamics, thermodynamics, and kinetics. Kinetics is all the way up there. Okay. It's a bit of a reach, but we'll work with it. We'll work it. Work it, baby. Very nice. There it is.
Change in free energy, we're just looking at our reactants compared to our products. We're ignoring this little bump. The change in free energy, which I'm going to indicate with a green line, extends between these 2 points. And same on the right side, just again, extending between the start and end points of our reaction. So I'll go ahead and indicate that this green line in both cases refers to our delta G values. And in a different color, let's say red, I'm going to go ahead and indicate the activation energy, which takes into account the change in energy between the high energy intermediate and the reactant. So in the case on the left, that change is indicated here with red. And on the right side, the change between the intermediate and the reactant is a bit longer. So we'll go ahead and indicate that here. Now, activation energy is an important quantity to take into account because in order for molecules to react, they must have enough energy to overcome this activation energy barrier. Essentially, in the case of a spontaneous reaction, for example, I kind of think of it like the energy one needs to get a ball to start rolling down a hill. We all know that gravity will make a ball roll down a hill, just like a negative delta G value is telling us that the reaction is very thermodynamically favorable. But we need to sometimes give the ball a push in order for the reaction to occur. And so that's kind of this little hill that it needs to go over before it can actually proceed. For a non-spontaneous reaction, the idea is essentially the same. We still need to have some activation energy. But in addition, because it requires an input of energy, we can kind of think about it as rolling a ball up a hill instead of down a hill. Now, in general, the idea is that the lower this free energy change is, the faster a reaction will occur. And remember, I'm saying faster, so I'm talking about kinetics. I'm talking about the rate of a reaction. So just to write that out, the activation energy, the smaller it is, the faster the reaction will proceed. Now, in biochemistry in particular, it's really important to distinguish between these two terms of thermodynamics and kinetics, which we've drawn out in our diagrams as the change in delta G or the change in activation energy, because many biochemical reactions in our body are kinetically unfavorable. That is to say, they have a very high energy of activation, even if they are thermodynamically favorable. This is why our bodies have enzymes, which essentially lower the activation energy of a reaction. So I went ahead and drew a dotted white line that's a little bit lower, so you can see that when an enzyme is present, the height of the barrier has decreased, and if it's decreased, the reaction will proceed faster. Now, there's one analogy that my chemistry professor used to tell us all the time that really helped me understand the interplay between kinetics and thermodynamics as they apply to whether or not a reaction will occur. So I'm going to go ahead and scroll down so we can briefly talk about this analogy, which is, I think, a fun way to think about all of it. So let's say you went to a dating website because you were looking for your perfect match, and this dating website told you that your perfect match lived halfway across the globe. And this was such a perfect match, and they had all these algorithms, and if this match were a chemical equation, we would say that you had a very, very negative delta G value. That means you would be very, very spontaneous, right? But you live halfway around the world, right? So in terms of chemistry language, we might say that you are kinetically limited. That is to say, you don't have any way of actually traveling halfway across the world
makes
to
sense.
meet
Interesting.
someone.
Okay.
So we might say that you have a very high activation energy. So from this discussion, perhaps the biggest takeaway is that neither kinetics nor thermodynamics solely determines whether a reaction will proceed. It's important to take both into account. And one of the applications of this in biochemistry especially is, remember that enzymes lower the activation energy of a reaction. So we can kind of think of enzymes as light switches. They can kind of regulate whether a reaction will proceed or won't proceed. But of course, a light switch only works if the light bulb itself is working. And so the metaphor for the working light bulb is saying that a reaction has a negative delta G value, or if the light bulb is not working, we're saying that the delta G value is positive. That is, it requires a new battery or energy for the reaction to proceed.
Huh. That was actually quite helpful in understanding the difference. Interesting. Okay, cool. Well. That's it. That's lesson 11. Damn. Alright, we're getting through this. Metabolism, finally. I believe this is the last lesson in Unit 5 out of like 12. This is like the first unit that we're starting with, because I don't feel like working on cars right now. Oh no, I love cars though. I do love cars, but not cars. Be clear, just to be super crystal clear. I like cars, but not cars. Okay. I know. I messed up. Kind of wonky.
Textbooks define metabolism, a topic in biochemistry, as a series of chemical reactions that take place inside of our bodies to sustain life. Now, this is a pretty broad definition of metabolism. So in this video, I really want to break this definition down to a more workable understanding of what metabolism really is. So first, I'm going to introduce another arrow in this diagram like this and say that really the requirements of life, let's say in a human being, such as, maintaining a constant internal temperature, reproducing, growing, and all that jazz, all of that ultimately boils down to the body's ability to utilize 4 essential biomolecules. And these 4 essential biomolecules, or as they're sometimes known as macromolecules, are proteins, fats, carbohydrates, or carbs, and nucleic acids, like DNA and RNA. And ultimately, all of these biomolecules perform different life-sustaining reactions inside of all of the cells in our body to ultimately promote life. So as you can see, we've already begun to break down this definition of metabolism. Essentially, what we're saying here is that metabolism is really the study of how we're able to obtain these important biomolecules to sustain life. So how do we obtain these biomolecules? Now, the simple answer to this question is, of course, that we eat food to obtain all of these important biomolecules. But there's an important word of caution here, which is that since most food comes from
You guys see the problem. I have no idea where to put this. We're gonna put metabolism here.
living organisms like plants and animals. These plants and animals also contain an array of proteins, fats, carbohydrates, and nucleic acids, but not necessarily in the same flavor or configuration that our bodies would prefer. So what do our bodies do instead? Well, in our bodies, we go ahead and eat the food. It's a very large head there, but you get the idea. And in our bodies, we break down this food through a process called digestion into the component parts of all of these biomolecules. So what do I mean by component parts? Well, the smallest subunit of proteins is called an amino acid, and our body breaks down all the different types of proteins that we digest into individual amino acids. And the same pattern continues for the rest of the biomolecules. So in the case of fats, we're talking about fatty acids, which are the smallest subunits of fats. And then for carbohydrates, which are long chains of sugars, one of the most common subunits of carbohydrates that our body loves is called glucose. So I'll go ahead and write that here, since you'll be seeing it a lot in the discussion of metabolism. And then finally, for nucleic acids, we're talking about nucleotides. So at this point, you're probably thinking, well, okay, I understand that our body can't use the same macromolecules found in food because maybe they're not in the right configuration, but how does breaking them down do anything for us? Now, the key here is to recognize that in our body, there's actually a delicate balance going on between the processes of breaking down molecules, such as in the process of digestion, and then taking these products and building them back up. So Essentially, you can see all of these subunits or monomers as Lego pieces that we're essentially reconstructing to build the right configurations of proteins, fats, carbs, and nucleic acids that our body needs. So that's really the key idea here, which is that metabolism is a balance between breaking things down and building them back up in our body so that we can customize, so to say, what type of macromolecules that we create.
Excuse me.
And just to throw in some vocab words, biochemists call the process of breaking down molecules in our body catabolism, and a similar sounding word called anabolism is used to describe the process of building molecules back up. And the way I kind of like to remember this is looking at the first letter of each of these words. I think of C, I think of cutting molecules up into tiny pieces, so breaking them down, and then for anabolism, A I think of as like the apex of like a building, for example. So we're building something up. Now this seems all fine and elegant.
That is not how I remember those. I would think cataclysm or catastrophe for breaking down stuff. And as for the other one, I just pray to God that I'll remember it. Yeah. I have some pretty good memorization strategies, but they don't always work out, so...
There's one more issue that we need to contend with, which is a consequence of having to balance breaking things down and building them back up. And that is that this process of building molecules back up requires energy, which I'm kind of indicating here with these yellow lightning bolt stars. So the question I want to answer in this last part of the video is, where does this energy come
Tossing
from?
all the stars into there.
Well, we also get this energy by eating food. So how does that work? So first, recall that the energy currency of the cell, and I'm going to go ahead and erase this just to give us some more space. The energy currency of our body is a molecule called ATP, or adenosine triphosphate. And it's high energy molecule, as it's often referred to, when it is broken down into
use
ATP,
energy to
so
make energy. Hell
it produces usable chemical energy that can fuel energy-requiring processes in our body, such as the building up process of anabolism. Now, in order for this process to continue nonstop in our bodies, ADP must be regenerated into ATP.
I mean, on the simplest
and
level, that is how it works.
We digest our food into all these subunits, and some of
it
these
is what it
subunits, such
is.
as glucose and fatty acids mainly, but occasionally amino acids, so I'm going to put that in parentheses, can essentially be used as fuels in our body. So, just like wood, for example, is a fuel for a burning fire, which produces heat, these fuels in our body
you can't break down amino acids, huh?
to produce the energy that's necessary to convert ADP back into ATP. And this
Oh wait, am I misremembering this?
And just to throw in another vocabulary word that you'll probably see, this process of taking these fuels, which I've indicated with this asterisk, and breaking them down into usable energy is a process that's referred to as cellular respiration. And recall that because cellular respiration involves breaking down things even further, it's also a catabolic process, so it falls under this category of catabolism. And just to tie everything here together at the end, notice here that another way to interpret this cycling between ATP and ADP is to say that catabolism fuels anabolism. So what do I mean by this? Well, essentially, catabolism, such as the process of breaking things down and
hold up.
extracting energy through processes of cellular...
So what does it branch off of catabolism or anabolism? Is cellular respiration part of what? Like, I know what cellular respiration is. I think you make ATP, then you break ATP down. Okay, I think what's going on here is that I need to just branch it off of metabolism. It's supposed to be both anyway.
respiration is coupled with this process of building things back up. And so in essence, one relies on the other. And as you can probably guess, these processes are really tightly regulated in our bodies because obviously you wouldn't want to be breaking down something while you're building something back up. And in fact, just to give you kind of a preview forward, catabolism and anabolism are often regulated, so controlled through the use of hormones. So I'm going to write here that hormones are a form of regulation and tell the body whether it should be in a catabolic or anabolic state.
Oh, okay. I thought it was the music that was reaching that level of loudness. No, the volume's perfect. Although I suppose it's kind of hard to hear me over the speaker. I don't know if that's important though. Like, I don't need to be that much higher above her. I'll just lower it just in case. One of my favorite molecules, adenosine triphosphate. Let's go. I mean, it's kind of cheating. It's kind of a necessary molecule for all life, so of course I'm gonna like it.
ATP, or adenosine triphosphate, is often referred to as the currency of energy, or the energy store, adenosine, the energy store
Make money.
in biological systems. And what I want to do in this video is get a better appreciation of why that is. So adenosine triphosphate.
Get
So at first, it
maidens,
seems
even
like
though
a
I
fairly
can't
complicated
play
term,
Elden
adenosine
Ring right now.
triphosphate. And even when we look at its molecular structure, it seems quite involved. But if we break it down to its constituent parts, it becomes a little bit more understandable, and we'll begin to appreciate how it is a store of energy. in biological
I like how the
systems.
boss music started playing as soon as this guy started talking. That's fantastic. Why do I hear boss music?
Part that is the triphosphates, or the 3 phosphoryl groups. So the adenosine is this part of the molecule. Let me do it in that same color. So this part right over here is adenosine, and it's an adenine with connected to a ribose right over there. So that's the adenosine part. And then you have 3 phosphoryl groups, and when they break off, they can turn into a phosphate. And so the triphosphate part, you have, so triphosphate, you have 1 phosphoryl group, 2 phosphoryl groups, 2 phosphoryl groups, and 3 phosphoryl groups. So one way that you can conceptualize this molecule, which will
Oh my god, just get me to Krebs cycle. Come on, dude.
is to represent this whole
Krebs cycle.
Represent that as an A. Actually, let's make that an AD, and then let's just show it bonded to the 3 phosphoryl groups. So, and I'll make those with a P and a circle around it. So you can do it like that. Or sometimes you'll see it actually depicted, instead of just drawing these straight horizontal lines, you'll see it depicted with essentially higher energy bonds. So you'll see something like that to show that these bonds have a lot of energy. I'll just do it this way for the sake of this video. But these are high energy bonds. Now what does that mean? What does that mean that these are high energy bonds? It means that the electrons in this bond are in a high energy state. And if somehow this bond could be broken, these electrons are going to go into a more comfortable state, into a lower energy state. And as they go from a higher energy state into a lower, more comfortable energy state, they are going to release energy. One way to think about it is if I'm about to jump, if I'm in a plane and I'm about to jump out, I'm at a high energy state. I have a high potential energy. I just do a little thing and I'm going to fall through. I'm going to fall down. And as I fall down, I can release energy. I can friction with the air. Or eventually, when I hit the ground, that will release energy. I can compress a spring.
Makes sense. That makes sense.
But then when I'm sitting on my couch, then I'm in a low energy state. I'm comfortable. It's not obvious, how I could go to a lower energy state. I guess I could fall asleep or something like that. And these metaphors break down at some point. But that's one way to think about what's going on here. The electrons in this bond, if you can give them just the right circumstances, they can come out of that bond and go into a lower energy state and release energy. So one way to think about it, you start with ATP, adenosine triphosphate, and one possibility, you put it in the presence of water, and then hydrolysis will take place. And what you're going to end up with is one of these things are going to be essentially, one of these phosphoryl groups are going to be popped off and turned into a phosphate molecule. And so you're going to have then adenosine, since you don't have 3 phosphoryl groups anymore, you're going to have 2 phosphoryl groups. You're going to have adenosine diphosphate, often known as ADP. Let me write this down. This is ATP. This is ATP right over here, and this right over here is ADP, di for 2 phosphoryl groups. Adenosine diphosphate.
All the
and then this one got
like
off.
parts of my body that could be super cold right now. My knees. Why would it be my knees? It's like right in the middle of my leg.
The electrons in this bond right over here
Why?
go into a lower energy state. They are
Or
going
sand.
to release energy. So plus energy. So here, this side of the reaction, energy released. Energy released. And this side of the reaction, you see energy stored. And so as you study biochemistry, you'll see time and time again energy being used in order to go from
Biochem is
ATP
hype.
and a phosphate to ADP. So that stores the energy. You'll see that in things like photosynthesis, where we use light energy to eventually get to a point where this P is put
It's
back
hard,
on,
but
using
it's
energy,
hype.
putting this P back onto the ADP to get ATP.
The
And then
MCAT
you'll
is
see
hard,
when biological
and the
systems
MCAT
use energy, that they'll use the ATP,
is important.
and they'll essentially hydrolysis will take place, and they'll release that energy. Sometimes that energy could be used just to generate heat, and sometimes it can be used to actually forward some other reaction or change the conformation of a protein somehow. Whatever might be the case.
So let's talk about one of the most famous molecules in all of biochemistry, ATP. So why is ATP so famous? Well, it's the energy currency of a cell. And the reason it's called the energy currency of a cell is because it powers, it essentially fuels many...
Okay. Grab some water. Might eat some outdated oranges. Oranges. Oh no, my voice. Dude, it's only been 12. It's not even been 12 hours yet. I've been streaming for 9 hours. Okay, well, I'll be back. And oh. I'm really bad at figuring out when fruits go bad. There's like a squishy spot on this. I'll never tell until I try it. It's a little concerning. I mean, I literally just got these oranges, so if they go bad this quickly, then I'm concerned. We'll see.
Life-sustaining reactions inside of our body. So some examples of these include biosynthesis of biomolecules. So remember, fats, proteins, carbohydrates, and nucleic acid are all essential to life, and building these molecules requires energy in the form of ATP.
Build them up.
Used to contract our muscles, and this is very important in order to allow living organisms to move. And additionally, ATP is also involved in some ion movement across cell membranes. And
why is it in like 240p? What's going on here?
Within the cell.
This resolution.
That somehow ATP magically powers all of these life-sustaining processes, but you really don't have to. In fact, in this video, we're going to review some topics from general chemistry to really understand.
I'm like straining to
on a chemical level
this open.
These reactions.
This thing.
Now, the topic we want to review in introductive chemistry is the thermodynamic parameter.
Feeling an orange, dude.
Free energy, or as it's more often written as just simply delta G. So now recall that delta G is a quantitative number, and it's a number that's measured in units of joules, which is.
We literally just learned about that.
And depending on whether this value is positive or negative, it tells us whether or not a reaction requires energy or whether a reaction releases energy. Now remember that delta G is equal to the free energy of the products of a reaction minus the free energy of the reactants in a reaction. So if the change in Gibbs free energy is negative, which means that the products have a much smaller free energy than the reactants, we say that this reaction
once again boss music over some random shit. This is the final boss. Someone edit over like an HP bar underneath this. The greater orange.
So how does this all relate to ATP? Well, it turns out that there's a reaction involving ATP that has a very large negative delta G value. That is to say, it releases a lot of free energy. Specifically, this reaction involves ATP combining with water.
Maybe stronger than Millennia herself.
Hydrolysis reactions. I'll just write that here to remind us. And the products of this reaction are a molecule called ADP and a free phosphate group. And like I mentioned before, the change in Gibbs free energy is very negative. So what's going on here? So ATP starts out with triphosphate, 3 phosphate groups.
I hope this hasn't gone bad.
It forms a free phosphate group that it cleaved off. So on first glance, it might seem that this reaction is not balanced because we don't have this hydrogen or oxygen on the right side of our equation, but I just want to note here that the negatively charged hydroxyl group becomes a part of the phosphate group, and the remaining hydrogen ion of the water combines with another molecule of water in solution to become a positively charged hydronium ion. And usually these two things are left out just for the sake of convenience, but I wanted to point them out here so that you wouldn't be confused by the stoichiometry. On the other hand, many biosynthesis reactions in the body have a positive.
Well, it's not terrible, so I'm assuming it's still good.
And an example of this type of reaction is when we take a monomer, such as an amino acid, for example, and we string them together covalently to form a polymer. So in the case of an amino acid, that would mean we're forming a long peptide chain. Now here's where our knowledge of introductory chemistry comes in. So in thermodynamics, the study of energy changes, there's an important principle that states that the overall delta G for a reaction, I'm going to scroll down here to give us some more space, so the overall delta G for a reaction is equal to the sum of the delta G values for the individual steps of a reaction. So let's actually go ahead and add these two reactions together and see what happens. So let's write that out. So we have ATP as a reactant as well as water, as well as our monomer subunits, and we are producing ADP, a free phosphate group, and a polymer.
It's a little stale though.
Now, what is the delta G for our overall reaction? Well, we just simply have to add the delta G values for each step. Now, I didn't give you, actual numerical values for each of these steps, but in general, the hydrolysis of ATP produces energy in excess of the energy needed for biosynthesis reactions, such as this one. So
probably because I was eating some or drinking some really sugary stuff.
Overall negative delta G value. In other
Or
words,
is it?
take
I don't know.
A energetically unfavorable reaction with a positive delta G value and turned it into an energetically favorable reaction with a negative delta G value. And we have done this by what we call coupling a reaction that has a favorable delta G value, such as ATP hydrolysis, with a reaction that has an unfavorable delta G value. I want to mention that the ability to add these delta G values tells us nothing about the path that the reaction actually takes. And in fact, generally speaking, almost never does the reaction proceed in 2 discrete steps like it's written here. Instead, this coupled process often occurs simultaneously. But as you can see, it's still beneficial to separate these two reactions into 2 discrete steps so you can prove to yourself
all at the same time, baby.
With its negative delta G value is able to fuel
Multitasking.
processes.
That video did not last me eating this orange. It's like a whole ordeal, man.
Let's talk about one of the most
Just
famous
like,
molecules in all of biochemistry.
I still have half of the orange
So
left.
why is ATP so famous? Well, it's the energy currency of a cell.
Bro.
The reason it's called the energy currency of a cell is because it powers, it essentially fuels many life-sustaining reactions inside of our body. So some examples of these include biosynthesis.
Okay.
Of
I'm not
biomolecules.
rewatching this though. I'll switch to a different video, then come back for the notes.
Oftentimes in biochemistry, we write energy-requiring processes, such as in the conversion of a monomer to a larger polymer, as one-step reactions that are ultimately fueled by separate reaction involving the breakdown, or hydrolysis. Remember, hydrolysis just means reaction with water, of ATP to produce ADP and a free phosphate group. Now, when we write it this way, it implies that the breakdown of ATP is entirely separate from the biosynthesis reaction that it's fueling, when in fact, in the body, coupled reactions such as these often occur simultaneously. In fact, almost always, ATP is directly involved in the chemical reaction that it's fueling by directly donating or transferring one of its phosphate groups to the reaction it's involved in. And in this case, in our example, it donates a phosphate group directly to the monomer. To understand this mechanism even better, let's go ahead and take a look at a more tangible example in which we take a monomer and convert it into a polymer. So let's actually go ahead and take a look at building up nucleotides into a polynucleotide chain, such as DNA. So for the sake of simplicity, I'm going to go ahead and just draw a symbol for our nucleotide instead of writing out the entire chemical formula. But let's go ahead and review what the components of a nucleotide are. So remember that a nucleotide is composed of a sugar and a nitrogenous base, along with a phosphate group. And I'm going to highlight this phosphate group because it will be particularly important in the covalent bonds that form between nucleotides to form DNA. In addition, the sugars have a hydroxyl group that are also involved in these bonds. I'm going to highlight that here as well. In the very first step of the reaction, two ATP molecules make an appearance, and specifically, they donate one phosphate group each to the phosphate group already on the nucleotide. Let's go ahead and draw out what our products look like. So we have our nucleotide and our untouched hydroxyl group, but now instead of one phosphate group, we have a total of 3 phosphate groups that are covalently bonded to one another. And because ATP gave up its phosphate group, we are left with 2 molecules of ADP. Now there's one point of confusion when we're talking about the breakdown of ATP into ADP. Many of us fall into saying that it's the breakdown of ATP into ADP that provides the energy for a reaction. But it turns out that this step of the reaction, in which we're essentially stacking up all these negatively charged phosphate groups that don't want to be next to each other, has a positive delta G value and cannot drive the reaction forward. But what does drive this overall reaction forward is the second step of the reaction. So let's talk about what happens in the second step. In the second step of the reaction, in comes a growing polynucleotide chain. So I'm going to go ahead and just draw 2 of these nucleotides. And remember that on one end of the growing polynucleotide chain is a hydroxyl group, which I'm highlighting in green. And on the other end is a phosphate group, which I'm highlighting in blue. Bonds between the nucleotides involve both the hydroxyl group in green and the phosphate group in blue. In this step of the reaction, the hydroxyl group of the growing polynucleotide chain essentially displaces the phosphate groups that were added in the previous step of the reaction. Now recall that this is possible because having 3 phosphate groups all crowded together like this makes the electrons in the bonds between the phosphate groups very energetically unstable. Think about all that negative charge that's close together that doesn't want to be. So let's go ahead and draw out what our final product looks like. I'm going to go ahead and copy and paste this polynucleotide chain so we don't have to redraw all of it. So here it is. Go ahead and erase
it's fine when I comb it, but
and on the hydroxyl end, which is this green end right here, we have covalently bonded our new nucleotide. And so I'm just
going into my left eye.
Is the one that
Oh my god. Oh my god, it's so annoying.
And the other hydroxyl group remains on the other end of the nucleotide, ready for another nucleotide to attach. Now let's not forget the paired phosphate group that was displaced in forming this covalent bond. This paired phosphate group, which has a name that you don't need to remember, it's called a pyrophosphate group, undergoes a hydrolysis reaction, that is, reaction with water, to produce 2 independent phosphate groups. Now, the particular reaction mechanism isn't that relevant, but just so you know, the water essentially inserts itself asymmetrically in between these 2 phosphate groups and basically donates a hydroxyl group to one and a hydrogen to another. And because the split was asymmetric, these 2 phosphate groups still remain identical to one another. Now, the key point to take away here is that this reaction with water, this hydrolysis reaction, is very energetically favorable. Specifically, we say that it has a fairly large negative delta G value. And so it is this step of the reaction that ultimately drives this entire reaction forward. Now, if that doesn't fully click in your minds, let's also think about this from another perspective. Let's think about this from the perspective of equilibrium. So remember, there's a principle in chemistry called Le Chatelier's principle. And Le Chatelier's principle says that a system, such as a chemical reaction, will respond to a change in the system, such as a change in temperature or change in concentration of reactants or products, by shifting in the direction that counters that change. So in this case, what is the change in our system? Well, the change in our system is that we are degrading our products. So remember, this phosphate group, this pyrophosphate group, is being broken down into individual phosphate groups. And so essentially what we're doing is we are removing the products of our reaction. And if we do this, the reaction will shift in the forward direction to produce more products. And the shift in the forward direction pushes this reaction forward. So ultimately, the reason I wanted to go through this mechanism with you was to really show you that ATP provides energy to a reaction by donating one of its phosphate groups or transferring one of its phosphate groups, and not through a simple hydrolysis reaction, even though that's often how it's written for convenience. Now, one question I had about ATP hydrolysis when I was learning it was that if this overall reaction here, this coupled reaction, is energetically favorable,
wait, that's it?
Overall reaction has a negative delta G value. Why doesn't ATP just donate its phosphate groups to any substrate in the body?
Is that the whole point of this?
So potentially could donate its phosphate group to water and expend a lot of wasteful energy.
To demonstrate.
Involves reminding ourselves of the 2 factors that control whether or not a reaction will occur. So the first one is energetic. So is the reaction
it's just here to demonstrate that the phosphate group is getting dominated? I mean, isn't that the whole point? I'm confused.
But the other factor that controls whether or not a reaction will occur is kinetics. And we usually talk about a parameter called the activation energy.
Am I just too advanced to understand this simple concept?
ATP has a very high
Or am I missing the broader
I'm just going to
point?
skim the surface of this topic since our video is devoted just to this topic alone, but essentially we say that this reaction is kinetically stable because it has a very high activation energy.
Banana for scale. Got him. Banana. Just scale it. Oh, what? That sounded a lot more gross than it actually was. I was trying to eat over here, man. While standing, I guess. Let's sit down. Ah. Still got plenty of stream left. Another 2 hours and we'll be halfway there. I'm not even done with this unit though. That's really annoying. But whatever happens, I need to like comb my hair. This is getting ridiculous. Some gel in this thing. Ever tried to learn ATP hydrolysis at 6 a.m.? Just me? Okay. I'm not even sure if I want this stream to still be like accessible after this. There's nothing like particularly wrong with it, but like, man, just not that interesting content. But hey, if this somehow stays up, and you're that one like fan, that one Nick super fan, well, congratulations. Watch the whole thing. Hope you're pre-med. You are interested in bio. Bio's really interesting anyway. Okay, hair gel to save the day. Now it shouldn't get in my eye like earlier. Nice. It's not supposed to. It's gonna be really annoying if it does. Man. I've watched a significant amount of Trash Taste's 24-hour stream, and like Garnt needed to eat, or wait, no, Connor needed to like eat natto or something. Just like I can't imagine like eating something that you absolutely abhor, regardless of what it is. Like if it tastes disgusting to you, then it is disgusting, right? Because I kind of like feel nauseous, but like not really at the same time. It's a weird feeling. I am acutely aware that my deprived state awfully similar to my drunken state. Or at least tipsy state. I don't think I've ever gotten fully drunk yet, but you know. Regardless, the point is feeling a bit woozy probably. But it is what it is. I think part of it is just the pure psychological stress of the challenge. Being that, like, I'm feeling more tired because I know that I'm not supposed to, like, because I'm up against this challenge of not falling asleep, I guess. So there's part of that. That might be part of it. But hey, I have my Saul Goodman haircut now, so I guess I'm getting a second wind here. Alright, let's do this. So, ATP. ATP hydrolysis. Let's go back to the last one and reorganize our thoughts so that I can make sure that I know what's going on.
Recall that delta G is a quantitative change in enthalpy as well as a change in entropy, which are two topics that you might be free phosphate group that it cleaves off. So on first glance, it might seem that this
Oh, great. I have something in between my teeth. Hold up. This has been the worst idea in the history of ideas. And yet I knew I would say that. So, really, who's winning? Okay, Output of energy, releases energy, hydrolysis. What? Okay. I mean, I guess hydrolysis is the pathway in which we go from ATP to ADP, so let's do this, I guess. Ugh. I think it doesn't help that I'm eating some, like, slimier foods. Kind of makes me gag a bit. Ugh. Like it. Not a fan. Like, eat some refreshing, or chew on some more refreshing gum or something. Watching people live stream, probably a very easy way to identify who's way too comfortable with talking to themselves. Should that imply anything? I don't know. Where's the studies on live streamers, huh? Maybe I should actually search that up. I feel like I'd be very interested to see if they learn anything about live streamers. It's one thing to study the parasocial relationship from the audience side, but I'm very curious about how it affects entertainers.
And the way our body deals with this is that our body has enzymes.
And yes, regardless of the audience size.
When it should proceed.
Silly me trying to stream 24 hours with 4 subscribers. I don't know, man. I just feel like it.
We are left with 2 molecules of ADP. Now, there's one point of confusion when we're talking about the breakdown of ATP into ADP. Erase the parts that ultimately drives this entire reaction by donating one of its phosphate groups or transferring one of its phosphate groups, and not through a. And we usually talk about a parameter called the activation energy, which I'm going to abbreviate here as E sub A. And it turns out in this example, ATP has a very high activation energy. And I'm just going to skim the surface of this topic since our video is devoted just to this topic alone. But essentially, we say that this reaction is kinetically stable because it has a very high activation energy. And the way our body deals with this is that our body has enzymes that can lower the activation energy and allow the reaction to proceed when it should proceed. And so in our example, each step of the reaction has a specific enzyme that facilitates or catalyzes each step of the reaction. And this is actually very beneficial for the body because by having an energetically favorable but kinetically stable reaction, the body can selectively control whether or not energy will be used by controlling the enzymes that are available for the reaction to proceed. And so it's a very elegant system, and I hope that ultimately this entire video has given you a better picture of how ATP fuels energy requiring processes on a molecular level.
Okay. I mean, ironically, the thing that I just put into my notes is the exact thing that they were trying to teach me isn't necessarily true all the time. Or not all the time. The point is, it's not the important part, hydrolysis. The important part is the phosphate going to a new group. Well, whatever. I'm not gonna be concerned about all that right now. I just want to like make sure I have all the terms on represented on this map, and then technicalities are things that we need to work out later.
And then see how what we learned in our chemistry class relates to the way that a biologist or biochemist might use these words, and hopefully we'll see that they're the same thing. So just as a bit of review, if you watch the chemistry playlist, oxidation, you can do, and actually there's a famous mnemonic for it, it's an oil rig, where the oil tells us that oxidation is losing, and I put it in quotes because you're not necessarily losing the electrons, I'll show you what I mean, is losing electrons. This is what you should have learned in your chemistry class. And then you also learned that reduction is gaining, and I'll put that in quotes as well, is gaining electrons. And I put that in quotes because you're not necessarily gaining
Oh
them,
no.
you're more hogging them. The reason why it's called reduction is because if you are gaining electrons, your
Oh no.
gaining them is being reduced. The reason why this is called oxidizing is because you tend to
Redox. Fucking redox.
that will hog electrons away from it. And I think a nice example would be fair to kind of make this a little bit more concrete. Let's say I took some molecular hydrogen, it's in a gaseous state, and I were to combust that with some molecular oxygen. This is what happened on the Hindenburg. They fill a balloon full of hydrogen, and you get a little bit of spark, expose it to oxygen, and you're going to have a big explosion. But in the process, for every mole of molecular oxygen, if you have 2 moles of molecular hydrogen, I'm just making sure the equation is balanced, you're going to produce 2 moles of H2O plus a ton of
hydrogen monoxide.
A lot of heat.
Don't you know that kills people? No, what? What the fuck? That's messed up. Everyone's who's taken, who's touched dihydrogen monoxide has died. Like, come on, man. It's terrifying.
For every oxygen is connected to 2 hydrogens. And the way to think about it is that oxygen is hogging each of these hydrogens' electrons. So hydrogen has this 1 electron in its valence shell. You know, the deal with most covalent bonding is give you an electron.
So I think the way to handle this is redox. Oxidation. Reduction. That's it. That's the whole thing. That's all you need, baby. That's literally it.
You give me electrons, we both have a complete pair. But we know, or hopefully we can review, that oxygen is much more electronegative than hydrogen. This is the glucose reminiscence that is left over from our cellular respiration video. You can ignore it for now, but I'm actually going to connect all this in a future video. If we look at our periodic table, if you remember from the chemistry playlist, electronegativity increases as we go to the top right of the periodic table. These are the most electronegative elements over here. These are the least electronegative. And all electronegative means is likes to hog electrons. Let me write that. Electronegative just means likes to hog electrons. So even though oxygen and hydrogen are in a covalent bond in water, they're sharing electrons. Oxygen is more electronegative, much more electronegative than hydrogen. So it's going to hog the electrons. And actually, if you take some elements on this side and you bond them with some guys over here, these are so much more electronegative than these left-hand elements. They'll actually completely steal the electron, not just hog it for most of the time. But when you talk about electronegativity, it just means likes the electrons. So when you look at this bond between hydrogen and oxygen, we saw from the periodic table, oxygen is a lot more electronegative. So the electrons spend a lot more time on oxygen. When we learned about a hydrogen bonding, we learned that it creates a partial negative charge on that side of the water molecule, and it creates partial positive charges on this side. And the electrons still show up around the hydrogens every now and then. When you talk about oxidation and reduction, you say, look, there's no partial charge. If one guy is kind of hogging the electron more, for the sake of oxidation states, we're going to assume that he took the electron. So for an oxidation state, we'll assume that the oxygen in water takes the electron, and we'll give him an oxidation state of 1 minus, where the convention is you write the charge after the number for oxidation state, so you don't confuse it with actual charges. So this has a 1 minus because from an oxidation state point of view, it's taking the electron, it's gaining the electron. That's why I put it in quotes, because you're not really gaining it, you're just gaining it most of the time. You're hogging electrons. And likewise, this hydrogen, let me be careful, this isn't, he got 1 electron from this hydrogen, he got another electron from this hydrogen. So instead of saying 1 minus, it should be 2 minus. It should be 2.
This is such an epically stupid track. Or, wait. Stupidly epic. Stupidly epic. Sorry. Oh no. My sleep deprivation. It's showing up. But not in the form I was expecting it to. But hey, it works. Come on, baby. Let's go. Let's keep on going, baby.
Non-oxygen atoms or non-oxygen elements, it tends to have a 2 minus or negative 2 oxidation state. So this guy's 2 minus because he gained 2 electrons, right in quotes, gained 2 electrons. We know that he didn't gain them, that he's just hogging them. These guys lost an electron each. So this guy's oxidation state is going to be 1 plus, and this guy's oxidation state is going to be 1 plus. So you could say, by combusting the hydrogen with the oxygen, that the hydrogens, before they had a 0 oxidation state, right? Each of these hydrogens had a 0 oxidation state. Now they have a 1 plus oxidation state because they lost their electrons when they bonded with the oxygen. So we say that these hydrogens have been oxidized. We say that the hydrogen has been oxidized. So due to this reaction, hydrogen has been oxidized. Why has it been oxidized? Because before it was able to share its electrons very nicely, but then it bonds with oxygen, which will hog its electrons. So the hydrogen is losing its electrons to the oxygen, so it's been oxidized. Similarly, the oxygen, due to this combustion reaction, due to this reaction right here, has been reduced. Why has it been reduced? Here it was just sharing electrons, it wasn't losing or gaining it. But here, when it's bonded with an element with much lower electronegativity, all of a sudden it can start hogging the electrons. It gains electrons. So this hypothetical charge is reduced by 2. It's been reduced by, well, I can write it down, by 2. And if I wanted to actually account for all of the electrons, we're talking about losing electrons and gaining electrons, we can write 2 half reactions. This should all be a little bit of a review from your chemistry class, but it never hurts to see this again. I'm going to throw this in the biology playlist so that you biology people can hopefully refresh your memory with this stuff. We can write 2 half reactions. We can say that we start off with 2 moles of molecular hydrogen, and they have no oxidation states, or they're neutral, so I can write a 0 there if I want. And then I end up with, on the other side, I end up with 2 moles of H2, but each of the hydrogens have a +1 oxidation state. They each have a +1, it's 1 plus oxidation state. Or another way to think about it is, each of these, there's 4 hydrogens here, right? This is, molecular hydrogen has 2 hydrogens, and we have 2 moles of this, so there are 4 hydrogens here. Each of the 4 hydrogens lost an electron, so I can write this, so plus 4 electrons. That's the half reaction for hydrogen. It lost 4 electrons, so this is another way of saying that hydrogen is oxidized, because it lost electrons, oil, oxidation is losing. And then the other half reaction, if I were to write the water, or sorry, if I were to write the oxygen, so I'm starting with a mole of molecular oxygen, and I'm adding to that 4 electrons, right? I can't make electrons out of nowhere. I'm getting the electrons from the hydrogen, I'm adding to the oxygen. And so the half reaction on this side, I end up with 2 moles, well, I can write it like this, 2 moles of oxygen, and each of them have an oxidation state of 2 minus. So these are the half
Okay.
reactions, and all this is showing is that the
All right.
over the course of this combustion
That's pretty
lost
simple.
electrons, and
That
the
makes
oxygen gained
sense.
the electrons that the hydrogen lost. So this tells us that oxygen.
I don't know why I'm always... no, I know why I'm scared of
Now, this is all very
oxygen.
good. This is all a bit of review.
Why I'm scared of oxidation. You can't be scared of oxygen. Oxygen poisoning is a thing if you go too far, but oxidation reactions are rather redox reactions in general. Scare the hell out of me because I completely bombed the AP Chem test on it. But I'm back, baby. It's time to get some redemption for the final boss. Final boss of the first game, right? Final boss of undergraduate. The MCAT. Come on, baby. Come on, baby.
And we see where carbon, nitrogen, oxygen, and phosphorus, and really all this other stuff is. You see that all the stuff that in biological systems hydrogen tends to bond with, the things it tends to bond with are much, much more electronegative. So if a carbon is bonding with a hydrogen, the carbon is hogging that electron. And then if that hydrogen gets transferred to an oxygen along with the electron, the carbon will lose, it lost the hydrogen atom, but it really lost the electron that it was hogging before. And now the oxygen can hog that electron. So these are really consistent definitions. And the whole reason why I showed you this example is because the biological definition kind of doesn't apply here. I mean, you could say, well, oxygen is definitely gaining hydrogens in this reaction. You can definitely say that oxygen is being reduced still, according to the biological definition. But you can't really say that hydrogen is losing hydrogens here, right? In this situation, hydrogen is just losing electrons. It's not losing itself. I guess you could say it's losing itself because it's being taken over. But the biological definition just comes from the same notion that when hydrogen bonds with most things in biological compounds, it tends to give the electrons. So if a carbon loses a hydrogen and gives it to an oxygen, the carbon will lose that hydrogen's electron that it was able to hog. Now the oxygen is hogging it. So the carbon would be oxidized and the oxygen would be reduced. Hope that doesn't confuse you. In the next video, I'll show you a couple more examples. And the whole reason why I'm doing this is to apply this to cellular respiration, so that you don't get confused when people say that, oh, the NAD is being reduced when it picks up the hydrogen, or it's being oxidized when it loses the hydrogen, and so forth and so on. I wanted to see that these are the same definitions that you learned in your chemistry class.
Okay. Man, I'm really gaining a lot of energy points. Hell yeah. Yo, I can upgrade my profile picture. Yay. Thank you, Khan Academy. Very cool.
I really want to go ahead and integrate some of the topics that you learned in general chemistry with topics in metabolism. Specifically, from a general chemistry perspective, I want to review electrochemistry. And then I'm going to connect it to one of the biggest topics in metabolism, the breakdown of glucose in a process called cellular respiration. And if you recall, cellular respiration describes the body's way of being able to efficiently produce the energy currency of the cell, which is ATP. Now, admittedly, this is kind of an unconventional way to first present the topic of cellular respiration, but what I think is really neat, and what I think you'll realize, is that we're really killing 2 birds with 1 stone here, because you understand the concept of electrochemistry, you also understand cellular respiration, and vice versa. So let's go ahead and get started by returning to the topic of electrochemistry from general chemistry class, and let's go ahead and review a simple oxidation and reduction reaction between solid zinc and copper ions floating around in solution. So the AQ just stands for aqueous solution, and the products of this reaction are zinc ion and solid copper. Remember that whenever we're talking about reduction oxidation reactions, they're commonly referred to as...
Wait a minute. So redox was going to be used so that we can learn about metabolism? Are you serious? Oh, I should have like looked into the future more. Oh no. Oh, you gotta be kidding me. Oh, it's all the way up there. Oh shit. Okay. Yeah, you know what? I think metabolism may as well just come on over to the chem section. Why not? Come on over. It's gonna be fine. The water's fine, you know? Metabolism's here now.
Redox reactions, we're talking about the flow of electrons. We must ask the question, what is gaining electrons and what's losing electrons? So copper here, which is positively charged, is gaining electrons and therefore being turned into solid copper, which has no charge. So we say it's gaining electrons, which means it's being reduced.
The playback is at 2 times speed, by the way. I don't know, it just sounds normal to me. Not gonna lie, this actually makes more sense than if they were talking slowly. Like, watch.
On the other hand, solid zinc is turning into a positive charge, so it's losing negative charge. So we say here that it's losing electrons. It's being oxidized.
Oxidize.
Loss of electrons is to note here that there is a flow of electrons
no, there's a
from the
flow
zinc,
of
which is losing electrons, to the copper ions, which are then reduced to solid copper. And this, of course, is where
This is way too slow, man.
electrochemistry
I'm
comes in,
I'm
because
dying over here, man.
electrochemistry allows us to isolate this flow
Come
of
on.
electrons. by
Yeah,
building what's
that's
called
more
an
like it.
electrochemical cell. So let's go ahead and build a electrochemical cell for this particular
I mean,
reduction
bro,
oxidation
my
reaction. So I'm gonna go ahead and draw two containers, which if you recall,
like the average BPM of the music that I listen to is at least like 140. This is just how I live my life. I live on 2 times speed, except for anime, because I think that that's more, you know, out of respect for art and for, no, for designed pacing, I guess. I mean, these are designed quite well too, but just not quite the same. Or maybe it's just with this I have more of a rationale for ignoring the given pacing and going at it for 2 times speed. Three times speed is my normal, actually. If I have the option for 3 times speed, I'll go for it. 2 times speed is just kind of like, it depends on the presenter.
and remember that they're connected
Sometimes it's too slow,
wire
sometimes it's
that
too
we can
fast.
isolate the flow of electrons. And there's also another component called a salt bridge, which connects these two half cells. Just as a brief review, remember that this allows a flow of ions between each cell so that there's not a buildup of charge from this movement of electrons. Let's go ahead and remind ourselves where these electrons are coming from. So remember, the electrons are flowing from the zinc, which is losing electrons, to the copper, which is gaining electrons. And this is really the ingenious part of the electrochemical cell. It allows us to separate what's losing electrons, in this case solid zinc, from what's gaining electrons, which
In this case, solid zinc.
and therefore allows us to isolate this flow of electrons through a wire. And of course, we also have zinc ions and
I swear I'm paying attention.
The take-home message is we're really separating what's getting
Oh my
oxidized
god.
from what's getting reduced. And ultimately, this
I
flow
can't.
of electrons through the wire, which is also referred
Why
to
is
as
the
flow,
audio so
allows us to
different?
perform energy-requiring processes such as in
In copper?
a light bulb. Now, here's
In the
another
copper?
point that I want to make in this video. In our body, we also isolate a flow of electrons during cellular respiration. But instead of lighting up a light bulb, we harness this flow of electrons to produce chemical energy in the form of ATP. So let's go ahead and take a look at how this works by looking at the overall reaction for cellular respiration. Specifically here, we're looking at the breakdown.
Wait,
of
I
carbon.
actually
So glucose
failed
is
to
a chemical
understand
carbon,
why
sulfur,
this
hydrogen,
is
and
important.
oxygen. And even though it's broken down in many multiple steps, the overall reaction for the breakdown of glucose is glucose combining with oxygen to produce.
Wait, why are we learning
water
about
and
this right now?
dioxide. And I'm gonna go ahead and put in our
You said that it was
here.
for
This overall
metabolism,
reaction
but
is also a reduction
I
of
don't
the
see
glucose
the
that involves a flow of
connection.
electrons, just like our zinc and copper example. The
Oh
flow of
wait,
electrons
they're
is
literally
also
talking about it right
take
now.
a minute and review kind of the oxidation and reduction
Cellular
from a biological
respiration.
perspective by looking at this reaction. So in the zinc and copper example, it was clear what was gaining and losing negative charge, but here we have to really think about electron density. And so when we look at this molecule of glucose, it is oxidized. That is, it loses electron density to form carbon dioxide. And the way we can rationalize this, I'm going to go ahead and say that this is oxidized. We look at the carbon. When it's attached to two atoms of oxygen, the oxygen atoms essentially steal the show because they are much more electronegative than the carbon. So carbon has very little electron density. It lost a lot of it from before when it was surrounded by hydrogens as well, which didn't steal the electrons as much. Now remember, if something's being oxidized, something must be reduced. In this case, our oxygen molecule, which is also referred to as kind of a final electron acceptor, is being reduced. It's gaining electron density. It's essentially gaining the electrons and the hydrogens that were lost from our glucose molecule. So here we have a gain of hydrogen and we form water. So the water is the reduced product of the oxygen. Now once again, we can summarize this flow of electrons by saying that the electrons flow from the glucose to the oxygen. So with this written out, I really want to go back to our electrochemical cell up here and point out that the solid zinc is really analogous to our glucose because both of these are losing electrons, and the copper ion here is really analogous to our oxygen because both of these are getting reduced. So I'm actually going to go ahead and erase the zinc and the copper and replace it with the glucose and the oxygen to essentially show you that there's kind of an analogy here between the electrochemical cell and cellular respiration. Now, clearly I'm not being very technical about the creation of this electrochemical cell, mostly because, as you can guess, this really isn't what's happening in our body, but it's really this flow of electrons that our body harnesses. And instead of lighting up a light bulb here, which I'm going to erase here, instead of lighting up this light bulb, our body uses this flow of electrons, it harnesses it to allow the body to convert ADP back into ATP, which is, of course, the entire goal of cellular
yeah,
respiration.
baby.
And ATP can
That's
fuel
what I've
all
been
the
waiting
cellular
for. That's what it's all
processes.
about.
We can continue to extend this analogy between cellular respiration and the electrochemical cell we've drawn by pointing out that even though we don't have, a wire or separate containers in our body, it turns out that our body has actually functional equivalents of both of these two things. So let's start off with a wire. So in our body, instead of a wire, we have something called the electron transport chain, which I'm going to abbreviate here as ETC.
Oh,
the
hey.
Transport chain contains an array of proteins.
Electron transport chain, shout out.
Having a functional equivalent for having separate containers, our body is able to compartmentalize many of the reactions involved in cellular respiration. And specifically, I want to point out, you'll run into the fact that cellular respiration in its final step, which is called oxidative phosphorylation, occurs in the mitochondria. So I'm going to draw an organelle that looks like a mitochondria. And the mitochondria
Oh, we're doing this now? Oh, shit. We're doing it now.
As well as the outside.
Okay, hold up. How do I want to play this? Oh, shoot. We're doing this now? Really? Okay. Wait. Big brain. Okay. And then I'll loop it over here. Okay. And then electron transport chain. Oh, this is such a mess. Oh, I hate this so much. Oh, the short-sightedness. Will I really be able to clean this up? I don't know. I mean, I guess the mess is kind of the point, right? Just to get the stuff on paper. Oh, thank God it's not actually on paper, though. Whatever this is. Holy shit, mitochondria is the power of the cell? What the fuck? That's wild, man. That's wild. Yeah, no, I hear it pretty clearly. Like, it's probably because most of it, or no, all of this is review for me. And I'm glad that I'm actually able to understand it all because it shows that I did learn something in my classes. The problem is, it's so weird because I just can't replicate this in normal conversation. When people ask me, hey, what happens in the mitochondria? I'm like, wheel go burr. Which you'll see eventually is just basically the extended version of the electron transport chain, creating that electron gradient that ultimately pushes against a kind of like a rotor that combines the phosphate groups to the ADP, thus forming ATP. But for some reason, they're kind of taking the long way around to explain that, so I'm kind of getting a little bit confused as to where we're gonna go next. It's kind of like, I don't know if I like this pacing, man. It's just kind of pacing kind of mid, not gonna lie.
That are more and more oxidized, and these metabolites essentially donate electrons at each step to molecules that are
Metabolites?
electron carrier molecules. And as a technicality, I want to point out that it's really these electron carrier molecules that ultimately donate electrons to the electron transport chain and ultimately to oxygen. So I'm going to go ahead and indicate this with an asterisk here and note that it's really these electron carrier molecules
There's two.
which directly donate electrons into this circuit.
What the heck? They're not shouting out my favorite characters. What the heck? This is BS, man. Oh, they're coming up right now. Oh, okay. Never mind. Oh, that's awkward. I didn't mean to say anything wrong about that. You know, it's good job.
called electron carrier molecules that really do play a vital role in this energy production process as well. But in order to talk about electron carrier molecules, we need to first review the main character of our story. So let's start off with glucose. So glucose, which has a chemical formula of 6 carbons, 12 hydrogens, and 6 oxygens, we know is broken down in our body to produce energy. And specifically, this breakdown process is an oxidation process, a process of losing.
I was about to ask,
glucose is oxidized to
is glucose the main character? And then I'm like, wait, no, it is, because it literally has a hero's journey. No, I can't listen to Rap God and understand what he's saying. Although, well, so this is the equivalent of listening to Rap God after having read the lyrics, which is completely different. So if I look at the lyrics of Rap God, like, and read it over a couple times, maybe even get tested on it, and then come back to it, like, I don't know, months later, maybe even years, maybe I'll understand a decent amount of what he's saying, you know? But, yeah, not really equivalent. I do like Eminem stuff, though.
Definitely review one of the previous videos on oxidation and reduction from a biological viewpoint, but the key idea is to notice that this carbon is far more oxidized or electron poor when it is attached to hydrogen and oxygen molecule than when it's attached to hydrogen. So that's the key idea here. Now, in our bodies, this reaction does not take place in a single step. Instead, it takes place in many steps. And so I'm going to actually literally draw a staircase with many steps to indicate this. And I just want to note that this collective process of breaking down glucose in several steps is called cellular respiration, and that there are videos just devoted to the details of this process. Alright, so now we're ready to talk about our electron carrier molecules. And we can think about our electron carrier molecules a bit like a molecular shuttle. I'm drawing a picture of an actual shuttle here just to remind us of this analogy. At each step of glucose's breakdown or oxidation, a new breakdown product of glucose is formed that is given a fancy name of a metabolite. And just for short, I'm going to abbreviate the rest of these as M. Now, the key point to realize here is that as glucose is broken down, the metabolites are more and more oxidized. That is to say, they have less electron density. And this is where our electron carriers come in. So recall that energy is extracted from this oxidation process by harnessing the flow of electrons. And it is the job of the electron carriers to harness the electrons that are lost at each step of the breakdown process. And so I'm going to go ahead and draw a metaphorical shuttle for our electron carrier molecule at each stage of glucose's breakdown. And these electron carrier molecules are, of course, carrying the electrons that are lost from the oxidation process. Ultimately, all of these electron carrier molecules are going to shuttle the electrons that are harnessed from the breakdown of glucose to something called the electron transport chain, which is in the mitochondria. And at this point, there are enzymes that can facilitate the transfer of these electrons to the final electron acceptor in our body, which is oxygen. And remember, it is this flow of electrons in the electron transport chain that allows the production of energy to produce ATP, the cell's currency of energy. So one question that you might have is, why does the body have such a complex mechanism that involves many steps and all of these electron carrier molecules to fuel the production of ATP? Well, let's reconsider the chemical equation that describes the overall breakdown of glucose. So glucose combines with oxygen to produce carbon dioxide and water. And just so that we conserve our mass, let's put in our stoichiometry. So this describes the overall breakdown of glucose, and we know that this reaction releases energy to fuel the production of ATP. Now, another way to view this process is as a simple combustion process, and combustion is exactly what it sounds like. It's what you think of when you think of something burning. And a combustion process just requires a fuel, and in this case, our carbon-rich or electron-rich molecule of glucose can be considered a fuel, and it also requires molecular oxygen, which we also have here. So to reword the question that I just posed, why doesn't glucose, the fuel, spontaneously combust in one step instead of being broken down in multiple steps inside our body? Let's first think about the
Glucose just explodes.
Spontaneously combusted in our body.
It's just like,
all of that would be unusable energy. It would be unusable because
yeet that shit into the fires, boys.
Is able to use in the form of ATP. And
Like, no. No, that's not quite how it works.
Energetically favorable, that is to say, it releases energy.
It'd be kind of epic if it did, though. I guess that would be, that might be how androids work.
Only happens when you overcook something at a high temperature.
And like high sci-fi, you like just burn the glucose, like literally just burn it.
At a temperature much lower than that needed for a combustion process, which is thankful because otherwise we would be producing all of this unusable energy. Instead, our body overcomes the high activation energy of this reaction by using enzymes at each step of the reaction. So I'm going to abbreviate this.
Wouldn't that be more steampunk?
A multitude of enzymes to break down glucose. So the first benefit, so I'm going to write benefit
but then
enzymes. So the first benefit is that we're able to produce a large number of metabolites, and this is important because it allows our body to essentially reshuffle all of the metabolic products into many different pathways so that we can reuse and recycle these products as much as possible. And the second benefit of using many enzymes to break down glucose is that we have a slow and controlled oxidation of glucose, which allows us to harness all of that energy, which is in the form of the electrons that are being oxidized in a very controlled manner, as opposed to the big burst of heat that we would get in a single-step combustion process. And electron carrier molecules play a big role.
Oh god, now I'm designing like,
enzymes, as you can see in our diagram.
I'm like designing robots in my mind that either run on like breakdown of glucose through combustion. Or would just the more practical way to do it is just like your solar panels on your shoulders or something, and then just like maintain the energy.
That is to say that they take away hydrogen, and when they're taking away hydrogen, they also take away electrons. And most
I use renewable energy though.
Along with a proton, which is what chemists call a hydride. And and its hydride has a negative charge, so it's often referred
What
to
am
as
I
a
even
hydride
talking
ion.
about?
And
What?
notice
Where
that
are we
it
right now?
has one proton
What is
plus two
happening?
electrons.
Oh
Now, the two
shoot.
electron carrier molecules, or coenzymes, that are most commonly discussed in the breakdown of glucose are two molecules that go by the name of NAD and FAD. And I want to write down the reaction that occurs between these electron carrier
NAD
molecules and the electrons
and
that they're
FAD.
accepting from the glucose molecule through this dehydrogenase enzyme. So first, it's notable that NAD has a positive charge in its most oxidized form, while FAD does not.
Wait,
And this
so
coenzyme reacts with two protons and two electrons each, in other words, reacting with a hydride ion plus an extra proton. And once the electron carrier molecules accept the electrons in this way, they become reduced. And the reduction products are slightly different for each. So in the case of NAD, it's reduced into NADH. So notice that we only have one hydrogen. And the reason for that is NAD can only accept one hydride ion. On the other hand, FAD can accept both hydrogens. So it's reduced to FADH2. The extra proton is just donated to solution in the case of NAD. And it is these molecules here, these reduced form of our electron carrier molecules, that shuttle the electrons to the electron transfer chain to allow for the production of ATP.
okay, hold up. So I understood that the first half of this.
Normally, activation energy. And you know this
Actually, I understand the second half as well,
Then
but
our body overcomes the high
it's more so I
and
don't
the second benefit of
really
using enzymes
know
is that we have a slow
what is
oxidation of glucose,
this.
which allows us to harness all of that energy, which is in the form of the electrons that are being oxidized, in a very controlled manner, as opposed to the big bursts of heat that we would get in a single-step combustion process.
Oh,
Carrier molecules play a big
I
role
mean,
in
yeah,
all
of course,
the
combustion
that
is
facilitated by
problematic.
enzymes, as you can see in our diagram above, because they are essentially serving as a temporary storage for all of the electrons that are being lost by glucose. And in fact, because of their close association with the
That's not even
enzymes that facilitate
that's
the
a
breakdown of
non-starter.
glucose,
Like,
electron
of
carriers
course,
are
it's
also
going to be
called coenzymes.
inefficient.
Coenzyme is exactly what it sounds like. It's a molecule, or it's a chemical functional group that
Oh my
coenzymes
god,
function.
bro, come
Now, the enzymes
on.
involved in the breakdown of glucose, for the most part, are in the class of enzymes that have a special name called dehydrogenases. And these dehydrogenase enzymes do exactly what their name implies. They dehydrogenate the glucose. That is to say that they take away hydrogen. And when they're taking
Dehydrogenate. Oh shoot. Okay. So I think go here. And then no, and then this leads to electron carriers, I think. Yes. Okay, so it's gonna get destroyed. We're gonna move this here. Electron carriers include NADH and FAD. FAD. FATH2.
away hydrogens, they also take away electrons. And most often they do this by taking away two electrons along with a proton, which is what chemists call a hydride. And this hydride has a negative charge, so it's often referred to as a hydride ion. And just another way to think of this is as one proton plus two electrons. Now, the two electron carrier molecules, or coenzymes, that are most commonly discussed in the breakdown of glucose are two molecules that go by the name of NAD and F.
Oh wait, is NAD the wait? Oh shoot, are these the electron carriers, or are they? Or is the electron carrier both that component plus the hydride?
Along with a proton, which is what chemists call a hydride. And this hydride has a negative charge, so it's often referred to as a hydride ion. And just another way to think of this is as one proton plus two electrons. Now, the two electron carrier molecules, or coenzymes, that are most commonly discussed in the breakdown of glucose are two molecules that go by the name of NAD and FAD. And I want to write down the reaction that occurs between these electron carrier molecules and the electrons that they're accepting from the glucose molecule through this dehydrogenase.
Okay, so the electron carrier itself is the NAD. Oh shit. Okay.
And each coenzyme reacts with two protons and two electrons each. In other words, reacting with a hydride ion plus an extra proton. And once the electron carrier molecules accept the electrons in this way, they become reduced. And the reduction products are slightly different for each. So in the case of NAD, it's reduced into NADH. So notice that we only have one hydrogen. And the reason for that is NAD can only accept one hydride ion. On the other hand, FAD can accept both hydrogens. So it's reduced to FADH2. The extra proton is just donated to solution in the case of NAD. And it is these molecules here, these reduced form of our electron carrier molecules, that shuttle the electrons to the electron transfer chain to allow for the production of ATP.
Whoa, whoops. Okay, there we go. Got it. Okay. Oh shit. Okay, I think we're almost done here. I think this is the last video in this unit. Oh my
So in
god.
the previous video, we talked about how an ATP molecule can, in the presence of water, hydrolysis will take place, and one of the phosphoryl groups could be plunked off, and then how that would release energy because these electrons are going to be able to go into a lower energy state. That, you know, they were kind of, you could, one thing that was uncomfortable, you can imagine that this was already kind of not that stable of a bond, that all these negative charges want to get away from each other, and once this is plunked off, then of course, when they get into a more comfortable state, energy is released. But you might say, well, I want more. I want to actually see the mechanism by which the hydrolysis takes place. And so that's what I'm going to do in this video. So let's start with our ATP molecule, and let's throw some water in there, H2O. So let's say this is water right here, oxygen, 2 hydrogens, and I'll do the 2 pairs of oxygen that aren't in bonds right over here in the outermost shell. And actually, let me draw one more water molecule right over here. And there's multiple ways that you can actually kind of depict this right over here. But let's say that one of these, let's say this water molecule right here, and obviously no organic, or actually no chemical reaction happens this cleanly. This is showing how it can happen if they just bump into each other in the exact right way. But this has got this pair of electrons. Let's say this pair of electrons is essentially given by this oxygen to this hydrogen proton. So we can draw it like this right there. So it nabs just the hydrogen. Then both of these electrons that are in this pair, that are in this bond, I should say, go back to the oxygen to form, essentially, you can think of this as a pair of electrons attached to the oxygen. And then that gives the oxygen license to allow these two electrons to form a bond with the phosphorus. Now the phosphorus is in the mood to form 6 bonds. It's already got 5, and this is a fairly uncommon situation for it. So that allows these 2 electrons, right, these 2 electrons to go to this oxygen, or just like that.
Heck.
And so as a result of
Yo,
this,
Ochem is actually so high.
what does it look like?
Literally just like...
Well, now you're going to have your adenosine diphosphate. Let me put it over here. And just to be clear,
Ochem is just like
gained, so it was this oxygen right over
the
here was
political drama of chemistry,
bond,
man.
2 electrons. Now it's getting both of the electrons.
Like,
So
it's
now
all
having
about
a charge
electrons
half
grouping
of
together
1/2
and deciding where they need to attack
But now it's going to get both of
while
them.
protecting their base.
The adenosine
Holy
diphosphate.
shit.
Now this phosphoryl
This is
group
why I
right
like
here, when you
Ochem.
draw it, it's going to look like this.
It's all about the logistics, man. Wait, what in the world is this game right now? Oh yeah. It's a certain I'm sorry if you haven't played Xenoblade 3 and you are hoping for completely no spoilers. I mean, obviously the fact that this song is in there is like, and the fact that you're listening to the song before you play the game. Sorry. Sorry, should have put a spoiler warning there. But hey, I'm not gonna say any more than that. It's a good song. Some good shit.
So double bond to that oxygen. We have this oxygen right over here. So that's there. We have that oxygen right over there. And of course the water molecule, or I guess now it's just going to be an OH group, is going to be, it has, let me see if I can make the colors interesting. So these 2 electrons have now formed a bond, and you have,
And again,
oxygen,
nearly
this hydrogen here,
made me
and
think of a different game.
this thing has 2 still.
Maybe think of like either
And of course,
Persona
you have
or
this guy right over here, who gained a proton.
what's the name?
So this
Catherine?
one,
Full body?
oxygen,
No.
hydrogen had 1 lone pair,
Eight?
but now it gave half of this lone pair to form a bond.
What is the name of the game that Atlas made?
Proton. Let me draw it like this so you can see it. So
Oh,
these
I don't know.
are now the 2
Whatever.
electrons in this bond with this hydrogen proton. So this right over here, this is a
Made me
positively
think of
charged,
that.
this is a positively charged molecule right over here. And you can imagine, maybe this thing breaks off, it can be viewed as a proton, or you can view this as just a positive charged molecule. But either way, this is the reaction that we just depicted. You have ATP being, hydrolysis takes place, you're left with ADP, you're left with a phosphate, a released phosphate molecule, and then you're left with a positive charge. You can either view this as kind of a proton, or that proton attaches and forms a hydronium ion right over there. And of course, in the process of doing all of this, as these electrons...
Oh, is this showing? This is just for the mechanism.
Sitting right over here and allowing this thing to break off, it releases...
That's funny.
It releases energy. It releases...
That's some funny stuff, man.
Energy, which is, in most biological
Very
systems,
nice.
the whole point of having the ATP molecules around.
I think what it is that I don't remember like specific terms that people would normally like use in order to describe this mechanism. Like the way that I would describe it is just like O attacks H, attacks O, and that attacks P, which leads to an attack of O, and then so P gets separated. Oh no. Oops. Like, I guess the end goal is for me to learn how to converse with fellow scientists so that we can be on the same page on stuff. But it's just like, bro, I'm just here to see the electrons fight, bro. And capture territory, man. What are you guys even talking about?
Okay, so the term carbohydrate refers to a chemical compound made up of carbon atoms that are fully hydrated. So carbo
What do you
for
mean?
carbon and hydrate for hydration or water. And
Carboxylic
because these biological
acid.
molecules are
What is
hydrates
that?
of carbon, you can find them fitting into the general formula C, so a number of carbon atoms, so N for just kind of a generic number of carbon atoms, and then a matching number
Benzyl
of
group?
water molecules, so H2Os,
Nani?
usually in the exact same number as your carbon. atoms. And because all of these carbons kind of have an associated water, you can think of this as being essentially a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. Now, when we have one of these carbohydrate molecules, we call it a monosaccharide. So monosaccharide. And monosaccharide essentially means one saccharide, and saccharide is just a synonym for carbohydrate. So saccharide is actually, it's derived from the Greek word for sugar. So you might hear a single carbohydrate referred to as a simple sugar. But in all these instances, we're talking about the same kind of molecule. And these molecules that we're calling carbohydrates, they do some
I don't know where to put that. Okay, what we can do is okay, how about we put this here, and then carbohydrates can be over here, leading towards, like, no. Oh, that's too, that's not. And glucose, but like, no. Just play it safe. Stick with metabolism here, connecting to cellular respiration. And then I'll include carbohydrates as a separate branch. Hydrates. Monosaccharide. Okay. Then we'll include the chemical formula. H2O.
pretty incredible and pretty hugely necessary things in our bodies and in most living things for that matter. And maybe one of the most familiar of these tasks is the fulfillment of our body's energy source. So carbohydrates fulfill our body's energy needs. And the main energy source for metabolism in our bodies is glucose. So glucose, and I bet you've heard of glucose before. If you might have heard it in the context of checking blood glucose levels for people with diabetes. But glucose is a monosaccharide made of 6 carbons. And you might also be familiar with kind of the structural rigidity of cell walls in plants. And that rigidity comes from the rigid carbon backbone of several carbohydrates linked together to form the
glucose is right there. Oh my god. Okay, hold up. So much for that. I literally... okay. Hold on. Yes, I know that glucose is a monosaccharide. But I was not expecting them to go that direction. I was thinking that they'd go a little bit more complicated here. And then I have to reroute. But no. I can go at it like this, which is the most simple way to do this, but why not? Let's do it. Sometimes simple is best. Oh my god, this is a mess. It's so... oh no, it's so... oh wait, it just fixed itself. Wow. I don't know how that happened, but I'm glad it did. There. Okay. We got him.
Saccharide. So polysaccharide, several carbohydrates linked together, and we call that cellulose. So cellulose is the polysaccharide, the carbohydrate that makes up the structural backbone of cell walls. And then, of course, one of the most beautiful carbohydrate roles in my mind is the use of ribose, which is a 5-carbon sugar that supports the transcribed products of our genes in RNA. And so you might have caught on that in all 3 of the carbohydrates that I just mentioned, we see the ending ose, O-S-E. And we see that in glucose, cellulose, and ribose. And that's because -ose is the suffix for sugars. And there are actually two prefixes that help us further break down of these compounds. And so the first prefix that we're going to consider is how many carbons are in the chain, so the number of carbons that are in the chain for this molecule. So for example, I'm going to draw glyceraldehyde, which is kind of generally considered to be the simplest carbohydrate, and it looks like this. And if this carbonyl group right up here was just another hydroxyl group, this would be glycerin, 3 carbons but with 3 hydroxyl groups. But instead it's an aldehyde, and so this molecule is for that reason named glyceraldehyde. And aldehyde is our functional group, and so if we're going to count how many carbons are in this molecule, we'll start with our functional group carbon, this carbonyl carbon up here, and we've got 1, 2, 3 carbons in glyceraldehyde. And so there are 3 carbons, and for that reason we would call this a triose. Tri for 3, and again, ose as our suffix for sugar. And if we added a 4th carbon, we would call it a tetrose. So 4 carbons in a carbohydrate chain is a tetrose. And then we add a 5th carbon, and that would be a pentose. So pentose, 4, 5. And if we added an additional carbon, we would have 6 carbons, and that would give us a hexose. Hex being the prefix for 6. And I mentioned before when I was talking about the energy source of our body that glucose is actually a 6 carbon carbohydrate. And so as an example of a hexose, I'll draw glucose here.
Wait, am I dumb?
This carbon chain, there's 6 carbons. We've got 1, 2, 3,
Wait.
4, 5, 6 carbons, and this is a hexose.
Glucose is
Now, in the case of glucose, the functional group is an aldehyde.
Oh, hold up. I think I'm dumb. Hold up. Is glucose Wait, what is a monosaccharide? It's a simple sugar, but like past that like short definition, what actually is an example of a monosaccharide? Oh no, it's lagging so hard. Any class of sugars that cannot be hydrolysized to make hydrolyzed to make a simpler sugar. Okay. So wait, what's a polysaccharide? Poly. A number of sugar molecules bounded together. So I think the difference here is that polysaccharides have like these like chains of like saccharides that loop around. What? I'm seeing a bunch of little like discs. These hexagons. So what does a monosaccharide look like? It's just a chain of carbon. But then a polysaccharide has a bunch of Okay, I think I sort of get it.
Just like our glyceraldehyde.
So these are all
so the functional group up here is an aldehyde. But what
monosaccharides.
You see, we can actually make it a ketone and still retain that 1 to 2 to 1 ratio, and it brings us kind of another pretty popular hexose called fructose. So this is another hexose. It still has that 1 to 2 to 1 carbon, hydrogen, oxygen ratio, but instead of having an aldehyde functional group, it has a ketone functional group right here. And so that brings up kind of the second naming prefix. We have to indicate whether we're working with an aldehyde or a ketone. So glucose would be more accurately referred to as an aldohexose. And that aldo is a reference to the fact that the functional group in this carbohydrate is an aldehyde. And fructose, then on the other hand, let me write fructose down. So fructose is a ketohexose.
Oh, here we go.
Keto is a reference to the fact that the functional group here is a ketone. And then if we want to just kind of exhaust this kind of second prefix idea, going back up to glyceraldehyde, which we said was a triose, because the functional group is an aldehyde, this would be an aldotriose. So aldotriose. So we name based.
Okay, we're doing naming schemes as well, which is always great to see. Okay, this is interesting. Maybe I can move this down and then tackle this. Put aldohexose, and then I'll just have myself figure it out later. It's genius. Just leave it for future Nick. He'll figure it out. He's figured out these things before. He'll figure it out again. Probably would have done this even if I wasn't this tired. Just tired. Oh.
Based on length of the carbon chain, the number of carbons that are in the chain, and the functional group that's in our carbohydrate. And the last kind of major component of naming is the stereochemistry of the highest numbered chiral center. So again, we start with a carbonyl carbon, and if we use a Fischer projection like we did with glucose, then we go to the highest chiral center, which would be this last one, and we decide the stereochemistry of that chiral carbon. And just kind of as a shortcut with Fischer projections, if the kind of highest substituent, in this case the hydroxyl group, is on the right hand side, then it's an R stereochemistry, and if it's on the left side, it would be an L stereochemistry. So let me try to make that a little bit easier. And I'll just kind of redraw glyceraldehyde, a nice small molecule, as a Fischer projection. So we've got our aldehyde, and then we've got kind of our next two carbons,
Oh, no.
3 carbons, and we have one chiral center in this carbohydrate, this one right in the middle. And our OH group, you can see, is on the right hand side.
Well,
this is an R configuration. And
this
for
is
carbohydrates, it was,
appropriate
they
considering the game that we're going to be playing in 17 minutes.
But he decided to
Chiral,
use an R,
which
and
I
the last
misremembered
right hand
because
kind of is extra. We decided to
enantiomers are the ones that
and then
are,
if we kind of drew this in a
you
mirror
know,
image, and we drew the enantiomer
that's
diagram,
where the
and we had our
imagery comes from.
aldehyde
See,
carbon up here,
there it is.
and our last two carbons, and their
So
hydroxyl
chiral
groups.
is relevant,
The
but
chiral carbon, the
not
hydrogen
exactly the
chiral
same
carbon,
term.
its primary
That's not
constituent on
what
the
that
left side
means.
at the bottom of this Fischer projection, and so this would be assigned an L. which
Chiral
is
just
what-
means that
And so as an example, I guess going back to the glucose, this would be a D aldohexose.
it has
because the
like a
hydroxyl group is on the right-hand
unique.
side of this molecule. And
What's
again,
that
with the
chiral
same thing, the
center?
last chiral carbon.
And what's chiral on its
the
own?
OH group on the right hand side, so this is a D
What?
ketohexose. Now, before I move on, I want to allow you to review Fischer diagrams because I know
I'm
I just kind of
confused.
breezed through it, and really they're important, especially with carbohydrates, because stereochemistry becomes quite important in the.
Oh dear, stereochemistry is always a way to make things a lot more interesting. Sometimes more than they need to be. Alright, I'm just gonna put D versus L configuration. And I think that'll basically covers it. Kind of.
Biological implication of carbohydrates. So I included a great video by Jay with Khan Academy on Fischer projections, and it's actually the video I used to learn about them. So I'd encourage you to kind of pause here for a second and give that video a watch on Fischer projections and get real comfortable with absolute configuration, and then we'll move forward with the discussion of carbohydrates.
I mean, I feel like Fischer projections are pretty like straightforward. You just kind of like turn the thing and look at it down the center and just split off all the branches. Of course, I'm saying that as someone that has a lot, whose life revolves around a lot more visual imagery, right? So that's why I make these maps. That's why I need to draw stuff as opposed to like write things down, you know. Just more comfortable that way. So, you know, if you can't imagine,
Okay, so we spend a lot of time on the front end of carbohydrates talking about their stereochemistry.
like, if you can't do object rotations within your visual imagery, you can't do object rotation through visual imagery, then it's just not, you know, it's just not a trait that you have. It's all good. Like, and I think you can train it. It seems like it's likely that you can train it unless you have aphantasia, in which case, I don't know. Very curious if you can just help train people with aphantasia into being able to do, you know, have visual imagery, or if it's just like forever incompatible.
Especially that last chiral center. And again,
But this isn't
plays a role in
cognitive
the biological
science,
function of
so
those molecules.
let's
For
go
example,
back to
humans
the
are
bio.
enzymatically programmed to break down and digest the D sugars. So for that reason, I want to spend.
I was very surprised at how much I like Coxide, though.
When learning about
Very fun.
carbohydrate stereochemistry and nomenclature. So first, I need to clarify that D and L refer to stereochemistry, but they don't speak to the overall optical activity of the molecule. So as an example, let's take a look at D3-ose. And D3-ose has an aldehyde functional group, and it has 4 carbons, so it's a aldotetrose. But you can see that the last chiral center down here has its functional group, this hydroxyl group on the right side, so it's a D carbohydrate. D threos. But it turns out that there are actually 2 chiral centers here, and whenever we have, you know, n chiral centers, whatever number of chiral centers we have, then we have 2 to the n possible stereoisomers. And in this case, there are 2 chiral centers, so we have 4 possible stereoisomers. And it turns out that this particular stereoisomer actually has kind of an overall optical activity such that it rotates plane-like counterclockwise, as opposed to clockwise like you would see with most R configurations. So even though this is D, it's actually a negative. It gets a kind of a negative sign for its optical activity. So this is D minus threos. And again, it's D because this lowest chiral center here has an R stereochemistry, so it's a D carbohydrate. Now the second big thing that I want to clarify is that it's important to note that the D and L configurations of a particular carbohydrate are enantiomers, which mean they differ at every chiral carbon, not just the last one. So we can take a look at this in the case of glucose. So glucose, again, is an aldehyde carbohydrate, so it's an aldose, and it's got 6 carbons, so it's an aldohexose, and this is the D configuration. The L configuration is going to look like this. So you can see, again, it has 6 carbons, so nothing's changing there. But as we reflect it across this mirror, every single chiral carbon is going to be the mirror image. So this is L-glucose. And again, the big thing that I want to clarify here is that it's not just this last chiral center down here. It's not just this last chiral carbon that is flipped for the D and L. The D and L glucose are true enantiomers. So enantiomers, which means that they're complete mirror images. They differ at every single chiral carbon. Now that being said, if the D aldohexoses, these glucose, if the D and L aldohexoses are enantiomers, that means that all of the D aldohexoses have to be diastereomers of each other because they're not superimposable and they're not mirror images. And I know that's confusing, but I've drawn out here all of the D aldohexoses, and we'll just kind of take a look at what I'm talking about. So we have the D aldohexoses here, and there's 8 of them that I've drawn. So in the case of glucose up above, I'm going to flip back up to it for a second. You see that D glucose and L glucose are enantiomers. They differ at every single carbon. Now, all of these are stereoisomers, but they differ at maybe just one. They don't differ at every single carbon from glucose. So here's glucose down here, and you can see D-allose. Well, it's just different at this one chiral carbon right here. Or you can see D-galactose up here. The only difference is the C4 chiral carbon from glucose. And so what you see is that these aren't mirror images and they're not superimposable. So all of
Oh my god, come on.
aldohexoses are
You're kidding me, right? Freaking, okay. There we go.
diastereomers. It's the same thing for all of the L aldohexoses. They're all diastereomers of each other. And you can carry that thought through the ketopentoses. All the D ketopentoses would be diastereomers of each other. And they would have a partner in the L ketopentoses that would be their enantiomer. So again, this is a terribly confusing idea, but I really think the best way would be if you could just pause the video for a second and take a look at all 8 of these and notice where they're different, and notice that they're not different at every single carbon, so they can't be enantiomers. Okay, I've said enantiomers and diastereomers too many times already, I'm sure. Now I mentioned just a minute ago that glucose and galactose are different only at the C4 carbon. Remember, we've got 1, 2, 3, 4, 5, 6, and similarly with glucose, 1, 2, 3, 4, 5, 6. So the only carbon that these differ at is the C4. And because they just differ at one carbon, we have a special word for these, and they're called epimers. So epimers are diastereomers that differ at one chiral center, and that's just kind of a vocab word that's probably going to come up several more times as you look at carbohydrate chemistry. Now you can take
Holy shit, I've never heard of that one. Okay. So it's epimers with lactose, right? Okay.
take kind of this thought of D versus L carbohydrates to the next level with critical thinking. If you consider all of the stereoisomers for an aldohexose, again, we're talking about aldohexoses right now, and how many chiral centers do these aldohexoses have? Well, you can count. There are 1, so 1, 2, 3, 4. Now, that's not numbering that. I'm just counting the chiral centers because this carbon up here, the carbonyl carbon, is double bonded to an oxygen, so it's not a chiral center. And down here, this carbon is bound to 2 different hydrogens, so it's not a chiral center. So all of these aldohexoses have 4 chiral centers, and that means they have 2 to the 4 or 16 stereoisomers. Now, half of those are going to have this OH at the bottom on the right side, and the other half would be left. So half of 16 is 8, and that's how we get to this idea that there are 8 D aldohexoses. And that's just kind of a thought that you can use, and you can translate that into pentoses. Pentoses are going to have 3 chiral centers, so there's going to be ultimately 8, and there would be 4D and 4L. So the last thing I want to do is cover just the common names for maybe the 5 most commonly seen monosaccharides, and I've similarly kind of pre-drawn their structures in, and I'll give you their names and kind of the mnemonics that I was taught to remember them by. So the first one that we have right here, number 1, is ribose. So ribose. And the way I remember this is a pentose. It's an aldopentose. It's got an aldehyde and 5 carbons. So it's an aldopentose, and all of the substituents, all of these hydroxyl groups are on the right side. So I remember that ribose is all right. Now the next one we have, hopefully you can see here because we've drawn it a couple of...
Oh no. Okay. 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 Oh no. Fuck. And this song is kicking in too? Seriously? Damn, okay. Going all in on this. Going all out, baby. Okay, monosaccharide. So ribose. I'm hoping and praying that we have like a section just dedicated to OChem terminology, because I'm kind of getting a little lost.
Different
'Cause I
times is
forgot all
glucose.
my
And I should
OChem,
mention
so
that this is D-glucose again, and I should mention that this is D-ribose here. But the way that I remember glucose is actually a little bit racy, so keep in mind that that I do not support flipping people off with your middle finger. But if you look at this,
Yo.
again, it sure does
Yo.
resemble somebody flipping off people. So you can say whatever insult you want to glucose. I'll just kind of like write some kind of expletive marks here to glucose, and you can remember that glucose. We'll just pretend that we're really frustrated with it, and we're kind of cursing it out. And again, I don't condone you using your middle finger, but thank goodness that organic chemistry can redeem even the most heinous of societal insults. And so we can remember that
Hey, yo.
glucose looks like if we're holding, if kind of this is our pointer finger, and you can curl your finger up and kind of stick your middle finger out with the fingernail down towards the page. I'm sure you can make the connection of how your fingers resemble glucose. And so that's kind of my mnemonic for that. This next one is mannose, and again, it's D-mannose. And if you position your fingers in the same way that you were with glucose, and now you just extend your pointer finger as well.
You know,
So
this
now we
kind of
have
works.
2 fingers extended and then 2 kind of curled up. We can see that it's like a man holding his gun. So we're the man, and we're holding our gun, and that's D-mannose. So man with a gun. And again, this is an aldohexose, just like glucose. And to keep using that vocabulary, these are
Like
diastereomers from each
the
other. And then this next one on the list is galactose. And it's kind of lame, but the way I remember that this is that
the
D-galactose
aldehyde
the C4
is
epimer
like
of
your
glucose.
thumb,
So
right?
galactose, I've got the C4 epimer of glucose. So
Oh
down
my
here, this is the
God.
only carbon, the only chiral center where it differs from glucose. So I
Oh
remember
wait,
it's the
whoops.
C4 epimer. And then last but not least,
Mannose,
we have fructose. And
lactose,
this kind of made an
fructose.
appearance.
Okay. Mannose. Can't freaking see. Okay. Glucose has a gun. No, it's Manos. It's Manos that has a gun. Glucose flips you off, though. Fuck you. Glucose. Fuck off. God damn, Glucose. Didn't realize you were such a gamer. Holy shit. Fructose? No. Fructose. There we go. Like high fructose corn syrup. Everyone loves that. I do not, because it kind of gets really annoying. That sticky sensation after drinking soda. Disgusting. Alright.
In an earlier video. So we've got D-fructose, and the way I remember D-fructose is that it's the ketose of glucose. So the ketose of glucose, and you can see that it very much resembles glucose, except that instead of an aldehyde, it has a ketone functional group. And these are maybe the most common monosaccharides that you'll see in an organic chemistry and kind of biochemistry context.
Biochem powers unite. Okay, how long is this video?
Alrighty, so we've been speaking so far about carbohydrates as chains of carbon, and these are chains of carbon atoms that feature an aldehyde or ketone
whatever.
functional
We'll
group, and
just
that falls
let
into
it
this
ride.
general kind of 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. And of course, I'll keep using glucose as an example. Now, I've also used the term polyhydroxylated to refer to the numerous hydroxyl groups that are in these carbohydrates. And really, I bring all of this verbiage back out to hopefully spark your ability to see that carbohydrates have all the makings of an internal or intramolecular, I guess, reaction between the carbonyl carbon right here and one of the hydroxyl groups. Because essentially what we have is carbonyl and alcohol chemical reactions just kind of waiting to happen. And what happens when an alcohol nucleophile attacks an aldehyde or a ketone? Well, you know, if there's an excess of alcohol, we end up with a product that is either an acetal or a ketal. But what happens if there's only one nucleophilic attack?
Oh
We
my
just have one
god, that
alcohol, and
molecule's
that's
drunk.
in the ring closing intramolecular reaction we have going
Oh
on. Now,
shit.
hemiacetal or hemiketal. And really, that terminology is just kind of a review of acetal and ketal chemical reactions that would fall under, I guess, if you're looking at an organic
Oh,
chemistry
yes, a
book,
review. Yeah,
aldehyde
yeah.
or ketone reactions, probably the
Mm-hmm.
carbonyl section. So let's show how this process is happening in the
Yeah.
context of our glucose
Yeah,
overload.
carbonyls.
First, I'm
I
gonna highlight
remember how
particular
those work.
hydroxyl oxygen that's going to act as the nucleophile. So we'll make
Oh,
that
no.
pink. And after being
Oh, no,
deprotonated,
they
so
think I
after
know how
losing
carbonyls
this
work.
proton, this oxygen is going to have an
Okay,
extra
wait a
set
minute.
of electrons right here, and those electrons are going to target that carbonyl carbon.
Yes.
So I'll draw the carbonyl carbon in green. And remember,
Attack
the
that
carbonyl
carbon.
has a partial positive charge on it. It has a partial
Destroy
positive charge.
them.
The electron density in this double bond is being hogged by this oxygen. So the oxygen has a partially negative
Yeet
charge,
that
and the carbonyl carbon is
oxygen.
partially positive, and that makes it
Or
a perfect
yeet
target for the nucleophile that's
the
been created in
electrons
the
back
deprotonation
to the
process
oxygen.
of this oxygen. And so after the oxygen's
Do
electrons
it.
attack this carbonyl carbon, what's going to happen is the electrons from this double bond are going to kind of kick back up to the oxygen up here, and
Yes.
eventually they're going to attract another proton and will form
And then
another
take
hydroxyl
back
group
the
or
proton.
a side electrons from that
Yes.
bond. Now you might be asking, and it's a perfectly valid question, why it's this particular
Beautiful.
oxygen, the one that I've highlighted, that's acting as the nucleophile. And you're going to see, as soon as I get the product drawn, that we formed a 6-member ring. So it really has a lot to do with
Oh
products
my
in the
god.
ring. If you remember, the basis for the formation of the ring in the first place was the increased stability over the straight carbon chain. So it makes sense that we're going to form the most
Is this
stable
how we're
ring
gonna
here.
make
Now, when we end up with a 6-membered carbohydrate
polysaccharides?
ring, such as the case with glucose here, we call it product
boy.
pyranose, the os again as a suffix for sugar, and then the pure part to indicate that this ring is sugar with 6 carbons. And then if the carbohydrate ring is a 5-carbon ring, we call it a furanose, which is a bit easier for me to remember because furanose and 5 both start with the letter F. So that's kind of the memory jogger for me. And maybe a good example for that would be ribose with its 5-carbon chain. But I'll kind of stop there because almost every ring forming carbohydrate that I can think of with biological implications at least forms either a 5 or a 6-membered ring. So pyranoses and furanoses. So just by convention, you can see that I've placed the O in this corner up here, and that places the formerly carbonyl carbon down here right below it. And it's actually no longer the carbonyl carbon, but it's still significant because it's the only carbon here that is bonded to 2 oxygen atoms, the highlighted oxygen, and it's bonded to another hydroxyl group as well. So I'll keep it distinguished in color, and we also distinguish its name now as the anomeric carbon. So that's the anomeric carbon, and then we can go ahead and fill in the rest of the substituents in the diagram. So a hydroxyl group, and another, and we call this diagram a Haworth diagram. So Haworth diagram, and the Haworth diagram doesn't show us the actual configuration of the ring because in reality, 6-membered rings are going to show up in a more stable chair shape, but it is beneficial in telling us which substituents are above or below the ring. So to keep this convention straight in my mind, I remember the phrase downright uplifting. So downright uplifting, kind of a play on I guess the phrase that's downright uplifting, but downright uplifting because as I fill in the substituents, those on the right side of the Fisher diagram will point down, and those on the left side of the Fisher diagram are going to point up. So we can actually see that one's up, and we'll make sure that this number's off right. This one's up as well, and maybe we'll name this, or maybe we'll start numbering with 1, 2, 3, 4, 5, 6, and we can do that over here. This would be 1, 2, 3, 4, 5, 6. So our 3 carbon in the Haworth diagram is pointed up, and our 3 carbon on the Fisher diagram has its substituent on the left.
Okay,
So
whoa,
downright
I need to fix my this diagram's going places right now. This is not good. Move my ring stuff to a different section. And put that below the electron transport chain. Just such a messy way to do this, but I'll have to make it work. What the heck did I just do? Okay. Oh my god, I'm ruining my diagram. This is terrible. This is actually disgusting. Oh no. Okay, there is my Death Stranding reminder. As if I need a reminder to remind myself to play a video game. Okay. Ring anomeric polysaccharide.
carbon group, which kind of forms this tail down here. I remember that if it's a D sugar, that group is going to point up in the Haworth, or in the Haworth, projection. So this is a D sugar, and you can see in the Haworth projection that this last carbon points up as well. And really, this is going to be the case for a lot of sugar chemistry that you deal with, because again, we're enzymatically programmed to digest D sugars, so we often end up with this last group pointing up. Now, the last thing I want to show you is the chair conformation. So the chair conformation, and that's because this is a kind of diagram that's going to give us a sense for the actual configuration.
Yeah, baby, the chair conformation is the best conformation.
As far as the substituents being above.
Hell yes.
So let me just kind of keep filling in the substituents.
And look at that thing, it's beautiful.
I'll number them off again, just so you can kind of see that there's some consistency here. So we've got 1, 2, 3, 4, 5, 6, and again, this 3 carbon right here is the only one with the hydroxyl
wait, no.
pointing up. And I guess I better change the color of our 1 carbon to keep that consistent as well. Now, I didn't indicate the position of the
Pyranose and
hydroxyl group yet, because I think it makes more sense to show it in this diagram. Remember,
why is it not Pyranose and
way back over here,
Mithranose, dude? What's up with that?
Carbon, and the other with the S configuration. So that last hydroxyl group can actually be in 2 different positions. On one hand, the hydroxyl group would be cis to the last carbon in the equatorial
He's terrible.
position. So it'd be cis to this last
0
carbon
out of 10.
over here, and it's in the equatorial position. And we call this the beta anomer. Then on the other hand, I guess, it could be trans to that last carbon group, which would place it in the axial position down here. So I guess it could be down here,
Beta.
in the axial position, and we call it the alpha anomer when the hydroxyl group is in the axial. And I kind of remember that a little bit easier, alpha for the axial position of that substituent. And I guess I've also heard that fishies are down in the sea and birds are up in the air. So if that helps you keep them straight, you might be able to use that also. Now, you've got to remember that what caused this ring to close in the first place was some amount of acid or base. And the amount of acid and base in water is actually kind of capable of doing that, because that's what facilitated this ring closing process in the first place. And in water, the ring can actually open and close spontaneously. And when it opens up, the C1 and C2 bond right here can actually rotate. And when it closes again, you can form either the alpha or the beta product. So this thing is constantly opening and closing to form the 2 different products. And we call that process where it opens and rotates and closes again mutarotation. So this thing is mutarotating in the water at all times. So mutarotation. And the outcome is that we end up with both configurations, the beta and alpha, in the equilibrium concentration. So for glucose, that's going to be about 36% alpha and about 64% beta. And the reason that the alpha configuration is less favored in equilibrium for glucose is because the transpositioning of the hydroxyl group creates some steric hindrance. But this is pretty individualized for different sugars. So I guess the most general rule, I suppose, that you could apply to all cyclic sugars would be to say that the beta anomer, that the beta anomer is the one with the anomeric oxygen in the cis position with respect to the last carbon.
What? Okay, hold on. Okay, so that's on the anomeric carbon. So I guess I can just go ahead and put beta and alpha. So what? Hold up. This man just went at it.
Carbon over here, and it's in the equatorial position, and we call this the beta anomer. Then on the other hand, I guess, it could be trans to that last carbon group, which would place it in the axial position down here. So I guess it could be down here.
What? What is it? What is it? Hydroxyl. Yeah, hydroxyl. Okay, that is not the... What the hell? Come on, man. Work with me here. Got the beta, and then we have the alpha. The alpha's underneath the beta. In the chair formation. Of course. Fantastic. Chair. And then you're telling me that they switch places all the time? Oh my god. That's crazy.
Would be cis to the last carbon in the equatorial position. So it'd be cis to this last carbon over
Sir,
here.
I know not what equatorial means.
The beta anomer. Then on the other hand, I guess, it could be trans
Okay,
to that last carbon,
I do know
which would
what
place it in
that
the
means.
axial position down here. So I guess it could be down here. In the
Okay,
axial position,
cool.
we call it the alpha anomer when the hydroxyl group is in the axial. And I kind of remember that a little bit easier. Alpha for the axial position of that substituent. And I guess I've also heard that fishies are down in the sea and birds are up in the air. So if that helps you keep them straight, you might be able to use that also. Now, you've got to remember that what caused this ring to close in the first place was some amount of acid or base. And the amount of acid and base in water is actually kind of capable of doing that, because that's what facilitated this ring closing process in the first place. And in water, the ring can actually open and close spontaneously. And when it opens up, the C1 and C2 bond right here can actually rotate. And when it closes again, you can form either the alpha or the beta product. So this thing is constantly opening and closing to form the 2 different products. And we call that process where it opens and rotates and closes again mutarotation. So this thing is mutarotating in the water at all times. So mutarotation. And the outcome is that we end up with both configurations, the beta and the alpha, in the equilibrium concentration. So for glucose, that's going to be about 36% alpha and about 64% beta. And the reason that the alpha configuration is less favored in equilibrium for glucose is because the transpositioning of the hydroxyl group creates some steric hindrance. But this is pretty individualized for different sugars. So I guess the most general rule, I suppose, that you could apply to all cyclic sugars would be to say that the beta anomer, anomer.
Oh, steric hindrance. Yeah, I can see that, actually.
Anomeric oxygen in the cis position with respect to the last carbon.
Okay, interesting. But not as interesting as trying to set up Death Stranding for the last time, because I think we're gonna switch to Signalis for the next one. Alright, that is the plan. Load the game. Norman is returning. Where is he? There he is. Now. Norman returns now. It's gonna do the thing again. Being in the top half of my thing. Top half of my screen. It's very frustrating. I don't like it when you do that. Makes me very sad. Especially because that's what happened last time and we spent an entire hour with it. Just like that. Because I failed to switch the scenes in OBS. I mean, really, I just need like a streaming deck. But it's a little hard to do that. Oh, this was published by 505. Cool, cool, cool. Time for it to load. All right. Any day now. Any day. Ooh, 98%. Yeah, it's great. It's wonderful. To be fair, it was 10 minutes earlier at the start of the stream. And now it's at like 3. It was about 3 minutes. It's beginning to believe. Right. Hello, Sam. I don't even remember what we're supposed to do. Oh. Fantastic. Should we use the toilet standing or sitting? I don't know. Use it standing. Feel so immersed. I literally just went over there earlier. In real life. And now, I'm going there. Oh, great.
As well as those of dooms and repatriants. When you relieve yourself, a certain amount of chiral matter is flushed from your system. Chiral matter can adversely affect hormone secretion and nervous function.
Okay.
There's a correlation between exposure and the development of phobias, as well as, in extreme cases, violent and suicidal impulses. Chiralium contamination has even been postulated as a trigger for the onset of dooms. Allow me to collect a sample of your excretions for my studies.
Ah,
They provide a
sure.
non-invasive means to monitor your exposure to chiralium and your overall health.
Wonderful. You go do that. That sounds great. What's the purpose of the sink? Brush my teeth or something? What are we doing here? Perform action. Checking himself out in the mirror, I guess. I do that all the time. All right. But what I don't do is drink Monster Energy. And so, I will engage in the fantasy of drinking Monster Energy drink. Of course. Fantastic. Oh, okay, so it just increases my stamina? And there's no limit to it? This seems somewhat concerning. I can appreciate that the nutritional facts they look accurate. They just scanned a real-life Monster Energy drink from the US into the game. It's just fantastic. And we gotta drink one more. Adds in my full price game. Yeah. Yeah, well, Kojima needed to fund it somehow. And he's recommending that we down 3 of them at once to increase our stamina by 25%. Fantastic. Look at that. I'm basically ready to take on the world. Oh, Examine weapon rack.
Hey there. Believe we've met? I oversee delivery operations as well as maintenance and repairs.
Oh no, I'm bleeding into the background again.
I'm Mama, in case I didn't mention it before. Those there are strands. Not just used for packing, by the way, but for identification too. Take a closer look and you'll see. See the red there? That's your blood.
Well, shit. So what, I just look at the different suits? Okay. Doesn't seem like I get to choose which suits I take out on missions. Maybe they'll just... Oh, wait, no, there's an equipment rack. Let's check on the baby first. Yo, baby. At least it's not trying to kill me, like in other games. Or is it trying to kill me? Can't tell with this lore. This lore, dude. I mean, of all the things to survive a post-apocalypse, it'll probably be Monster. Suda, baby. Fantastic. After playing Peekaboo, just need to get our equipment together and we'll be headed out. Fantastic.
This case will remove any chiral matter still clinging to your suit. Speaking of suits, blue is for delivery personnel, red for medical, orange for corpse disposal, and black for security.
Ah, okay, okay. That actually makes sense. Well, in that case, I don't have any reason to change the color scheme, right? Because I should be taking out blue for my next mission. assuming that it's just like a normal transport mission. Oh, no. Wait. Okay, these are all tips. All right. I was worried that there was like a lot of lore critical files and that there were 98 of them or 49 of them. 98. Okay. No orders, no nothing. I have to be careful about the music or else I'll get copyrighted. Case. Oh, just as I was about to leave, Die Hardman shows up.
Well, have you had a chance to think it over?
Nope.
Chock full of chiralium, but safe for you Dooms guys to wear. That's all you'll need to go forth and reconnect the world. To make us whole again.
I'm a porter. I don't care about connecting anything. Or making knots. But I'll do what I have to help Amelie out.
Now my face isn't blocking the stream to avoid copyright. Visual copyright is fine. The problem is auditory copyright.
Listen up, Sam.
I'm really just on the screen because this is how I feel like streaming.
The infrastructure's there, but the Cairo network is offline. Right now, it's only capable of transmitting voice communications. Sometimes wired, sometimes wireless, and a small amount of data. So, unless the necessary data stored on site, our chirograms won't show up. Emily and the other Bridges members you've seen around the place are grams generated with local data. In case you didn't know. Anyway, all you've got to do is find the knot, connect your Cupid, and bring Cairo communications online. Once you connect it to the terminal, you'll be able to initiate zero-time massive data transmission with the UCA network.
Zero time.
Just like that, you'll reconnect us not only to each other, but to our past.
It's supposed to smell like anything?
Compiled and restored, like bringing a dinosaur back to life from a fossil.
Oh my god. I'm only half paying attention. I can't tell if it's because I'm tired or because this is somewhat obtuse.
Since the death Stranding will be ours again. And that, my friend, is how we'll
But I have a 7-hour video waiting for me at the end of this playthrough to explain the whole damn thing for me. Oh dear.
You'll also be able to use Cairo printers.
Oh, I see.
Able to send all kinds of things through the wire.
Except anything original. Or that's got a soul. Nothing real. Just copies.
True. Which is why we'll still need porters like you. Before and after we're made whole, we'll need men in the middle.
No rest for the wicked, huh?
Yeah, well, idle hands and all that. We'll give you the details at the dispatch terminal. Make sure you check it upstairs. Drop's waiting up there, too.
These shoes look like they've done some serious miles. Shall I dispose of them?
Wait. Hold on. There's something I want you to look into. My blood seemed to set those things off. Happened more than once.
Set them off? How do you mean?
I don't know. You took my blood, didn't you? You tell me.
All right. I'll see what I can do.
Well, he could at least define that a little bit more clearly. Last time we were trying to get through BTs is just a bit of a struggle. I didn't see any setting off of BTs there. I just saw a whale trying to chase me down through a black void. I mean, to be honest, storytelling's solid. It's fine. It's just, you know, not all information is gonna be clear, but at least there's some information to work off of. Like this. I can't help but think of... Is that like Amelie? Wait, no, he's talking to BB and he's referring to... Oh, wait, what? Oh, whoops. I think it's because my audio is like desynced. Which only enhances the experience, obviously. Okay. I was like, what? Why is he calling the lady BB? I mean, like, is that Amelie in the... on that table? Or in that bed? And the BB is like Amelie's child? Because we saw that little photograph. I mean, he keeps looking at it. Assuming that that is Amelie, whose face got blotted out. Take on a new order. Sure can do.
Sam, this is Di Hartman. Your current objective is to extend the chiral network from here to Port Knot City. But don't think you can make a beeline straight for it. Signal won't carry that far. To cover the distance, we need to utilize knots. Think of it like tying ropes together to make a longer rope. The first of these knots is a Bridges Way station. Go ahead and take a look at the order. Deliver whatever they need, and connect the Cupid.
Right. More drug delivery. Fantastic. Sounds like an average Saturday afternoon.
Contain information critical to your success. Make sure to review them before getting underway.
Okay. Smart drugs, oxytocin. This is the reason why I was interested in this game. Effective at easing symptoms known to arise from little interpersonal contact, such as stress and a lack of affection for others. Man. Hell yeah. This is some cool ass shit. I appreciate it. Alright. Accept your order to bring the oxytocin. Or we can just have everyone be a little bit more comfortable with hugs, I guess. But no. We're gonna just... Oh god.
Sam, we've supplied you with some rope and a ladder for this run.
Or I could just bring all of the hormones with all of the ladders.
Like a halfway decent bridge if you need to cross a river.
Okay.
Right now, I figure you're thinking about how you want to handle this order.
So I'm assuming I can just reorganize. Wait, what? Oh, I see. Okay. I don't really have a choice. I'm just gonna dump it all on my back. It's absolute mad lad. It's absolute unit.
Order assigned.
feel like these containers are probably way too big than they need to be. Way larger than they need to be, I mean. What is that supposed to mean? Excuse me? Ah, yes, urinate, my favorite tool. Okay. Wait, how many how many ladders do I have on me? Seems like somewhat excessive. Oh, I see. Okay, this is just to force me to equip that. Okay. I see. Wait, where am I going? What am I doing? What's the plan? What is happening? Okay, that's the incinerator, so I'm trying to get over there. Then where is there? Didn't I... Okay, yes, this is where I need to go. Create a sign. Why would I need to create a sign? Well, got a journey ahead of us, and 30 minutes to spare. Let's see if we can make it. Thank you, disembodied voice. Yes, I do. Someone's already used... I've already used it. Oh, wait. I don't think he's talking about this sign, actually. I think he's talking about some other thing. This thing, yeah. Yay. Can I use this? There we go. Okay. So I'm guessing I probably want to just walk around the coast and then loop around. Seems like a pretty good plan. Let's go ahead and do that. Oh, wait. Guess I gotta like everything. Even though I'm pretty sure that's an NPC that I just, liked just now, but... We'll start messing around with, real people stuff. As soon as we get over to that next, the next goal.
Even the best porters have been known to lose their cargo. But you're better than the best. You've got what it takes to finish what they started. If you come across any abandoned shipments,
Yeah. America. Of course. Because Norman Reedus.
All our packages are tagged with Bridges IDs for easy tracking. And your Oja deck is equipped with a scanner which might detect said IDs. Which is a roundabout way of saying, but that thing on your shoulder can help you locate mislaid shipments.
That was interesting. Wait, what? Okay, wait. Then I see. To the baby. Because I just ran into a rock without stopping. Oh, I see. You just shake the baby, I guess. Yay! I like how he just was running into that rock and just didn't do anything. Alright. That's cute. My controller's like lighting up. Okay. But is the cargo safe, though? I do not care about the child. I want to know if the cargo is okay. Because that will cost me all of the marbles. Okay. This is a little bit of a... Okay. Nerve-wracking moment, but it worked out. So I see that he has these little, like, shoulder pads. I thought you can put cargo on there. I don't really see an option for that. I can carry it with my left arm. And then I guess I just have to... Yeah, just has to go on my back. Which, to be fair, is probably the most efficient way to do this for now. But I'd appreciate more flexibility. That'd be pretty cool. Or maybe I just haven't found the right item type yet. What is that? Off in the distance. Oh god, there's a lot more cargo. Thanks, Die Hardman. Yeah, of course. Of course I'm gonna get all of this cargo. Oh, you know that they just planted this here. So that I get tempted to pick up everything. Which I do feel very tempted to do. You can't do this to me. I'm weak. I can't handle the temptation like this. Come on, man. I have to pick up everything. It just has to be done. Run, Sam. Run. There's so much stuff we need to pick up. Okay. What is that, like a trash bin? Time to toss the baby into it? What are you talking about? What is that? I ain't gonna listen to Guillermo del Toro and his advice on babies again. Okay. Dropped... No. And return... What? Okay, what is this? So what, I can just drop off cargo here and, okay, make a delivery.
Good work, Sam. I see you brought in some lost cargo. Feel free to submit it here for processing.
Huh. The ringtone is like a lullaby. Interesting.
Don't think you can carry it to its destination, you can always put it in storage. It'll be safe and secure until another porter comes along to finish the job. Even if it has to pass through a dozen more hands before it arrives, you can rest easy knowing you did your part.
Okay. I can deliver lost cargo here. No, this is a storage room, so I would... I see, okay. I mean... I could probably just... I'm probably fine. I think I can bring it over there. I don't think I'm overvaluating over... Oh god. I am very tired, though. I've overestimated myself, maybe. I don't think that's the case, though. I see how it is. They put all of the lost cargo on the mountain to incentivize me to travel up it. Oh my god. Okay. Well, this ain't gonna be about the easy route. Makes me wonder what the difficulty setting actually does, though. Wait, speaking of which, I don't really understand what urinate does. Whoops, that is the wrong... What? Why did I pull up my glasses? Sorry, what does this do? I mean, sure, I'll just... What? Wait, how do I... How do I piss? How does one... Oh, so that's how I crowd. Should have known. Should have known. Wait, there's an entire... There's a very specific input for this. I see. Okay. Wonderful. Got a steady stream going. It's one... It's one of those things where the character turns around. I see. I see how it is. Well, to be fair, if I was able to see the front, I would instantly be banned off of YouTube. So, thank you, Norman, for saving our eyes. Why is it flashing red like it's a bad thing that I just let out the entire tank? What's up with that? I thought that was a good thing. Come on, man. I don't want my boy to walk around with a full bladder all day. God damn. Oh, shit. What is it, Die Harbin? I actually did forget about the ladder you issued me. Ah, yes, a seasoned pro. Yes. Not like this is my first time playing this game or anything. You can tell I'm a seasoned pro by the sheer amount of shit that is all over my back. What is happening? I thought there was a... Oh man, there's more cargo down there? Are you serious? Okay, no. You know what? No. I don't want to do any more of this. Oh, they're telling me to use my ladder here. I mean, I guess I'll just... I was already planning on doing that, but okay. This position, and then... Oh wow, that goes really far up. Very nice. Fine, I'll like it. Not that I needed it, though. Let's go! There is so much cargo here. What is going on? Oh my god. Cargo management screen. Cargo, what? Accessing the cargo management screen from the cufflinks will allow you to optimize by pressing it. Okay, wait. So, is this the cufflinks? Cargo, yes. Okay. I can have them auto-arrange it for me. Okay. Okay. Yeah, there we go. Now we're optimized. Not familiar enough with the physics of the game. Wait a minute. I didn't save, did I? Silly me. Right. How did that happen? What did I press wrong? Why is it not... Why do I have to scroll all the way down to confirm? That's weird. Oh, I have to press and hold to confirm. Okay, that makes more sense. Right. That piece of cargo just... It's so much wider than the other... Let's try this again. Okay, then I can confirm... There we go. There we go. Now everything makes sense. Oh, shit. Where am I even going? What happened in between then and now? What was I even planning on doing? What is happening? Norman, speak to me. Speak to me, Norman. I need to know the plan. What is the game plan, sir? Oh my god. What is the game plan? You know, I can't help but feel this is somewhat impractical to climb with this amount of stuff placed squarely on my back. It seems a little bit, silly. Seems like this wouldn't work in real life. Alright, so I think we're leaving the area where I can have support. Yep, leaving the chiral network, so it's on my own now. Oh my god, is that a too... That's too wide. Oh my god. Is it worth it? It's not worth it. It's definitely not worth it. God damn. It's jiggling! You know, there's a problem. When the car goes jiggling, you've got to be kidding me. There's lost... I'm risking my entire life in the name of lost underwear. I mean, I guess it's worth it, right? Disc media. Disc media. Seriously. What happened to the oxytocin? Okay, it's relatively undamaged. Oh wait, no. I want to confirm. Yes, I will continue like this. This'll be great. It's gonna be great. Gonna be just... Oh, crap. That is a sharp hill. Ah, I can use my... Oh, crap. I bet someone that's played this game before is probably watching me and going like, no, there's an easier way to do this. But you know what? Sometimes you just gotta live dangerously. Or with 77 kilograms of stuff on your back. Basically the same thing. This is quite dangerous. Yeah, Sam, this is, okay, wait, okay. Alright. And then if I hold everything together, I might just make it. Assuming that I don't have to wade down, a river. Or something, that'd be silly. Oh, this is not gonna work. The river's so wide, too. Oh, you've got to be kidding me.
So bad.
Bruh. I'm sure they taste delicious, and you're just complaining about nothing, Sam. Come on now. Okay. Alright, I see... What? Oh. I see a oh no, I shouldn't have pressed that button because now I see that there's more cargo on the way. Oh, there's more cargo on the way and now I want it. I want it. I mean, I don't think it's physically possible to carry more stuff. This is ridiculous. What in the world is... What is this game right now? But I want to carry that. It's right over there. Come on, it's possible. I can do it. Don't tell me what I can't do. What is that? Is that the baby? Wait, I can get likes from the baby? I mean, okay. Okay, that exceeds my weight limit. I see how it is. All right. It's time to make the most difficult decisions. What do we let go? I mean, I feel like disc-based media is important, but at the same time, it's like, man, do we need that? I don't know if we need that. Just boots. Climbing anchor. Ladders for days. Okay, what is the weight? I see. That's some heavy underwear. Really? I mean, seems kind of important. It's not that important, right? Come on, man. I mean, fall resistant wood. I think what we gotta do. Oh shit. So what happens if I wear stuff that's like over? I see. Okay, that's not great. I don't like that. Okay, I have to offload everything, don't I? All right, 1, 2, 3, 4. And then I'll just be right at the amount of stuff that I can carry, which is perfectly fine, so that I don't have to like walk like a snail on my way out. And I forgot to save again, didn't I? You know, I just feel like if I back out, that probably means that I would like to do the thing that I have already specified would happen, but fine, that works too. I like how there's this little cutscene of me just dropping stuff off. I can respect that. And I can appreciate how visceral that feels. Watching him trying to, like, fix his balance. Oh my god, man. What? Wait, that lowers my weight so I can pick up the stuff. Yeah, I can do it. I can pick up more stuff. I can do it. I can do it. Yeah. I can. This will be great. This will be great. It's possible. I believe. Gotta save that disc media, dude. Oh my god, what is happening? Yeah, okay, okay, okay. Okay, this is interesting. This is very interesting. I'm playing this game with bated breath. I honestly can't believe it. This is actually working. By all accounts, this doesn't make sense. But his climbing animation is too OP. Oh my god, come on, man. You can do it, you can do it. I believe in the Sam Porter Bridges. You can do it. Come on. Can I make the trip? Can I make the trip across the river? I don't think I can. I don't think I can make it. So I'm gonna use up another ladder. Are these expensive or something? Or do I have a limited amount? Oh, shit. That was on accident. I forgot how that worked. What the hell? Okay. Oh my god, I have to press and hold. There we go. That's what it is. Okay. And then I'll use the ladder. Oh, fantastic. Oh, I'm such a genius. Holy crap. Yes. 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 I can't believe this is working. Yes. No, Sam. You can do it, man. I believe in you. Yeah? Okay. Wait a minute. Oh, okay, okay. Ooh, yeah. He gets it. He knows what's up. He knows we narrowly avoided death just now. Come on, you can do it. We're almost there. Almost there to Sam. Come on, man. Yes. Oh my god, I feel so powerful right now. Oh my god, this shouldn't work. No, 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 don't choke, don't choke. No, no, come on, 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 come on. Yes! Yes! Yeah! Make that delivery. Deliver requested the cut. No, wait, hold up. Deliver. Unusable. What? Hold up. Okay. I will deliver that cargo first, sure. Wait, are you telling me that this random shit that I'm carrying on my back is not... Can I just not send it here? Wait a minute.
To know what it meant for the organization, or the UCA, or us. The oxytocin will take the edge off, and that'll be a big help. Won't be long before we're back on track. You may have luck. This is great. Thank you again. Yeah, everything looks to be in good order. And soon our way station will be too, I hope.
Okay. Okay, but what about my other deliveries, though? Wait a minute. Oh my god.
You're with the sec.
Wait, whoops.
Dead. Caught in the void out.
No way. That one blast got them all? And what, they just sent you out on your own? I mean, if it was just a delivery, that'd be one thing. Porters come through from time to time, but... The second team was meant to bring the Cupid. They were going to connect us up. 3 years we've been waiting for help. 3 years. And
Well, I got it.
Shit. Tell me you brought more than the oxy, at least.
I've got the Cupid.
You... Really? So it's true. They finally got it working. Well, then you just might be the answer to my prayers. So what are you waiting for? You put us on the goddamn grid.
Alright. If you insist. Floaty keychains. Activate. Or floaty dog tags, I guess. Oh, random math. And like... That's gonna connect America. Mathematics? No. Duh, baby. I'm just gonna elect to ignore the fact that there's such a huge chunk of the map left. It's only been 3 hours. Or has it been already 4? Oh my god, it's light outside. Oh, it's really bright outside. Damn, is this what morning looks like? This is why I needed to do this. I haven't seen morning in forever.
Connection to capital, not city, confirmed.
Okay.
It's really happening. Just like Amelie promised. We can finally do what we came here to do.
Hell yeah.
You're headed west, right? Gonna be a lot of people happy to see you. Guess you'll keep on till you hit the coast, huh? Which means you'll get to see Amelie in person. Oh, that's something. That is really something. Me, I've never met her. Only seen her holo messages back when I was with the first expedition. Well, anyway, you best be careful on the road.
Yeah, I will. Considering the amount of fucking stuff on my back right now. This is ridiculous.
Thank you, Sam. By bringing that way station into the Cairo network, you've put it in direct contact with those of us back here in Capital Knot City.
Right. What's next?
West of the way station are a couple of structures our previous expedition put in place. And beyond them is your next destination, Port Knot City. It's on the shore of a crater lake that formed after the first void out. Your objective is to link up Port and Capital Knot. But to do that, you'll need to utilize our facilities as additional waypoints. They'll be essential for establishing a stable connection. Given the distance we need to cover, we're looking at using a distribution center and a power station, as to which you should head for first. Start with the distro center.
I mean, you literally won't let me do the other way because the goddamn map doesn't even make sense to go to the other one first, so... Great. think I would like to hand off my lost cargo.
Your hard work's paying off, Sam. Thanks to the increase in network capacity, HQ is now in a position to provide you with additional materials and support. Should prove useful.
Okay. Right, but yes, there we go. This is what I needed to do. Oh my god. Oh, it's finally done. Oh, there you go. Here you go, people. All of your stuff. All of it. It's all here. All the disc, all the... Okay, we had to toss aside the underwear. I'm sorry. Okay, jeez. Oh my god, look at this shit. This is ridiculous. I will now receive more likes. Great. Rare metal delivery. Okay, sure.
Sam, if you're in the middle of a run and decide you need to secure your cargo temporarily or entrust it to another porter, you'll want to make use of a post box. In addition to cargo, post boxes can also be used to share gear and weapons, if you're so inclined. Now, I'm sure you've used them before, but humor me and build one. Maybe between Capital Knot and that way station. If you're lacking a PCC, I'm afraid you'll just have to scrounge it up yourself.
Maybe. Okay. Alright, yeah.
To build a post box, you'll need to use a PCC. It goes without... Sam, you can use that PCC to build a post box.
Okay, yes, that's what you just told me to do. Thank you, Die Hardman. I understand. Yes, thank you. Alright. Load up everything. Wow. Okay, that's much more manageable. Okay. Great. Now that I don't have as much of an absurd load, this might just work out. Yes, what is it, Die Hardman?
Head to the site I designated and build that post box, would you? You should have no...
Okay. I'm gonna let you finish. And then once you are done with everything, I'm going to save. And then I'm going to get out of here. Goodbye. Thank you, Die Hardman. Very cool. Quick game. I'm out of here. And I'm also done with the stream for now. I'm going to set up the next stream really quickly, and then we'll dive right into more studying. But for now, we're just going to close it. I think we can keep on playing Death Stranding, honestly. So that's good. That's nice. All right. I'll see you on the flip side. For now, bye.